all my wonderful PU2 champions. I hope you're doing great. So many children are struggling with biology with a huge syllabus out there, which is exactly why we thought of doing this type of video where, dear children, there will be chapter-wise question, 1 marker, 2 marker, 3 marker, 5 marker, whatever comes from those chapter. It is going to be covered up over here, and this is for everyone who needs that company to study. So there will be live that is ongoing, but please remember this is actually a recorded video. There is no anything that is being hidden over here. It's actually a recorded video which we are putting in front of you in the form of live, such that you have a proper time setting out there and you study with me throughout this particular lecture. So I hope this helps you really well. Make sure you stay consistent, make sure you do each and every chapter out there, and if at all you think that this particular video was useful, definitely let us know. If you have any doubts, definitely let us know about that as well. Yeah, so wishing you all the best. Please take care of yourself. Let's go ahead. So let's go ahead with the first chapter that we have, that is sexual reproduction in flowering plants, and here goes the first question. Statement number 1. Pollination does not guarantee the transfer of right type of pollen. Pollination means what? Transfer of pollen grains from the anther to the stigma. Now, it can be right type, it can be wrong types coming from some other flower and things like that, right? Come from other species also altogether. So, pollination does not guarantee the transfer of right type of pollen. Yes, it does not. This is actually true statement, right? So, this is true statement. Next, compatible pollen pistil interaction guarantee double fertilization. Listen to me. So, basically, onto the stigma, the pollen grain is going to come and land, right? Now, they form the pollen tube. And the pollen tube will be coming over here into the ovary. Inside that you have the ovule. Inside that you have what? The female gamete. That is the whole story. Pollen tube will be having the male gametes as well, right? When the male gamete comes and fuses with the female gamete, you say that the syngamy has taken place. Zygote is being formed. Yes, that is the first fertilization. Now, the next fertilization happens when the next male gamete comes and fuses with the polar nuclei. That is known as triple fusion, because 3 nuclei are being fusing up there, right? When 2 times fertilization happens, once with the female gamete, another one with the polar nuclei, my dear children, we say that double fertilization has taken place. And here the statement is, whenever there is a compatible pollen pistil interaction, okay, it will definitely guarantee double fertilization. Do you think so? What do you think so? Suppose it's not just one pollen grain, but multiple pollen grains landing. They are all forming tubes, my dear children. So does that mean that all of them are going to do double fertilization? No, Baba. There is no guarantee that compatible pollen pistil interaction will definitely make sure the double fertilization will occur. Sometimes the pollen tube form, but they stop right there. Other the pollen tube can form, go ahead. So we never know, that can happen, that may not happen. So there is no guarantee to it. So this particular statement is wrong, because it says that the compatible pollen pistil interaction guarantees double fertilization. The answer is wrong. So statement 1 is correct, but what about statement 2, Baba? Statement 2 is going to be wrong. Understood? Read the question carefully. Okay, and then everything is going to be sorted. Yes. The next question that I have over here is statement number 1 says, apomixis is the production of seeds from unfertilized ovules. Is it? What do you mean by apomixis? In apomixis, what is going to happen, my dear children? Suppose this is the ovule. Inside the ovule, what do you have? You have the embryo sac, right? Inside the embryo sac, what do you have? You have the female gamete. You have the female gamete, you have 2 antipodal cells and blah blah blah, so lots of things like that. Usually the female gamete which is there, it is haploid in nature. It has half the number of chromosomes, and it actually waits for the male gamete to come, so that both the haploid cell comes together to form a diploid zygote. Diploid zygote. That is how it usually goes. But you know what? There is a change in the trend. Over here, this particular seed thinks that, okay, or basically this particular plant thinks that, okay, I do not need any male partner to form the zygote. Why not? Why not? Here, instead of being haploid, let me be just diploid. Let me be just diploid. So the egg cell which is there, the female gamete which is there, my dear children, what is going to happen? It is already diploid in nature. So does it need anyone else to become a diploid zygote? No, Baba. It itself transforms into what? A zygote. a zygote, and when a zygote is formed inside the ovule, you will now not call this as ovule, rather you have a different name for it, which is known as a seed. Whenever a zygote is formed inside the ovule, you will now call this as a seed. Over here, even without the help of the male gamete, it formed the zygote, and now it is going to be known as a seed also. So apomixis is the production of seeds from unfertilized ovules. Do you think that this particular ovule underwent fertilization? No, there was no fertilization. So this is correct. It's the production of seeds from unfertilized ovules. This particular statement is absolutely correct. Now next thing, embryos produced from the apomictic seeds are not generally identical to the parent plant. What? Listen to me carefully. Here only a female parent is being involved. Female gamete is being involved, right? There is no fusion of gametes. If there was fusion of gametes, I'll say it's sexual reproduction that is happening. And at the end products of sexual reproduction, you always get someone new who is having a combination of genetic material from both the parents, right? At that particular point of time, I'll say that they are not generally identical to the parent. For example, I'm not exactly like my mother or my father. Like it's a hybrid, right? Why? Because sexual reproduction is being followed in the case of human beings. Now here also, do you think sexual reproduction is happening? There is no fertilization, Baba. So there is no sexual reproduction. If there is no sexual reproduction, can I say it's a type of asexual reproduction? Yes. And in asexual reproduction, what happens? Only one parent is involved, and the progenies which are going to be formed, they are going to be just like that parent only, because they are just passing the genetic material, right? So they are going to basically form clones, and I'll say that they're genetically very much identical. So embryos produced from apomictic seeds are not generally identical to the parent plant. Wrong statement. Again, a wrong statement. Statement number 1 is correct, and statement number 2 is what? False. Got it? Yes. Now, the moment you feel that, okay, ma'am, speed is going a little bit fast, what you can do is go to the settings. Okay, I do not know on what side it is. Go to settings and make my speed less. The probable reason that I might be going fast is that there are so many questions to be completed, and probably I'm thinking in the back of my head that you do not have much time to watch this video. So that could be the only reason. It's not because I need to do it fast. I am usually someone who goes really slow while teaching. Okay, so it might be just because of for you guys. So please forgive me if at all it is happening, and please change the settings out there. Yeah. Okay. Now, even in the absence of pollinating agents, seed setting is assured in. Seed means fertilization has taken place probably, right? That means zygote has to be formed. That is what you mean by seed formation. Now, zygote to be formed, obviously you need the pollen grains to come and land on the stigma, such that it can give the male gametes, and the male gametes and the female gametes can fuse. But who will bring the pollen grains? Obviously it has to be some pollinating agents. But here it is said that even in the absence of pollinating agents, seed setting is assured. Seed definitely is going to be formed in which plant? Cucurbita, papaya, commelina, maize. In which condition do you think this is definitely going to happen? Without pollinating agents, seed setting. My dear children, if suppose the flower is like this, closed, it has the female part, it has the male part. From the male part, the pollen grains lands on the stigma, that is on the female part. Here, do you need any pollinating agents? No. That means these flowers has to be what? Closed. Ma'am, are these flowers for real? Yes. What do you call them? You call them as clistogamous flowers. Okay, you call them clistogamous flowers. Here Cl stands for what? Closed. Here Cl is going to stand for closed. Okay, so definitely pollination is going to happen, and after that seed setting is also going to be happening. Yes, the only drawback that is going to be there is like it will be just like the parent plant. So parent plant, suppose it has some toxic character, it cannot be pest resistant or this and that, any bad characters are there, it will be just passed on. Okay, and in your NCERT there are 3 plant's name given, and you have to remember it. I remember it like this: cow, but with a V. So C for Commelina, O for Oxalis, and V for Viola. Commelina, Oxalis, Viola, they have 2 types of flowers. One is the closed flower that you just saw, and one is the open flowers which are there. And here the normal type of pollination will be happening. They will require pollinating agent, they sometimes not require pollinating agents. Okay, so it all depends. Anyway, it is going to be open. This type of open flowers are known as chasmogamous flowers. What do you call them? Chasmogamous flowers, and they are known as clistogamous flowers. They are usually found towards the root region. This is given in your NCERT. Low key, sometimes you might not see it. So please understand, they are mostly found towards the root. Okay, probably within the soil itself they might be found, but just above the soil they might be found. Clistogamous flowers. Okay, so over here you have the answer, and that is Commelina. Commelina is the answer for this. And as I said, here I have put in a real-time picture. These are the open flowers that are known as chasmogamous flowers, and here you have the closed flowers. If at all you see, it's just above the the soil, yeah, or it can be beneath that also. Fine. Yes. Next question. Select the correct sequence of anther wallets. Anther, if at all you have seen, my dear children, there are multiple wallets, 4 of them are there. Okay, I'll require you to remember it like this. It's very easy. So have you been to ear, nose, throat doctor? Whenever you have any problem in the ear or nose or throat or anything like that, you go to an ENT. You go to an ENT. Similarly over here, you have EMT. Okay, so E for epidermis, that is going to be the first layer. So let's see. Epidermis, epidermis, epidermis. So this one is the odd one out. Okay. Next one, after epidermis, you have endothecium. Endothecium, endothecium, endothecium. Only this one has. Let's go ahead. After endothecium, you have middle layer. Okay. And after middle layer, you have tapetum. Oh my God. So your answer for this particular question is going to be B. Always remember, epidermis, endothecium, middle layer. These 3 of them are best of best friends. Why? Because when the anther, which has pollen grains, my dear children, the anther has to dehisce, basically break open, so that all the pollen grains go outside. Who are the 3 members of this particular gang who break open for the anther? It is basically epidermis, endothecium and middle layer. So I'll say that they are responsible for dehiscence of anther or for the breakdown of anther. What about tapetum, my dear children? Tapetum, on the other hand, is Papa guy, is there for nourishment. Nourishment of what? The sporogenous tissue. So it's a nummose layer, right? So here you have the sporogenous tissue, which is made up of multiple cells, microspore mother cells, which will later form the microspores, which will later form the pollen grains. So to provide the nourishment to that, you have tapetum. There are certain features of tapetum that you need to remember. First of all, since it has to provide nourishment, it will be having a very dense cytoplasm. Okay. Apart from that, what about the nucleus? Nucleus, suppose it's a cell, they have 2 nucleus. That means it's a binucleate condition. What condition? Binucleate condition. 2 nucleus is found over here. So that means karyokinesis took place. Nuclear division is known as karyokinesis. That took place. But cytokinesis didn't take place. Cyto means cell. Cell division didn't take place. Okay. The karyokinesis took place out there. So then cytoplasm is there. They have binucleate condition. This is a feature of tapetal cells, which is there for nourishment of the sporogenous tissue. Okay. Or the cells of the sporogenous tissue. Works? Works, ma'am. Awesome. Okay. Now, so I have spoken about this already and I have given you, this is a question bank question. Number of the question bank question is also given down there. Now, select the correct sequence of events in microsporogenesis. In microsporogenesis, how is the thing going on? So let me, please try it out by yourself. Pause the video. I'm going to go ahead. You might see the answer, but I just want to check if at all an animation is given there. Yes, it is there. So I'll explain you with this animation. That is much easier. So my dear children, as I told you, in sporogenous tissue, you have microspore mother cell. Microspore mother cell. They are what? They are diploid in nature. They are having diploid number of chromosomes, right? They undergo meiosis. Meiosis happens in 2 stages. First meiosis, second meiosis. When the first meiosis happens, the number of chromosome, it becomes half. That is why you call meiosis as reductional division. Okay. Now the next meiosis happens. There are 2 cycles that happens. First meiosis, second meiosis. Again, do you think it will get half? No, Baba. It will remain the same. N. Only thing that you need to remember is that these microspores which are there, they are together. They form a tetrad. Tetrad means 4 of them are together over here. They are microspore tetrad. That is how it is being formed. Later on, when the anther is going to get dehydrated, when the water is going to be lost, all of them get separated. And after getting separated, what do they form, Baba? Each of them individually are going to form our own pollen grains. Our own pollen grain. That is the story. So sporogenous tissue first, then microspore mother cell, then microspore tetrad, and finally you got the microspores that will later form the pollen grains. When these type of question comes, Read all the options carefully. All the options carefully. So they might miss this thing, but write all this thing in sequence, right? So please check out for that. If at all this is being missed and everything else is correct, it's okay. It's okay. If at all this is common for all, sporogenous tissue has never been mentioned over here. You should know what is the next thing. You get it? That is what I am trying to tell you. Yeah? Yes. Okay. Now the next question. From the statements given below, choose the option that is true for a typical female gametophyte of a flowering plant. What is a female gametophyte? The pollen grains that we saw, my dear children, do you think that was a male gamete? No. It forms the male gamete, which is why it is known as male gametophyte. Similarly, in the case of females, you have a female gametophyte which is going to form the female gamete. This female gametophyte has a name. It is known as what? Embryo sac. It is known as embryo sac. And it looks something like this. So embryo sac also, let's read the options first and then I'll go ahead with the answer, because I think again animation is given over here. So it is 8 nucleate and 7 celled at maturity. 7 cells are there but 8 nucleus, how can that be possible? Do you think that is possible? Okay, let's see. It is free nuclear during development, as in the nucleus. Whenever a cell is here, so many cells are being formed, right? 7 cells are being formed. It says that the nucleus of these cells first divided and then later on the cell walls formed around it. That is what you mean by free nuclear division. So it's not like immediately cell wall was being formed. First of all, the nucleus divided, later on the cell wall was being formed. That is what you mean by that. Okay, free nuclear division. Now, it is situated inside the integument but outside the nucleus. What do you think? It is situated inside the integument but outside the nucleus? According to what I know, it goes like this. So you have the ovule. The outermost thing that you have is the integument. Inside that, you have the cells of the nucleus. And inside that, you have the embryo sac being formed. So this is the embryo sac. Okay, there is a female gametophyte. Outside that you have nucleus. Outside that you have integuments. But what is given over here? It is situated inside the integuments, sure, but outside the nucleus. Embryo sac is not outside. Embryo sac is inside the nucleus also. So this is also correct. It has an egg apparatus situated at the chalazal end. What is egg apparatus? Egg, my dear children, the female gametophyte will be having a female gamete, right? The female gamete is guarded by 2 cells which are called as, so what do you call them as? I forgot the name. Oh my god. Synergids. Yeah, synergids. So synergids plus the egg cell, that is the female egg, you call this as what? The egg apparatus. And egg apparatus can never be located at the chalazal end. Mostly, the most important things are located towards the micropylar end. So always remember, the integuments have an opening over here which is known as micropyle. Okay, and the opposite side of this is known as chalazal end. Everything important is going to happen up over here. Your egg apparatus is going to be forming up over here on the micropylar end, not on the chalazal end. So this is definitely going to be wrong. But what about these 2 options? We have to see that, no? Is it 8 nucleate and 7 celled at maturity? Let's see that. Okay, so over here, understand, so just how you have microsporogenesis happening in the case of males, similarly you have megasporogenesis happening in the case of females. At the end you get 4 megaspores, okay? 3 of them are going to degenerate, only 1 is going to be left, and when the one which is there, it is going to go ahead and form the embryo sac. You call this development as monosporic development. Why? Because only 1 guy is involved over here. Monosporic development, right? So this is the cell. This is the megaspore, the only megaspore that is left. Here you have the nucleus of that. The nucleus undergoes division. How many times? Thrice. So first division, you got 2 nucleus. Second division, again you got 4 nucleus. Third division, 1, 2, 3, 4, 5, 6, 7, 8. 8 nucleus are going to be formed. At the end of this, you get 8 nucleus, my dear children. Very, very, very important thing. And over here, you do not see any cell wall formation, which is why I'll say it is what? It is free nuclear condition. It is free nuclear condition. That means there is no cell wall formation around it immediately. And 3 mitotic division is what you say. 3 mitotic division. Now, later on the cell wall and everything is going to be formed, and you will see something like this. So basically, 3 of the cells go up there at the chalazal end. This end that you see is known as the chalazal end. And these 3 cells with 3 nucleus, you call them antipodal cells. Similarly, 2 cells come over here, you call them synergid cells, each of them having their nuclei. Okay, so 3 plus 2, 5 cells are done. 1 more cell comes in, and that is the egg cell. Now, that also have nucleus. Yeah, so 5 plus 1, 6 cell. Now where is the 7th cell, my dear children? Where is the 7th cell? This whole thing that you see over here is the 7th cell. So you have 7 cells, but this 7th cell has 2 nucleus. Instead of having 1, we have 2 nucleus. So all of them individually had 1. Until 6 it was fine. Until 6 of them it was fine. Now in the 7th cell there are 2 nucleus. So this is going to be the 7th nucleus and this is going to be the 8th nucleus out there. You call them as polar nuclei. 2 polar nuclei of the central cell. So isn't it 8 nucleate and 7 celled in condition? The answer is yes. Okay, the answer is yes. This is very, very important. So all the statements eventually are going to be correct. So let's see. So A and B, yes, it is correct. A and B is correct. Okay, and what about so did I actually say it is correct? Oh my god, I explained you something else, and if at all I have said that this is correct, I'm so sorry. Okay, so A and B are the only one which is correct because it is situated inside the integument but outside the nucleus. No, that is not true because it is inside the nucleus also. Yeah, and as egg apparatus situated at the chalazion end. Baba, this is the egg apparatus that you see. Okay, just a second. Yeah, this is the egg apparatus that you see. It is towards the micropylori end, not towards the chalazion end. As I told you, everything important happens towards the micropylori end. Even the division starts in the micropylori end stage only. Okay, now let's go ahead. And over here we had the 7th question. Identify the correct sequence of arrangement of cells in mature embryo sac micropylori end. Micropylori end. So in micropylori end, what do you have? Synergids is the first thing that you have. Then you have the egg cell. Then you have the very big central cell. Then after that, at the top, what do you have? You have the antipodal cells. So this ends the answer. Simple. Antipodal cells cannot be in the micropylori end. Wrong. Synergids, then central cell, then egg cell. No. Synergids and egg cell comes together. You call this as egg apparatus. Antipodals, again wrong. So basically, your only answer for this particular question is what? A. Yes. Again, am I explaining? I'm so sorry. Yes. Question number 8. In viola, both autogamy and geitonogamy is prevented. Viola. Commelina oxalis, viola. Cow. Remember plasmogamous and clistogamous flowers? Here it is said that in viola, both autogamy and geitonogamy is prevented. What do you think? So, just give me a second. Okay. So, we are right over here. Yeah. So, viola, my dear children, here in this particular case, listen to me very carefully. So there can be open flowers which are something like this, right? And you have autogamy. Autogamy means self-pollination. So that is definitely going to happen. What about geitonogamy? Geitonogamy means, suppose this is a flower, there is another flower on the same plant. So from here to here, if things are going from the same plant, 2 different flowers are undergoing pollination, this is known as geitonogamy. And here it is written in viola, both autogamy and geitonogamy is prevented. It's wrong, right? It's wrong because it can happen. Viola has closed flowers and also open flowers. Open flowers can do all this thing. Even in closed flowers, there's only one way for fertilization or pollination, pollination, and that is autogamy. So autogamy cannot be anyway prevented. So this particular statement is wrong. Now statement number 2, geitonogamy is similar to autogamy since pollen grains come from the same plant. Is it like that? So in autogamy, again, in the same plant, okay, in the same flower it is happening. In geitonogamy, same plant, different flowers. But anyway, same plant means same type of genetics are going to happen. Gametes are going to be male and female gametes. Both of them have the same genetics, right? So basically, can I say genetically it is similar? Yes. Geitonogamy is similar to autogamy since pollen grains comes from the same plant. Yes. Genetically, actually, it is similar. If at all they had mentioned that also, it would have been nice. But please read the options really, really carefully. Sometimes they could twist and turn and they could give you something else which is actually wrong. Okay, so here it is correct. It is similar to autogamy because they are mentioning what is the reason for that. Pollen grains are coming from the same reason. They are not saying geitonogamy and autogamy are same. They are saying it's similar, not same. You get it? So there is some differences, but there is some similarity as well. That is what you mean by that. So this particular statement is correct and the other one is wrong. So I'll say statement 1 is incorrect and statement 2 is correct. What do you mean by xenogamy? Basically, geitonogamy means 2 flowers of the same plant. Xenogamy means 2 separate plants itself. So xenogamy and geitonogamy, both of them are cross pollination, but which one of them is true? It is that one that is absolutely true. Got it? Yes. Fine. Next thing, the following picture is an example of. This particular picture is an example of what? This is given in your NCERT and we should know. This is given in a question bank also. Any idea? So over here, my dear children, you need to understand one very important thing. Okay, there are so many gynoecium, as in female reproductive part. You call the gynoecium as carpel, you call this as pistil also. So I'll say there are multiple carpels over here or multiple pistils, but generally we use the term carpels. Okay, so there are multiple carpels over here, and there is one very important thing. All the carpels, okay, all the carpels which are there, they has a unified ovary. Okay, unified ovary. So their ovary is all fused together, my dear children. And you call this condition as syncarpous condition. When they are fused, you call it syncarpous. There is a condition where there are multiple carpels, but all of them have their individual ovary. That is known as apocarpous condition. But over here, it is syncarpous that is being shown. Okay, syncarpous condition. What is this particular plant? Do you know? This is a plant from where you get what do you call opioids such as heroin and morphine and things like that. The name of this particular plant is Papaver somniferum. This is having multicarpal condition and also syncarpous gynoecium. So ma'am, is there a condition like apocarpous as you told? Is the example given in the NCERT? Yes. That example is of Michelia. Have you seen Chambagam? In Malayalam we say Chambagam. In Hindi we say Champak. Okay, so it smells so good. The white color flowers, they have apocarpous condition, and that is known as Michelia. And that is exactly known as Michelia. Okay, got it? So that is apocarpous. This is syncarpous. Do not forget it. Okay, fine. Next one. Yeah. So I have given the picture also. That's me. Okay, so this is Papaver somniferum. This is how exactly it looks. Okay, exactly this is how it is going to be looking, my dear children. Inside that they have the fused ovary, and over here, if at all you see, in the case of Chembagam or Champak, if you open this, it looks like this. Now this is the picture that is given in NCERT. Over here you have multiple carpels and each of them have the individual ovary, they are not fused. Okay, so this is known as apocarpous condition. Yes. Let's get into the first chapter, that is sexual reproduction in flowering plants. Here you get 1 marker, 2 marker and 3 marker as well as 5 marker question, but we are going to deal with 2 and 3 markers. And the first question goes like this: write the significance of sporopollen. I hope you guys know that there are multiple layers in the case of pollen grain. The first layer that we have is plasma membrane. Outside that there is a continuous layer which you call as what? Intine. Outside that there is a discontinuous layer which you call as exine. And the exine which is there, my dear children, is made up of one very important thing, the most resistant organic material which is known as sporopollen. Why do you call it most resistant? Because even if I put alkali, if I put acid, if I give UV radiation, whatever I do, my dear children, the pollen grains will still be safe. Why? Because it is the toughest resistant material out there. And that's exactly why pollen grains can be stored in the form of fossils as well. So you have to write that answer in this particular way itself. That is one of the most resistant organic substances, forming the exine of the pollen grain and protecting them from high temperature, strong chemicals, UV radiation and also microbial attack. Because of its extreme durability, pollen grains with sporopollen get preserved as fossils also. And if at all you can make the diagram of it, it would not take too much of your time if you think it's required. Okay? Fine. Let's go ahead with the next question out there. Give reasons. Micropyle remain a small pore in the seed coat of a seed. Apple and cashew are not called true fruits. Okay, we'll go one by one. First of all, micropyle remains as a small pore in the seed coat. So listen to me carefully. This is your ovule. This is the ovule. Inside that you have the, this is the ovary. Inside that you have the ovule. Ovule has 2 integuments around it, right? And this place where the integuments are not joining, there is a free space out there, that is known as micropyle. Listen, inside the ovule, you will have embryo sac, which in future will be having female gamete. The male and the female gamete fuses. It forms a zygote. You will now not call the ovule as ovule, rather you will now call this as seed, because this is fertilized ovule, which you call as seed. So the integuments which are over here, they will also transform into something else that is known as seed coat, testa and the tegumen. And here in this area, there will be a space that is micropyle. Why do you think there will be a space over here? Such that, okay, the water which is there, the moisture which is there, it can be trapped easily. and whenever the seed needs to go ahead and germinate, it can go ahead with that particular process. So basically for exchange of gases as well as for water intake and things like that, during germination you need the micropyle just like that without any damage out there. Okay, we'll read out the answer and then we'll go to the next question. So micropyle of the ovule persists even after fertilization and becomes a tiny pore on the seed coat. It helps in the entry of water, as I told you, and oxygen during seed germination. It remains unchanged as a small opening. It remains like that. Cool? Yes. Now apple and cashew are not called true fruits. Why? Because in the case of normal fruits, this ovary that you see over here, inside the ovary you have the ovule, right? So ovule becomes seeds. What about the ovary? The ovary becomes the fruit. Right, that is a general thing. Ovary, it becomes the fruit. But in some cases, the parts apart from the ovary can also give rise to a fruit. For example, in the case of apple. For example, in the case of cashew. Over here, what is happening? You might have seen this particular part on a plant onto which the whole plant is resting. This is known as the thalamus. The thalamus also can take participation in the formation of fruit. In that particular case, it won't be known as a true fruit. Rather, we'll call this as false fruit. Why? Because it's not just ovary that is participating in the formation of fruit, but also the other parts of the flower. Got it? So true fruits develop only from the ovary after fertilization. In apple and cashew, the edible part develops from the floral parts other than the ovary. Other floral parts other than the ovary, like the thalamus in apple and the pedicel tissues in the cashew. But in your NCERT, it is all written that thalamus is something that is going to participate in the formation of fruit in the case of apple, cashew, and also strawberry. These three examples are there. ACS, or however, or even if you want to remember it like this, that works. Sac. Okay, this is super important, and yeah. So you can also remember that all the parts which are there, you do not have to go ahead with pedicel and things like that. You can just remember it's the thalamus. Fine. Now, hence they are known as false fruit or accessory fruits, but not true fruits. Works, ma'am. Cool. Next, differentiate between pericarp and perisperm. Okay, what is pericarp? The wall layer of the ovary. When I say, okay, but before that, just a second. Okay, I've shown you a picture also. I think, okay, I'll do this question first and then we'll discuss that. So, this is the ovary, right? Ovary becomes a fruit. Listen, the wall of the ovary is actually known as pericarp. What do I call it? Pericarp. And we all have eaten mango in our lifetime, right? If at all you have eaten mango, you know that. Basically, there is a outside thing that we shed off. What is this known as? This is known as epicarp, or you also call this as exocarp. Okay, then there is a fleshy portion that we eat up. What is that known as? That is known as mesocarp. And then there is a hard thing which is there, a very hard thing which is there, inside which you have the seed. Inside that you have the seed and everything up there, right? So this hard material which is there, it is known as endocarp. Three of them together, you say they are walls of the ovary and you call them as pericarp. You call this as pericarp. Listen, the seed will have its own wall that is separate. This is the walls of the ovary. Now, in some plants, it can be fleshy like this. In some plants, it can be dry also. Okay, or basically in some fruits, it can be fleshy like this. In some of them, it would not be fleshy. You will not be able to have something like this. You get it? But anyway, the outer layer which is there, totally you call this as pericarp. Now, what is perisperm? That is also a question. So for that, again, I have to move on to this particular picture. So if at all you see here, this is the ovule. Inside the ovule, one cell is there which is going to undergo division and later it forms the embryo sac, right? So over here, just imagine that embryo sac is being formed. You see the term nucellus over here. There are multiple other cells which is just there for providing nourishment. These cells are known as cells of the nucellus. These cells are known as cells of the nucellus. So once an embryo sac is formed over here, okay, once an embryo sac is formed over here, what usually happens is that the cells of the nucellus are being utilized up very much and it goes off. The cells of the nucellus, it goes off. But sometimes it can persist there. It can just be there, my dear children, providing nourishment to the embryo as well. So until now, until the formation of embryo sac, it usually is there. But after that, it usually get disintegrated, it's being used up and things like that. But in some cases, it can still persist. The cells of the nucellus can persist until embryo is being formed, okay, for the help of the embryo growth out there. And that is known as perisperm. And where do you see it? You see it in B square. B square stands for beet as well as black pepper. beet as well as black pepper. Okay, so differentiate between pericarp and perisperm. The wall of the ovary develops into the wall of the fruit and is known as pericarp. Epicarp is the outermost layer. Okay, I told you mesocarp is the middle layer, endocarp is the innermost layer. Now perisperm is what persistent nucleus is known as. Persistent nucleus is known as perisperm and it is found in black pepper as well as beet. B square. Done? Done, ma'am. Perfect. Next question. Distinguish between albuminous and non-albuminous seed. Albuminous seeds means sometimes. Okay. So we have two things that is being formed at the end of double fertilization. Once double fertilization happens, we get two things. One is zygote and the other one is the endosperm. Primary endosperm cell is something that we get, they further divide to form a tissue of endosperm, right? Why do you have endosperm? For the nourishment of zygote, such that the zygote could later form the embryo. That is why we have the endosperm, for providing nourishment out there. So what happens is that when this transformation happens, when zygote forms the embryo, the endosperm is being used up. What happens? It is being used up. Sometimes it is used up completely. And in that particular case, I'll say there is no endosperm left behind. And these seeds which do not have any endosperm left behind, I'll call them as non-albuminous seeds. On the other hand, in some of them, the endosperm might still persist out there, and that is known as albuminous seeds. Okay, now that is the first difference that we have. Second, it provides nutrition to the embryo from the stored endosperm. So in albuminous seeds, since endosperm is there, that will be providing nutrition. But in the case of non-albuminous seeds, endosperm has been utilized up. No, now what is there? Listen, so endosperm has been utilized up for the formation of embryo, but embryo thinks that, okay, I have to have my stored things. So basically, they will be having their storage units, which are known as cotyledons. So these cotyledons, my dear children, will now provide nourishment to it. I do not have endosperm, but I have cotyledons. So nutrition is stored inside the cotyledons, not in the endosperm. Done. Now example is there. So usually we just consider the dicots which are there. Dicots which have two cotyledons. We assume that, okay, endosperm is totally utilized, probably it is to make the cotyledons out there. So dicots come under non-albuminous and monocots comes under albuminous. But it's not like that. There are exceptions to it as well. For example, castor. Castor which is there, it is actually a dicot. It has two cotyledons, but it has still endosperm also. You get it? So dicot, castor is there, which is dicot. Wheat, maize, barley, all of them are monocots. They will have just one cotyledon. Rest of it is just endosperm itself. So in monocots, definitely endosperm is still persisting. There is some exception to it. You have a dicot over here, which is albuminous, and that is castor. Now non-albuminous, you have bean, pea, groundnut. All the dicots, generally all the dicots come under this only. Dicots mean two cotyledons will be there. So they come under this bean, pea, groundnut, all of it you can break into two. Right. So always remember like that, but know the exceptions as well. Fine. Got it? So this is one very important question. Next one, draw a label diagram of mature two-celled pollen grains. Over here you have to explain everything properly. Please know that this is a pollen grain. Inside the pollen grain you have a very big cell that is known as vegetative cell. Okay, inside that the nucleus is, the shape of the nucleus is what? Irregularly shaped nucleus. Then what do we have? We have generative cell. What is the shape of the nucleus over here? Elongated nucleus. This is what? The plasma membrane. This is the plasma membrane. Then you have what? A continuous layer. What do you call this layer as? Intine. Intine is made up of what? Pectin as well as cellulose. PC. I already told you, Priyanka Chopra. You can remember like that. Then you have exine. What is exine made up of? Exine is made up of one very important thing which is known as sporopollenin. Yeah, and over here you have a space that is known as germ pore. Okay, generative cell later divides. How do you get two cells over here? With the help of mitosis. Similarly, one more mitosis happens. The generative cell divides into two, and now you have two gametes. You will be having two gametes over here. So in total, how many cells? One. That is vegetative cell, and 2 and 3 gametes. 2 and 3 will be there. So 3 cells will be there at the end of it. So 60% of the angiosperms, the flowering plants, they release the pollen grains in this 2-cell stage. But 40%, rest 40% release the pollen grains in 3-cell stage, where the generative cell have divided into 2 again. Got it? So know these basic things. And the vegetative cell which is there, it is also known as tube cell. Why tube cell? Because my dear children, male gametes are being formed. It has to form a pollen tube now to transfer the male gametes. So the pollen tube is being formed with the help of intine. So intine which is there, it basically moves out from here through the germ pore. But who gives the cytoplasm and everything? Vegetative cell gives the cytoplasm. That is why it is also known as tube cell. It helps in the pollen tube formation. Yeah, so inner content is from vegetative cell. And then through that pollen tube, you have your male gametes being transferred as well. Works? Works. Fine. Now, draw a label diagram of LS of an embryo of a grass. By grass, what do you mean? You mean a monocot. You mean a monocot. Okay, so you have to draw the diagram of the LS of an embryo. It's very, very simple. Listen, it's an embryo. So obviously, it will have an embryonal axis. That means there will be an area that will be giving rise to what? The shoot. There will be something that will be giving rise to what? The root. And the radicle always will be having a root cap. Why? Because it has to go into the soil. Right. Apart from that, since it is a monocot, it will just have one cotyledon, and that cotyledon is known as scutellum. It is known as scutellum. Apart from scutellum, what does it have? It will have little amount of endosperm still left behind. Little amount of endosperm still left behind. And since it is a monocot, it will be having few extra layers around the plumule, which is known as coleoptile. What do you call it? Coleoptile. P over here and P over here. Similarly, over here, below the radicle, you have coleorhiza. R over here, R over here. You can remember like that. Now, ma'am, how do we come with this particular picture? Very simple. Look at this. Okay, look at this particular image. So you have got the basic idea. Plumule is there. Over that you have coleoptile. Radicle is there. Root cap is there. Coleorhiza is there. On one side you have scutellum, and then endosperm is there. Right? Now we are going to have this diagram being made. Make a whale over here. This whale has a fin. Then with this whale has a tail and a smaller fin right there. This fin that you see over here is known as scutellum. That is your cotyledon. This is another cotyledon which tried to form but was not able to do that. Okay, so this is rudimentary cotyledon out there, and you call this as epiblast. Fine. Now listen. Over here, make a triangle like this. Color it up. Again, make a triangle like this. Color it up. The bigger triangle which is there, you can just mention it's coleoptile. The smaller triangle which is there, you can just mention it is what? Plumule or shoot apex. Right? Now, you join this, let it go here, let it come back, and then go to one particular tail, let it come back and join over here. Okay? Here, this particular thing that is there is radicle. The radicle which is there is covered by root cap. And the root cap which is there, again make a triangle like this and mention it is covered by coleorhiza. And then your diagram is totally done. That's the only thing that you need. Grass is a monocot, do not forget it. Cool? Cool, ma'am. Perfecto. Now draw a neatly labeled diagram of typical anatropous ovule. How do you make that? Anatropous ovule is basically a little bit, the programming of it is a little bit different. Listen to me carefully, it's very simple. You have the ovary. The ovary has a placenta. Why? To provide nourishment to the ovule, right? So this is the placenta that is there, and the placenta is attached to the ovary with the help of something that you call as funicle. You call this structure as funicle, right? The ovary will be having integuments around it. How many? 2 of them. Cool. Outer integument, inner integument will be there. By the way, the point of attachment which is there, you call this as hilum. The point of attachment of funicle is known as hilum. Yeah. Inside the ovule, what do you have? You have the cells of the nucellus. You have the cells of the nucellus and also one megaspore mother cell which is going to form the embryo sac later on. Right. That is a general thing. That is how it is there. The opening place which is there, you call this as micropylar end, or you call this opening as micropyle, and you call this end as micropylar end. The opposite of micropylar end will be known as chalazal end. Chalazal end. So this is a typical ovary which is there. Suppose I am shifting the position of the placenta over here to here, and the funicle is right over here, and the hilum is right over here. Now you have got something like this that is known as anatropous ovule. Look, this picture and this picture might look different, but it's same only. So this is your embryo sac which is there inside the ovule. Here you are opening, that is micropyle, okay, and there are 2 integuments out there. This particular thing that is there is funicle. It is attached to the point that is known as hilum. Placenta is not being shown over here. Got it? And the opposite end is known as chalazal end. So that's all. Okay, that's exactly which is there in a different way. It is being put down. That's all. Works? Works, ma'am. Fine. So now what do we have? Why are some seeds referred to as apomictic seeds? Mention its advantage to a farmer who uses them. Okay, why do you think seeds are known as apomictic seeds? Any idea? Any idea why some seeds are known as apomictic seeds? Very simple. I'll explain it to you. Okay. So I told you there is ovary. Inside that you have ovule, right? The ovule which is there, usually when it undergoes fertilization, inside that you have embryo sac, and when it undergoes fertilization, you say that basically it's now a seed out there. Suppose there is no fertilization that is happening. There is no fertilization that is happening up there, okay? And still a seed is being formed. Then, is that possible? Yes, that is possible. So usually what happens is that you have a male gamete and a female gamete that fuses together to form a zygote. So male and female, the male which is there, it will be haploid. The female which is there, it is also going to be haploid. Haploid comes together. That is how you get diploid zygote. What if the female gamete which is there, it is already diploid in nature over here? Somehow the female gamete which was formed inside the ovule is already diploid in nature. Do you think anyone else to fertilize it? No, Baba. It can transform itself into what? Particularly zygote. And finally, when a zygote is being formed here, is there any fertilization involved? No. I will call this as a seed. A seed without fertilization, which is very unique. This process is known as apomixis. Okay, this process is known as apomixis, and these type of seeds where no fertilization occurring, still you get a seed that is known as apomictic seeds. Okay, so some seeds are known as apomictic seeds because they are found without fertilization, that is without fusion of gametes. The embryo develops without meiosis and syngamy, that is without meiosis. When meiosis happens, that's when it becomes half, right? So here there is no meiosis, there is no halving of chromosome, and there is no even fusion of gametes as well, producing genetically identical plants to the parent plant. So obviously only one parent was involved, and this female particular plant is only giving rise to the progeny. So whatever features were there in this particular plant, that is going to be transferred to the baby plant as well, right? So can I say genetically it will be similar? Definitely, which is why I'll consider this as a type of asexual reproduction. I can never consider this as sexual reproduction. I'll call it asexual. Why? Because there is no fusion of gametes, and genetically the children are identical out there. Where is this very much common? It is very much common in Asteraceae. Please remember this, and also grasses. In Asteraceae and grasses, this particular process is very, very, very common. Okay? Now, is this the only way in which it can form like this? Like a seed can be formed? Listen. Apomixis means you have to form the seed without fertilization, right? So one is where the female gamete itself, was diploid in nature. The other case is where I already told you, so I'll just rub it off and I'll explain you. There is ovule, and inside the ovule, sometimes the nucellus persist. The nucellus, they persist. Nucellus is a normal cell. So a plant, if at all I say it's diploid, the cells of the nucellus will also be already diploid in nature. Inside that you have an embryo sac where the female gamete is there. Suppose the cells of this nucellus gets inside, multiples of them, they get inside. What is going to happen? Now, once they get inside, they are going to form embryos over here inside. Again, without fertilization, what is happening? The same thing is happening. Embryo is being formed. That means it is now known as a seed, right? So this is also how it can happen. And this condition where multiple embryos are being formed together, this is apomixis only, but this condition where from nucellus, entry of the nucellus, multiple embryos are being formed, over here I will say that this condition is known as polyembryony. So in apomixis you can see polyembryony, you may not be able to see polyembryony. You get it? There's a chance. Polyembryony is literally very common in citrus fruits. For example, orange, mango, etc. This is something that is very common. Fine, got it. Now, advantages of apomictic seeds to the farmers. Hybrid vigor is maintained. Suppose there is a plant which has all the very important features out there. Okay, this particular plant has very important features out there. What is going to happen? Now what is going to happen? Basically, I am creating clones of it. All the important features are going to go ahead. Similarly, right, it will not change at all. There is no fertilization that is happening. There is no scope of changes. So farmers can reuse hybrid seeds every season without the loss of yield. That is one very good thing. Second thing, cost effective. Farmers do not need to buy hybrid seeds. For if I go to the market and I try to buy the hybrid seeds, do I get it cheaply? No, that will be very, very costly. So obviously that is also there. Uniformity in crops. All plants grown from Apomixis seeds are genetically uniform. Okay, from the Apomixis seeds, if at all I am growing some plant, all of them will be same, like clone only. So again, it ensures uniform growth as well as development. So I do not have to be concerned about anything. So one is hybrid vigor is maintained, as in whatever good quality is there, that is being maintained, because there is no fusing of gametes. If there is fusion of gametes, obviously genetically there will be some changes. That is not happening over here. Cost effective, why? Because you do not need to go to the market to get hybrid crops out there or seeds out there, and also uniformity in the crops which are being formed from this Apomixis seeds. Works. Okay, now there is a thing, remember, this sometimes give you a question that production of fruits without fertilization is known as apomixis. No. Production of fruit without fertilization is parthenocarpy. Production of seed without fertilization is apomixis. So please read your question very carefully before going and attempting it. So let's go ahead with the first chapter, sexual reproduction in flowering plants. I've already made a video on all 1 marker, already made a video on all 2 and 3 marker, and now it's time for the 5 marker questions out there. And here goes the first question. Plan an experiment and prepare a flow chart of the steps that you would follow to ensure that seeds are formed only from the desired sets of pollen grains. So basically, I am going to make a seed with the desired sets of my pollen grains, whatever pollen grains that I would like to, I am going to make it go land on the stigma, and I'll make seeds depending on what plants I want to fuse it. So what do you mean by that? Basically, I am going to do artificial hybridization. So this is the first thing that you need to crack it, right? So you have to plan an experiment for that, as in you have to basically explain all the steps of artificial hybridization. Now, name the type of experiment that you carried out. Papa, what is the type of the experiment that we carried out? It is artificial hybridization. And write the importance of such experiments. Importance will be what? Basically, suppose I want some desired characters in one particular plant. I can get that using what? Artificial hybridization technique. That is a desirable character, right? It can be useful for crop improvement programs. I want varieties of different crops which will sustain really well in drought condition, in high salinity. All of it, I will be easily able to get it. Now, the thing is, you have to explain about all of it. So how do you explain it? This is the flowchart that you need to follow. You need to write about all of this. What's the first thing that you need to do is that you have to do the selection of the parents. Choose a desired female parent and the male parent. Okay, the female parent could be there and a male parent. Suppose there's a female parent that I have taken, and over here, my dear children, there's the female part as well as the male part. Okay, and there is another plant where basically I have, so this is my male parent, and this I want to be the female parent. The problem over here is that this female parent is also having pollen grains, or basically is having the male reproductive part. So that means the pollen grains can land on the stigma and fertilization can happen this way. But actually what I want to do is I want to collect the pollen grains from here and I want to put it over there. That's my logic out there, because I want both of their characters to come in the next generation. So the first thing that I have to do is that whenever a flower is in bisexual condition, that means it has both the sexes, male and the female sexes, I would have to remove the male part from here. If only the plant that I am taking over here is bisexual, the first thing that I would have to do is removal of the male part, which is known as emasculation. Emasculation. Okay, I am removing the masculine part. So emasculation is being done. I have removed it. Now what do I do after this? So selection of parents is one thing. Suppose this female parent is bisexual, I do emasculation. Apart from that, the next step that we would have to follow is that first one, select the parents. Second one, emasculation. Now the third step is going to be bagging. Okay, what do you mean by that? So suppose over here, if I remove this, there are multiple other flowers. The pollen grains can land from here and come on this particular plant, right? Is it okay? No, because I just want the pollen grains of this plant to come over there. So what do I need to do is I need to bag this up. I need to bag the whole thing up such that no pollen grains can land on this, and also it is to make sure that the stigma which is there, it is becoming receptive. Basically it is becoming mature enough to take that pollen grain whenever the chance comes. Okay, so the next third step which we have is what? Bagging. Okay, now after bagging, what do we do, Baba? After bagging, we would be removing the bag. We are going to remove the bag and what? And dust the pollen grains over here. So I'll be removing the bag and I'll be dusting the pollen grains right over here from this particular plant. From this particular plant, I will be putting that. So dusting of the pollen grains, or basically transfer of the pollen grains, that is going to be done. From what? From the desired male plant, right? Now after that is being done, I have to make sure no other pollen grains comes and lands over it. So what do I need to do? Again, I have to bag it up. So the next process after doing this would be what? Rebagging, Baba. It will be rebagging. I will be rebagging it. And after rebagging, I also have to do one more very important thing, which is known as tagging. After this, I have to do one more very important thing, that is known as tagging. What are we tagging over here? We are tagging from which parent did we do the cross, so that we know we have an idea about what type of progeny is going to be formed later. You get it? These are the steps that you have to write. And also please understand, if at all it is not bisexual, it is unisexual flower, there is only one sex. In that particular case, you do not have to do emasculation. Only in bisexual condition you do that. So here I have written everything whichever is needed for you guys to write. Make sure you get this PDF. You will get the PDF by joining the link given in the description session. That is about, there might be a link for to download the lecture notes and things like that. Join the WhatsApp channel. So join the WhatsApp channel and get your PDF. Works. So this is the whole thing. I hope this is super clear to you. Yeah. And the name of the experiment is artificial hybridization. Also remember, it is sometimes known as controlled cross-pollination as well, because in a controlled way you are doing cross-pollination. Okay, so not be confused if at all that came. And also remember, if in any place it is written that what is that technique used in crop improvement program to produce desired quality of plants, it is basically this only, that is artificial hybridization. Always remember that. Now, importance of such experiment: they prevent contamination by unwanted pollen and help in producing hybrids with what desired characters, such as higher yield, disease resistance, better quality, etc., etc. Works. Perfect. Now the next question that we have over here is, picture related to pollination is given below. Answer the questions that follow. So this is a picture that is given. What is happening? An insect is there sitting on the plant, right? Now, will this pollination confirm fertilization? Justify your answer with suitable reasons. What do you think? Every time whenever pollination happens, do you think that will lead to fertilization? What do you mean by pollination, Baba? There is stigma, there is style, and there is ovary. Suppose the pollen grains are landing up over here. This is what you mean by pollination. This is pollination, as in transfer of pollen grains from the male to the female part, right? What do you mean by fertilization? Where basically the male gametes which are there inside the pollen grain will be fusing with the female gametes that is there in the ovary. That is known as fertilization. So, pollination has to happen, after which pollen tube will be forming. The pollen tube will be getting inside and transferring the male gametes. But always remember, this is not necessarily going to happen. After pollination, some pollen tube might form, some might not form. So pollination does not guarantee fertilization. Always remember, for fertilization to happen, obviously you need pollination, but every pollination which is there will not be guaranteeing you fertilization. This is a very important thing. Okay, so answer for this is, pollination only brings the pollen grains to stigma. It is just a transfer process, right? Fertilization occurs if the pollen is compatible, it germinates on the stigma and forms a pollen tube that reaches the ovule as well. Listen, even after forming the pollen tube also, listen, there is also one more question. After pollen germination, is it for sure that double fertilization is going to happen? Listen, even if the pollen tube is being formed, we cannot definitely say that yes, fertilization will be happening. It might happen, it might not happen. You get it? It might or it might not. Always remember it. So yes, even if it germinates on the stigma and forms a pollen tube that reaches the ovule and transfer the male gametes, then probably fertilization could be taking place. So if the pollen grain is incompatible, it will not germinate and fertilization is not going to happen. So anyway, if the pollen grain lands, suppose pollination has happened, but do you think fertilization is happening in this condition, where the pollen grain itself is incompatible? So there are certain pollen grains which are not compatible, right? In that case, the pollen tube itself is not going to be formed. If at all you are getting confused, the only answer to this question is no. Okay, pollination does not guarantee fertilization. And I am telling you one more thing. Additionally, even if pollen tubes are formed, that does not guarantee fertilization. Even if compatible pollen pistil interaction occurs, pollen tube form, that does not guarantee double fertilization. Okay, there is more to it. Multiple pollen tubes are formed. It should be coming inside the ovule and doing the whole process. Then only we'll say that yes, double fertilization can take place. Cool. Now, what are the floral rewards provided by the plants to the insects to revisit? So, what is the reward that is given by the plants so that the insects can come again and again and again? First thing that we know is nectar. Nectar is one thing. Second thing, pollen grain itself. So, ma'am, if at all it gives pollen grain, so how does pollination will be happening? We just want to transfer the pollen grains, right? It will eat up everything. No, it does not eat up everything. There will be some pollen grains on its surface as well, which could be used for transfer. So yes, nectar, pollen grains are given. Apart from that, you might have heard about yucca and moth. There, it provides a safe space for the moth to lay the eggs. So it can be in the form of shelter also. Okay, so the floral rewards which are there can be nectar, can be pollen grains. Apart from that, it can be shelter as well, which you see in yucca and the pollen grain. Okay, yucca and the moth. Now, mention the characteristics of flower in this pollination other than the floral rewards. What is the characteristic of an insect pollinated plant? For an insect to pollinate, obviously you have to make sure that the flowers which are there, they have a good odor. Okay, they have a good fragrance also, so that it can attract the insects. Apart from that, most of them, my dear children, would be large and colorful in nature. If at all the flowers are small, they will come together to form the inflorescence. They will come together, form a cluster of inflorescence. Always remember that. And if at all the plant is being pollinated by by flies and something like that, then in that particular case, it will be having a foul odor, because foul odor is going to attract the flies. So please know about all these few things out there. So large, colorful and fragrant flowers to attract the insects. Sticky or spiny pollen grains. Yeah, that is also there. So the pollen grains which are there, it has to stick on the surface of the pollinator, right? And then only pollination will happen. So which is why it will be sticky and spiny pollen grains. Stigma is sticky to capture the pollen brought by the insect. Yes, stigma is also going to be sticky, such that whatever pollen grain is there on the surface of the pollinator, that can be transferred into the stigma also. Now, foul smelling flowers in plants pollinated by flies and beetles. And please write, if at all it's small flowers, then it will be forming cluster of inflorescence. Works? Works, ma'am. Perfect. Next question. Complete the given tabular column with respect to the male and the female gametophyte of angiosperms for the mentioned character. So this is the male gametophyte column. This is the female gametophyte column. This is the character that is given over here. You have to give the answer. For example, number of cells present in the male gametophyte. What is male gametophyte over here? First, let's decode that. Male gametophyte, Baba, is the one that is going to form the male gametes later. It is going to form the male gametes later. So the male gametophyte which is there, it is pollen grain. It is the pollen grain. Okay, ma'am. Then what about the female gametophyte? Again, the female gametophyte is the one that forms the female gametes. So what is that? It is the embryo sac. Always remember this thing. Okay, now what we are asked, number of cells present in the mature male gametophyte. In the male gametophyte, that is the pollen grain, my dear children. How many cells do you remember it's there? There's a very big cell which you call as vegetative cell. There's a small cell that is known as generative cell. This generative cell, in this particular case, you have just two cells, 1 and 2, right? Always remember, this generative cell can later divide into 2 and give rise to 2 gametes, right? It is the one that forms 2 gametes. So then in that condition, 1, 2, 3 cells will be there. So either you can write 2 or it can be 3. 2 or basically 3. Always remember that. Now, don't just write 2 and come. So mature, we'll finish this first, yeah, then we'll go there, okay? So number of gametes present in. So how many number of gametes are there, Baba, in one pollen grain? There are 2 number of gametes, so I'll write 2, okay? Now type of cell division involved in the development of. What is the type of cell division that is involved over here in the development of the pollen grain out there? I can say it is mitosis. So, If you are very much confused, okay, if you're very much confused, you can write through microsporogenesis. Through microsporogenesis, meiosis happens and you get microspores. The microspores undergo mitosis to form what? To form the pollen grains. So actually it is mitosis itself, but if at all you're confused between meiosis and mitosis, be specific. In microsporogenesis, for forming the microspores, you have meiosis. Later, it will be undergoing mitosis for forming the pollen grains. Okay. So actual answer is mitosis. If at all you can give like that itself, it will be more correct. But yeah, if you want to explain the previous portion also, you can. But make sure you underline mitosis specifically. Okay, to make the pollen grains, you need to do mitosis itself. Before that, yes, meiosis is happening. Now, ploidy of the cells of. What is a ploidy? It is diploid, haploid. What is the case over here? Always remember, all the cells which are over here are going to be haploid in nature. They will be having only half the number of chromosomes, which is why it is denoted as N. So 2N is diploid. So half of 2N is going to be what? N, that is going to be haploid. Number of chromosomes over here is always going to be haploid. So I'll say ploidy of the cells is going to be what? Haploid, Baba. And please mention haploid is denoted by N. Cool. Is this particular thing being done? Let's tally it over here. So just a second, okay? Where is it? Yeah. So mature male gametophyte has number of cells 2, 1 vegetative and 1 generative, or 3, 1 vegetative and 2 male gametes. Done. Number of gametes present, there will be 2 only, 2 gametes are there. Type of cell division, mitosis from microspore to the gametophyte. So I have mentioned over here, mitosis from where to where? From microspore to gametophyte. Before that, it is meiosis. Okay. Now ploidy of the cells would be haploid. Khatam. Okay. Now let's come back over here and let's talk about the mature embryo sac. Number of cells present in the mature embryo sac. How many number of cells are there? We say, no, always we say 7 celled, 8 nucleate condition. Do you know about it? Let's see. So over here, I have a quick animation. So suppose this is the embryo sac. 3 cells are at the top with 3 nucleus. It is known as antipodal cells. These 3 cells are known as antipodal cells. They are towards the chalazal end. This end, okay, is known as chalazal end. Always remember, they have the nucleus. All the nucleus over here, all the nucleus individually, if I talk about the nucleus, all of them are going to be haploid in nature. That means I say that these 3 cells are haploid in nature. Agreed. Now, because since they just have one nucleus, so all of them haploid. Now here you have 2 cells, you call them as synergid cells. What do you call it? 2 synergid cells. Again, these cells are also going to be haploid in nature. Because here it is N, here also it is N, haploid. Fine. Going ahead, you have one more cell, that is the egg cell, or you also call this as female gamete. This is also going to be N, haploid in nature. Okay. Now what do you have? Now you have a very big cell. And this very big cell has 2 nuclei. So the very big cell looks like this. This entire thing that you see over here is a very big cell. What is the name of this very big cell? Do you have the name also? No. So I'll mention it over here so that you do not forget it. It is known as the central cell. The central cell, Baba, how many nuclei does it have? It has 2 nuclei. None of them had 2 nuclei, but this guy has 2 nuclei. Each nuclei, I told you, is haploid. So what is going to be the ploidy of central cell? Haploid. N and N. N plus N, it becomes what? 2N. So this cell is the only one which is having diploid number of chromosomes. Rest all of them are haploid. If you talk about the nuclei personally, they are haploid only. They are known as 2 polar nuclei. Both of them are there, which is why 2 polar nuclei. Got it, ma'am. Okay. Now how many cells are there here? 1, 2, 3, 4, 5, 6 and 7. And 7 cell. So can I write over here that basically, just a second, I'll go forward, okay? So there are 7 cells. Number of cells present, 7. 3 antipodal, 2 synergid, 1 egg cell and 1 central cell. Done. Number of gametes which are present over here, my dear children. How many number of gametes are there? Only this egg cell is the female gamete. So I'll just say 1 gamete is there. So, okay, 1 gamete is there. Type of cell division involved over here in the formation of female gametophyte. Do you know how these cells are being formed? It is with the help of mitosis. So 3 mitotic division happens, and that is how. 3 mitotic division happens, and that is how you get these many cells. Always remember, 3 mitotic division. And when the mitosis happens, one more thing that you need to remember is that the nucleus keeps on dividing. Here it's a very unique feature. The nucleus keeps on dividing. You call this as free nuclear division. Later on, the cell walls are being formed like this. So it is free nuclear division that you see. Free nuclear division is seen in the case of female gametophyte. Why am I telling you this? You might get MCQ questions based on this. Anyways, okay, so you had that. Now, type of cell division, I told you mitosis from megaspore to the female gametophyte. How does megaspore form? Through, again, meiosis. So here also microspores form through meiosis only. Here also megaspores forms through meiosis only. But later, both of them, they are going to undergo mitosis. Here, it is thrice. Okay. Now, ploidy of the cells which are there. Haploid. What about that? All of the cells are haploid except what? The central cell. Except the central cell which is going to be diploid because it has 2 nuclei. Works? Works, ma'am. Fine. So always use do not go around writing the answers without thinking for these type of questions. You have to think and then only give your answers. Do not be like fast I have to do. No, don't do that. Fine? Okay. Now the question, there is one more part of this question, that is, which advantage offered by the seeds help angiosperms to adapt to changing environmental conditions? So they are basically asking you about the advantage of a seed. Okay, seeds of what? Angiosperms, as in flowering plants. So what advantage does seed of the flowering plants give to them such that they can survive even if the environmental condition is super bad? What could be that advantage? What is there in the seed? Seed have a seed coat. Seed has seed coat, which is going to provide what? Protection. Protection against anything that is there. So protection and dormancy. The seed coat protects the embryo and allows to remain dormant during unfavorable condition. Apart from that, suppose it is dormant, it does not have any food. Does it have a food resource? Yes. Seed, my dear children, always have stored food in the form of either endosperm or maybe in the form of cotyledon, which can help it survive. Now, apart from that, if at all you talk about the seed, basically seed helps in easy dispersal. So seeds are adapted toward dispersal. They are very lightweighted, which is why they can be dispersed by wind to different locations where the conditions might be a bit more favorable, and it can allow that organism to grow up there. So these are the few things which are there that you can write for the advantages of the seeds which are there. Cool, man. Works. Okay. Here 2 times mitosis happens, one to form generative and one to form vegetative cell. That is 1 mitosis. Second, the generative cell again divides into 2, that is again 2 mitosis. Here after end of 3 mitosis you get this thing. Okay, always remember that. And these 3 cells which are there, they are known as egg apparatus. Why egg apparatus? Because the egg cell is over here and these 2 synergids are going to help in the process of fertilization. Why? Because the pollen tube are going to be formed through the synergids. So the synergids are going to disintegrate, and in synergids you have one more very important thing that is known as filiform apparatus, which provides the path. It provides the path. Works? Works, ma'am. Cool. Now explain the different outbreeding devices developed by the flowering plants. So what are the outbreeding devices that are developed by the flowering plants, my dear children? Any idea? First of all, outbreeding means what? The ones that will be preventing self-pollination and promoting cross-pollination. Okay, so why do you think that plants need to prevent self-pollination? Because suppose there are some toxic traits in me and my kids are being hung. Basically in the case of plants, if at all it's like that, there is some bad plant over there and the plant reproduces, what is going to happen? The same features are going to be there in the small baby plant also, and it might not be able to sustain really well. So which is why self-pollination could be really bad at times, and the plants would be having something that is known as inbreeding depression, as in because of continuous self-pollination, they will be having some very toxic characters within them such that they will not be allowed to stay in a particular environment, they will be dying off. Okay, so which is why to prevent this and to promote cross-pollination, we have outbreeding devices which are there in the plants, and you are being asked about that. So how do you frame your answer for this? Listen, mechanism to promote cross-pollination, the first one is time difference. Pollen release is happening, but the stigma is not receptive. So even if the pollen release happens, since the stigma is not receptive, pollen tube is not going to be formed. So that is one method in which I can prevent what? Self-pollination. Okay, so the next thing that we have is spatial separation. What do you mean by spatial separation? So over here, you have your where is it? No, it is not working. Okay. You have the female part, right? This is the female part. Suppose you have the male part over here. It's not too long. It's just somewhere over here. What is going to happen? The pollen grains which are there, they have to move like this and land on the stigma. That is not clearly very much possible, right? So that is what you mean by spatial separation. Plants, my dear children, have adapted a way where the anther is positioned in a way such that it cannot transfer the pollen grains. So anther and stigma are positioned differently. That is spatial separation. The third thing that we have is self-incompatibility. What do you mean by that? It's a genetic mechanism which the plant uses, such that even if pollen grains land on the stigma, it will not allow the pollen tube to be formed. There is some genetic mechanism that is blocking from the pollen tube formation that is there. That is known as self-incompatibility. Genetic mechanism preventing what? Self-pollen from fertilizing the ovules out there. Very, very important. Now, the fourth one that we have is production of unisexual flowers. Production of unisexual flowers. How does that help, ma'am? So suppose one plant is just having the female flowers, as in it is just having flowers with the female part. Obviously it has to rely on some other plant for fertilization to happen, right? Yes. So this type of plants which have two separate sexes, first of all, please know them. They are known as dioecious plants. What do you call them? Dioecious plants. Example, papaya. Example, papaya. So papaya plant, if at all you have seen, there is female plant and there is male plant. There is female plant as well as male plant. So in this particular case, male and the female flowers are going to be on different plants, and that is definitely going to help in what? Promoting cross-pollination out there. So here what all things are going to happen, my dear children? In the case of papaya, autogamy is definitely not happening. Autogamy means self-pollination from here to here. So since there is no pollen grains, which is why there is no autogamy that is going to happen. What about gynoegamy? Gynoegamy basically means from here there's a flower and there's another flower. Do you think pollination can happen between two flowers of the same plant? No. Again, there's just female part over there. How do you think pollination can happen? So autogamy and gynoegamy is prevented. What is promoted? Xenogamy. Xenogamy means pollination between two different plants altogether. Two different flowers of two different plants. Okay, so always remember, in dioecious plants, autogamy and gynoegamy is prevented and xenogamy is possible. Okay, this is dioecious plant. The example for this is papaya. Now you have monoecious plants. Monoecious plants means a plant having both the sexes, female as well as the male sexes. Okay, and how do you think here you can prevent cross-pollination? Sorry, here you can prevent self-pollination. You cannot exactly prevent self-pollination, but let me tell you, to some extent it is possible. Suppose in a plant, okay, in a plant, I am saying flowers are not like this. The male and the female flowers are not together in the same flower. Rather, on the same plant, you have two different flowers, one with female flower and one with male. One with female and one with male. Okay. So that is being seen in the case of castor. So monoecious plants are the one that has both the sexes in one plant. Either it can be in this way or it can be in this way. We are talking about this. In this, anyway, autogamy is going to happen. Okay. Here, what are you saying? There are less chances. You are making the chances less for autogamy to happen and for promoting cross-pollination. From other plants, here easily things can be coming up. There is less competition of self-pollination. Right? You get it? So to some extent, you can basically do that. And this is the example of castor. In castor, it's not like this. It's like this. There are separate male and separate female flowers, but on the same plant. Again, over here, autogamy is prevented, but gynoegamy is not. Gynoegamy is not. Pollination can still happen from the same plant parts, but yeah, it is reduced to so much extent, and it helps in proper cross-pollination to happen from other plant also, because there is no pollen grains of itself, right, in the same flower. So example for this is castor and maize. CM, Chief Minister, you can remember, male and female flowers on the same plant prevents autogamy, but not gynoegamy. Know about this. If you do not understand this, you will not be able to answer this. Okay. Works. So these are the things that will be promoting cross-pollination. These are the outbreeding devices out there. Cool, ma'am. Let's go ahead. Now, double fertilization is a unique event taking place in flowering plants. Briefly explain. How do we explain double fertilization? Any idea? Very, very simple. Listen to me carefully. So, I'll come back over here, and I hope you do not mind if at all I remove this. So, what exactly happens is that I told you your pollen grain which is there, it has two gametes, right? So there is one gamete and there is another gamete. This is male gamete, this is also male gamete. Two male gametes are there. 1, 2. When the pollen tube formation occurs over here, what happens is that one male gamete which is there, fuses with the female gamete. Okay, the first one. Now there is one more male gamete. One more male gamete that is there is going to fuse with the two polar nuclei of the central cell. So first fertilization, second fertilization, 2 times fusing is happening. That is why I call it double fertilization. What do you get when the male and the female gamete are going to fuse? You are going to get a zygote. Okay, what is the ploidy of the zygote, ma'am? So always remember, gametes are going to have half the number of chromosomes. So here the male gamete is having N, female gamete will also have N plus N, you will get 2N, diploid over here. Okay, diploid zygote is being formed over here. What about this? So here, my dear children, in the case of the male gamete, it had haploid number of chromosomes. Here the 2 nuclei had haploid number of chromosomes. 2 nuclei has haploid number of chromosomes. So what is going to happen? N plus N plus N. You are going to get something that is having 3N number of chromosome. And what do you call this particular cell as now? What do you call this particular cell which has 3N number of chromosome? It was a central cell. Central cell fused with the male gamete. Now you will call this as primary endosperm cell. Primary endosperm cell, which is triploid in nature. Which is what? Triploid in nature. This nucleus which is there, I'll just call this nucleus with the fusion of all this thing. When the nucleus is being formed, I'll call it primary endosperm nucleus. So primary endosperm nucleus is of primary endosperm cell. This primary endosperm cell, Baba, later it will be forming endosperm. Why do you need endosperm? Endosperm is super important for the zygote, for providing nourishment to the zygote, such that the zygote can form an embryo. That is how it is usually going to go. Please remember, this is super important. Works. Okay, so basically this type of fusion which is happening over here with the male and the female gamete, I'll call this fusion as syngamy. This fusion which is happening between 3 nucleus out there, or basically 2 cells out there, I'll call this as triple fusion. But why triple fusion? Since 2 nucleus are being fusing. What is the end result of this? Primary endosperm cell with triploid nature. Okay, or you call it primary endosperm nucleus, if at all you're just talking about the nucleus. Anyway, the cell which is there, which is being formed, it will be later forming endosperm, and the endosperm forms first. Endosperm is going to be, primary endosperm cell is later going to form endosperm tissue, and this endosperm tissue is going to help with the zygote, such that the zygote will be transforming into the embryo. Cool. So that is double fertilization. Now again, how do you frame this answer? So in flowering plants, 2 male gametes enter the embryo sac through the pollen tube. Agree. 1 male gamete fuses with the egg cell to form a diploid zygote. This is known as syngamy. Okay. The second male gamete fuses with the 2 polar nuclei in the central cell to form the triploid primary endosperm nucleus. And this is of the cell, that is primary endosperm cell. The primary endosperm cell later divides to form what? The endosperm tissue. Okay, and this endosperm is going to help in nourishment of zygote to form embryo. Fine. Because 2 fusions occur in the same embryo sac, it is known as double fertilization. A feature which is very unique to what? Angiosperms. Cool. Done, ma'am. Now, there is one more question that is given to you. Arrange the following terms in correct developmental sequence. So pollen grains, sporogenous tissue, microspore tetrad, polar mother cell, and male gametes. That means you are being asked about one very important thing, that is microsporogenesis. The steps involved in microsporogenesis. So what is the step? First thing that you have is basically lot of cells over here. These cells are known as pollen mother cell, PMC, or I can also call it microspore mother cell. Either way, these cells which are there, they belong to something, a tissue which is known as sporogenous tissue. So tissue is made up of multiple cells, right? So these cells are going to form sporogenous tissue. You can remember that. So basically, if I want to go one by one, I'll say sporogenous tissue is there. In that you have pollen mother cell. This pollen mother cell which is there, it is diploid in nature. It is what? Diploid in nature. It will undergo meiosis. Meiosis happens in 2 stages. First stage, it will give you 2 cells, and in this stage itself, the number of chromosome becomes half. It was 2n over here, it becomes half over here. Okay, now these cells again divide, you get 2 cells over here. The number of chromosomes remains the same. Here also it divides, again you get 2 cells. Here the number of chromosomes remains the same. This is going to be known as meiosis.
लाइव मेरे स्टूडियो में चल जाएगा कैसे?
Welcome. When you start. Just 5 minutes.
Half the number of chromosome. If 2 polar nuclei is there, central cell has 2 polar nuclei. 1 polar nuclei will be having 3n, just like any other nucleus out there. There is 2 nuclei, so I'll add 1 more 3n. 2 polar nuclei there. 3 plus 3 is equals to what, Papa? 6. Now 6n is the totality of the central cell out there. Now this 6n plus 2n is equals to what? 6n plus 2n. 6 plus 2 is equals to what? 8n. This is going to be the ploidy of the primary endosperm nucleus. Again, I am telling you, primary endosperm nucleus is being formed when 2 polar nuclei of the female. 1 polar nuclei will be having half the number of chromosome. Another polar nuclei will also have half. So 3 plus 3. Half is this number, right? So 3 plus 3 is equals to 6n over here in the central cell, right? And the male gamete is having half the number of chromosomes, that is 2n. So basically 6 plus 2 is equals to 8. You got it? So this is going to be octaploid. You need to write your answer like this exactly. I've given everything which is needed for you. So yes, I'll just write 1 by 2. Cool? Cool, man. Perfect. Now, one more question is there. I thought I'll discuss it here, and that is double fertilization is reported in plants of both castor and coconut, groundnut. Double fertilization is happening both in castor as well as groundnut. Okay. However, the mature seed of groundnut is non-albuminous. Groundnut which is there, it is non-albuminous. What do you mean by that? Non-albuminous means mostly when I talk about a seed. Seed has what? Inside it has embryo. How did the embryo come? Embryo came with the help of zygote. Zygote transformed into the embryo, right? Now, to provide nourishment to the embryo, or basically to form the embryo, you had endosperm. You had some endosperm. Some of the organisms use up the endosperm completely. Completely they use up the endosperm to form the embryo. From zygote to embryo transformation, you need endosperm to do this, and some utilize the endosperm completely. You do not have any endosperm left inside the seed. You do not have any endosperm left inside the seed. You call this as non-albuminous condition. There is no endosperm up there. Now when you talk up, this is the case of groundnut, because groundnut is non-albuminous. Now what about castor? In castor, my dear children, the endosperm which was there, it is not completely utilized. It is still there. It is still there in that particular case. Okay, so since endosperm is still persisting, I will be calling this as what? Albuminous. I will be calling this as albuminous. Please understand, groundnut as well as castor, both of them, they are dicots. Dicots means basically usually dicots use up the endosperm to form 2 cotyledons. So they usually have 2 cotyledons. Mostly endosperm is totally utilized for this purpose only, for formation of 2 cotyledons. But in the case of castor, even after being a dicot, it has 2 cotyledons, but also it has some endosperm left behind. That is why I'll call this as albuminous. Usually, okay, leaving back of endosperm is found in the case of monogots, but this guy is an exception. This guy is an exception, and I'm not saying mostly it happens. I'm saying usually this is the case of monogots. But yes. So explain the post-fertilization event. So how do you explain it? Right. In both castor and groundnut, double fertilization occurs. Zygote develops into embryo. Primary endosperm nucleus develops into endosperm. In castor, the endosperm persists in the mature seed. Hence, the seed is albuminous. Right. Here in groundnut, the endosperm is completely consumed by the developing embryo. Hence, the mature seed is what? Non-albuminous. So you just have to give the explanation of the definition out there. Now, what do you have? Banana is a parthenocarpic fruit, whereas oranges show polyembryony. Explain, how are they different? So, parthenocarpy means what? Usually, Baba, what happens is that we have ovary, inside that we have ovule, right? So, ovary which is there, it transforms into a fruit, and ovule which is there, that transforms into a seed after fertilization. This is after fertilization. Until then, you will call this as just ovule and ovary. After fertilization, you will call this as seed and fruit. But in some cases, my dear children, where is it? Yeah. In some cases, without fertilization, as in there is not going to be any formation of seed over here. still the ovary will be transforming into fruit. Ma'am, how is that possible? Under the influence of some hormones, okay? So under some influence of some hormones, which is auxins and things like that, here the ovary transforms into a fruit without fertilization. So fruit without fertilization is known as parthenocarpy. Seed formation without fertilization, that is known as apomixis. Please know the difference, okay? Seed and fruit difference is there. Now, that is about banana. Now, when you talk about oranges, oranges show polyembryony. What do you mean by that? So, in the case of orange, my dear children, again, you have the ovary, you have the ovule. Inside the ovule, no, sometimes the nucleus cells might be there. There might be cells of nucleus. And here you have the embryo sac where you have the female gamete and things like that, right? Sometimes, inside this female gametophyte, that is embryo sac, the cells of the nucleus might get inside it. Once they get inside it, they are going to form the embryo. And you know what? They will be forming multiple embryos. Polyembryony is what you call this. And this is usually found in citrus fruits such as oranges, mango, etc. etc. Ma'am, over here, since it is getting inside the embryo sac and forming the embryo, here also fertilization is not happening, right? No. So can this be an example of apomixis also? Yes. So apomixis can happen in this case. Can happen in this case where polyembryony is being seen, but it is not necessary that every time it has to be done in this way only. Apomixis can happen in another way where the female gamete which is there in the gametophyte, instead of being haploid, it is actually diploid. The female gametophyte, female gametes are usually haploid, right? But here it is diploid, which is why it does not need any male gamete. And that's also a way in which you can, basically you can transform this female gamete into what? A zygote. A zygote can form in embryo there without fertilization. So that is one way of apomixis. This is the second way of apomixis, which you call as polyembryony. Cool? Yeah. Apomixis is generally seen in Asteraceae and grasses. Polyembryony, a specific condition, is seen in the case of citrus fruits. Okay, ma'am, how do you write the answer? So, the ovary develops into fruit without fertilization, that is parthenocarpy. This occurs under the influence of what? Plant hormones. Can it be induced? Yes. So, in this fruit, do you think seeds will be there? No, Baba. Fertilization didn't take place, no. So, that's why seedless varieties of fruits will be formed over here. Oranges is polyembryony. More than one embryo is formed in a single seed, and additional embryos arise from the cells of the nucleus that I just told you. Hence, seeds are present containing multiple embryos. And that's your answer. Okay. Oh my God, we have completed one chapter. Kudos to everyone who have come this far. And my dear children, I hope you are understanding, hope you are enjoying as well. So it's like more of like a drama from my end because I'm alone in the studio and I'm talking to you and I'm trying my best to make you and me highly engaged. So if at all you think it is working well for you, do not forget to share it with your friends and do not forget to hit the like button as well. Yeah? Yes. So human reproduction is the next chapter, and let's see the first question that we have from this chapter. The secondary oocyte formation from primary oocyte occurs in which of the follicle? This is MQP1 question. Okay, question number 2. MQP means model question paper. Okay. So secondary oocyte formation from primary oocyte occurs in which of the follicle? So only at two places the division takes place, or basically I can just say one place. Okay, one place. So you know primary follicle is there. Primary follicle is there. Inside the primary follicle, what do you have? You have the primary oocyte. Secondary follicle is being formed. In the secondary oocyte also, what do you have? You have the primary oocyte only. It has not completed its division. Okay, primary oocyte which is there, it has to divide into two things. It is an unequal meiotic division where you get the first polar body and you get the secondary oocyte. The secondary oocyte, that is what you have to get. Okay, so this particular transition is happening, but the division did not complete. Okay, completely the meiosis was not able to get completed off. It is stuck in meiotic phase, meiosis 1. Okay, now what happens? The secondary follicle will transform into tertiary follicle, and this is the place where everything changes up. Why do you think everything changes up? Because finally the primary oocyte is able to complete its division and form what? The secondary oocyte. Finally, the secondary oocyte is going to be formed up over here, and that is super important. Okay, now the tertiary follicle which is there, it is going to transform itself into the mature form of follicle which is known as Graafian follicle. And the Graafian follicle, what do you think? This will undergo division. So now the secondary oocyte is yet to divide and form the second polar body, and also one thing that is known as ovum. But this division also does not take place, so it is still stuck in secondary oocyte phase. When do you think this division is going to take place? When ovum is going to be formed? Just before fertilization. Okay, just before fertilization, the ovum is going to be formed and the sperm and the ovum is going to fuse. Okay, so always remember, tertiary follicle is the place where finally it is able to divide to secondary oocyte. So if the question is secondary oocyte formation from the primary oocyte occurs in which other follicle? It occurs in the tertiary follicle. I hope you never forget that. Yeah, in ovulation, in which form is it going out? The Graafian follicle contains the secondary oocyte. So secondary oocyte is something that is going to be released. Okay, it has not completed its division to form these two things. It happens just before fertilization. Okay. Now next thing, the cells that secrete testicular hormones or androgens are known as. The cells that secrete testicular hormones, do you have any idea? So in the testis, you have testicular lobules. In the testicular lobules, you have 2 to 3 highly coiled seminiferous tubules. If at all you zoom it in, it looks like this. So this is seminiferous tubule, this is seminiferous tubule 1, this is seminiferous tubule 2. In between this, do you see a space? Do you see the space? This space, my dear children, is known as interstitial space. And over here you have interstitial cells, or you also call them as Leydig cells. Leydig cells. What do they release? They release the androgens, the male sex hormones. Why? Because under the influence of these androgens, finally sperms are going to be produced over here in the seminiferous tubule. So in the interstitial space, you have the Leydig cells that are going to help you with all of this thing. Okay, so please remember that. Here you have, in the seminiferous tubule, you have spermatogonia. Apart from that is the sperm mother cell. Apart from that, you have the Sertoli cells, the sperm, nurse cells out there, which is there for nourishment of the spermatogonia and the sperms. So please remember, both SS cells are inside, and in the Leydig, in the interstitial space, you have just the Leydig cell. Works? Works, man. Okay. Now, yes, next question. Which of the following statement is true? Okay. LH surge induces menstruation. What do you think? LH surge means increase of LH is going to induce menstruation. No. LH surge is responsible for one very important thing. Here, always imagine the follicles are maturing and you have one Graafian follicle which is out there. This Graafian follicle has what? It has the secondary oocyte out there. Under the influence of high amount of LH, luteinizing hormone, the follicle is going to rupture and this particular guy is going to come out. Secondary oocyte is going to come out, and that process is known as ovulation. Here it is written, menstruation is induced by LH surge. Wrong statement. Now, regression of corpus luteum increases progesterone. Regression means what? Basically, the corpus luteum is going to decline. The amount of it is going to regress. It is going to be less. Do you think that is going to increase progesterone? First of all, let's talk logically. Okay, so once LH surge has taken place, this was the Graafian follicle first. It was the Graafian follicle, agreed? This Graafian, whatever is left behind, is now known as corpus luteum. So the egg is released, the corpus luteum is being made also. Not the egg, actually secondary oocyte is being released. Now what are you waiting for? You are waiting for the sperm to come. If at all the sperm and the egg comes in contact, what is going to happen? Basically, if suppose fertilization happens, you will form the zygote, then the embryo. The embryo needs nourishment. How do you provide the embryo nourishment? By the help of endometrium. Right, so you have to make sure the endometrium is super ready. Okay, ovulation has taken place. Now fertilization can happen at any point of time, but you also have to make sure endometrium is super ready. Who makes sure that? The corpus luteum, that is whatever is left behind the Graafian follicle, that forms the corpus luteum, and the corpus luteum is going to release two very important things. One is progesterone and the other one is estrogen. But this is the progesterone that majorly helps in what? Helps in, in making the endometrium really thicken. Okay, and the endometrium starts to secrete some very important things, such that if embryo comes in contact, basically it provides all the nourishment that is needed. Right? This is what usually happens. But you need to understand one thing, that corpus luteum is also being maintained because there is LH being maintained. There is LH surge that happened. Later LH, it comes to this particular phase where it is there and is there for the nourishment of corpus luteum. Not nourishment, basically for making sure that the corpus luteum stays like that. The corpus luteum in turn produces what? Progesterone. But there is a catch. Progesterone, when it is produced, it does not like LH to be there, which is why it tells the papa of LH, okay, papa of LH to stop the secretion. Papa of LH is what? GnRH, gonadotropin-releasing hormone from hypothalamus. So it says that stop the secretion of LH. Now if LH level goes down, that means corpus luteum is also going to go down. If corpus luteum goes down, progesterone will also be less. So regression of corpus luteum increases progesterone? No. If corpus luteum is going less, that means progesterone is also going to go less. So this is wrong. Okay. Diploid egg is formed after second meiotic division. Listen, over here, there is no diploid thing that is going to be formed. Egg cannot be diploid first of all. Egg and the sperm has to be haploid in nature. These haploid thing comes together to form a diploid zygote. So this particular statement is anyway going to be wrong. Now embryo differentiate from the inner cell mass of the blastocyst. All of it, this question if at all you see, you need an understanding of multiple things. Menstrual cycle, you need an understanding of how the oogenesis takes place, and you need an understanding of how basically gestation takes place, or I can say implantation takes place. So there are three different concepts being asked over here. That means we have to be really strong with our concepts. So embryo differentiates from the inner cell mass of the blastocyst. Always remember, when I say the zygote transforms into an embryo and the embryo comes and attaches to the endometrium, I mean that there is a particular stage that comes and attaches, and this stage is known as blastocyst stage. In the blastocyst stage, there are two types of layers. There is the outer layer of cells, which you call as trophoblast, and there is inner cell mass out there, or the inner mass of cells, which you call as inner cell mass. Now once it comes and attaches to the endometrium, you know endometrium cannot always provide the nourishment. There should be something out there that will be there for nourishing for a longer time, and that is a placenta. So your next goal is to make the placenta. Who helps in the formation of placenta? The trophoblast, my dear children. It helps in the formation of placenta along with the mother's maternal tissue out there. So from embryo side, it is trophoblast that is going to help. From the mother's side, it is going to be some maternal tissues that are going to help. Now what is left behind? The inner cell mass is left behind. This actually forms the embryo. This one actually forms the embryo. Rest is there for placenta formation as well. So embryo differentiate from the inner cell mass of the blastocyst. The answer is yes. This is actually correct. Sometimes you say that inner cell mass forms the embryo proper. So do not be worried. It means that it is actually the one that forms the embryo later on. Got it? Yes. So only the option is correct. Rest all of it, my dear children, are wrong. Next question. Scrotum helps in maintaining the temperature of testis, which is lower than the normal body temperature. How much degree lower than the normal body temperature? 2 to 2.5 degrees Celsius. So how many options do you have which are correct? This and this. So can I exclude this? Yes. Just cut off the options which you think that is not correct. Okay. Now what do you have? Each of the testes has about B compartments. How many compartments are there in the testis? What do you mean by compartments? You mean team lead. I always remember like that. So how many team leads are there? Testicular lobules, there are 250 of them. Testicular lobules or team leads, there are 250 of them. So here again the options are same, so we cannot choose. Now it measures C centimeter in length. What is the length of the testis? This is what you mean by length. This is the width. Width is 2 to 3 centimeter. Length is 4 to 5 centimeters. So through 3, 4, 5. 2, 3, 4, 5. Always remember that. So over here 4 to 5 centimeter is actually length. So your answer for this particular question becomes what? B. It becomes B. Okay, do not be confused with the width and the length. Length is 4 to 5 centimeter. Cool? Yes. Fine. Next question. In spermatogenesis, if FSH hormone is not secreted from the anterior pituitary gland, which stage is affected? In spermatogenesis, when sperm is being formed, if FSH hormone is not secreted from the anterior pituitary, okay, which stage is affected? What do you think so? Which stage is going to be affected? Let's see. Listen, as a basic thing, okay? There is hypothalamus. hypothalamus produces gonadotropin releasing hormones. Okay, now these gonadotropin releasing hormones are going to act on anterior pituitary. Now the anterior pituitary on the other hand is going to release two things. One is LH and the other one is FSH. Cool. LH which is there, it is going to act on the Leydig cells, and FSH which is there, it is going to act on the Sertoli cells. Remember, Leydig cells in between the seminiferous tubules, Sertoli cells inside the seminiferous tubules. So here L, here L, here S, here S. You can remember like that. Okay, the Leydig cells on the other hand is going to release what? The male sex hormones which you call as androgens. Done? Done. Okay, what about the Sertoli cells? What are they going to do over here? When they are acted by FSH, they are going to release some factors. Some factors. Okay, so two of the things are being produced. Here androgens, that is the male sex hormones, and there the factors. You need to understand that basically this thing is happening within the seminiferous tubule. Factors are being produced within the seminiferous tubule. What is happening in the seminiferous tubule? The spermatogenesis is happening over here. Spermatogenesis is happening over here. And for that, you need high level of androgens. So the androgens gets inside. Okay, why do you think the factors are being produced over here? Factors are here for making sure that the level of androgens are being maintained. Why? Because if at all androgens are not there, this process is not going to happen. Especially this particular process. What is that process, ma'am? So in spermatogenesis, if at all you know, the spermatogonia differentiates. You have one cell that is known as the primary spermatocyte, which is in 2N nature. This primary spermatocyte undergoes meiosis. You get two cells, N and N. Okay, this is going to be what? Your secondary spermatocyte. Again, here half of the number of chromosomes have gone. Okay, only half is left. Now again meiosis happens. You get four cells. Are they the sperms? No, they are known as spermatids. Spermatids, they are like kids. They are immature kids, chotu kids. They are immature. They are globular in shape, but the actual sperm is elongated in shape. Now you need to transform this into what? This way, which means that you have to make them transform into spermatozoa. You need to make them transform into spermatozoa. So spermatids to spermatozoa transformation has a specific name. It is known as spermiogenesis. Why are you teaching this, ma'am? Why are you teaching this, ma'am? Basically, in spermiogenesis, you need to make sure the level of androgens is maintained really high. So throughout it is important, but majorly during this particular process. So in spermatogenesis, while the whole formation of spermatogenesis is happening, if FSH hormone is not being secreted from the anterior pituitary, what stage is going to be affected? Formation of primary spermatocyte? No, that is going to happen. Formation of spermatozoa? Yes, that is not going to happen, because as I said, here in the process of spermiogenesis, this whole is spermatogenesis, but this particular portion is spermiogenesis. At this particular place, you definitely need a lot of FSH, because FSH acts on the Sertoli cells to produce factors, and these factors make sure testosterone level is being maintained. So this is the thing that is being affected. Now formation of spermatids? No. Formation of secondary spermatocyte? No. Your only answer for this question is going to be B. So for this, again, you need an in-depth understanding of spermatogenesis. Later when the sperms are being formed, the sperms will be moving out of the seminiferous tubules, and that is what you call as spermiation. I remember it like aviation. Similarly, you can also remember, but yes, spermiation means basically the sperms are being moving out. Okay? Yes, ma'am. Okay. Now, to produce 2000 sperm and 400 ova, how many spermatogonia and oogonia are required to produce 2000 sperm? Remember, from one spermatogonia, as I said, it transforms into primary oocyte and things like that. At the end, you get 4 sperms. At the end, you get 4 of the sperms. So here the question is 2000 sperms are there. So 2000 divided by 4, if at all I do, I get an understanding of that there are 500 spermatogonia, which divides, each of them divides into 4 sperms. That is how I get 2000 sperms. Okay. What about oogonia, my dear children? You have been asked about oogonia. Remember, one oogonia transforms into one primary oocyte. This primary oocyte undergoes unequal division. Remember, first polar body and the secondary oocyte. This first polar body does not undergo any division. This does not undergo any division. Now only this is left. This also undergo unequal division, which is why you get 1 ovum at the end. You do not get 4 ovum and all, because here also did not undergo division. Here also did not undergo division later on. So from 1 oogonia, 1 ovum. So if at all you are saying that 400 ova is being produced, 400 divided by 1, it will be 400 oogonia only. Got it? So 500, where is it? 500 spermatogonia and 400 oogonia is going to be your answer for this question. Your answer is B. Cool? Yes. Next question. Which among the following has 23 chromosomes? That means half the number of chromosomes. How do I know that? Because I know 46 number of chromosome is what? Diploid. So half of it is going to be what? Half of it is going to be 23. And where do you see this? You see this either in the case of ovum or the sperm. Apart from that, as I said, meiosis happens in 2 stages. In the first stage itself, my dear children, what happens? Diploid turns into haploid. So basically, secondary oocyte or secondary spermatocyte, all of them are going to be haploid. Half the number of chromosomes are going to be there. So spermatogonia, definitely not. It is a diploid cell. Zygote, definitely not. It is made of fusion of haploid, diploid. Secondary oocyte, yes. Secondary oocyte, why? Because I told you, primary oocyte undergoes first meiosis. End is what? Reductional division. Okay, that means it will have half the number of chromosomes. Later, it will be carrying the same number of chromosomes in the ova. Or in the case of males, it will carry the same number of chromosomes in the case of the spermatids which are going to be formed. So it is secondary oocyte or secondary spermatocyte, which is the answer. And you will not be getting all the options together, that is secondary oocyte and also ovum. That is definitely not going to happen. Okay, you will only get either secondary oocyte or the ovum. Yeah. Okay, so next question. Identify the wrong pair with respect to menstrual cycle. Formation of corpus luteum, 15th to 28th day. So if at all I say the cycle is of 28 days, when do you expect that the ovulation is going to happen? On the 14th day. That means this is a period when there was high amount of LH. Now, after this, when the LH level goes down a bit, basically the Graafian follicle has ruptured. Now you are going to form the corpus luteum. So corpus luteum is formed after the 14th day. Formation of corpus luteum 15th to 28th day. Yes, it happens during that particular point of time. This is correct. Secretion of progesterone. Obviously, the corpus luteum is the one that releases progesterone. So I'll say again it happens during the same day, 15th to 28th day. Regeneration of the endometrium 5th to 13th day. 5th to 13th. So listen, this is a cycle. From 1st to 5th day, that is there, you will have the menstrual phase. Menstrual phase basically means that your endometrium is going to fall down. Okay, it is going to break down. Now, after the 5th day, my dear children, slowly the endometrium is going to regenerate, because again we are preparing for the next cycle of ovulation to happen, right? So regeneration of endometrium from 5th to 13th day, yes, from this particular time it happens. So this is also correct. Okay, disintegration of endometrium 5th to 13th day. Okay, this was correct, this was correct. Disintegration of endometrium. Disintegration means again breakdown of endometrium from 5th to 13th day. No, regeneration happens. Disintegration took place over here in the 1st to 5th day. After that, again it is being formed. Ovulation happens, it becomes even more thicker, and then when if suppose fertilization didn't take place and things like that, again it will fall down. So here it is time for regeneration. It is time for regeneration. Got it? And here is the time for thickening. So here it is written ulta. So this is wrong. Disintegration of endometrium from 5th to 13th day. No, it happens in the 1st to 5th day. Yes? Yes, ma'am. Okay. Next question. The secondary oocyte after ovulation is covered by non-cellular layer of what? Listen to me. It's not like after ovulation, but in the graphene follicle, I told you there is secondary oocyte which has not completed its division, right? This secondary oocyte which is there in the graphene follicle makes a layer around it. And there's a unique feature about this particular layer. It's non-cellular. That means there are no cells up over here, or I'll call this as a cellular layer. It is not made up of any cell out there. You call this as zona pellucida. You call this as zona pellucida. So C is your answer for this particular question. Okay, later around it, since it is there in the graphene follicle, no, the cells of the graphene follicle, they also contribute to one more layer formation, and that is known as corona radiata. That is cellular, and that is known as corona radiata. This is how it is going to be released from the graphene follicle with all these things. Okay, so non-cellular layer is there, that is known as zona pellucida. It is acellular also, please remember. Cool? Yes. Next, during pregnancy, the fetus develops limbs and digits after what? Limbs and digits. This is a very important question, as in they can give you any month and you will be asked this question, so you should know. So during pregnancy, the fetus develop limbs and digits. So first month, the first thing that we check, okay, first month, the first thing that we check when someone dies is what? Heartbeat. Similarly, when someone is born, we check the heartbeat. So towards the end of first month, you have the heart being formed. Now the second month, my dear children, you have the limbs and also the digits being formed. Limbs and the digits are going to be formed. They are not going to be separate, they might be together, but they are going to be formed. By the third month, my dear children, you will be having all of this definitely there. Apart from that, external genital organ, that means male or female external genital organ will be there. Apart from that, the organ and the organ system, most of them are going to be formed over here. Organ and the organ system, most of them are going to be formed here itself. Okay, now fourth month is not given in your NCERT. Until third month, we say first 3 months is known as the first trimester. And always remember, 1 month is equals to 4 weeks. In 1 month, how many weeks do we have? 4 weeks. So 3 month means 3 into 4. 3 into 4 is equals to what? 12 weeks. So by the end of 12 weeks, what do you have? You have all this thing being formed. So they can ask you in different formats, just know that. Okay, now the 4th month is skipped. 5th month, baby says that, God, basically I want to move around. Until now I heard you, okay, but now I want to move around. So you see the first movement of the fetus in that month. And also one more thing, baby says that I want some hair to do some fashion show. So over here it has the hair on its head, hair on the head, and also the first movement in the 5th month. Now in the 6th month, that means the next 3 months, in the 6th month, my dear children, baby asked the God for hair, no? So God gives lots of hair on the body also. So you will see tiny hair on the body. Apart from that, eyelashes are going to be formed. And my dear children, eyelids are going to be separated. These are the few things that you can clearly see. This is basically the second trimester. This was the first trimester. This is going to be the second trimester. You also call this as 24 weeks. Why? Because 6 into 4, 6 months. Okay, 6 months into 4, 24 weeks as well. Just know that. Here the question is, during pregnancy, the fetus develops limbs and digits. Limbs and digits, in the second month means 2 into 4, 8 weeks. So after 8 weeks. Okay? Yes. Okay, next question. Statement number 1. The process of release of sperms from the seminiferous tubule is known as spermatogenesis. What do you think is the answer? The process of release of sperms? Arey Baba, release of sperms. Remember, aviation, spermiation. Spermatogenesis is formation of sperm, right? So this is wrong. The spermatids are transformed into spermatozoa. Is it? Or is it spermatozoa to spermatids? Remember, kids to zoa. Okay, so it was immature, now it is being mature by the process of spermiogenesis. Is that correct? Yes, that process is known as spermiogenesis, and here you need a lot of factors from Sertoli cells. Remember? Yes. So statement 1 is wrong, statement 2 is correct. Fine. Next question. Choose the correct option from the table given below for the formation and dissolution of the labeled part in the given diagram. Okay. Zona pellucida is the thing that is being labeled over here. So you have to say about the formation and also the dissolution, as in breakdown and also making. So how it is made? It is made with the help of secondary oocyte. I told you it makes. So there are two options. I'll cancel out these two options. Now, dissolution as in breakdown of Zona pellucida. When does it happen? Before fertilization, after fertilization. Before fertilization, do you think Zona pellucida will go away? Why do you think Zona pellucida is there at the first scenario? Need to understand that. Basically, Zona pellucida has some receptors onto which your sperm head is going to attach and going to release some enzymes such that it can cleave through and get entry. Once a sperm enters, what is going to happen? The Zona pellucida is going to become very, very thick such that no other sperms can enter. Okay, later fertilization is going to happen. So for fertilization to happen, this guy has to be here. So it cannot be before fertilization. Now after fertilization has taken place, after zygote is being formed, slowly the Zona pellucida is going to be going off. So after fertilization, it gets dissoluted or basically dissolved. Okay, yes. So your answer for this question is going to be D. Every time in an MCQ question, read the question carefully. Very, very important. Most of us mess up with the MCQ question because we might know the answer, but we are in an anxious state so that we read it very, very fast. So don't do that mistake at all. Okay, very, very important thing. Now, so I think with this we completed your human reproduction chapter, and with this I want to tell you one very important thing. That is, my dear children, we have launched a KCET crash course, which is 30-day comprehensive crash course for everyone who is preparing for KCET. Now I know not a lot of biology students are in for KCET examination, but still I am telling you, if at all you have not started up your preparation really well or strong, we are here with you. There will be complete PU1 and PU2 syllabus coverage. There will be previous year questions along with the solutions. There will be PYQs in the form of test also, that suppose you're done with the chapter, you can go give the test of that particular chapter. There will be 10 full-length mock tests, and all of it starts from 21st of March, just after your board examination finishes. Yeah, and the pricing for this is rupees 2499, but it will not be the same, okay, after 25th of February. So if at all you're planning to take it up, make sure you enroll right now. Yes, and if you want a free mock test PDF, just chumma, okay, if at all someone is interested, there, go to the link. Okay, go to the description section. I somehow not able to tell that. Okay, and then click on the link, fill in your number and download the mock test. But as of now, I don't think you will require it, but check it out. Fine. And now we move on to the next chapter, that is human reproduction. So one chapter is done, and by the way, everyone who are new to this channel or probably have not watched any of the videos until now, I had taken a live today, which was on top 50. You can see in the live session there is top 50 questions. In those top 50 questions, I have taken so many hot questions, high order thinking questions from each of the chapter. I'll request you, at least if you're not able to watch that video again, I'll request you to at least get the PDF of it and read through it. Watching it will be definitely a better option because it's top 50 question, most curated one. But again, it's your choice. Yeah? So do that. Yeah? Now, human reproduction. Let's go ahead. So there is 1 marker, 3 marker and 5 marker question that comes from it. So I'll just deal with 3 marker from here. Let's go ahead. The first question, what is menstrual cycle? Mention one pituitary and one ovarian hormone that plays a very important role in the menstrual cycle. Okay. So, what do I do is that I explain it to you. Okay, just listen to me very carefully. It's very, very simple. So suppose your cycle which is there, it's from 1st to 28 day cycle.
You are claiming for some internal reservation. Make sure
half. When you reach half, that is 14th day, that's when you can expect ovulation to happen. That means release of egg to happen over here. Right.
Wishing you all the best, and not forget to hit the subscribe button.
In this particular case, you need to understand from 1st to the 5th day out there. 1st to 5th there will be menstrual phase. What do you mean by menstrual phase? As in, over here, the endometrial lining which was there, it is going to shed off. It is going to shed off. Why? Because estrogen and progesterone level is super less during this particular period. And estrogen and progesterone is something that maintains the endometrium. Whereas on the other hand, there are 2 other hormones, FSH and LH, whose level is slowly increasing during this particular period. So they are going less, they are going more. Slowly it is going high. Okay? I hope you got it. So there is no one to maintain the endometrium, that is why it is falling down. Who maintains the endometrium? Estrogen and progesterone. These two hormones are ovarian hormones. They are released by our ovary. These two hormones that you see, they are released from the pituitary gland, so which is why I'll call them pituitary hormone. Cool. Now, what happens is that since FSH and LH level is rising high, so from 5th to the 13th day out there, we can expect something happening. And that is what, first of all, what do you call this phase as? You call this phase as follicular phase. Why? Because the primary follicles which are there in our ovary, they are slowly developing into what? Into Graafian follicle. Into Graafian follicle, which has the secondary oocyte, the matured form of follicle. While it is happening, the follicles, my dear children, the cells of the follicles, they are going to release some hormones. Okay, so under the influence of what? So FSH and LH is getting high, especially FSH, my dear children. They are the ones that is responsible for this particular phase. And now they are going up. And parallelly, as I said, in this follicular phase or during this follicular phase, some things are being released. What is that particular thing? Estrogen and progesterone, which is why what is going to happen? The endometrium which was falling down, it will slowly regenerate. There is regeneration of endometrium, and I'll call this as proliferative phase as well for the same reason. So what is happening? Under the influence of FSH, the follicles are generating, but the follicles which are there, they are releasing what? They are releasing estrogen, which is why the level of estrogen is now slowly going to go high. And when it does, there is regeneration of endometrium that is happening. It's not fully regenerated, but still. Now, by the end of this particular time, okay, by the end of this follicular phase, what exactly happens is that level of estrogen goes up really high. So usually they work opposite. They work opposite to each other. Whenever estrogen is high, they go down. But there comes a phase where the level of estrogen is super high, such that instead of inhibiting them, they have a positive effect, and the level of LH as well as FSH, it shoots up, especially LH. Especially LH, it shoots up, and now you have what? LH surge. LH surge, and that's when the LH surge happens, the Graafian follicle which is there, it ruptures and releases what? The secondary oocyte. Secondary oocyte is being now released. Now whatever is left behind, what do you call this as? You call this as corpus luteum. You call this as corpus luteum. So basically from the 15th day onwards, you have the formation of corpus luteum. Why did it happen? Because under the influence of LH surge, the Graafian follicle ruptured. So now LH is high only. It cannot come down slow, right? It is high only, and that LH itself, along with FSH, it maintains the corpus luteum. It maintains the corpus luteum, which is why first thing I'll call this as a luteal phase. I'll call this phase as luteal phase. There is one more name to it, and I'm going to talk about that. Now, the corpus luteum, on the other hand, my dear children, will be releasing two hormones. The major hormone which is there is progesterone, and also little amount of estrogen is going to be produced. So until now, after this, the progesterone level never increased. But as of now, my dear children, the progesterone level is going to super increase. And when the progesterone is there, you need to understand the endometrium becomes very, it was just regenerating over here. Here it becomes very, very thick, and also start releasing some things, secreting some very important things. Why? Because it feels that suppose ovum and the sperm meet, zygote will be formed, the zygote will come down, it will form the embryo. The embryo needs nourishment. Who will provide nourishment? The endometrium. That is why it becomes thickened and will start secreting things, which is exactly why I'll call this as the secretory phase as well. Got it? Did you get it? Now what? Now what? So listen, when progesterone estrogen is being released, progesterone hates LH and FSH. It says that I do not want these guys to be produced. Suppose these guys are not being produced, there is an inhibiting effect over here, negative effect. What is going to happen? LH is going to go less. If LH is less, do you think corpus luteum can be maintained? There will be no one to maintain it. Corpus luteum is going to be less. If corpus luteum is less, progesterone also will not be made, which is why again what is going to happen? Progesterone estrogen level is going to go down, and then it again shuts down. That is the whole story. So what are the two hormones out here? It is FSH and LH, pituitary hormones, and ovarian hormones, estrogen and progesterone. Who plays a major role in follicular phase? FSH plays a major role because you want to generate the follicles. Okay, and during this estrogen is also being released, but FSH plays a very important role. Okay, then by the end, because the follicles are also producing estrogen, the estrogen level goes super high, and that's when LH, which is there, LH which was a normal level, it goes up. There's a LH surge leading to ovulation. Always remember, behind ovulation, you cannot write estrogen directly, you have to mention LH first. Okay, now even though because of estrogen high, that's what LH also got high, but still you mention it like that. Now, after that, LH comes down and LH maintains the corpus luteum. Okay, corpus luteum is going to release two hormones, progesterone and estrogen. Again, more estrogen, but even more progesterone. That progesterone will make secretion in the case of endometrium. And yes, and the progesterone hates LH and FSH, which is why it shows a negative impact on it. Now, when LH goes down, corpus luteum level goes down. If corpus luteum is not there, progesterone will not be there. Again, the same cycle is going to be repeated up. This is the whole story. Got it? The very, very important thing. Now over here, if at all you want, you can take a screenshot of this. Yes. So the menstrual cycle is a monthly cyclic change in the ovary and uterus that occurs in human females for about 28 days. Agree. Starting from menstruation to the next menstruation out there. Functions of the hormones in the menstrual cycle. First thing, FSH, the pituitary hormone. What does it do? Stimulates the growth and maturation of ovarian follicles in the ovary. This is the primary function. Secondary function, induces the follicles to secrete what? Estrogen. Now this induces the follicles to secrete estrogen, which is also super important. What does the estrogen do, which is an ovarian hormone? Causes repair and proliferation of the uterine endometrium. And later, secondary function, it leads to LH surge leading to ovulation. Done. This is what you need to write if at all you get a question like this. Again, you can get different question. You might be given dates and you might be asked, disintegration happens during what time? Disintegration of endometrium happens during 1 to 5th day. That is menstrual phase. Regeneration of endometrium from 5th to 13th day. So please know about all this thing. Okay? Yes. Now, what is placenta? List its function. What is placenta, my dear children? Placenta is a vascular connection between the mother and the fetus, and it is something that is temporary. It is temporary. It is there for nutrition, for respiration, for excretion, and also for hormone secretion. So we also call placenta as temporary. We also call placenta as the temporary endocrine gland. Temporary endocrine gland. So what does it do in nutrition? It transfers nutrients from the mother to the fetus. It transfers nutrients from the mother to the fetus. Respiration, it supplies oxygen to the fetus and removes carbon dioxide. Right? Yes, it gives oxygen, takes away carbon dioxide. Excretion, it removes all the waste out there. Okay. And endocrine function, there are 4 hormones which is released by placenta. Please remember the name. First one is HCG, human chorionic gonadotrophin. Second one is HPL, human placental lactogen. Third one is progesterone. So initially it was ovary that releases it, but when placenta is being formed, placenta takes over the job. So it is progesterone. Apart from that, you have one more thing, and that is estrogen. So 4 hormones are released by placenta. None of the other hormones which is mentioned in your NCERT are released by the placenta. There is a name of the hormone, relaxin. Relaxin is produced by ovary during the later phase of pregnancy. It wants the ligaments to be relaxed so that the baby can easily move out. Yeah, and there are certain hormones that increase in many folds during pregnancy. One is cortisol, the other one is thyroxine, the other one is prolactin. Thyroxine, cortisol and prolactin, all of them are super important for the metabolic activities of the fetus as well as the mother. Yes, okay. Next one, name the pituitary and testicular hormones involved in spermatogenesis. Name the pituitary and testicular hormones involved in spermatogenesis. Any idea what are the hormones that are there in spermatogenesis? Well, if you not have any idea, listen to me carefully. Okay, so yes, it goes like this. So what exactly happens? We have hypothalamus, which is the grandfather of all the glands, endocrine glands out there. Hypothalamus releases one thing, that is gonadotrophin releasing hormones. That means it goes and signals the pituitary gland to release the gonadotropins. That is why it is giving gonadotropin releasing hormone. That is a signal that, hey, you release gonadotropins. What are the gonadotropins, my dear children? 2 of them are there. Okay, and always remember, pituitary gland has 2 lobes, anterior and posterior. This one is the anterior lobe out there. So in anterior pituitary lobe, what do you have? You have LH and FSH. They are the gonadotropins out there, right? LH is going to act on the Leydig cells. FSH is going to act on the Sertoli cells. Where is Leydig cells found? So if at all you say the testicular lobule, my dear children, you have seminiferous tubules in the testicular lobule. Inside seminiferous tubules, you have the Sertoli cell as well as the spermatogonia, right? And here in the space, you have the Leydig cells which produces hormones. This hormones has to get inside, and then only sperm formation will be taking place in the seminiferous tubules, right? So Leydig cells, L over here, S over here, remember that. Leydig cell is going to produce the male sex hormone. The most famous among the male sex hormone is what? Testosterone. Testosterone. What about the Sertoli cells? They are going to release one very important thing, that is factors. That is factors. What are these factors? Androgen binding factors, as in factors that is going to make the androgens bind, the male sex hormones bound to them. So why is all this happening? So listen to me carefully. Inside the seminiferous tubule, sperm formation is taking place, right? The spermatogonia, primary spermatocyte divides into 2, you get secondary spermatocyte, again divides into 2, you get basically spermatids at the end. What do you get? You get spermatids, right? These spermatids which are there, they are globular in shape. They are immature. You need to convert them or transform them into mature form, and that is known as spermatozoa. Okay, spermatozoa. Spermatid kits which are there, they are transformed into spermatozoa, and this process which is there is known as spermiogenesis. The whole process is spermatogenesis. This is known as spermiogenesis. So this is happening where? In the seminiferous tubule. Where are the hormones being made? In between the seminiferous tubule. So hormones gets inside, testosterone gets inside the seminiferous tubules, and basically the level of it has to be maintained for all this process to happen, especially for this particular process to happen, for spermiogenesis to happen. That is why, to maintain the level of hormones, you have the factors. Factors say, testosterone, you do not go anywhere, you stand here only, such that the whole process can happen smoothly, especially the last process. So if at all you are getting a question that if FSH is not being released, which stage is being affected, you should know it is the formation of spermatozoa which is majorly being affected, even though all of it, but majorly formation of spermatozoa is being affected, and this is known as spermiogenesis. Okay, cool. I hope you will not forget it. Yeah. So name the pituitary testicular hormones. What is the pituitary hormone? LH and FSH. And name the testicular hormones. It is basically testosterone. That is the male sex hormone out there. Done? Done. So just know the function. You can get any questions on the basis of it also. Now, parturition is induced by a complex neuroendocrine mechanism. Comment on it. Okay. So, just a second. Parturition means what? Expelling of the baby. Movement of the baby outside the uterus. Now, why do you say it's neuroendocrine mechanism? Because your nervous system is involved. Also, your endocrine system is involved over here. Hormones are going to be released over here. Can you believe that? Yes, that happens. So, you have to be really, really careful while I talk about this. This is going to be the uterus, Baba. In the uterus, the baby is going to be formed. Agreed? The fully developed fetus which is there, it will slowly try to move out of the uterus. It will slowly try to move out of the uterus. When it does, what is going to happen? There are some receptors over here in the cervical region that becomes active. It becomes active. The fully developed fetus tries to move out, but before that, one more very important thing happens. Basically, it is going to release some hormones. Okay, and there's some chemical coordination that is happening between the fully developed fetus as well as the placenta. So these two guys, fetus as well as the placenta, they have some chemical coordination between them such that there is a release of prostaglandins, a type of hormones out there. Okay, there are type of hormones that are prostaglandins which causes initial contraction and relaxation of the myometrium, of the muscular layer. So both of them, fetus and placenta, both of them have some chemical coordination, which is why there is going to be release of hormones which would lead to initial contraction and relaxation that could make the baby move down. When the baby moves down, my dear children, the receptors are going to be active. What receptors? Some stress receptors are going to be active. This receptors, until now everything was hormonal in some way, but now when receptors are active, they are going to send a neuronal signal, nervous signal to what? To the pituitary gland. Okay, to the pituitary gland of whom? Of the mother. Okay, anterior lobe or posterior lobe? It is posterior lobe of pituitary gland. And now, my dear children, the mother is going to release one very important thing that is known as oxytocin, a hormone. So, nervous signal, again a hormone. This oxytocin which is there will be coming and binding onto the myometrium, the muscular layer, the smooth muscular layer, right? It will come and bind on it, and what is going to happen? Contraction, flexion, and the baby will move out. Again, the same receptors become active, again more oxytocin, and it is a positive loop that is being created until and unless the baby moves out. So first thing, fetus and placenta gives the initial signal, and that is known as fetal ejection reflex. What do you call this? Fetal ejection reflex. That is the first thing that comes in, and then only you have neuronal signal, that is a signal towards the pituitary gland of the mother for release of oxytocin, and then again hormonal. Fine, you have to explain this, you will get your marks. Yeah? So fetal signals, parturition begins when the fully developed fetus and the placenta release hormones signaling the mother's body to prepare for delivery. Now, the maternal pituitary gland secretes oxytocin which stimulates strong uterine contraction. Before that, you can just say that receptors in the cervix becomes active. Receptors in the cervix becomes active and send signal. And send the signal to what? To the pituitary gland. And that's when all of this will be happening. Now that is something that comes in the middle. Okay, please remember it. Yeah? Yes. Perfect. So other important topics which are there, which could be coming in this particular chapter is hormones involved in gestation, parturition, etc. All the hormones, please go through it once. I've explained HCG, HPL, estrogen and progesterone. Those are the placental hormones. Apart from that, as I said, ovarian hormone, that is relaxin. Apart from that, you have this thing. What do you call that? Apart from that, cortisol, prolactin, and thyroxine. Okay, that is increasing manifolds in the blood of the mother. Please know about that also. Yeah. And male accessory glands, ducts. So accessory glands, seminal vesicle, prostate gland, bulbourethral gland. Seminal vesicle secrete what? Fructose. This is for nourishment of the nourishment of the sperms, to provide energy to the sperms. Prostate gland adds calcium, okay, as well as some enzymes to seminal plasma, and bulbourethral is for the lubrication of the penis. So these three things together, it forms seminal plasma. That is the fluid, and when sperm is added into it, we say that basically it gives rise to semen. There is formation of semen up there. So please remember this, very, very, very important. Yeah. Now, month by month development of the embryo. So first month, what happens? First month, there is heartbeat. So when someone dies, we check for the heartbeat. Similarly, when someone is born, we check for the heartbeat. First month, heartbeat formation happens. Second month, what happens? Limbs and digits are going to be formed. Limbs as well as digits are going to be formed in the second month. Third month, that is the end of first trimester. Trimester means first 3 months. Now, in each month, always remember, 1 month which is there is equals to what? 4 weeks. So 3 months, if I say, so 3 into 4 is equals to what? 12 weeks. 12 weeks. After the end of 12 weeks, major organs, limbs and external genitalia is going to be formed. Organ system, the limbs and the external genital organs. External genital organs is going to be formed. You will get to understand male or female or everything else. Okay? Now, 4th month has been skipped over here. 5th month, the baby says, God, I want to move, I'm just bored. So first fetal movements and also first hair on the head is found on the 5th month. Now what happens in the 6th month? God said, you wanted hair? No, have hair on the body also. So there will be small tiny hairs on the body, and apart from that, the eyelids are going to separate. Eyelashes are also going to be formed by this particular point of time. Yeah, this is end of the second view of the female reproduction system. Over here you have to mention each and everything, as in the ovary, the parts of the fallopian tube, and every single diagram labeling should be there, including the uterine fundus region. Okay, so I'm not explaining much. The diagram is given, you have to practice it. Okay, and by the way, do you want a separate video on the important diagrams? You can let me know in the comment section if at all you need it. So in this 5 marker, you can get one diagram from this chapter. Always remember that. Okay, so draw a neatly labeled diagram of sectional view of mammary gland. This is the mammary gland. Again, please know the flow of it generally, which is like this. Dal fell on mat. So basically, write mat first, and then you put dal. What is mat standing for? This is mammary lobes. Mammary lobes have alveoli. Alveoli opens into tubules. Tubules opens into duct. Duct is going to form ampulla. Ampulla is going to open into lactiferous duct. That is how it goes. So dal fell on mat, or mat dal, whatever you want to remember, you can remember. And yes, so 2A is there here, alveoli, there ampulla. So ampulla ends with L, so you can remember like that also, that after this A you have L, that is lactiferous duct. So yeah, so here also the flow is the same itself. So see these things, you have multiple lobes. The lobes are opening into small alveoli. Alveoli, my dear children, will be opening into what? Tubules. Tubules which are there, these small things which are there, they are forming what? A duct over here, and the duct is going to come together, from multiple ducts are going to come together, form the ampulla. Ampulla will be forming what? Lactiferous duct. Over here you have the nipple, you have the areolar tissue, and the back side you have rib. In between the ribs you have muscles out there, and fat is also towards the side. Okay, fat is also there towards the side. So this is general idea. Yes. Yeah? Yes. Next thing, write the schematic representation of spermatogenesis. This I can explain to you right now, because I think it's something that might need explanation, right? So let's go ahead. First of all, when I say spermatogenesis, the first thing that should be coming into my mind is it starts from puberty. And over here, what do you have? You have some cells which are known as spermatogonia. These cells are normal cells. It will be deployed in nature. Now these spermatogonia which is there, it will undergo mitosis. What will it undergo? Mitosis, to form multiple cells, to increase their number, right? Now when they increase the number like this, one cell among them becomes differentiated. I'll say it matures. It feels that I want to form the sperms later on. This cell is going to be known as what? The primary spermatocyte. The primary spermatocyte. How many number of chromosomes are going to be there? Baba, 2n only. Why 2n? Because this is mitosis. This is not meiosis, it is mitosis. This primary spermatocyte is going to divide. And from here onwards, meiosis is going to happen. And you know meiosis happens in 2 stages. So this is the first stage when you get 2 cells. What is an haploidy of these cells? It becomes half. After the first meiosis, it becomes half. Haploid numbers. So from this point onwards, it becomes haploid. And what do you call these cells as? Secondary spermatocyte. From primary, you got secondary spermatocyte. Now what happens? You need to do meiosis 2. In meiosis 2, what is going to happen? Let's see. You get 2 cells over here. You get 2 cells over here. Here the number of chromosomes remain the same. Here also the number of chromosome remains the same, right? And this is going to be what? Meiosis 2. This is going to be meiosis 2. What do you call these cells as? You call these cells as spermatids. Ma'am, isn't it known as spermatozoa? No. These are spermatids. They are usually globular in shape. They have not attained the elongated shape until now. So how do you remember who came first, spermatids or spermatozoa? Spermatids, they are like kids. They are immature. They are small kids. So now the immature. So until now you had spermatogenesis. Still you have spermatogenesis. Listen, this is a last stage which is there, where the spermatids, the immature spermatids, are going to transform into mature sperm. What do you call them as? You do not call them sperm, rather you call them as spermatozoa. What do you call it? Spermatozoa. And ma'am, is this technique having a specific name? Is this process having a specific name from here to here? Yes. This is known as spermiogenesis. It is known as spermiogenesis. This is very, very important. Do not get confused between spermatogenesis and spermiogenesis. This is spermiogenesis, that is spermatogenesis. Later, the spermatozoa which are being formed, they will be moving out to the seminiferous tubule, and that is known as aviation, that is spermiation. Okay, that is movement of the sperm outside, or spermatozoa outside the seminiferous tubule. Works? Works, ma'am. Perfect. So always remember that until here it was 46 number of chromosome, but from here it becomes half number of chromosome. From one spermatogonia, how many sperms? 4 sperms you get. Okay. Now we go on to the next question. Where's that thing? Okay, just a second. Yeah. Draw a labeled diagrammatic sectional view of human male reproductive system. Again, you have to draw the whole diagram up there. Make sure you not do any mistake. Do you need my help? I'm so sorry for doing that, but yes, I have cold. Right? So if you need my help, I can help you with that in some ways. So listen, first of all, you make the testis. The testis has testicular lobules. The testicular lobules will be having seminiferous tubules. The seminiferous tubules which are there, they are going to open into a duct. The first duct out there, which you call as rete testis. What do you call this as? Rete testis. Now rete testis says, oh my god, the sperms want to move out, let me exit, let me exit, let me exit. So who helps in that? It is vasa efferentia. E for exit, you can try to remember. It is trying to exit. These are vasa efferentia. But you know what? They are blocked over here because now you have the third duct out there, which is known as epididymis. This is actually 6 meter long duct out there. And this is where the maturation of sperm actually happens. Now from here you get a duct that goes into the body. So everything was happening in the scrotum until now, but now it goes into the body. What is this duct? This is known as vas deferens. So D for distance, it is traveling to distance. Where does it go, Baba? It coils around the urinary bladder. So this is going to be the urinary bladder, which looks like this. Okay, it coils around that, comes over here. It is going to receive a duct, an opening from a gland. This is known as seminal vesicle. Similarly, a seminal vesicle is on that particular side also. Now both of them unitedly are going to open, having a common duct. This duct's name is what? Ejaculatory duct. Ejaculatory duct. So this is what? A gland, accessory gland, you call it seminal vesicle. Now we have a duct. Two of the ducts form and opens up over here. What is this particular thing opening? This is basically the urethra. This is the urethra. So ma'am, urine comes through this and sperm also? Yes, they have a common passage. Urethra is being covered by the male part, which you call as what, Baba? What do you call this particular part as? This is the penis. And over here you have the glans penis, the enlarged portion of the penis. And here you have a loose fold of skin, which is known as foreskin. Okay. Now, here I told you there is one gland. So there are two glands, two paired glands. You call it as seminal vesicle. Seminal vesicle. After that, there is a donut shaped gland over here. Do not write donut shaped, just remember it. It's single, it is not paired. You call this as prostate. Prostate gland. And then here you have again a paired gland, and that is known as bulbourethral gland. Bulbourethral gland. You also call this as the Cowper's gland. Okay, and that is exactly how. So this side and that side also you have to make and explain. Cool. Works. So yes, you had to mention these parts. So I have mentioned almost all of it, so you know that. Next thing, given below is a flowchart showcasing ovarian changes during menstrual cycle. Fill in the spaces with the hormonal factors. By the way, I have solved the questions in 2-marker, 3-marker session also, where I have explained about menstrual cycle. Watch those videos. Okay, so everything is linked. So obviously every single time when a video is being made, we cannot go on repeating things, otherwise the video will be too long, right? So which is why you need to understand some part of this chapter will be there in another video. Please refer to that video. Okay, and here I am going to talk about the next thing. So given below is a flowchart of ovarian changes during menstrual cycle. Fill in the spaces with the hormonal factor. Let's see. So this is for the menstrual cycle. Primary follicles which are there, they mature into Graafian follicle under the influence of A. So what is that? That helps in the maturation of follicles. What is that hormone? Follicle stimulating hormone, right? It is follicle stimulating hormone that is going to help with this. So I'll just write it FSH. They help in the maturation. Now what? The Graafian, where is it go? The graphene follicle which is there, it is going to rupture, and that is how you get the ova. So graphene follicle and then ova is written. So you have to guess it is about rupturing of graphene follicle to produce ova. Who helps in the rupturing of graphene follicles? Ma'am, estrogen. No, Baba. Estrogen is going to tell LH. The LH is going to go on increasing basically because of estrogen. LH is going to have a spike. This LH spike is leading to what? Ovulation or the rupture of graphene follicle. So I should be writing LH, right? Yes. So over here the answer for this question is going to be what? B is going to be LH, luteinizing hormone. Done. Now what do we have? Now you have to think, okay, I'm not going to tell the answer. You have the ova. Basically now it forms the corpus luteum. How does ova form the corpus luteum, ma'am? Imagine graphene follicle which was there, it ruptured. Ova is being released, or basically secondary oocyte is being released. What is left behind? It is the corpus luteum. That is what you mean by this. Now the corpus luteum, arrow this, and it is written C. What is this thing which is released by the corpus luteum? What do you think is released by corpus luteum? It is a hormone that is known as progesterone, and progesterone will make the endometrium even more thickened. So secondary oocyte is being released. Any point fertilization can happen. Suppose fertilization happens, there should be someone to nourish the embryo. Who does that? Endometrium, which is why the endometrium has to be very thickened up and also should be secreting things, which is why you need progesterone. Progesterone majorly does it. So corpus luteum releases two hormones actually, progesterone and estrogen, but majorly progesterone. So you can write both progesterone and estrogen, but majorly please write this one is the major hormone which is going to play an important role over here. Works. Now, women experience two major events in their lifetime, one at menarche and one at menopause. Mention the characteristics of both the events. At menarche, menarche is what? The start of the reproductive phase. It is the start of the reproductive phase, right? Menopause is when it's the stop, it's the end of the reproductive phase. So beyond that, she cannot reproduce and give fertile offspring out there. So basically that is menopause. So write the characteristics of both the events. How do you write it? It's very simple. By the way, I have written everything which is needed for you to understand this particular question. And menarche, the first onset of menstruation at puberty, marks the beginning of reproductive phase in the case of females. You have to write that just all. Menopause is the permanent cessation of what? Menstruation, as in permanent stoppage of menstruation. It marks the end of reproductive phase, isn't it? Yes, ma'am. Done. That's all. That's all. Now, draw a sectional view of seminiferous tubules of human and label the following cells in the seminiferous tubules. So, sectional view of seminiferous tubule is something that you might have missed. Do not miss it and please know it. First of all, before decoding the sectional view, you should know these things also. Cells that divide by mitosis to increase their number. Which cells are there which increase their number in seminiferous tubule? During spermatogenesis, first one, spermatogonia, they increase the number, right? So can I write spermatogonia over here? Now these things which are being asked, you have to mark it there. So you have to write it first, then you have to mark it there. That is how you get the marks. Now cells that undergo meiosis 1. Primary spermatocyte, which is differentiated in form, it is the one that will undergo meiosis for the first time, right? So the one that undergoes meiosis 1 is primary spermatocyte. Okay. Cells that undergo meiosis 2, secondary spermatocyte, that is the one that will undergo meiosis 2 to form spermatids. So I write secondary spermatocyte. Okay. Cells that help in the process of spermiogenesis. Spermiogenesis is what? Spermatids to spermatozoa formation. And ma'am, what cells are there that helps in that spermiogenesis? It is not spermatids, not spermatozoa. Basically, the cells which are there, they are Sertoli cells. Remember, Sertoli cells are there for nourishment of the spermatogonia and the entire process which is going to take place there. It is the one that takes care of it, especially the last process, that is spermiogenesis. So under the influence of Sertoli cells, so you know what happens? FSH hormone is there. FSH, follicle stimulating hormone is there. It acts on the Sertoli cells. Sertoli cells are going to release some factors. These factors are going to make sure that the hormones are enough for the whole process of spermatogenesis to happen. So for maintaining the hormones up over here, you need to make sure factors are being released. How do you get hormones? There is one more LH, luteinizing hormone. In the case of males, this is how it works. LH goes to act on Leydig cells. Leydig cells releases the male sex hormone, that is testosterone, or you broadly call them androgens. These androgens are now, this is happening inside the seminiferous tubule, this is outside. Okay, sorry. Sertoli cells and everything is there inside the seminiferous tubule, and this is happening outside. So androgens from outside comes inside the seminiferous tubule for the whole process to happen. But to make sure that their amount has been maintained over here, you need some factors. Who release those factors? Sertoli cells. Especially those are being needed in the process of spermiogenesis. So cells that help in the process of spermiogenesis are going to be what? Are going to be Sertoli cells. Why? Because they are the one that releases the factors which make sure that there is enough amount of testosterone over here. So can I write Sertoli cells? Yes. Do I need multiple understanding? Yes. I do need understanding of multiple concepts, and only I can write this answer. So please know that. Okay. Now cells produced due to spermiogenesis. Answer. Cells produced due to spermiogenesis. At the end of spermiogenesis, what do you get? Spermatids transform into spermatozoa. So cells produced due to spermiogenesis is what? Spermatozoa. It is spermatozoa. Got it? Yes. Now how do you draw this diagram? This is how you are going to draw it. So you are having spermatogonia right over here. The spermatogonia which is there, it is going to divide, going to form primary spermatocyte. The primary spermatocyte which are there, they are going to divide, form secondary spermatocyte. So here it's just cell which looks almost the same. Right? It looks almost the same. So you have to form multiple cells over here. In between that, you have to make other type of cells. These cells are what? Sertoli cells. That provides the nourishment and also produces the factors which make sure that the level of hormones are being maintained up over here. Now, towards the end, my dear children, you can show spermatozoa as well as spermatids. Spermatozoa and spermatids. Spermatids actually here in this picture has been mentioned like this only, but you can basically show a globular structure also. It's not a big problem. Okay, so please do that, but according to NCERT, whatever is given in NCERT, go according just to that. So yes, fine. When you write the answer, write here A, mark here B, C, whatever is there accordingly. So this was also a question that was given in your book. Now apart from it, there are a few other things. Menstrual cycle, as I told you, hormones involved in gestation, parturition, month by month development data. All of this is super important. In 2 and 3 marker session, I have explained it. Go watch it. Go anyway, any chapter you have to watch the 1 marker as well as all 2 and 3 marker questions. Otherwise, there are chances that you might miss out on concepts. So do not do that mistake. Okay? So I'm not explaining it here. Otherwise, it will take too much of time, right? And it will be repetition. So yeah. Now we go ahead, the next chapter, that is reproductive health. You're champions! So did you drink water? I hope you're doing great. And by the way, everyone who have reached until this particular point, let me know in the comment section that ma'am, level 1 crossed. I will be really happy to see that. Actually, I'll be really happy when you cross the last level out there, yeah. But still, you can keep on updating in the comment itself. I'll be really happy with that. I said so many times really. Okay, so basically I just got a headache and I went and put in so much balm around all different parts of my face, which is why I'm not even able to open my eyes now. Okay, so yes, let's go ahead with the first question that we have, and this is from your MQPs. What I will, yes. So oral pills as contraceptive acts as contraceptive effectively. Oral pills as contraceptive effectively functions by inhibiting what? Okay, so oral pills, when I say that they are contraceptives, how does it show their effect? How they are actually doing their effect? What do they inhibit? And maybe acid, richness, nostoc, ovulation, colostrum, diversity. So you all know it's basically going to be ovulation itself. But why do you think that oral pills are going to inhibit ovulation? Listen, oral pills has a combination of progesterone or estrogen together. Otherwise, it can be plain progesterone and estrogen alone as well. Anyways, these hormones which you see are generally required for the process of ovulation and also for the maintenance of endometrium. Now, when you are providing it from outside, basically that is going to create what? An imbalance. And since there's an imbalance that is going to happen, it will do the opposite of what it was doing. So since usually it helped in ovulation, now it won't. Usually it helped in formation of thick endometrium, now it is not going to be forming thick endometrium. Instead, you will be having thin endometrium being formed. And this thin endometrium cannot support, cannot support any fetus out there, which is why obviously pregnancy will not be happening. So that is how usually it works. Okay, so oral pills act here. By inhibiting what? Ovulation. Do you know your ma'am is this brilliant that she almost put the balm inside, the roll-on balm inside the eyes. Oh my god. Yes. Now, which of the following is not a copper releasing IUD? You have to spot that. Which is not? Not. Children sometimes what they do is that they try to find that which one is copper releasing. So please know it is asking about which one is not. Okay. LNG20, LNG20, CUT, QT is copper T basically, and copper T is copper releasing only. CU7 is again copper releasing. So here CU2T looks like this, onto which you have the copper round. CU7 looks like this, onto which you have the copper round. That is the only difference. Now multi-load 375, what about that? This is also copper releasing IUT, and over here you have 375 mm or something like that of copper being releasing, which is why not mm, some unit was there, which is why you call this as multi-load 375. So this is also copper releasing. So what is left behind? LNG20. LNG20 is levonorgestrel. Basically it's a synthetic form of progesterone, my dear children. It is coming under hormone releasing IUD. Okay, and there is hormone releasing IUD, there is copper releasing IUD, there is non-medicated IUDs, right? So this comes under hormone releasing IUD. It functions similarly just like how oral pills were working, because over here also whenever you're giving synthetic hormones, for example progesterone, it creates an imbalance leading to ovulation not happening, or thin endometrium being formed, or the cervix being very cervix having one mucus around it, mucus cap around it, such that the sperms cannot move inside. So that is a general thing that is there. So this is hormone releasing, and 3 of them are copper releasing. Copper, what does it do? It messes up with the motility of the sperm and also the fertilizing capacity of the sperm. So copper, they go and stick like to the sperm's end and say that you cannot move. That's how it works. Okay, so you can remember like that if at all you want. Yes. Now, statement number 3, question number 3, statement number 1, gift is an in vitro fertilization technique. Is it? Gift means gamete intra-fallopian tube transfer. Do you think it's an in vitro fertilization technique? In vitro means what? In vitro means inside the laboratory. Gift is where you take the gametes and you transfer the gametes into the fallopian tube. Why? Suppose there is a female X. Okay. She is having a proper uterus, everything, but she is not producing viable types of ovum, or an ovum which is there is not of good quality, which is why fertilization is not happening. Rest everything is really cool up there. So basically in gift, what do? Some other lady who has a normal ovum will be gifting her ovum. That is what gamete. Okay, gamete is being gifted into what? Inside the fallopian tube, it is being transferred as well. This particular gamete that is being gifted is being transferred into the fallopian tube. Why? Because you are relying on your own body for fertilization to happen. The sperm will be coming by natural sexual intercourse, and the sperm will be basically fertilizing the ovum out there. So here it is not in vitro, rather it is in vivo. Opposite of it. It's inside the body, naturally occurring. So this is a wrong statement. Statement number 2. In gift, gametes are fertilized outside the female body. Are they same thing is being said? Okay, do you think gametes are fertilized outside the female body? Is it? Gametes are fertilized inside the female body. That is why I told you it's in vivo, not in vitro. So both the statements are wrong, but I do not know what I have marked. Yes. So statement 1 and statement 2, both of them are wrong. Incorrect. Okay? Yes. Next question. Out of the options given below, choose the correct stage for transfer to the fallopian tube for successful IVF results. Out of the options given below, choose the correct stage for transfer to the fallopian tube for successful IVF results. When do you transfer the embryo? So, in vitro fertilization, that means fertilization in the laboratory happens. After that, you get the zygote. This zygote can be single celled, right? Or later it can divide to form the embryo. Now, embryo up to 8 cell stage, okay, can be transferred into the fallopian tube. Here you have been asked about that only. Up to how many stage you can transfer into the fallopian tube? It's only up to 8 blastomeres or 8 cell stage. The cells are known as blastomeres. Now if it is beyond 8, you cannot put it in the fallopian tube. Why? Because ectopic pregnancy can happen. That means in the fallopian tube the baby can be found, and that can be dangerous for the mother as well as child. It will be dangerous for the mother and the child. So embryo which is up to 8 blastomeres can be put in the fallopian tube. If at all it is beyond that, you have to put it in the uterus. It is known as intrauterine transfer. In that case, you call this as intrauterine, inside the uterus transfer, intrauterine transfer. In this particular case, you call this as ZIFT, Zygote intra-fallopian transfer. So it is not zygote, it can be an embryo up to 8 blastomeres also. So either zygote or an embryo up to 8 blastomeres is transferred into the fallopian tube. Works? Works, ma'am. Okay. So your answer for this question is A. Now, reasons for human population explosions are given below. Reason why human population is increasing is given below, and you have to select the correct answer statements from the options. Okay. Rapid decline in maternal mortality rate. Mothers are dying less, which is why population is increasing. Yes, mothers are dying. There's a proper care being given. It's a good thing only. Okay. So this is definitely helping. Now, rapid decline in infant mortality rate. Again, the babies are also dying less, which is why the population is also stable or increasing. Now, rapid increase in death rate. If people are dying more, do you think it will lead to population increase? No, Baba, it will lead to population decrease, right? So this is wrong, because here it is written rapid increase in death rate is leading to population explosion. No, that cannot happen anyways, right? Now, increase in the number of people in the reproducible age. Again, if people are there who are going to reproduce in the future and their number is more, obviously the population explosion is going to happen. So I'll say 1, 2 and 4 statements are correct. 1, 2 and 4 statement, that means your answer is going to be C, Baba, right? Your answer is going to be C. Yeah? Yes. Now the next question that we have is question number 6. Which of the following sexually transmitted disease is not completely curable? So there are some STDs or venereal disease or genetic tract infections which can be treated, which can be cured also. But 3 of them are there which cannot be cured. And 3 of them has H in it. One is HIV AIDS, other one is genital herpes, and the third one is hepatitis B. Hepatitis B. Out of this, if at all you see any of these options, your answer is that. Man, there is no HIV, there is no herpes, there is no hepatitis B. You see genital herpes, that is itself the herpes. So your answer for this question is going to be C, Baba, which is not completely curative. Yeah? Yes. Okay. Next, an example for non-medicated IUD is non-medicated IUD. That means this is an intrauterine device. You put it inside the uterus only, but it is not going to release anything in your body. It's not going to release any hormones or medicines in your body. What is that? So CUT, copper T is releasing copper. LNG is releasing levonorgestrel, the synthetic form of progesterone. Multilode 375 is also releasing Cu, that is copper ions. Lipase loop is there, which is going to be the answer. It is the example for non-medicated IUD. So basically, suppose this is the uterus. In the uterus, the shape of the uterus is like this. Similarly, you put something like this over here, which looks like a loop. This is known as lipase loop. Okay, and it is not there to release anything. Basically, when something foreign is being put in our body, what happens? Our body reacts to it by producing immune cells, okay, that fight against the pathogens. Now suppose the sperm enters the cell, enters our body. What is going to happen? Our immune cells feel that this guy is also a problematic guy, so we will kill them. Phagocytosis is going to happen. The cells are going to be killed. They're going to be taken in and be killed by the immune cells out there. Yeah, so lipase loop is there for generally activating our immune system is what I'll say. Yeah. Now the assisted reproductive technologies in which a sperm is directly injected into the ovum. Sperm is injected into the ovum. Why do you do that? In the laboratory condition for fertilization to happen, right? What do you call this process as? You have a specific name. Remember, do not mug it up. This is the egg. This is the cytoplasm of the egg. And suppose you are putting in the sperm with the help of a pipette. You call this as intra, that means inside. Inside what? Intracytoplasmic. Inside the cytoplasm, what you are doing? You are injecting the sperm. So sperm injection. Intracytoplasmic sperm injection. So your answer for this question is ICSI. ICSI. Intracytoplasmic sperm injection. In this particular part of the chapter, if at all you go around mugging things, you will be forgetting it. So please think about it and you will get your answers. Yeah? Yes. Next, coitus must be avoided from day 10 to day 17 of the menstrual cycle. The reason is, okay, this is a natural method of contraception and it is said that it does not have any side effects, but it has high failure rates. Yes. So why do you think coitus or sexual intercourse is being avoided from day 10 to day 17? Because, my dear children, I told you, if your cycle is of 1 to 28 days, the mid-cycle is 14th day where ovulation can happen, but we are not sure that ovulation will happen on 14th day. It can happen over here. It can happen until here also. So 10 to 17 days, I'll say it's the fertile window where the egg can release any single time. And always remember, Suppose the egg is not released, but the sperm is being put inside your body. That means sexual intercourse have taken place. Sperm after ejaculation can stay in your body for 5 days. That means 72 hours. Now, later on, if the egg is being released, the sperm is still there, they can fuse. You get it? Which is why we say that it should be avoided. So, sperm remain viable for 7 days. Ovum may be available for fertilization. Yes, that is the reason. Ovum may be available for fertilization because that is the ovulation period. That is a fertile period that is out there. Okay? Yes. So, if at all, yeah. So, ovum loses viability on these days? None of these? No. So, I hope you you got to understand the answer. Why 10 to 17 days? Because here you're specifically talking about ovum. Ovum could be available. Sperm, though, by any time it can be available, but ovum is available only during these days. Okay? Yes. Now, yeah. Condoms have additional benefit as compared to other contraceptive because. What do you think? Additional benefit as compared to other contraceptive because what? What is the reason for that? Or what does it do? It is made up of rubber and latex. Other things are also made up of rubber and latex. We have so many things made up of that. So example, diaphragm and things like that. Okay. Provides privacy to the user. Almost all the things provides privacy to the user. Protects against STIs, sexually transmitted infection. Do you think that is the additional benefit of having a condom? Yes. Because when you use a condom, suppose it is a penis, it is being covered by a condom. Any genital fluid is trapped over here in the condom itself. Genital fluids are not being exchanged, and if genital fluids or blood, anything is not being exchanged, you say that there are lower chances of getting an STI. You get it? So it protects against STI, that is the best part. It prevents implantation, no baba. That is a whole different thing. So C is your answer. It prevents against STI. That is an additional benefit that you get from condoms. And that is the only one that can provide that benefit. Now, there's a statutory ban on amniocentesis as. Why there's a ban on amniocentesis? It reveals the chromosomal anomalies. Chromosomal anomalies means problems in the chromosome. If it reveals, it's actually good now we get to understand that, right? And we can treat it. So that's not a reason. It can be used for determining the sex of the unborn. It can be used to reverse the sex of the fetus. No, obviously not. It can be used for correcting the genetic disorders. Yes, it can be. But do you think it has been put a ban on it because of the sole reason? No. The reason is that basically it can be used for determining the sex of the child as well. And when they get to understand that it's a female child, they go ahead with female feticide. That is why there was a ban being put on amniocentesis. So always remember, amniocentesis were actually found out for finding the genetic disorders out there. And by genetic disorders, I mean hemophilia, sickle cell anemia, Down syndrome and things like that. Not jaundice. Jaundice is not sickle, what do you call, it's not a genetic disorder, okay? And apart from that, I can also find the sex of the child, but that was not the primary reason for inventing amniocentesis, right? The primary reason was for finding genetic disorders, but people went ahead, found the sex of the child and killed when it was a baby girl. So that was a very, very big problem. So that's why a ban was being put. Okay, yes. Now, select the options with correct identification of the given contraceptive measure and the hormone essentially present in it. So this is the contraceptive thing that is being there. And what do you think is the, yeah. So is it an IUD, intrauterine device? Do you put it inside the uterus? No. Everything that you put inside the uterus looks like the uterus cavity is. Some shape would be there like that. What about CUT? Copper T is definitely not the answer. That is IUD, right? And subcutaneous implant, progesterone and progesterone estrogen combination. Intramuscular implant. What do you think this is? This is a subcutaneous implant. It's an implant which you put it below under the skin. What does it release? Over here it is said you have to find the name of the given contraceptive measure and also the hormones essentially present in it. So the hormone is what? Progesterone or progesterone estrogen combinations. So subcutaneous implant, and apart from that, it is what? It's a combination of progesterone or progesterone estrogen combinations out there. Okay, and again it does the same thing just like oral pills. Prevents ovulation, and it will be not allowing the endometrium to be thickening up. There will be cervical mucus thing that is going to be formed. It does not allow the sperm to move around. So always remember that. Yeah. Next thing, a lady is 8 weeks pregnant. 8 weeks pregnant. 8 weeks means how many months? 8 weeks means how many months? 2 months, right? Okay. Various tests done confirm that if a child is born, it would suffer from a serious illness. What is the best suggested option in this particular case? It's been 2 months only, and the baby might suffer from a serious illness. What could be done? Amniocentesis is to be performed, emergency contraceptive pills is to be taken, MTP is to be performed, tubectomy is to be performed. Listen, probably amniocentesis might already been performed. That is why you say that the child is suffering from the serious illness. The next thing that you should be doing is MTP, medical termination of pregnancy. This has to be done. This has to be done. Medical termination of pregnancy has to be done because you know that the child, if at all it is born, would have a serious illness. So that is a thing, and it's just 2 months. So it is definitely safer for the person also. Just a second, okay? Over here I have written amniocentesis is to be performed. Just give me a second. I'll be back. Okay, so I just changed the color of the answer so that when you get the PDF, it's not wrong. Okay, so it is MTP is to be performed, not amniocentesis, because with the help of amniocentesis, obviously you have found that serious illness is there, or basically with some other method also you could have found that serious illness is there. Now what is the best suggested option in this case? The next thing that you should be doing is what? MTP has to be performed. That is the next thing. Okay, medical termination of pregnancy. Cool. Now which of the following contraceptive devices is having a common mechanism of action? Has the same mechanism of action? Vows and condoms, both of them, they are physical barriers. CUT and multiload, both of them are copper releasing IUDs. Progesta cert as well as cervical cap. Progesta cert as well as cervical caps, definitely not, because cervical cap is a physical barrier, and progesta cert is, my dear children, something that is related to hormone releasing thing, right? So not. Mala D and lipase loop. What do you think is the answer for this particular question? Please think about it and then only tell the answer. Think very nicely. It is this. Why? Because walls and condoms, they are physical barriers, but walls acts as a specific location. Condom, my dear children, again over here you have a whole coverage. In the case of walls, it is kind of different. Okay, there might be vaginal walls and things like that. This used to cover the vagina. Now condom is not like that. It can be used in the case of males as well as females, and it has a different way. It also protects against STIs as well. So there is a difference. When you talk about CUT and multiload 375, both of them has a common function, and that is to produce what? Copper ions. Copper ions are going to mess around the sperm motility as well as the fertilizing capacity, which is why this would be more correct. Malady is not there in your NCERT, which is why you should not even consider that. So yes, that is the only answer that you need to have. Cool? Yes. Next question. If a female has rudimentary ovary but can provide a suitable environment for the growth of the fetus, the ART technique or the ART, assisted reproductive technique, that can be used is, so here she has a rudimentary ovary. That means her ovary cannot produce enough amount of good ovum. Okay, rudimentary ovary means underdeveloped ovary, might not be producing ovum itself. That can also be a case. Now in this particular condition, you need to notice one more very important thing, that there is she can provide a suitable environment for the growth of the fetus. That means uterus and everything is fine. What is the best assisted reproductive technology that can be suggested over here? It can be IVF-ET. IVF-ET means what? In vitro fertilization, embryo transfer. Embryo, why do you need to transfer the embryo directly? You can just give the egg because she cannot produce the egg, right? So it is going to be gamete intrafallopian transfer, GIFT. Just provide the gamete, rest everything is fine. Not artificial insemination, because in artificial insemination, what do? You put the semen inside the vaginal area or inside the reproductive tract. If at all inside the reproductive tract, you will call this as intrauterine insemination. If in the vaginal area, generally you say artificial insemination. But that is the case when basically the sperms are naturally not going ahead and doing the fertilization process, or when the motility is less, you take it from the donor and you provide it. So it happens in that particular condition. Okay. Now CUT and multiload 375. What about this? No. There's only one answer, that is the straight question. Cool? Yes ma'am. Cool. Now we are going ahead with the next chapter, but before I go ahead with that, I just want to tell you one more very important thing. Until when is it safe to do MTP? It is safe to do MTP until 3 months. And by 3 months, I mean 12 weeks. But until what time can it be done? That is also a question. So until what time it can be done? It can be done until 24 weeks. That means 6 months. But until what it is safe? Okay, very, very important. Do not mess around over here. Got it? Yes. And yeah. In which year MTP was legalized? And in which year MTP Amendment Act was being put? So please know about that. MTP was legalized in 1971 is what I remember. Okay, and basically if at all you add +1 over here, it becomes 20, and here if at all you do ulta, it becomes 17. So in 2017 the MTP Amendment Act was being put on, according to which if at all a person is performing MTP at this particular time, you need the permission from at least one medical practitioner, and if at all 6 months, then you need the permission from 2 medical practitioners. Now let's go ahead with the next chapter, that is reproductive health. Here you will get one marker, one 2 marker, and one 3 marker. So be prepared for the 2 and 3, and let's go ahead with the most probable one, that is mention any 4 assisted reproductive techniques that can be advised to couple with infertility problems. If at all someone is infertile, what are the techniques that you can suggest? First one that is there is IVF, in vitro fertilization, where the sperm and the egg is taken. Suppose fertilization is not naturally happening inside the body, then yes, you can take both the egg and the sperm from both the male and female and try to fuse it in the laboratory condition. Always remember, if at all you feel that female and males are not producing gametes also, in that particular case also, you can do IVF using the donor's egg and the sperm. Yeah. Now, this is known as test tube baby. Why? Because it is produced in the laboratory condition. Now, when you get the zygote can be put inside the fallopian tube, because usually the zygote transforms into an embryo within the fallopian tube itself. Okay, so zygote intra-fallopian transfer can be done. It can be a zygote that we are transferring. It can be also an embryo. So zygote, okay, just a second. It can be a zygote or an embryo up to 8 blastomeres. Up to 8 blastomeres is going to be transferred into the fallopian tube, and that is known as ZIFT, zygote intra-fallopian transfer. Now what do you have? You have the next thing, that is IUT, intrauterine transfer. IUT is basically when inside the uterus you are going to transfer. Why? Because the embryo is more than 8 blastomeres. This is very important. If the embryo is more than 8 blastomeres, we are going to transfer it into the uterus directly. That is intrauterine transfer. This is the condition where the female can provide a safe atmosphere for the baby to be born. Now suppose it's the female who is having problem. The male is fine. The female is not able to produce gametes, but she can give suitable environment for the baby to grow. So what do? We take a donor's egg and transfer it into the fallopian tube of the female. Fertilization is going to happen normally itself. Fertilization which is there, it is going to happen normally itself within the body, not outside the body, within the body. Why? Because we are just giving the egg out there, because she is not able to produce the egg. Rudimentary ovaries could be there, the ovaries would not be fine, which is why we are giving the egg, and the fertilization process will be happening within, right? That is GIFT, gamete intra-fallopian transfer. Apart from that, if at all you feel that male is having problem, okay, there is problem in erection, there is problem in ejaculation, there is motility problems. So listen, either if at all the sperms are fine, just that erection is not happening, so probably those sperms can be taken and artificially can be put inside the reproductive tract, that is the vagina, or can be directly put inside the uterus also. You call this as intrauterine insemination, inside the uterus insemination. Otherwise you call this just artificial insemination if at all you're putting in the vagina. Fine. Now the next thing, it can be possible that you take the sperms from another donor. If at all you're doing that, if you're taking the sperms from some other donor out there and you're giving, then in that particular case, again, you can do the same thing only. That is being done when the sperms produced by the man is not of good quality, or probably is not motile, or there is no sperm at all in the semen. In that condition, that can also be done. But these are the techniques that can be really used for treating infertility. Cool? Yes. Next thing, mention the reasons for infertility in the case of humans. What are the reasons? There can be physical reason, as in probably damage in the reproductive organs. There can be congenital reason, as in there might be some genetic problems, as in hormonal imbalance. Okay, you might have heard about PCOS, PCOD. It can cause a lot of trouble because there's a hormonal imbalance in the body. Now there can be disease related causes. For example, you have an STI, which is why that can lead to infertility. You get it? That can also be happening. Now, or it can be drug related. If at all you are taking taking some drugs, it will have some side effects, which is why there can be infertility issues. It can be immunological, as when your body's cells are not able to understand and basically going on killing your own cells. Okay, you know, autoimmune disorders, things like that, right? So immune response against the gametes, that can also happen, or it can be psychological causes also, because of stress and things like that, that can also lead to infertility in the case of humans. So these are the few things that you need to keep in your mind. Very, very, very important. Cool? Cool. Works. Next thing, mention any 2 events that are inhibited by the intake of oral contraceptive pills to prevent pregnancy in human. Okay. Listen, when I say oral contraceptive pills, okay, when I say oral contraceptive pills, when I say hormone releasing IUDs, hormone releasing IUDs, or when I say implants as in you might have seen, you put it under the skin. Hormone releasing IUDs, intrauterine device which is going to release hormone, such as progesterone, levonorgestrel, and things like that, right? LNG 20 you might have heard, right, in hormone releasing IUD. Oral contraceptive pills, and these guys have one thing in common, and that is basically you are going to create an hormonal imbalance, because you are giving progesterone, you are providing progesterone or progesterone estrogen combination. So usually progesterone is super helpful for maintaining endometrium, but now since the balance is not being created, it is not going to maintain the endometrium, rather it will make the endometrium very thin. If it is very thin, do you think implantation can take place? The embryo will come over here and get implanted? No, that is not happening. So first thing, if at all fertilization has taken place, anyways implantation is not going to take place. Now suppose estrogen, you know, is very much important for ovulation to happen. The level of estrogen is not being maintained over here, which is why what is going to happen? Ovulation is also not going to happen. Ovulation is also not going to happen. That means no fertilization also. No egg, that means no fertilization also. That is also being affected. Next thing which is there is that cervical mucus plug is being formed. As in, you know, there is a uterus. In the uterus, my dear children, there is cervix region. In the cervix region, there can be a mucus plug that can be formed. And if at all mucus plug is being formed, the sperms cannot get inside the female uterine tract or female reproductive tract, and that is known as formation of a mucus plug. So these are the few things that can be taking place if at all someone has oral contraceptive pills. Remember the thing about oral contraceptive pills, they are to be taken every single day except 1 to 7 days when the female wants to menstruate. Okay, rest of the days it has to be taken. There's one oral pill which is once in a week pill, and that is Saheli. Okay, it is non-steroidal pill. Please remember about that also. It is non-steroidal, it does not release any hormones. It functions in a very different way. Okay, it blocks the estrogen receptors. So it is not messing around with the hormone release. Basically, estrogen will not find a place to come and bind because it blocks the receptors out there. So it's a once in a week pill with no side effects, and yeah, that is very unique about it, Saheli, and it's not steroidal also. Yeah? Yes. Next thing. What are emergency contraceptives? Right, their hormonal combination. What is emergency contraceptives? Something which you use during emergencies. For example, during rape, which is a very unfortunate thing, or during any other process, someone has sexual intercourse without protection. At this particular point of time, if at all they do not want to conceive, they need to take emergency contraceptives. Something that can prevent pregnancy or prevent conception within 72 hours. You get it? So basically, oral contraceptive pills are pills used to prevent pregnancy after unprotected sexual intercourse or contraceptive failures. They are intended for use within short window, that is within 72 hours after the event. What are those things? Oral pills. Okay, oral pills, hormone releasing IUD, implants, they all come under what you call emergency contraceptives. Why? Because they are the ones they can create an imbalance out there. As I said, okay, fertilization has occurred. It does not allow implantation to happen. You get it? And why within 72 hours? Because the sperm which enters the body can stay in the body for 5 days. For 5 days. That is why 72 hours. You get it? And within this 5 days, any time if ovulation is happening, my dear children, fertilization could also take place. That is why within this particular period, it has to be taken up. Works? Yes. So, administration of progesterone estrogen combination, oral pills or hormone releasing IUD, anything can be used over here as emergency contraceptives. Cool. Next question. Write any 3 principles to be followed by people to be free from sexually transmitted infections. What are the 3 ways that you can be free from sexually transmitted infections? Please avoid multiple partners. Second thing, correct and consistent use of condom, because this is the only contraceptive that can prevent conception, and also STIs. Apart from that, please do prompt medical checkup and treatment and talk to doctors out there. It could also leave you safe. Yeah? And this is something extra, avoiding sharing needles and razors. This is basically because some STIs, for example HIV and hepatitis, also transfers through blood, not just through body fluids but also through blood. In that case, you have to be sure about this particular thing also. But in general, these 3 are the ones that have to be followed. Now, if at all you are asked this question, name 3 diseases, sexually transmitted infections, which cannot be cured, which is non-curable. What will be your answer be? 3 sexually transmitted infections which are non-curable. What is your answer? H. It will be what? HIV. And then what? You have hepatitis B. And you have what? One more thing, that is herpes. Genital herpes. These are the few things that you have to keep in mind. They are non-curable. Yeah? Yeah. Okay. Define venereal disease and mention the modes of transmission of STIs. I already told you venereal disease or STIs or reproductive tract infections are basically infections that are transmitted through sexual contact with the infected person. Some of the diseases such as HIV as well as hepatitis B can be transferred through these things also. Apart from unprotected sexual intercourse, these things also, that is sharing contaminated needles, transmission of infected blood or from the infected mother to the fetus during pregnancy, childbirth or breastfeeding. Okay, please know the symptoms of it: itching, pain, swelling in the genital area. These are the few initial symptoms. Later on, it can lead to pelvic inflammatory disease. It can lead to stillbirth, that means baby is born but dead. Okay, it can lead to ectopic pregnancy, that means pregnancy at different place apart from the uterus. Suppose pregnancy is happening in the fallopian tube. Obviously, it can be dangerous for the mother as well as the child. That is ectopic pregnancy. Okay, apart from that, infertility issues, problems in the menstrual period cycle and things like that. So those also are the problems that will be happening in STIs. Yeah? Yeah. Next thing, what is medical termination of pregnancy? Mention any 2 medical grounds on which pregnancy can be terminated. Okay, what is medical termination of pregnancy? It means that it is induced abortion. You are terminating the pregnancy. It is not happening naturally. You are doing it. It is induced abortion, not a natural form of abortion out there. You have to do it under the approval of medical supervision. Okay. Now, any 2 grounds on the basis of which pregnancy can be terminated. First thing, when it is dangerous for the child. For example, it has child has some genetic abnormalities or some physical abnormalities, serious ones, such that the child will not be able to survive during that particular time, or if at all it is a risk to the mother's life. At this particular point of time, these are the 2 medical grounds on which pregnancy can be terminated. Okay, always remember MTP was legalized in the year 1971. 1971 it was legalized, and if at all you do +1 it gets 20, and here if at all you do ulta it becomes 2017, and that's when Medical Termination of Amendment Act was being put, because people were doing MTP so frequently. Since they got to understand the sex of the child during amniocentesis, through amniocentesis they were doing MTP. So now there is an act being put, and also so many people were acting like doctors. There were unqualified people who were doing this, and so many deaths were being reported, which is why Medical Termination Amendment Act was being put in 2017, according to which if at all someone is doing MTP within the first 3 months, that is within the first 12 weeks, then you need to take permission from at least 1 medical practitioner out there. And if at all you are doing in 24 weeks, that is 6 months, you need to take permission from 2 people. Which one is safe? Definitely this one is safe. Up to what time it can be done? Up to 24 weeks it can be done. But please remember, it is safe only up to 12 weeks. It is safe only up to 12 weeks. Cool? That's all. Perfect. Now, the popular and the effective contraceptives includes IUD. Mention the types of IUD with each example of each. Okay. Any idea? The popular and the effective contraceptives includes IUDs. Yeah? It's very much common. Now, IUD means what? Intrauterine devices. The types of IUD with an example of each. The first one is non-medicated IUD. That means something that is not going to release anything in our body. Basically, Suppose this is the uterus. Within the uterus, you put a loop-like thing. This is known as lipase loop. Why do you put it? You are basically creating a hostile environment inside the uterus. You are creating an environment signaling our body that something foreign has come in, and that is this. Okay, why? Because you want your body to release some immune cells. Immune cells. Now what is going to happen? Suppose sperms get inside the body. The immune cells are going to eat up the sperms. That means they are going to do phagocytosis of the sperm. So it is just to what? Increase the immunological system, and such that the sperms will be undergoing phagocytosis. So non-medicated IUDs, the example is lipase loop. These are simple inner devices that can prevent sperm from reaching the egg by creating a physical barrier in the uterus, by creating a physical barrier and also a hostile environment. Hostile environment leading to phagocytosis. Leading to phago. This term is very important. Phagocytosis. Cool. Now next thing. Copper raising IUDs. Example, copper T. You have CUT, CU7. How does it look? Exactly similar. So over here, you will be having the uterus, right? It looks like a 7 shaped thing, that is copper 7 to which copper wire is being attached, or it can look like a T, again the copper wire will be attached, or it can be multi-load 375 where it looks something like this and there will be more copper being released over here. But what is the use of copper? Copper messes up with the motility of the sperm. It says a sperm, you cannot move anywhere, I'll just hold you over here. So copper is there for messing up with the motility of the sperm, and also it reduces the fertilizing capacity of the sperm. So fertilization is prevented. So basically these IUDs release copper ions in the uterus which are toxic to sperm, reducing their motility and viability, or basically this prevents fertilization. Why? Because fertilizing capacity is also reduced. Fertilizing capacity is also being reduced over here. Now what do you have? You have hormone releasing IUD. Example for that is LNG20 and progestasert. LNG20 and progestasert. Okay, what do they do? I already told you: mucus plug formation, ovulation prevention and implantation prevention. LNG20 is levonorgestrel, progestasert, LNG20, both of them are basically synthetic progesterones out there which you give inside the body to create an imbalance. Done? Yes. Next thing. What is contraception? Briefly explain: 2 natural and 2 surgical methods. So what are 2 natural methods? One is periodic abstinence. What is periodic abstinence? Where basically no one will have sexual intercourse during the fertile period. So when I say there is a window over here, 14-day fertilization, ovulation could happen. That means from 10 to 17 days, you should not be having sexual intercourse. Why? Because it's a fertile period. Any day ovulation can happen. So when someone refrains from doing sexual intercourse during the fertile window, this is known as periodic abstinence. Do you involve any devices over here? No. This is a very natural method that is there. Yeah. Then there is withdrawal method, or you also call this as coitus interruptus. Coitus interruptus means you are interrupting the sexual intercourse. How? By removal of the penis outside the female genital tract. Why? Because you do not want to ejaculate within. So that is known as withdrawal method. So listen to me, this is natural, which is why there are high failure rates. They are not at all reliable. They have 0 side effects, but it is not at all reliable. Apart from this, there is one more thing, that is lactational amenorrhea. That means a mother who is lactating, who is breastfeeding, in her body, the level of a hormone prolactin is going to be very much. Prolactin is going to be very much. So under the influence of prolactin, always remember FSH and LH level is going to be down. So follicles are not going to develop. That means ovulation is also not going to happen, right? So which is what is generally happening. So we think that 6 months, okay, 6 months after the childbirth, when the mother is highly lactating, at this particular point of time, it should be working. Lactational amenorrhea, that means there will be no menstruation. But sometimes it fails. But sometimes it's fail again, it has high failure rate. Cool. Now surgical methods, if at all you're asking about that, there is tubectomy and vasectomy. Tubectomy means what? You're cutting the fallopian tube. Now will the ovum is the ovum released every month? Yes. Ovulation happens. Does menstruation happens? Yes, it happens. But the ovum and the sperm will not be able to meet. Why? Because there's a barrier, there is no rod, there is a gap in between. What happens to the ovum? It gets disintegrated. Okay, that is tubectomy. Now vasectomy is basically when you're cutting the vas deferens. Again, will the sperm be formed? Yes. Spermatogenesis definitely happens, but again it gets disintegrated, won't be there, and the semen is also produced over here. Please remember, semen is also being released, but it will just have the seminal plasma, the secretion of accessory glands. There won't be any sperm. There would not be any single sperm out there. Got it? Yes. So spermatogenesis still happens, ovulation and menstruation still happens over here also. Now what do we have? Reproductive health. This chapter have 5 marker questions, Baba. No, there are no 5 marker questions, only 1, 2 and 3. So it's already been covered in the 1 marker and 2, 3 marker session. Now what do we have? Molecular basis of inheritance, and between that you have principles of inheritance. We'll do it after this chapter. So here also you have some 5 marker questions coming in. Let's see those. Mama. Okay. Children, I hope you guys are understanding. As I said, my energy is a bit low, which is why I'm not able to tell some jokes and things like that, but I hope that you are really getting engaged, you're understanding things well. Anyways, exam time, so you have to pay attention, right? So yeah, that's all. Let's go ahead. DNA is genetic material in both prokaryotes and eukaryotes, yet the packaging process in their cells is not the same. Explain why. DNA is a genetic material in both prokaryotes and eukaryotes, is it? Yes, Baba, in prokaryotes as in bacteria. Okay, the primitive organism. Eukaryotes as in us. In us we have DNA, they also have DNA. But one very important difference that you always need to remember is that in them, the number of the amount of genetic material which is there, it is going to be very less. For example, Escherichia coli 4.6 into 10 to the power 6 base pairs. 4.6 into 10 to the power 6 base pairs. Our case 6.6 into the power 9 base pairs. So we have too much things in here with us. We have less genomic content. That is one very important thing, which is why packaging can be pretty much easier in them, and packaging for us. Listen, why it is difficult for us? Because we have a cell, inside the cell we have nucleus, inside the nucleus this DNA has to be fitted in. And when I say DNA has to be fitted in, our genomic size is more. It is almost 2 meters long. Can I fit this 2 meter long DNA inside the small nucleus? It's very difficult. Which is why it is difficult in us. But in the case of prokaryotes, do they have a nucleus? No. They do not have any nucleus. Their genetic material is going to lie around in the cytoplasm itself. There's a specific region which is there, but it is not membrane bound and things like that. Which is why it does not have to be made too compact. So first thing is that prokaryotes do not have large genomic size. Second thing is that they do not have membrane bound organelles such as nucleus and things like that. Got it? Yes. So DNA is genetic material in both prokaryotes and eukaryotes, but its packaging differs due to difference in genome size and also the cell organization. First point. Now, in prokaryotes, DNA is naked, circular and present in the nucleoid region, the open region which is there, specifically where genetic material is there, you call this as nucleoid region. Do you think they have histone proteins? No. Histone proteins are needed whenever you want to wrap around the DNA such that it becomes compact in nature. So here there is no need of histone. In prokaryotes, no histones are there. And because of small genome and absence of a nucleus, you do not need that. Now in eukaryotes, DNA is associated with histones, okay, and almost 200. So DNA wraps around the histone protein. So 200 base pairs of DNA are being covered in this only. This whole structure which is being formed is known as nucleosome. Okay, DNA wraps around histone octamer. 8 histone proteins will be coming up, and that's how DNA is going to wrap around. This whole structure is known as nucleosome out there. These nucleosomes, they coil further, so you will have multiple nucleosomes. So you might be able to see something like this popping up. Here you have nucleosomes, and basically the DNA will be wrapped around like this. It shows like a bead on a string like structure. There might be beads on a string. Basically it is nothing but the chromatin. The chromatin will later condense to form the chromosome. Everything, the chromatin, chromosome, whatever you're talking about is nothing but the DNA and the proteins. It's just the DNA and the proteins. Always remember that. So yes, these nucleosomes coil further to form the chromatin, which finally condenses to form the chromosome. So the large amount of DNA can be efficiently packed inside the nucleus. That is why both of these things are different. I hope that's clear. Yes, this is your model question paper 1 question. Okay, please know the structure of nucleosome. This is very, very important in 2 and 3 marker. I have explained it. Yeah, fine. Now explain Griffith's transforming principle experiment. How do you explain Griffith's transforming principle experiment? You might be knowing about it. Please remember, I have told Griffith's transforming principle experiment over here. That does not mean you should not be looking for Avery's experiment and Hershey and Chase. Hershey and Chase could be coming in, we never know. So please know about these 3 experiments. Here just Griffith's experiment is being talked about. Okay, so what exactly is being done is that one strain of a bacteria is being taken. What do you call this bacteria as? Streptococcus pneumoniae. It causes pneumonia. Okay, now this Streptococcus pneumoniae which is there, it has two strains, one a rough strain and one smooth strain. The rough strain which is there, it is a plain strain out there. Smooth strain which is there, it has an extra covering around it, which is made up of polysaccharide, as in carbohydrates basically. It is mucus polysaccharide, which makes it or gives it a smooth appearance. Now, this guy, since it has an extra covering around it, it is the demon, or I'll say it is the one that causes death. It is the virulent form, the one that causes death in the organism. If someone is infected with this, it will be non-virulent, which is why those organisms can be easily living. So over here in Griffith's transforming experiment, what was being done, that first thing that he did is he took a live R strain, as in he took an R strain, live one, and he put it inside the mice. I call it as Puchu. My students might know it. So inside Puchu, when it was put, Puchu lived, my dear children. Why? Because R strain is non-virulent, does not cause any problem. Okay. Then S strain was taken and then it was put inside the mouse. What happened? Puchu died. Why? Because it was the virulent form. Okay. Then what did he do? He treated S strain with heat. He treated S strain with heat, and obviously that will be bringing some changes. And then he put inside Puchu. Puchu was so happy. Puchu didn't die. Okay, it didn't die. Okay. So then Griffith was like, okay, when I put live R strain, it didn't die. When I put heat killed S strain, then also it didn't die. Why not I put a combo of it? Let's see what happens then. He put that, and to his surprise, he found that Puchu died. And one more thing was noticed. He put live R strain and heat killed S strain. But inside Puchu, he found live S strain. Did he ever put live S strain? No. So basically something might have been coming from here into the R strain and might be transforming this live R strain into what? S strain inside the organism. So there is something that is getting transferred, and that something which was getting transferred could be the genetic material, because when that thing was there, it is giving information to produce something that will lead to the death of the organism. So this thing that was getting transferred was considered to be the genetic material, but he didn't know whether it was DNA, RNA, protein that was genetic material, which is why Avery, MacLeod, McCarthy came into the picture. They proved it partially, okay, but then people didn't believe it. Then Hershey and Chase came with their experiment on bacteriophage and E. coli, and then they proved it. Please know, these two experiments are equally important. Please go through it. If you have any doubts, ask me. I'll make a separate video also. I do not mind it. So please do that. Okay. So this is what you have to write. The live R strain bacteria were transformed into virulent S strain. Griffith concluded some heritable material was transferred from S strain to R strain. So please know about that. Works? Works. Fine. My nose. Okay. Now state the arrangement of different genes of lactose operon in E. coli, lac-operon, and describe the role of lactose in regulation of lac-operon. Now this is also super simple. Listen to me. So first thing, so I'll explain with this diagram. That will save a lot of your time. So there is a gene over here which you call as inhibitory gene. What do you call this? Inhibitory gene. This gene is a regulatory gene. Okay, so this position you have a regulatory gene. It regulates. It can regulate positively, negatively. But over here, since it is going to regulate negatively, I'll call it as inhibitory gene. This inhibitory gene has a promoter for itself. Onto this promoter, when RNA polymerase will come and bind, what is going to happen? Transcription will be happening, and whatever mRNA will be found has to be found. And here after translation, you get the protein. What is this protein? Repressor protein. What protein? Repressor protein. Okay, done. Until here clear. Now you have another set of gene over here. For that you have a promoter region, and you have one more region which is known as operator. O for operator. This type of setup is usually found in prokaryotes. Okay. And over here you have multiple genes. Z, Y, A. I remember it as Zyada. Zyada means more. So Z, Y, A, that is the flow of things. Z basically, which is there, it codes for an enzyme that is known as beta galactosidase. Ma'am, what does this enzyme do? So E. coli, suppose E. coli needs some energy. What do we do when we need some energy? We drink some juice and things like that. We get carbs, right? glucose basically. Similarly, when this organism E. coli needs energy, it has to take either glucose or basically something that could give glucose. So over here, suppose it has lactose in the medium where it is growing. It takes up the lactose and breaks down the lactose with the help of beta-galactosidase into what? Glucose and galactose. So lactose is made up of glucose and galactose. So when I break down lactose, I get glucose. You get it? So that is the use of beta-galactosidase. Now, first of all, lactose is outside in the medium. It has to be taken inside. For that, you have to increase the permeability of the cell, which is why you have permease enzyme. When this enzyme is produced, the permeability increases and you have what? More lactose. Now, there is one more enzyme that is known as transacetylase. It's not this, it is transacetylase. Transacetylase deals with everything toxic that comes in between. Obviously, when you are doing so many things, there are some toxic buildup happening up there. So, deal with that. So, we have transacetylase. Okay, these are the three genes which is there, Z, Y, A. What is this? Beta-galactosidase. What is this? Permease. And what is this? This is transacetylase. Okay, now, suppose I am growing the organism, that is E. coli, in a medium that does not have lactose. Obviously, do you need all these enzymes as of now? No. So, do you think transcription should be happening? No. Which is exactly why the repressor protein over here will be going and binding on the operator. Now suppose this is my hand and this is RNA polymerase. RNA polymerase will come bind over here to do the transcription. But do you think it will be able to do the transcription? Why? Because this guy is up over here. The operator is up over here. Sorry, repressor is on the operator, which is why there is no possibility of transcription to be done. Which is why all these things are not going to be formed. In this condition, I will say it is switched off, lack operon. This is lack operon, and it is what? In switched off condition. What type of regulation is this? Negative regulation. This is negatively regulating it, right? So this is negative regulation that is happening up over here. Now suppose I have changed the medium and the medium has lactose in it. What is going to happen? The lactose slowly will be getting inside the cell. Once the lactose gets inside the cell, it says to the repressor protein, hey macha, don't go and bind there, da baba. You come here, you bind with me, because they both are best friends. So always remember, the repressor protein which should be binding up there, it's now free, it's not binding there. Rather, the lactose protein is coming, sorry, not lactose protein, lactose is not a protein, lactose is a carb. Okay, the lactose which is there, that is binding up with the repressor out there. In this condition, what is going to happen? Transcription will smoothly happen because over here there is no blockage. Which is why I'll say that lactose which has just got in, it is what? An inducer. It is what? An inducer. It is helping in transcription to happen, right? So lactose is an inducer. Always remember that. Apart from lactose, you have different form of lactose. There is allolactose that can also act like an inducer. Please remember that. Okay, done. So this is an inducer. Now what happens? Beta-galactosidase is being formed, permease is being formed, transacetylase is formed. When permease is being formed, it increases the permeability, more and more lactose will be coming in. But ma'am, there's a question, how did the first lactose come in? So it's when I say that it's switched off condition, it's not completely switched off. Little amount of permease will be there. Okay, little amount of transcription will be happening. It's a general thing. In your NCERT, it might not be written, but please know that. Okay, so please know all about this. Please know the flow of this. Very, very important. Okay, Piposia, whatever you want to remember, you can remember. Piposia. Okay, very, very important. Then this is when I say it's switched on condition. And remember, in your NCERT, it is written lack operon shows negative regulation. There is positive regulation also, but it is beyond as to what is to be taught over here, which is why it is not. Is what NCERT also says. Do not worry. Okay, it's not just me, it's also NCERT. Works, ma'am. Perfect. So that has been mentioned over here. Write your answers properly. I've given you all the answers, how to frame your answers. Just know about it. Okay. Now, another 5-marker question, that is, given below are sequence of nucleotides in a particular mRNA and amino acids coded by it. Okay. 5 AUG, UUU, UUC, GAG, UUA, GUG, UAA. And methionine, phenylalanine. Okay, so AUG codes for methionine, UUU phenylalanine, UUC also phenylalanine. So you are being asked what are the properties of genetic code that can be correlated from the given data. So what are the properties that you can understand from this? First thing, AUG which is there, it is what? Going to be an initiator codon. it is the one that initiates the process of translation such that amino acids are being coming up. And apart from being initiated codon, it also codes for an amino acid that is known as methionine. So can I say AUG, which is there, it has what? It has a dual function. It has a dual function. This is the first thing that I can write. Now, if I see UUC, two codons are there coding for the same amino acid. What do I call this particular feature? Multiple codons coding for same amino acid. This feature is known as degenerate feature. This is known as degenerate feature when multiple codons code for the same amino acid. Now, do you see any full stop, commas, anything up there over here? No. So can I say that it is read in a contiguous fashion, as in a continuous fashion? That is what I mean by contiguous fashion. Okay. Now the fourth thing that is up there. What is the fourth thing? Any idea about the fourth thing? If you see, each codon codes for only one amino acid, right? GAG is not coding for anything else, just glucine. Oh, sorry, glutamine, not glucine, glutamine. Okay, or basically over here, just a second, okay. It is glutamic acid. Okay, now we're coming to this. UUA is also coding for leucine. So it goes like that. What do you call this feature when an amino acid is when a codon is coding for a single amino acid? This is known as unambiguous nature of codons. Unambiguous nature of codon. What do you mean by that? One codon codes for only one amino acid. Listen, multiple codons can code for one amino acid, but one codon is definitely coding for one amino acid. That is known as unambiguous nature of the codon. Now what feature do you have? If at all you see this, UAA is not coding for anything. So what is this codon? This is basically, and after this there is no amino acid also being formed. So can I say UAA is a stop codon? Yes, I can say that. UAA is what? A stop codon. It does not allow translation to further go up beyond that. Okay, so it's a stop codon. Apart from UAA, do we have another codons are there which are stop codons? Yes. So UA, UGA and UAG. Okay, UA, UGA and UAG. These are the few things that are being formed. Always remember that. Okay, so, and one more very important thing, if at all you see all the codons which are there, they are what? Triplet in nature. They are triplet codons. They come in a number of 3 groups. Okay, so the codon is triplet. 61 out. We have 64 codons, by the way. After that, 61 of them are going to code for amino acid. 3 of them, UAA, UGA and UAG are stop codons. So know that. AUG has dual function, know that. Some amino acids are coded by more than one codon, hence it is degenerate, correct. The codon is read in mRNA in contiguous fashion, correct. UAA, UAG and UGA terminator codons, correct. Now, it is not given over here, but still I am telling you. The codons which are there, they are going to code for the same amino acids all throughout all different type of organisms out there. This is known as universal feature of the codons. If at all you get a question that this codon is coding this amino acid in bacteria and the same amino acid in human beings, what feature does it show? It shows universality of this particular codons. So they are universal. Exceptions are always there. Exception is what? The mitochondrial codons and some protozoans. So in mitochondria also you have codons. There you see a different whole thing happening. Similarly in protozoans also you see a different thing happening. Okay? Okay. Perfect. Okay. Next thing that we have is a police squad from the crime department approached the forensic science department to help identify the culprit. DNA samples were collected from the crime site and of the suspects. Explain the crime site and of the 2 suspects out there. Explain the process involved if the culprit has a monozygotic twin. Can this method give results? Okay, first of all, let's go line by line. Explain the process involved. If at all there is something happening, suppose there is a crime that has taken place over there. I am the criminal. I did the crime. You are also there. I said that you did the crime. I said that you did the crime. Let me name you. Your name is Raghav. Okay, your name is Raghav. Any Raghavs up over here? Please let me know. Okay. So over here, me and Raghav are the suspects. Okay. And from the crime site, basically from the crime site we got some blood. This was being taken. This was being taken and it was assumed that it is of what? The murderer. That is me. But as of now, no one's know that. So basically, the DNA was being checked out. Okay, and they made a complementary strand of it. What did they do? They made a complementary strand of it and they added radioactive thing over here. This complementary strand is known as what? A probe. It is known as a probe. This was already been done and is kept there. Now, you have a doubt between, the polls had a doubt between me and Raghav. So basically, first of all, our DNA was isolated. So I'll write isolation of DNA. That was being done. After isolation of our DNA, using restriction enzymes, our DNA was being cut. Using restriction enzymes, the DNA was being cut. The same restriction enzyme was used over here also, if at all cutting was there. Done. Now after that, using gel electrophoresis, our DNA strands were separated. Okay, so now I'm going to make the scenario. It looks like this. Suppose this is me and this is Raghav. This is the DNA that we have, the DNA bands that are being formed, okay? Cool. Similarly, this DNA from the murder site also, from here also, I took the DNA, I cut it with the same restriction enzyme, I made them separate. Okay, listen, this and this exactly matches, but as of now, I cannot prove it. Listen to me carefully. What has to be done next? So gel electrophoresis was been done. After that, you have to transfer it onto a nitrocellulose sheet. You have to transfer it onto a sheet. So this is nitrocellulose sheet. That is known as southern blotting. You call this as southern blotting. After you transfer it up, what do? You basically put the radioactive probes. This probe which was there, it is being put over there. Radioactive probe. And this process is known as hybridization. Why? Because you are putting a probe and it will bind to the complementary base pairs out there. So what exactly happens is that, listen to me carefully. The probe goes and binds up over here. The probe goes and binds up over here. It binds up over here and it will also bind up over here. You might ask why? Listen, we all have some repetitive sequences. There are some sequences which repeats in our body. In you, it might be repeating in different number of times. In me, it might be repeating different number of times. Does not mean that we do not have any sequence which is not similar. We will have it. But over here, if at all you see that, if I have a quick comparison between this and this, it is exactly similar. Both of them looks exactly similar, the bands. Here it is different. With that, what I get to understand is that probably the murderer is jasmine itself. Murderer is what? Jasmine itself. But again, you put the radioactive probes. After that, you need to do one more thing. That is, you need to view it under an autoradiography machine or UV light thing. So please know that also. Autoradiography thing or UV light. So DNA fingerprinting works on that. That is, we have unique DNA out there. Okay, we have some unique DNA out there which makes us all different. And basically with the help of that we can find out the culprits. We can find out if at all there is who is the father, who is the mother and things like that as well. So please know about that. And for this particular process, you will require things such as blood, hair follicle, skin, bone, saliva or sperm, etc. These are the specimens that are generally being used. And also remember, in the blood, RBC is of no use. In blood is made up of multiple cells, right? Red blood cells are of no use because red blood cells does not have nucleus. That means no DNA as well. Please remember that. So process involved is DNA fingerprinting. I hope you know that. Here what do? Isolation of the DNA. Then digestion by restriction enzymes, you cut it. Separation by gel electrophoresis. Transfer to the membrane or southern blotting. Hybridization with the DNA probes. And visualization. So how do you get to know that this probe is there? Basically, you made it from the DNA that you got from the site, right? That is why it is going and matching up there. So yes, got it. I hope you have got it. Each step you have to write. There is one more part of this question. If the culprit had a monozygotic twin, what do you mean by monozygotic twin? as in the ones that look similar, very similar, right? So basically when they look similar, that means inside that the DNA is also very much similar. Why? Because there was a zygote, this particular zygote split into two and they develop into two embryos. So whatever genetic material was here, it's the same over here, same over here also. So in this particular case, do you think DNA fingerprinting will be of any use? No, because both the twins will be having same type of genetic material. It will be very difficult to find out. So if the culprit had a monozygotic twin, no, sorry, no. Why? Because DNA fingerprinting cannot distinguish between monozygotic twins. This is because monozygotic twins have identical DNA sequences. Hence, their DNA fingerprints are exactly the same. There is one more type of twin where basically two zygotes. So basically in that particular case, you might have seen those people who do not look similar, right? So in that particular case, ovum was released from both the ovary. Okay, sperm came and fused both the ovaries, two sperms, separate here, separate here. Okay, now you have got one zygote here, one zygote here. So that is individual zygotes that are there. Over here, only one zygote. In monozygote twin case, only one zygote is there. This zygote itself divides into two. You get it? So that is the difference that is there. So please know the difference and answer accordingly. Okay. Now, I hope this type of questions are helping you well. You feel that, okay, I'm practicing some new type of questions as well. Yeah? Do let me know. I'll be really happy if at all I get to know that it's helping you. Now, answer the following. Construct a complete transcription unit with promoter and terminator on the basis of the hypothetical template strand. So, what is given over here? This is going to be the template strand. Please read your question really well. Now, write the RNA strand transcribed from the above transcription unit along with the polarity. Okay. So first thing that you have to do is make a template strand. So make a transcription unit. So just realize whenever you have arrow on this side, it means that it is a 3'N. Okay. Okay. So where was I? Yeah. So sorry. As I said, I'm not feeling well. Yeah. So this is 5'N and this is 3'N. So wherever you have arrow, that is 3'N. This is going to be the template strand, right? Now, I'm just making a rough diagram. Do not judge me, okay? 3'N, 5'N. If this is my template strand, I would have to make a coding strand as well. That will be in the opposite polarity. So this will be 5' and that will be 3'. This is going to be what? My coding strand. Now listen. At the 5' under the coding strand, you have a sequence that is known as promoter region, onto which RNA polymerase is coming and binding and doing the process of transcription. Here, onto the 3' end of the coding strand, you have the terminators region. In between, you have to fit all this in. So, in between, in the 3' end, what do you have? A, T, G, C, and the list goes on. How do you make a complementary strand? A base pairs with what, Baba? A base pairs with T, T base pairs with A, G base pairs with C, and you have to make a strand like that. That is going to be a coding strand. This is how your transcription unit is going to look. Make sure you make the polarity really well. This is super important. And read the question. Here it is given, hypothetical template strand is given to you. If hypothetical coding strand is given to you, thing will be different. What will be your answer if at all coding strand is being mentioned over here? Think for that as well. Okay, just know the scenarios. Now, this is the transcription unit. What do you have to make? You have to make an RNA thing that is transcribed. So basically, which one is going to undergo transcription? Template strand is the one that undergoes transcription, right? So you have to make a complementary RNA strand. Do you make it again from this strand? Listen, you just have to replace wherever T is there, you have to replace U, because you are making an RNA and it will be complementary RNA, right? So wherever T is there, you will have U out there in the new RNA strand. So this is how it is going to look. Do not be worried. If at all you're confused, come on, Baba. Yeah, this is a promoter, that is a terminator and everything. Here you have the new RNA strand being made. So if at all you see this, it was TAC here. You had UAC. So wherever T is there, you're replacing it by U. That is the only difference out there. And when you make a new RNA strand, it has to be from 5' to 3' direction. And you will always use the template strand for that. Coding strand is not going to get transcribed. Remember it. Else works. Perfect. Now list the salient features of human genome. This is being asked usually. So please know the salient features of human genome. The human genome contains how many base pairs? 3,164.7 million base pairs out there. 3,164.7 million base pairs. The average gene size consists of what? 3,000. A gene, if at all you see, it is having 3,000 base pairs. But there is an exception, and that is dystrophin. It has 2.4 million base pairs. Everything is in million over here. Now, total number of genes is what? Estimated to be 30,000. How was the previous estimate? Previous estimate is what? 80,000 to 1,40,000, but it is not. It's just 30,000. Now, the functions are unknown for over 50% of the discovered genes. For 50%, okay, for over 50% of the discovered genes, we do not know their functions. Their functions is not known. Less than 2% of the genome codes for protein. When I say, okay, I have discovered many genes. See, 30,000 genes I have discovered. For 50% of them, I do not know what is their function. And not just that, when I talk about the whole genome, less than 2%, okay, it's not more than, please remember it is less than, these terms are important. Less than 2% of gene actually codes for what? Protein. Know that. Now, repeated sequence make up very large portions of the human genome. That is why we say less than this codes for protein, rest everything is what? Could be repeated sequences. What are these repeated sequences? They are stretches of DNA that are repeated many times, 100 to thousands. They thought to have no direct functions, but they definitely helps us in understanding the evolution pattern which is there. Okay, they helps us in understanding evolution pattern. Now, so over here, okay, so they do not have any direct coding function, but they shed light on chromosome structure, dynamics and evolution. Chromosome structure, dynamics and evolution. Now, one more point is there. Chromosome number 1 has the most number of genes. What is the most number of genes? 2968. So over here only 7 is being mentioned. 2968. Here do not see 7, just everything is there. Okay. Chromosome number 1 has the highest number of genes. That is the first one to be sequenced also. And chromosome number Y, that is found in the case of males, XY, Y gene. Okay, sorry, Y chromosome which is there. It has the least number of genes, and that is 231. Do not go and write 213. It is 231. So here it is 2968. There it is going to be 231. Very, very important. Now, scientists have identified 1.4 million locations where single base DNA difference is there. That is single nucleotide polymorphism is what you say. Is that. Okay, so over here 1.4 million locations is where you find it. These are the very important things that you need to know. This is the salient features of human genome. Works? Works. Next thing. Explain the process of translation in protein synthesis. So, what is translation, Baba? By the way, let me just see how many questions are left. So, are we soon going to finish it up? Yes. Okay. Explain the process of translation in the protein synthesis. How do we explain that? First of all, for translation to happen, what do you need? You need an mRNA. mRNA is in which direction? 5' to 3' direction. Okay. Now, what do you need? mRNA which is there, it will be having codons. AUG and the list goes on. You need anticodons. From where do you get that? From a tRNA. So you need tRNA also. Okay, tRNA has an anticodon loop, and basically it will be having amino acid attached to it. Fine. And what do you need? You will need, apart from this, you will need ribosome. Because ribosome provides the site where tRNA will be coming and binding. So it looks like this. Your mRNA is between two ribosomal units. The tRNA will be coming and binding in the specific sites of these units and helping in the whole process to happen. The first thing that needs to be done over here is this. Is known as charging of tRNA. What do you mean by that? It means that tRNA has a 5' end and a 3' end. Okay, it's a RNAs which is a 5' end and 3' end. At the 3' end, the amino acid will be coming and binding. This process is known as charging of tRNA. It is also known as amino acylation. What do you call it? It is known as charging of tRNA, or it is also known as amino acylation. This is one very, very, very important thing that you need to remember. Now what? Now there is another thing that is taking place over here. There is anticodon which has also been prepared. Please remember the secondary structure of tRNA. We thought it is like a clover leaf model, but actually it is not. Okay, it is an inverted L. It looks like an inverted L. And also tRNA is known as soluble RNA, which is why sRNA is also what you call it. Fine. So this charging of tRNA amino acylation requires a lot of ATP. Remember that also. So this has already been prepared. Every tRNA which is there, it has an amino acid joined to it. Charging of tRNA, this has to occur in the first place, in the first phase. This is not first step, I say it's the first phase. In the first phase of translation, this has to be already done and kept. Now what? So now we have the mRNA. Onto this mRNA, one smaller ribosomal subunit comes and attaches. Small ribosomal subunit. Then you have the AUG sequence, initiative codon sequence. Over here, the tRNA will come and attach. Then what? Then the next ribosome, the larger ribosome will also come and attach over here. Larger subunit of the ribosome comes and attaches. And now we say that the initiator complex have been found. The ribosome which is there, always remember it is made up of rRNA, ribosomal RNA. Apart from that, there are 80 proteins which are being involved. 80 proteins are also going to make up this ribosomes are there. So first the small ribosome attached to the mRNA, then the tRNA, then basically the larger subunit attached. Now we say that initiator complex is being set up. So we'll read until this point so that you have an understanding. First is activation or charging of tRNA. Amino acylation of tRNA is the process of attaching an amino acid to the corresponding tRNA. This is known as charging of tRNA. It occurs in the first phase. Next thing, initiation. This is the first phase. Now we are going to begin up. So in initiation, mRNA binds to the smaller ribosome subunit. Okay. The start codon is recognized by the initiated tRNA, the first tRNA out there, carrying what? Methionine. The larger ribosomal subunit assembles, forming the initiation complex. Done. Now what happens, my dear children? You have formed the ribosome. Slowly, the ribosome is going to move. So until it had, see, each tRNA has an amino acid, right? Suppose a new tRNA is coming. What is going to happen? These two amino acids which are there, they are going to join, and this bond is going to break, which means that this tRNA is free to go, and you have a new chain of amino acid being formed. So same thing is going to be repeated again and again until a terminator RNA, that is basically your UAA, the stop codons, anything is being reached. Until that particular point, this will be going in. But you should know that what is that something that will be helping in the formation of bonds between these amino acids, right? So that is the next thing that we are going to see. So elongation. tRNA brings amino acid to the ribosome, matching codons on the mRNA through its anticodon. Okay. Peptide bond, that is the bond between two amino acid. Peptide bond forms between the adjacent amino acid, catalyzed by the enzyme. Okay, you do not have to write the name of the enzyme, just know it is catalyzed by the enzymes out there. Now, the ribosome also acts like a catalyst. The ribosome as a whole, it also acts like a catalyst. Why, my dear children? Because it has, as I told you, it is made up of rRNA, and there is one type of rRNA which is called as 23S rRNA. 23S rRNA. This is there in the bacteria. It is basically acting like an enzyme which you call as ribozyme. So this particular thing also helps in the formation of peptide bond in the case of what? In the case of bacteria, the rRNA. Okay, for the formation of peptide bond. So generally peptide bond is formed by the enzymes which are present. You know, multiple proteins are present in the ribosomes. So yeah, out of that anything can help in the formation of peptide bond formation. In the case of bacteria, you have 23S rRNA, which you also call as ribozyme, helps in the formation of peptide bonds. Now, ribosome moves along the mRNA in 5' to 3' direction. In which direction does it move? From 5' to 3' direction it is going to move. Please have an understanding of that also. Got it? Perfect. Now what? Termination. When a stop codon, that is UAA, UAG, UGA is reached, at that particular point of time, no tRNA will come and bind over there. Release factors might come and bind, but no tRNA comes and binds over there, and that's exactly how you got a polypeptide. Got it? Yes. That's all. Those are the things that you need to remember. Now, one more thing which is there is that this is an mRNA. Before the 5'N, before the 5'N, and after the stop codon, before the initiator codon, after the stop codon, or after the 3'N, you have some regions which are known as UTR, untranslated regions. They are not going to undergo translation, but they are super important. Why? For the stability of the mRNA, for the initiation process to happen very smoothly, for everything, for the process of termination to happen smoothly, you need UTR. Untranslated regions, they are not going to get translated. Works? Works. Perfect. Now, Question number 9, which is DNA replication is said to be semi-conservative. Why? Describe the experimental proof of measles and install to show DNA replication is semi-conservative. First of all, measles and install did their experiment on which organism? E. coli. Okay, it was E. coli. So E. coli, my dear children, was grown in a medium which had N15. So over here, the two actors which are there, one is N15 and one is N14. Nitrogen basically is what? Is a part of our DNA. Yeah, nitrogen is there in our DNA. So nitrogen has been taken up. If at all I grow an E. coli in a medium which is having nitrogen, that nitrogen will be taken up for the formation of DNA. Okay. So there was a medium whereas NH4Cl is used where the N can be N15 or N14. And over here, what do I do is, I have a logic. The logic goes like this. What I am trying to prove is that if I say this is parental DNA, the parental DNA is going to separate out and a new strand is going to be formed for sure. But will the old strand be also there? Yes. Only half of the old strand is there, which is why I call this as semi-conservative mode of replication. And I am trying to prove this. Not me, actually, Michelson and Stahl tried to prove this. How did they prove this? So basically, they grew the E. coli in a medium that was containing N15. Now, N15 will be taken up for forming the DNA, and N15 isotope is a heavy version of isotope. So what is going to happen is that when I put this DNA in cesium chloride medium, in a medium that is cesium chloride, CaCl medium, the DNA is going to come somewhere over here. They move down, they segregate some over here. Why? Because they are heavy. They are very heavy. That is why they move over here. So I get a heavy chain. Okay. Now basically I change the medium. I grow the organism in N14 medium. What is going to happen? And every E. coli is going to divide in every 20 minutes. So after 20 minutes, I saw that there was no lower band with heavy band. There was actually a hybrid band. How? Listen to me. So these two strands which are there, red in color, they separated. The new strands formed up. The new strand was N14. The older strand was what? N15. Which is why now it's not like this. Rather it's like this. This is non-parental type. Do they look like the parental type? No, they look different. And similarly, their heaviness is also different. Over here, they're somewhere in the middle. So I got an understanding, but still I want to see once more. Again, I let the E. coli divide. So 20 more minutes. So 20 plus 20, I have 40 minutes now. Every cycle is of 20 minutes. What did I find? These two strands are going to separate one green strand, one red strand. Similarly, one red and one green. New strands are going to be formed. But what strand? N14 only, because I'm still growing in N14 medium. Now what you will find? You will be finding a heavy chain, a hybrid chain. Apart from that, you will be finding a light one. So N15 was a light one, N14 was a light one, which is why you will be finding a light chain also. So now they do they have any parental thing? If at all I compare with this, yes. But if at all I compare with this, no. In the last generation, I have nothing that resembles like the parent. Okay. Is the density different? Density is also different over here. So please know that. So with this conclusion, with this understanding, got to know that, yes, it is. Since he found hybrid variety every single time, he got an understanding that, yes, parental, DNA is going to be there. One strand, only one strand of the parental DNA is at least going to be there. Otherwise, you will not find hybrid, no. You'll only find the new ones and only the light ones. You have the hybrid for a reason. Got it? Yes. Now, similar type of experiment was done by Taylor and his colleagues, but they did not use nitrogen, rather they used radioactive thymidine. Please know that. They used radioactive thymidine over here, and just a second. Yeah. Work. Come on. Okay, there's some problem. I'll fix it. So radioactive thymidine was used, and the plant which was used was faba beans. You also call it as Vishya faba. Please know about that also. Okay, generic thing. Fine. Now you have the next question out there, and I think it's time to fix the... Okay, next question, it went. Okay, and it goes like this. Name the process involved in the following. So you have to tell the process name which is involved in the following. One parental DNA strand is conserved by the daughter cell. Semi-conservative mode of replication, right? Only one strand is there, other strand is new. Separation of DNA fragments. With what process did you do separation of DNA fragments? Gel electrophoresis. Yes, with that process you do separation of DNA. Now, removal of introns in the hnRNA. hnRNA means what? When I say transcription is happening, DNA transcribes into hnRNA. So whatever AMAP SHANAP is there in the DNA, it's transcribed over here. So there might be coding regions, exons, and there might be non-coding regions, introns. Everything will be there here also. But when you say I am going to form an mRNA which will undergo translation, I have to make sure there are no introns. So if introns were over here, I have to remove these introns with the help of splicing. This process is known as splicing. Now, attachment of Rho factor in to RNA polymerase during transcription. What do you call that? So listen, transcription, you have terminator and a promoter region, right? So in bacteria, for attaching to the promoter RNA and for starting the process of initiation, you need a factor that is known as sigma factor. Similarly, for termination, you need another factor that is known as Rho factor. And you call this process as Rho dependent termination. Okay, Rho dependent termination. That is what you call this process. Now, polymerization of amino acids to form polypeptide on mRNA. On mRNA, when you form a polypeptide, what do you call this as? You call this as translation. So this is exactly written over here. This was the question, so I thought I should be putting it over here so that you can also get a practice. Okay, so please remember, go through each and everything, very important. Okay, now what do we have? Okay, other important concepts which are there, which you should not be missing. As I said, Hershey and Chase, different type of RNA polymerase, there are 3 of them. Please know about that. I have explained previously. And genetic content in different organism. Again, in 1 marker, you will be able to see this. In 2 marker, you will be able to see this. In 1 marker, you will be able to see this. Hershey and Chase experiment, I have done in masterstroke. But yes, again, if you need some help regarding that, I can help. Okay, do let me know about that. I'll do help you. Yeah? In a separate video, I can do that. Now we go ahead to the next chapter, that is molecular basis of inheritance. So my dear children, before I go ahead with that, I always want to tell you one very important thing. It's not like any teacher who is in YouTube, who is in school, anyone who says that this particular questions are something that is going to come, you need to understand that they mean that the concepts could be repeated, the questions will not. So your concepts have to be strong. Don't think that just by relying on questions, your whole thing is done. No. You have to make sure your concepts are strong. We have recorded masterstroke videos. Watch it if at all you need. Make your concepts super clear. Then go ahead with question practice. This is important. If at all you're doing question practice, do that. But along with that, your concept clarity is also important. Okay? Yes. Now you have molecular basis of inheritance, and you only get 1 question from this chapter. 1 MCQ, which is why I'm not taking much, but yes. Okay, let's see. So another chemical name for the nitrogenous base thymine is already also known as what? So listen to me. In the case of DNA, you have thymine as a pyrimidine base. Okay, so in DNA, what do you have? You have A, G and T, C. Take care. A, G, take care. Okay, so you can remember like that. But in the case of RNA, you do not have thymine, you had uracil. RNA came first or DNA came first? It was definitely RNA that came first. Later on, some modification happened. That's how you got the DNA. So uracil was there firstly. Then uracil got modified into something that is known as thymine now. How modification took place? At the 5th position, a methyl group was added, and that is why it is known as 5-methyl uracil. Thymine is also known as 5-methyl uracil for the same reason. In the 5th position, a methyl group is being added. Got it? Yes. Next thing, if an mRNA 3-AUG undergoes translation, how many types of amino acids are going to be formed? If an mRNA, which is like this, undergoes translation, how many types of amino acids are going to be formed, are going to be present? That is a question. Now usually the mistake that we all do is AUG, okay, methionine, then UU phenylalanine. So 1, then 2, this all thing is same only, so I will not count. And UAA is a stop codon, so basically that is not going to code for anything. So two types, okay, 2 types of amino acids are going to be formed. Is it 2 types of amino acid? That is there in the option, but that is not the answer. How many types of amino acids are going to be formed over here? Please understand, see the polarity, it is 3' and 5'. Translation happens from 5' to 3'. So you have to read in this particular frame. So it is AAU, 1 amino acid, UUU, another type of amino acid. You just give me a second. Another type of amino acid. This is UU, same type of amino acid. Similarly, UUU, same type of amino acid. So I'll say phenylalanine is the next type, and then GUA is the third type. So you get 3 types of amino acids being formed over here. You get it? So B is your answer for this question. Okay, check out properly, then only go ahead. Otherwise, you will lose your marks, baba. Okay. Next, match the following: Alec Jeffrey, F. Sanger, Jacob and Monod, and Griffith. Always in match the following question, read all the options first, then only go ahead with matching of the options. Okay, and always try to match A and D first. It will be easy for you then. Alec Jeffrey was the person who went ahead and gave DNA fingerprinting method. Jeffrey has double F over here, that's how I remember that. You can remember it other ways also. Griffith, my dear children, Griffith was a famous guy for doing the transforming principle experiment. Not transferring, it is transforming principle experiment. So A goes with 4. A goes with 4. I have 2 options. Let me cut out these 2 options. And D goes with 2. A goes with 4 and D goes with 2. Here I've got my answer. But you have to be very sure your A and D options are correct. Now F. Sanger is responsible for annotated DNA sequencing. You remember whole DNA was being sequenced with the help of this technique. So that is F. Sanger who gave up this particular technique. Jacob and Monod, both of them has O. O stands for lac operon. Jacob and Monod basically with the people behind lac operon. Okay. So what is your answer? Your answer is going to be B. Next question. GGU, GGC, GGA, GGG, all code for glycine. All the 4 codons are coding for glycine. Which salient feature of this genetic code does it prove? Which feature does it prove, my dear children, of the genetic code? You have so many features of genetic code, right? There's a feature that is known as degenerate feature, where multiple codons can code for the same amino acid. So here, these are the multiple codons and they are coding for the same amino acid that is known as glycine. So this feature is known as degenerate feature, or you call this as degenerate codons. But always remember, one codon codes for only one amino acid. GGU, whatever you do will be coding for only glycine. That is known as unambiguous nature. This, when you talk about multiple codons coding for the same amino acid, you say it is degenerate nature. Done. Okay, cool. Next go. In a single-stranded DNA, if the ratio of A and G is 60 and 40, then the ratio of T and C. Whenever you have this type of question, you go like this: A and G and T and C. If A and G is 60-40, then what is going to be T and C? T and C is also going to be 60-40, because how many number of A is there, that many number of T will be there. How many of G is there, that many number of C is going to be there. That is how it goes. Is it? Remember, read the question carefully. In a single-stranded DNA, A can be multiple times, and so does T. T can be also multiple times. Here, A and T ratio is not relatable. You get it? How many numbers of A are there? But that we cannot say how many number of T is there. You get it? Because here you're talking about single-stranded DNA. So none of the above is going to be the answer for this particular question. Got it? Very, very, very important. Fine? Yes. Now, the next one. The components of deoxyguanosine is there. So, if at all you have studied my master stroke series, you will get to understand the answer for this question very, very easily. I've told. Deoxyguanosine. Deoxy means here which sugar is involved over here? Deoxyribose sugar is involved. Okay? Deoxyribose sugar, that means it's a DNA. Cool. Now, over here when I say guanosine, deoxyguanosine, I do not say deoxyguanosine triphosphate. If I would have told that, then phosphate group would have been included. But here, there is no term phosphate included. That means it is just the ribose plus the nitrogenous base, which is what? Guanine. Which is guanine. When both of them come together, the deoxyribose as well as guanine comes together, I will call this as deoxyguanosine. Okay, deoxyguanosine. Fine, so your answer for this question is going to be this. Why not this? Because here it is written phosphate, here also it is phosphate, and here it is ribose sugar, here it is deoxy mentioned, right? So it cannot be ribose sugar for sure. Got it? Okay. If at all it was plain guanosine, then what would have been your answer? If at all it was plain guanosine, what would have been your answer? then the component will be guanine and ribose. Because for ribose, you do not call it riboguanosine, you do not call it something like that. You say guanosine only. Guanosine is basically nucleoside. That means sugar plus guanine. That is a nucleoside, and since in the case of RNA you do not have any deoxy term, you just call it guanosine, adenosine and cytosine and things like that. When you add deoxy, that means you're specifically talking about DNA. That's all. Okay, fine, please know that. Next, a mature processed mRNA transcribed from a gene is 636 nucleotides long. How many nucleotides are there? 636. Okay. Assuming it codes continuously without UTR and stop codons, that means there is no untranslated regions, there is no stop codons, and it is continuously being translated. How many amino acid does it encode? So 636 nucleotides are there, no stop codons, nothing. So I'll say 636 divided by 3 because 3 codons come together to form one amino acid, right? So basically your answer will be 212. 212. 212 codons are there, 212 amino acids will be there. Got it? Now if at all you consider stop codon, so you have to minus 1. But here you have clearly saying that stop codon has not been mentioned. So your answer is this. Cool? Cool. Next, statement number 1. Deoxyribonucleotide triphosphate acts as substrate during DNA replication. What do you mean by that? So basically here is your DNA strand. Okay, A, G, C, T and the list goes on. So when I say that I am bringing nucleotides for replication process to happen, for a new DNA strand to be made, you need to understand that these nucleotides are in the form of DNTPs. What do you mean by DNTPs? DNTPs means you have the deoxyribose sugar over here, the nitrogenous base attached over here. Okay, so it can be, suppose it is adenine over here, then it is thymine over on this particular side. Yeah, and in the fifth carbon, my dear children, there is not just one phosphate group. This whole thing is a nucleotide, I agree. But please remember, it is actually triphosphate that comes. Now next time another nucleotide comes over here, my dear children, the third carbon of this and this particular phosphate will be making a bond, breaking this particular bond out there, breaking this particular bond. And it produces high amount of energy which allows joining of this particular point. You get it? So it is DNTPs that comes over here. DNTPs are basically the substrates and they are also the energy source. Why energy source? Because when the phosphate groups as your bond break down over here, it provides the energy for the formation of the next bond. Okay, so deoxyribonucleotide triphosphate DNTPs acts as substrate during DNA replication. Correct. And statement number 2, deoxyribotriphosphate also acts as source of energy in the process of DNA replication. Correct. It's both substrate and energy source. So I'll say both statement 1 and 2 are correct. Got it? Yes. Fine. Now, and that is how basically nucleotide chain is being formed every single time. Okay. In a pea plant, each character is governed by a gene which is pea plant. Monocystron, polycystron. When the term comes in the question, just understand you have to think about prokaryotes and eukaryotes. Okay. Polycystron, P for polycystron and P for prokaryotes, as in you're talking about bacteria and all the things. Monocystonic, that means you're talking about eukaryotes out there. What do you mean by monocystron? Basically, this cystron is only going to have one gene and this one gene codes for only one protein. This is known as monocystonic nature of the genes. In the case of bacteria, you have polycystrons. That means they will have multiple gene segments, and these gene segments, my dear children, will be producing multiple proteins as well. That is what you mean by polycystronic nature. Okay, that is found in prokaryotes, monocystonic in eukaryotes. Now in pea plant is what? Pea is a plant and plants are eukaryotes. Anything apart from bacteria is what? A eukaryote. So just understand that anything apart from bacteria will be monocystonic in nature. Okay, yes, fine. Next thing, which of these are correct? UTR are located on the DNA, untranslated regions. Translation happens where? In mRNA. So do you think untranslated regions is there on the DNA? No, Baba. Is there in the mRNA. The UTR is present both at the 5' end, before the start codon, is it? Yes, it is present. And also at the 3' end, after the stop codon. Is it? Yes, correct. UTR provides stability to the mRNA. Does it provide stability to the mRNA? Correct. UTR is required for efficient translation. Do you think you require UTR for efficient translation? Again, correct. You require it for efficient translation. So 2, 3 and 4 is correct answer. 2, 3 and 4, that means D option is correct. Remember, these are the functions of UTR. It helps in initiation, it helps in exit also, that means termination also. It is present at the both 5' end before the start codon and after the stop codon at the 3' end. Please remember, where is this 5' end start codon, 3' end stop codon? That is also important. Sometimes they can give you that and get you confused. UTR provides stability to the mRNA. Yes, it does. So yes, your answer for this question is D. Cool? Cool. Next, in a DNA strand, if there are 100 nucleotides and 30 are adenine. In a DNA strand, if I am saying that 100 nucleotides are there, out of that 30 of them is adenine, okay, and the number of thymine, guanine, cytosine are okay. So we just have one data and we have to find what is this number, what is this number, and what is this number. How can we find that? Remember, how many, this is DNA strand, as in double stranded DNA is what you're talking about over here. How many ever adenine is there, that many thymine is also going to be there, because A base pairs with T, right? So here also it will be 30. Now remember, you are given that 100 of them is there. In total you have 100. Out of 100, 60 of them are adenine and thymine. How 60? 30 plus 30 is equal to 60. So what are you left with? You are left with 40. Now this 40 you have to divide between both of them, because this, if this has 20, this will also have 20. 20 plus 20 is equals to 40, and you have got the answer. Thymine is going to be 30. You please know the order. Thymine is 30, guanine, my dear children, is going to be 20, and cytosine is also going to be 20. So your answer for this question is going to be A. It is going to be A. Got it? Now we are getting entry into molecular basis of inheritance. Over here you can get either 2 marker or 5 marker. So these are the two things. So we are going to discuss about the 2 marker questions right up here. Yeah? So let's begin. First thing, name the methodologies used in human genome sequencing. So when if at all you want to do human genome sequencing, what is the method that you use? First thing, listen to me very carefully. DNA which is there, it has the coding regions and the non-coding regions. Non-coding regions are known as introns and the coding ones are known as exons. The transcription, when it happens, you get RNA. The mRNA, the last form of RNA which is there, the mRNA, mature form of RNA, will be just having the coding sequences, because by that time we have removed the non-coding ones, right? So if at all we are just sequencing that, that is known as expressed sequence tags. So only the genes that are going to be expressed as RNA were identified and they were sequenced. Okay, those were identified and they were sequenced. Fine, that means those that underwent transcription were being sequenced. That's all. Now, next one is whole genome sequencing. Whole genome sequencing, this basically means what? Every single thing, non-coding, coding, each and everything is going to be sequenced. Got it? So I will want to make a quick correction out there. Listen. So over here I have the DNA. The DNA transcribed into an RNA out there, right? So basically this one which is going to undergo transcription is going to be sequenced. Only the ones which undergoes transcription. Not every part of your DNA undergoes transcription. I hope you know that. Yeah, there are non-coding regions as well. Okay, so there are repeated sequences which do not code for anything out there. So the ones which are coding, that only were sequenced, and that is known as express sequence tags. Whereas on the other hand, whole genome sequencing means entire genome, coding, non-coding, everything is being sequenced, and this was done through sequence annotation method. This method was given by Frederick Sanger. Done. That is given over here. Express sequence tag, only the genes expressed as RNA were identified and sequenced. Whole genome sequencing, sequence annotation, the entire genome coding plus non-coding was sequenced, and then functions were assigned to each of them. That is the one. Next thing, draw a label diagram of nucleosome. How does nucleosome look? First of all, why do you need a nucleosome? Because the DNA is pretty much long. It is 2 meters long, and if at all you want to fit the DNA into a small nucleus, obviously you will need some proteins. And what are these proteins? These proteins are histone proteins. What are these proteins? Histone proteins. So what do you do is that you have 8 histone proteins out there. Okay. 4 of them in the front, 4 of them in the back, and there will be 1 histone protein over here also. This one is known as H1 histone. This one is known as H1 histone. Now the DNA which is rare, it is wrapped around the histone protein twice. How many times? Twice. And when I say twice, my dear children, you need to understand that 200 base pairs of DNA are being covered over here. 200 base pairs of DNA are being covered. But why is it wrapping, ma'am? What is the reason for that? Listen, DNA is negatively charged. Why? Because it has a phosphate group out there. So it is going to be attracted with something that is negative. And you know what? Here, the histone protein which is there, it is negative in nature. Sorry. Just a second. Come back. So DNA which is there, it is negatively charged. Yeah. It has to wrap around something positive, and your histone protein which is there over here, it is made up of positively charged amino acid, that is Los Angeles, that is lysine as well as arginine. As well as lysine as well as arginine. Always remember that. So draw a label diagram of nucleosome. You have to draw it like this, not like this. And please mention all of this thing. You know that, right? Yes. Now, which sequence of base is transcribed from DNA are found both in hnRNA and mRNA? I told you. So basically what happens, you have the DNA. The DNA will be having introns, that is non-coding regions, and coding regions, that is known as exons. As it is being transcribed to form a hnRNA. hnRNA will also have introns and exons. Now, I do not want this introns. Why? Because they are not coding for anything. Now, when the RNA has to undergo translation, it just needs the coding sequences. So, what do? I make the hnRNA undergo a process that is known as splicing, and I remove all the introns, and I just have the exons out there, that is known as mRNA. Apart from splicing, you have capping, where you add some groups at the 5' end and you also add adenine group at the 3' end. That is all this there. But here you have been asked what is common between this and this. So what is common between this and this? Exon. Exon is common between hnRNA and mRNA. Yes. So the coding sequence. The sequence of bases transcribed from DNA that are found both in hnRNA and mRNA are exons. Exons are what? The coding regions that remain in the mRNA after splicing, while the introns are going to be removed. Yes. Fine. Now mention the levels where gene expression can be regulated in the case of eukaryotes. Always remember, when you talk about eukaryotes, eukaryotes will be having a nucleus, inside which the DNA is going to be transcribed into what? An RNA. So the first process is that if at all I want to regulate the expression of genes, I can regulate over here at the transcription level, at this particular level, right? The next thing, I told you hnRNA is being formed, then mRNA is being formed. hnRNA is being formed, then mRNA is being formed. The next process is processing level. When I process it, splicing, capping, tailing, any of these processes, I can stop it. If at all I do not want the gene to express. So at that level also I can regulate. The third thing is transport of mRNA. So this mRNA which is there, it has to move out for translation to happen, right? So for that, transport has to take place. So transport of mRNA from the nucleus to the cytoplasm, that's also how, if at all I can stop it. That's also how I can, regulate the expression of that. Now the last one that I have is translational level. Translation level means once it is out over here, I will make sure that no ribosome is coming or attaching over here, making some changes over here. Again, I can regulate it. Okay, that is translation level. Always remember, this is possible in the case of eukaryotes. In the case of prokaryotes, they do not have nucleus. Transcription, translation, everything happens together in one particular area. It occurs simultaneously as well. So please know that. Okay, this is just for eukaryotes. Fine. Next thing, write a note on the role of sigma factor and the rho factor in transcription in the case of prokaryotes. Okay, so what do you mean by rho factor and sigma factor? So the DNA which is there, it has to be transcribed. Who transcribes it? RNA polymerase. RNA polymerase, does it need the help of someone? Yes. Who is that someone? It is sigma factor. Okay, so RNA polymerase is shy. So shy, S for sigma factor. It needs sigma factor to initiate the process of transcription. So basically in the transcription unit, you have a promoter region onto which the RNA polymerase has to attach, then that's when the whole thing starts. So along with RNA polymerase, who will come and attach? The sigma factor will come and attach for the initiation process. Then that guy will be going off. Okay, now you have the RNA being formed. Now towards the end, you have a terminator region, and then to terminate the process, again, it's like crying, oh my god, I am DNA, RNA, everything were together, but now everything is over, we are having a breakup. So it needs a rho factor. So rho factor comes over here, attaches, so that the whole termination process goes off really smooth. So there is sigma factor and there is rho factor. Please remember, sigma for initiation and rho for termination. Yeah, so that is given over here. Now write the difference between euchromatin and heterochromatin. What is euchromatin? Listen, The nucleosomes, suppose they have space between them. Okay, first of all, the nucleosomes which are there, they are like this, they are formed like this. This structure which is there, bead on a string shaped structure is known as chromatin. This is the compact form of the DNA out there. Again, you can compact it a bit more, and that is how you get chromosomes, right? So chromatin is being formed over here. If at all you have enough space between the sequences of the nucleosomes, my dear children, you say that it is loosely packed, and this is known as euchromatin. This is known as euchromatin. So euchromatin is loosely packed, and since it is loosely packed, if I put a stain, it will stain lightly only. It will be lightly stained, loosely packed, lightly stained. Next thing, here since there is space for RNA polymerase to come and attach to do transcription, it is transcriptionally active. That means transcription can easily happen up over here. So 3 things that you know about euchromatin is that basically what? In a typical nucleus, some loosely coiled regions are there with light stain, and transcriptionally they are very active. That is known as euchromatin. Now we come to heterochromatin. What happens in heterochromatin? The nucleosomes which are there, they are very tightly packed. They are very tightly packed. Do you have any space over here? No. So I'll say they are tightly packed. Yes. Second thing, they stain very, very dark. They are going to stain very, very dark. The third thing which is there, do you have any space for enzyme to come and attach to do transcription? No, it is transcriptionally inactive as well. So that is heterochromatin. Done. Now name the virus used in Hershey and Chase experiment. It was bacteriophage. So bacteriophage eats up the bacteria, which is why you call it bacteriophage. Okay, it's a virus, it's not a bacteria. Cool. Next, list the goals of Human Genome Project. What are the goals? Identify all the genes in the human genome sequencing. Determine the complete sequence of the DNA. So identify all the genes, determine the complete sequence, store it in a database, improve tool to find the data out there, transfer it to related places out there, and also address the ethical, legal and the social issues which could arise from all of this thing. And please know, remember the organisms whose DNA has been sequenced apart from human beings. So some bacteria out there, some plants like Arabidopsis, rice and things like that, C. elegans. So please remember their names, they can also pop up. Okay, so if at all you're watching this video, you can pause it and go check it out once. Now, other important concepts which are there, Hershey and Chase experiment, please go through it. Different types of RNA polymerase in the case of eukaryotes that I'll be talking about here. Genetic content in different organisms. So genetic content is quite different in different organisms. Bacteriophage lambda has something else. Bacteriophage phi has something else. So please know about it. I have done several videos. If at all you have any problem with the concepts, please watch the master stroke one shot. Okay, and if you feel that it's too difficult for you to complete the whole one-shot video, go target the area where you find difficulty in and watch it. It is definitely going to be helpful for you. Now, when I say RNA polymerase, listen, RNA polymerase is there for transcription, right? So, in the case of prokaryotes, there is no different type of RNA polymerase, only one. In the case of eukaryotes, we have 3 different types. So, RNA pol 1 is responsible for transcription of rRNA, ribosomal RNA, and they are of different units. One is 28S, one is 18S, and one is 5.8S. Okay, then we have what? RNA pol 2. Okay, RNA pol 2. Second priority is to do the transcription of hnRNA, that is heterogeneous nuclear RNA. When DNA to RNA forms, no, it is hnRNA that is being formed. So that is the second priority. Now RNA polymerase 3, RNA pol 3 and T and 3 looks here is the same. So it is tRNA, transfer RNA, that is being transcribed with the help of this. Apart from it, one type of rRNA is also being formed, and that is basically 5S rRNA. So here everything was ending with 888, but here it is 5S rRNA, which is why this is different. And also small nuclear RNA. Small nuclear RNA is being transcribed from this. This is very important information. Please keep this in your mind as well. Works? Super important. Yeah? Fine. Okay. Now we are going ahead with the next chapter, that is Principles of Inheritance and Variation. If at all you have reached up over here, type you have crossed, level 2 crossed. Okay, level 2 crossed. Okay, so let's get into this world. So question number 1, progenies with A, B and O blood groups are not possible from which of the following mating parents? You have been asked that A, B and O blood groups should not be produced, are not possible from which of the following parents. So for A, B to come, you understand, for A, B to come, one parent should have at least one gene that is A, and the other parent should have another gene that is B. Right. For O blood group to come, my dear children, one parent should have O and other parent should also have O. Then only O blood group will be coming in. Now, I have to make sure that basically O I cannot guarantee, okay, but for this I can take the help of the information given over here. If I have A on this side and B on other side, that means there are chances of AB blood group to be formed. So here you have been asked which one progenies with AB are not possible from which of the following parents. So you have to exclude the ones from which it is possible, right? So from here it is possible because A is here, B is here. From here also it is possible because A is here, B is here, A is here, B is here. So wrong. A is here, A is here, fine. A is here, B is here, opposite side, so again wrong. So basically, my dear children, you are just left with this particular option. Here, if the parent is O blood group, that means both this are OO. Here the parent is AB blood group. That means it is like this. This and this fuses, you get IA, IO. That means it is going to be A blood group. This and this fuses, you get IP, IO. That means it is going to be B blood group. Why? Because B is dominant over O. This and this fuses, again IA, IO. A is dominant, A will be shown up. When this and this fuses, again you will get B blood group. But anyway, AB and O is not there, which is why your answer for this question is going to be A. Got it? Yes, got it. Fine. Next question. What is the possible blood group of an offspring when both the parents have AB blood group? Now you tell me the answer. If both the parents are having AB blood group, my dear children, A, B, B, A and B. Okay. This and this comes together, you get IA, the blood group is A blood group. This and this comes together, you get IA, IB, the blood group is going to be AB. Both of them are co-dominant over here. This and this, again IA, IB, again showing co-dominancy AB. This and this comes together, it is IB, that means B blood group. So how many possible things are there? Oh, okay, just a second, okay. What is the possible blood group of an offspring when both the parents have AB blood group? It will be AB, A and B. AB, A and B. So your answer for this question is B. This is the answer. Okay? Yes. Fine. Now, genes located very close to one another on the same chromosome tend to be transmitted together and are known as genes which are very close to each other, like this. They are known to be they are basically going to be transmitted together. Why? Because on the chromosome they are very close to each other. Even if there is cutting and splicing and things like that happening, my dear children, what is there? These two genes are always going to be close. What do you call them as? These are known as linked genes. And when they are very close to each other, you call them as tightly linked genes. Tightly linked genes. And one very important information from Jasmine Baba, and that is basically tightly linked genes will have lesser recombination. That means they will not be forming new combinations. They are very close to each other. There is no point of formation of new combinations. So I'll say lesser recombination. Remember that. And if they are loosely linked, they are far away from each other, they will have more recombination. Okay? Okay. Next, try it out. So I have given a question for you to try it out. Which three scientists independently rediscovered Mendel's work? Any idea? So Mendel was very sad because his works were not recognized. Later on, after 37 years, my dear children, his works got recognized. By whom all? So please let me know in the comment section below. You have got the question. You need to give me the answer. It is given in the NCERT. Just before chromosomal theory of inheritance, you will get the answer. If at all you do not know. Yes. Now, a plant having the genotypes capital A small a, capital B small b will produce. So you can give me answer like this, homework. Okay. So plants having a genotype capital A small a, capital B small b will produce dash kinds of gametes. How do I know how many type of gametes are going to be produced? So the question is very, very simple. If at all you know the formula goes like this. 2 raised to the power n. N is the number of heterozygotes. The number of heterozygotes. And what is it? Any idea? This is a heterozygote because here it is capital A small a. Both of them are different alleles. So I'll say this is heterozygote. So number 1 heterozygote. This is also heterozygote. Number 2 heterozygote. So I'll write 2 raised to the power 2. Right? 2 raised to the power is equals to what? Is equals to 4. So 4 type of gametes are going to be formed. Your answer for this question is B4. Okay? Okay. Fine. Now the next question that we have, which of the following is not a pair of opposing characteristics Mendel studied? Which of the following is not a pair of opposing characteristics that Mendel studied? So Mendel has 7 pairs of contrasting characters that he studied. Okay, which one is not? Axil and terminal flowers, this he studied. Which one is dominant? Axil one is dominant. Axil means over here from the position where you have buds and all, you have the flowers. Terminal means at the tip. So which one is dominant? This one is dominant. Okay, full and constricted pods. Full means nice pod, okay, which has the green peas in that is full. Constricted means it is like this. Which one is dominant? Full one is dominant. Green and yellow pods. So when you go to the market, which one do you pick? Green color ones or yellow ones? It will be green. So that is dominant. This is also correct. Pink and white flowers. Mendel, if he was here, he would have given me the answer and said that, Papa, what question is given over here? It is purple or basically violet and white flowers, not pink and white. Okay, it is violet. So this one is the odd one out. Okay, and over here, these are the features that are given. Please remember there is a formula gravity for remembering all the dominant traits. G for gravity, G stands for green. So in the case of seed color, in the case of pod color, green is dominant, right? In the case of seed shape, R is dominant. So gravity R. So all the, so I'll write it. I think that will be better. So in seed, we think that green is dominant, but actually in seed it is yellow, which is dominant in color. Okay, so please know that. In pod it is green. Yes. So G stands for green. R stands for round seed. A stands for axial flowers. V stands for violet flowers. And I stand for inflated pods, or what do you call as full pods. T stands for tall, and Y stands for yellow seed. Okay, so these gravity is basically for all the dominant traits that you need to remember. Cool. Now, chromosome maps or genetic maps were first prepared by chromosome maps. So basically, it's like on the chromosome at this position, this gene is located. Who discovered that? It was Sturtevant. Sturtevant is what? Alfred Sturtevant is a student of Thomas Hunt Morgan. Thomas Hunt Morgan gave the idea about linkage and recombination. He said that genes basically which are very close to each other, they show lesser recombination. So with the percentage of recombination, if at all the percentage of recombination is more, that means they are far from each other. And if that is understood, basically I can locate the genes also. So basically, if I am saying the recombination percentage is more, that means the genes are very far from each other. And if I had the data for recombination percentage, with that I can also understand where are these genes located as well. Okay, so the work was done by Thomas Hunt Morgan. He gave down two things, that is this, as I said, linkage and recombination. Sturtevant went ahead and gave an idea about the actual thing, that is genetic map. Okay, or gene map. Yes. Next thing, which of the following is incorrect regarding ZWZZ type of sex determination? ZWZZ type of sex determination, if at all you remember, my dear children, ZWW stands for woman. Over here is the female who are going to produce 2 types of gametes, one Z and another W. Usually in our case, in the case of human beings, it is XXXY. XY is produced by males. Males produce 2 types of gametes. But here, females are the ones producing 2 types of gametes. This is known as female heterogamety. Heterogamety means different type of gametes. And it is only seen in the case of birds. In the case of birds. So it occurs in birds, correct. Females are homogametic. Arre baba, they are not homogametic. Males are homogametic over here. Females are heterogametic and males are homogametic. Females are heterogametic, males are homogametic, correct. How can there be? Okay, you have to find what is the incorrect one. Yeah. Okay, just a second. Yes. Total number of chromosomes is same in both male and female. Yes, it is. It is very much same in both male as well as female. So this is also correct. What is wrong over here? Only one option is wrong over here, and that is this one. Okay, just a second. So when I mark things wrong, I put the answer like this. This is the answer. Female or heterogamety, that is correct. Okay, this one is the wrong one. Cool? Yes. Okay. Can we go ahead with the next question? Please know all the different types of sex discrimination. In honeybees, the number of chromosomes are not same in the case of male and female. In the female, there are 32 chromosomes. In the case of male, there is only 16 number of chromosomes. Okay? Yes, half the number of chromosomes. Okay. Now let's go ahead. A woman with normal vision whose brother is colorblind. Okay. Now this is color blindness related thing, so you have to be very, very careful. A woman with normal vision whose brother is colorblind marries a man with normal vision. They have 4 children, 2 sons and 2 daughters. One son is colorblind while the other son and both the daughters have normal vision. The pattern of inheritance for the diseases. So over here, it's such a simple question. Listen to me carefully. They just put all this drama to confuse you guys. First of all, to scare off you guys. Here they have clearly mentioned color blindness. So you know color blindness is a disease. What is color blindness? It is sex-linked recessive disorder. We all know that color blindness and hemophilia. Color blindness and hemophilia, both of them are sex-linked recessive disorders. So can I just write sex-linked recessive? Do I need to even think for a second? Okay, so your answer for this question is going to be A. Fine, ma'am. Okay. Now the next thing, if both the parents are unaffected carriers for thalassemia, which is an autosomal recessive disorder, what are the chances of pregnancy resulting in an affected child? So they have mentioned it is autosomal recessive disorder. That means it is not going to affect any sex chromosome, it is affecting your autosomes. In your autosome, some mutation is there, that is why this disease is there. And they have also mentioned that both the parents are what? Unaffected carriers of thalassemia. Okay. So in thalassemia, your hemoglobin is affected. So I'll just write like this, okay? So suppose this is a normal one and this is the affected gene. This is a normal one, this is the affected gene. They are the carriers. The carriers will have one affected and one normal. The normal will be dominant over this, which is why they will be just a carrier, they will not have the disease, right? So you have been asked how many of them are going to be affected in the children. That is the question. So let's do the calculation. This and this goes HBA is going to be a normal child. This and this, it will be HBA and HBT. They will be a carrier child. We do not know he or she because it is autosome linked. It is not sex chromosome linked, right? Now this and this, HBA, HBT. Over here, again my dear children, they are going to be carriers only. Lastly, you have HBT and HBT. This one, both the genes are being affected. Okay, both the chromosomes are being affected, which is why, my dear children, there is no chance that a normal baby can be produced over here. So I'll say out of 1, 2, 3, 4, one child can be affected. Out of 4 children, one can be affected. That means, can I say 25%? Yes, I can say that. You get it? So please know, whenever you're speaking about autosomal recessive, there's no male and female over here. All of them can be equally affected. We do not know the sex of the child. You just have to look for the percentage of the ones which are affected. Okay? And they will be carriers. They're going to be carriers. And carriers are not counted as affected. So yes, if they're asking you about carriers, please mention about that as well. Next thing that we have is principles of inheritance and variation, and again you have got a 2-marker question coming off from this chapter along with 5-marker and 1-marker. So let's deal with that first. First question that we have is recombinants obtained in both Mendelian and Morgan experiment, yet the processes involved are different. Comment. So recombinants as in new combinations were found in both the experiments, Mendel's experiment and also Morgan's experiment, but they were different. Why? So listen, in Mendel's experiment, what did he do? Basically, he crossed round yellow with wrinkled green. In the first one, all of them were round and yellow, but they were round and yellow like this. Okay, they were not like this. They were round and yellow like this. Fine. So now, when these plants crossed, okay, when similarly I cross one particular plant with this, I got non-parental character. I got new character which was not there in the parent at all. So I crossed round and yellow and wrinkled and green, but I got round and green and wrinkled and yellow along with the parental varieties as well. Why did I get it? Because of one very important reason. That is, here if at all I make the chromosome, it looks like this. Capital R, small r, capital Y, small y. Right? When the gametes form, from this pair, only one can go into the gamete, into one gamete, half the chromosomes are going to go. So here it is capital R, from here it can be capital Y, this is one gamete. Again, from here it can be capital R, from here it can be small y also. You get it? So what do you get to understand from it? Independent assortment. Alleles of one pair of character, that is shape, are going to independently assort from another pair of character, that is what color. There is no challenge in that I'll only go together, which is why you are able to see non-parental characters out there. So in Mendel's cross, recombination occurs due to independent assortment of the genes. Yes, there was independent assortment that is taking place, whereas in Morgan's experiment this was not the case. Okay, in Morgan's experiment, recombination occurs due to crossing over between linked genes. So in Morgan's experiment, he said that basically if I have a gene over here which are very close to each other, and if at all I exchange this, both these genes will be always together. They will go together only to the other part, right? So I'll say recombination is less, new combination is less. But suppose one gene is here, one gene is here. Now if at all I do crossing over, what is going to happen? Basically this part, okay, I'll just show it to you. So over here, if I do crossing over, one gene is over here and another gene which is there, it is over here, which I mark in white. Suppose this and this part gets crossed over, exchanged. What is going to happen? I get a new chromosome which looks like this. The yellow part went here. This gene is over here. Am I not getting a new combination of chromosomes out there? Yes. Why? Because these genes are very far from each other. So over here, why is new combinations arising? Because of one very important thing, that is crossing over between the linked genes. There is a crossing over that is happening. That is why we get new parental varieties. Sometimes there won't be much recombination happening. If at all the genes are very close to each other, my dear children, they will go together only. They will not go to separate things. For example, again, there is two chromosomes. These two genes are very close to each other. If at all I do crossing over, what is going to happen? Again, I'll get these two chromosomes here, these two part over here, because they are very close to each other. Then recombination is going to be less. But here, recombination or non-parental varieties are being formed of only one reason, and that is what? Crossing over. Okay? Yes. Fine. So differentiate between linkage and recombination. What is linkage? It is the tendency of genes located close to each other on the same chromosome to move together, right? Recombination means what? It's a process by which new combinations are going to be formed. Linkage, it reduces the chance of new gene combinations. Recombination, it increases the chance of new gene combination. Linkage and recombination both was given by Thomas Hunt Morgan. I've explained it to you in the live in a detailed manner. I'm telling you again, if at all you feel that you need a more explanation, see the live. Yeah? Of top 50 questions. Now identify the symbols used in pedigree analysis. Over here you have a square and a circle and two lines are given. Square stands for male and circle stands for female, and over here when two lines are there, it means mating is happening between relatives. If one line is there, normal mating. If two lines is there, mating between relatives, you also call this as consanguineous mating. Okay, now there is one more thing. A male and a female and a child is being formed, and this is colored. That means the child who is being formed, which is a male, is having a disease. So parents with a male child affected with a disease. That is what you mean by that. Yeah? Yes. And suppose something like this is given, that means sex is not being specified. And if something like this is given, 5 children are being there, but sex is not being specified. If I color it up, 5 children are there who are affected and the sex is not specified. Cool. Now, what is haplodiploid sex determination mechanism? Mention an animal which exhibits this. Haplodiploid sex determination mechanism means if at all an organism have half number of chromosome, it will be this sex. If at all it has full number of chromosome, it will be this sex. Okay, that is what you mean by haplodiploid sex determination. Sex is determined on the basis of number of chromosome that they have, and this is very easily seen in the case of honeybees. In honeybees, my dear children, the females which is there, it will be having 32 number of chromosome, right? Now, when it has to form gametes, it will undergo meiosis. Yeah, that's how gametes are being formed. It will be having half the number of chromosome in each gamete, 16 and 16. What about the males? Males were already having 16 number of chromosome. They are already haploid in nature. So this is diploid. This female is going to be haploid. Males are always going to be haploid. This guy cannot undergo meiosis again. It just undergo mitosis. Okay, there is no meiosis here, only mitosis. 16 number of chromosomes are here. Now suppose the male and the female gamete are fused. 16 plus 16, 32. If it is forming like this, then it is a female. Why? Because it is diploid. It will transform into a female. What about the male? How it is being formed? So the female gamete, without fertilization, if at all no fertilization is happening, we say with the help of parthenogenesis. There's an R over here that is being missed. Okay, with the help of parthenogenesis, you are going to make a male. Okay, so there is no fertilization. Please remember that is how you get males. And since there is no fertilization, we say that this male does not have a father. Does it have a father? No, Baba, it does not have father. Does it have a grandfather? Yes. How a female is being formed? With the help of a father as well as a mother. That is why it has a grandfather but does not have a father directly. Got it? If at all that is not being processed, slow down. Watch this part of the video once more. That is definitely going to help you out. Works? Works. Awesome. Now define point mutation. Give an example for point mutation. Point mutation means mutation is happening only in a single base pair. Only in a single base pair. For example, I say there is GAG over here. Now instead of A, it is replaced by U. It is replaced by U. What is going to happen? This was coding for another amino acid that is known as glutamic acid. That is known as glutamic acid. Now it is going to code for another amino acid that is known as valine. This is a point mutation where only one base pair is going to be changed. Got it? Cool? Yes. So this is sickle cell anemia as well, where one amino acid is being changed. That is why the whole hemoglobin sequence would be hampered. Perfect. Now, what are the conclusions drawn by T.H. Morgan from the crossing experiment in Drosophila? He said that if at all the genes are tightly linked to each other, they will show lesser recombination. That means if they are close to each other, they will show lesser recombination. If they are very far from each other, they will show higher recombination. So I always give this example. Please listen to me carefully. You and your best friend. If at all you are close, there are less chances that another friend can come into the circle, right? So lesser recombination. You and your friend, if at all you are not close, there is more chance for another person to also come in. More recombination. Got it? That's all. So yeah, we were here. Principles of inheritance and variation. Let's go. So 2 and 5 marker questions are there. 2 marker I have already discussed, and the 5 marker you will be seeing over here. First question that we have is, T.H. Morgan selected fruit flies for his genetic experiment. Why? Why did he take fruit flies for his experiment? First of all, they were very easy to grow. Second thing, they had a very short lifespan. So if at all I want to observe some changes, I just have to wait for few weeks. Okay, just 2 weeks, because in 2 weeks the lifespan is going to end up. Next thing, a single mating will be producing too many progenies, so basically that was also helpful. Next thing, there's a clear differentiation between male and females. It was very easily distinguishable, so that is also one very important reason. So these are the things, please write it down somewhere if you forget. Now, what are chromosomal disorders? Give an example. So listen. Chromosomal disorders are one and Mendelian disorders are the other one. Mendelian disorders basically means what? Suppose I have a chromosome over here. This chromosome and gene is there. This gene has some mutation, which is why the proteins are not being made properly. This is Mendelian disorder. This leads to Mendelian disorder. Chromosomal disorder means I needed to have 2 chromosomes, but I have one more chromosome. Extra things being made. Or probably I need to have 2 chromosomes, but I have only 1 chromosome. Chromosomal disorder. Suppose I have only half of it, other half is not there, leading to what? Chromosomal disorders. These are the things which comes under chromosomal disorders. So the disorder in human beings occurs due to absence or excess or abnormal arrangement of one or more chromosomes are called as chromosomal disorders. And the best example for that is Down syndrome, Turner syndrome, and also Klinefelter syndrome. Down syndrome, Turner syndrome, and also the Klinefelter syndrome. Got it? It's very simple, okay? That is how you have to write this particular question. Now this question is actually easy compared to others. So let's go ahead with the difficult ones. Slowly we'll move it, okay? Now give reasons for the following. A simple cut results in non-stop bleeding in hemophilia affected individuals. Why? So if someone is affected with hemophilia, no? If I get a small cut down there, the blood is going to go. Usually when we get a cut, there's clotting happening in our body, right? It clots suddenly. Okay, we have proteins for that. But in them, it's not like that. Why? Probably because the proteins that are responsible for clotting are not being made. That means the genes that are going to code for those proteins might be affected. You get it? So I will say that a simple cut results in non-stop bleeding because a single protein that is a part of a cascade of proteins, a single protein which is a part of a group of proteins which is involved in clotting is being affected. That is not being formed. That is the reason why we have non-stop bleeding in patients who have hemophilia. Now, Turner syndrome affected females are usually sterile. So, Turner syndrome is the case where you should be having 2 chromosomes, as in 2 X chromosomes for being a female. But over here, there is a loss of 1 chromosome, which is why we say this is XO condition. And since there is loss of 1 chromosome, I'll say that this particular female, even though she has feminine characters out there, she will not be having a proper ovary. Her ovaries are going to be rudimentary. They are not going to be properly formed. So I'll say it is rudimentary ovaries which are going to be there. And this rudimentary ovaries cannot give rise to what? Ovum. Got it? That is why she is sterile. She cannot produce offsprings because the ovum itself, the gamete itself which is there, it's not of any use. So Turner syndrome affected females are usually sterile because they have rudimentary ovaries. They might not even produce ovum. That is also a case that could be there. So how do you frame the answer again? We need to know. Let's see. So females are usually sterile because they have only 1 X chromosome, leading to underdeveloped ovaries and lack of normal gamete production, which is there. Safe answer. Now, in Morgan's dihybrid cross experiments on Drosophila showed that flies having genes for yellow body, the flies having genes for yellow body and white eyes exhibited lesser recombination. What do you mean by that? General thing that I want to tell you is this. Listen, suppose there are 2 chromosomes. My body is boiling. Okay, so there is A and there is B chromosome over here. There is capital A and capital B over here. These 2 genes are very close. These 2 genes are also very close. If at all I cut it and I exchange the parts, what is going to happen? Basically, I get a new chromosome which looks like this. Capital A and capital B comes on this side. Small a and small b comes on this side. Aren't these genes still together? Aren't these genes still together? Do you find any new combinations happening apart from, upper part is, yes, according to it, it will be new combination, but if at all you see these 2 genes, they are still close. So I'll say lesser recombination is happening there. Why? Because these 2 genes are tightly linked. They are tightly linked. When they are tightly linked, obviously lesser recombination will be there. And the example that I always use is that you and your best friend, if at all you're too close, there are less chances for someone else to enter your team out there, right? So lesser recombination out there. On the other hand, if at all the scenario was something like this, where you had capital A here, capital B here, small a here and small b here. These 2 genes are very far away from each other, right? Now if at all I cut this part and exchange, what is going to happen? Basically something like this is going to happen. Capital B, small b. And here since it's an exchange, small a, capital A. You see small a and capital B and capital A and small b is there. This was never the combination. So there is a higher amount of recombination out there in loosely linked genes. They are loosely linked and they are tightly linked. So here when I say in Morgan's dihybrid cross experiment on Drosophila, it showed that flies having genes for yellow and white eyes exhibited less recombination. That means probably they might have been very close to each other. They are very close to each other. Please remember it. Okay, and the other one, so there is one more thing, miniature wings. So miniature wing gene is very far from each other, which is why we say they have more recombination. The highest amount of recombination is how much? Do you know? It is 50%. So in this, they show almost around 37.2% recombination. That means it is too high. In this, basically only 1.3% recombination. Only 1.3% recombination. That means it is too less. So know that. Yeah. Okay. How do you frame the answer again? Yellow body and white eye genes in Drosophila show less recombination because they are located close to each other on the same chromosome, exhibiting what? Genetic linkage. That's all. Okay. Now we have one more question. Inheritance of skin color in the human show different phenotypes. Why? Inheritance of skin color. So do you think my skin color and your skin color is the same, or you and your friends is the same? No. Why? Because actually we show an exception to Mendel's law. Mendel said that 1 gene, 1 protein, right? But here it's not like that. Here what is going to happen is that multiple genes are going to code for one character, that is your skin color. So you have suppose capital A, capital A, capital B, capital B, capital C, capital C. Your genotype is going to be capital A, capital A, capital B, capital B, capital C, capital C. This is the genotype. This genotype shows highest skin color. Suppose your genotype is changed over here. It is not capital A, rather it is going to be small a. Similarly over here, it is not capital B. Here it is going to be small b. Here it is not capital C, here it is going to be small c. What is going to happen? The genotype is going to change. It is going to be capital A, capital B, small b, capital C, small c. This will be having lighter skin color than this. The more number of recessive alleles that you are using, my dear children, it will be lighter skin color. The lightest will be something like this. So here what do you see? Your skin color is determined by multiple genes out there, which is why since human beings will have different combination, we all have different skin color as well. Okay, so inheritance of skin color in humans show different phenotypes because this is showing polygenic inheritance. Skin color shows polygenic inheritance, but multiple genes has a role to play. And it is not just skin color, it also your height. Human height is also following polygenic inheritance. Okay, so it shows different phenotypes due to polygenic inheritance where multiple genes contribute to the phenotype, leading to a range of skin color variation. Different skin colors being formed out there. Works. Okay, next thing, accumulation of phenylalanine in the body of phenylketonuria affected individuals. Why do you think accumulation of phenylalanine happens? Why do you think accumulation of phenylalanine happens? Because an enzyme which is needed for converting phenylalanine to tyrosine is not being formed. Okay, this enzyme, the gene that codes for this enzyme is being mutated. Now, do you think enzyme is going to be formed? No. Do you think conversion will happen? No. So, phenylalanine is going to be accumulated in the form of phenylpyruvic acid for the same reason. And this accumulation will lead to mental retardation as well. It will lead to mental retardation as well. So, phenylalanine accumulates in the body due to defect in the enzyme phenylalanine hydroxylase, which converts phenylalanine to tyrosine, causing what? Toxic buildup out there. Okay, this is also an example of pleiotropy. It's also an example of pleiotropy. I'll tell you. Shall I tell you? Yes, I'll. So, listen to me. Phenylketonuria, what happens is that phenylalanine which is there, it has to convert to tyrosine with the help of the enzyme phenylalanine hydroxylase, PH enzyme. Now this enzyme is not being found because the gene is being mutated right there, right? So this thing is going to be built up, okay? They convert into phenylpyruvic acid and they get accumulated in our brain and causes mental retardation. It causes mental retardation. Now one more thing is being affected. Tyrosine which is there, it is super important for a pigment to form in our body. And this pigment is responsible for our hair, for our skin and our hair pigmentation, hair coloration, right? Since this pigment is not being found because the tyrosine is not being found, your skin and your hair will be having reduced pigmentation. So one gene is being affected and two characters are affected. One gene is affected, which is why two characters are affected. This is known as pleiotropy. So usually according to Mendel, one gene, one character, but here one gene, multiple character can also be affected. So please remember it's an example of pleiotropy as well. Works. Pleiotropy, polygenic, do not get confused. Now, few autism-linked recessive gene blood diseases occurs in human population. Among them, some are related to qualitative and some are related to quantitative problem of synthesizing blood proteins. Explain. So, let's see. Yes. So, they are autosome recessive gene disorders. So, I am just dividing into two. One is going to be sickle cell anemia. Another one is going to be thalassemia. So what is going to happen over here and here? Know the difference. You have chromosome number 11 where you have HBB gene. You know HBB gene codes for what? Codes for beta globin chain. Beta globin chain of hemoglobin. So ultimately your blood, your RBC has hemoglobin and hemoglobin is made up of alpha chain and beta chain, right? And this hemoglobin is inside the RBC, right? So here the beta globin chain which is there, okay, it is not being produced nicely, as in there are some changes in the beta globin chain. What changes and why did it happen? Because there were some mutations over here. Okay, let's see what is the problem that is occurring over here. Okay, so it was supposed to be GAG codon at this 6th position. So there are multiple codons. The 6th codon was supposed to be GAG, which codes for glutamic acid. Glutamic acid. Now, my dear children, instead of GAG, it is GUG. There is a point mutation. There is a change in the single base pair. Point mutation. Instead of A, it is U, which is why the codon will be now coding for another amino acid that is known as valine. And each amino acid has to come together to form what? A polypeptide. Over here, valine is going to come in this particular case and going to form the beta globin chain. Do you think it's a normal beta globin chain? No, it is not. It is a distorted one. Now, this beta globin chain comes along with an alpha chain and forms a hemoglobin. So, this hemoglobin is not proper. This hemoglobin goes into an RBC. In this RBC, the normal shape should have been different, but here it is going to be different. Just a second. Okay, dear children. Sorry, someone came in. Okay. So RBC is over here. Suppose it has this bad hemoglobin, what is going to happen? There will be a change in the structure of the RBC. So RBC has a biconcave shape, but now it will be spindle in shape. And when it becomes spindle in shape, what is the problem? It will go and bring clots in the blood vessels out there. And also remember, all of this change in the shape is happening because of one big reason, that is, there is a problem with the hemoglobin. When there is a problem with hemoglobin, there will be reduced oxygen carrying capacity. There will be less oxygen out there, which is exactly why the shape of the RBC also becomes changed to a spindle shape. Got it? This is the basic logic out there. For sickle cell anemia, always remember this is an example for point mutation that is happening over here, and A comes before U, so it was A first and then it was replaced by U. Similarly, G comes before V, so glutamic acid was the first thing, now it is replaced by valine. Right? Here, it is an autosomal recessive disorder. Why? Because these genes which are there, HBB genes, it is on chromosome number 11, which is an autosome. They are autosome. They are not 6 chromosomes. They are regular chromosomes that is known as autosomes out there. And basically, I call it a recessive disorder, which means that both the chromosomes should be affected for someone to have this particular disease. Otherwise, they will be just a carrier. So in recessive disease, both the chromosome have to be having the recessive alleles, then only the disease will be got. Otherwise, that person would be just a carrier. Got it? I hope that is clear. And also over here, what is the problem? The quality of hemoglobin which is being produced, that is a problem. So it is a qualitative disorder. It is not quantitative. It's a qualitative disorder, that is in the case of sickle cell anemia. Now when we talk about the other disease, that is thalassemia, what is the problem up there is that there are 2 chromosomes. This is chromosome number 11 and this is chromosome number 16. On chromosome number 11, what do you have? You have HBB gene, HBB. On chromosome number 16, my dear children, you have basically HBA gene. That means this chromosome will have genes which will be helping in the formation of beta globin chain, and here you have the genes that is coding for alpha globin chain. And there are 4 of the genes over here. There are only 2 gene locations over here. Please remember that. So I'll call this as HBA1 and this is HBA2. HBA1 has 2 genes. HBA2 also has 2 genes. Now listen. Here the problem is that suppose this gene is being affected. What is going to happen? There is a normal gene that will still help in the formation of beta globin. So beta globin chain is going to be produced, but the quantity of this is reduced. Suppose both of them are affected. Then beta globin chain is not going to be produced at all. You get it? So this is a quantity thing. Similarly over here, suppose one particular gene is affected, what is going to happen? Still alpha globin chain is going to be produced, but the quantity is going to be it is going to be less. Suppose Two of them are affected, again the quantity of alpha globin chain which is produced is less. Suppose all four of them is affected, alpha globin chain is not produced at all. When alpha globin chain is not being produced at all, do you think you can make hemoglobin? No. If beta globin chain is not produced at all, do you think you can make hemoglobin? No. So remember always, this is going to be a quantitative disorder. It's what, my dear children? It's a quantitative. It's because of the quantity of the globin chains which are being produced. Okay? And when you have so many genes being affected, that is why when you say that you have a major thalassemia patient, if only one gene is affected, other gene is still functional, you say that you are minor thalassemia patient. So please remember that thing also. This is also autosomal recessive trait. Why? Because over here also it's a autosomes genes which are being affected. Got it? Now how do you frame the answer for this? In sickle cell anemia, mutation in the HBB gene on chromosome 11 causes the production of abnormal hemoglobin. This defective hemoglobin leads to sickle-shaped red blood cells, impairing the oxygen transport and causing blockages in the blood vessels. Here the defect is caused because of the error in the quality of the protein that is being made, right? Whereas in thalassemia, it is caused by mutations in gene responsible for hemoglobin synthesis. HbA1, HbA2 for alpha genes, HbB for beta genes. There can be mutation in one allele of a gene, one allele of a gene, or there can be mutation in what? In more than one allele or both the alleles of a gene. It leads to reduced or no production of specific hemoglobin chains out there, causing anemia and fatigue. So anemia and fatigue is for this particular thing, thalassemia. Sickle cell anemia, it's a life-threatening disease. It can lead to death also. Similarly here also, if all the genes are affected, it can lead to death. Thalassemia also. Here the defect arises due to errors in the quantity of the gene which is being produced. So yes, so please remember, it's a qualitative and this is a quantitative disorder. That's all. Cool. Next thing that we have is define aneuploidy. Give two examples of aneuploidy. What do you mean by aneuploidy? Aneuploidy means either the loss or gain of the chromosome. That is what you mean by aneuploidy. Now if it is a gain, you call this condition as trisomy. If it is a loss, you call this condition as monosomy. Now you have been asked, mention the karyotype of Klinefelter syndrome of an affected individual and write its symptom. So in Klinefelter's, what is going to happen, my dear children? You should be having XY chromosome, right? These are sex chromosome. But here you have one more X chromosome leading to XXY condition. Okay. Generally in our body, how many chromosomes are there, Baba? 46 chromosomes are there. 46 chromosomes there. Out of 46 chromosomes, 44 of them are autosomes. 2 of the chromosomes, that is 45th and 46th, they are the sex chromosome. They are what? The sex chromosome. Always remember that. Now over here, when I say you have an extra X chromosome, how do you denote it? Basically, you can write like this, that is 45 autosomes, comma, XXY. This is known as karyotype. This is known as karyotype. So how many autosomes do you have? Not 45. Here it is going to be 44. You have 44 autosomes in XXY. This is 44th chromosome, 45th, 46th, 47. In total, do you have 47 chromosome? Yes. Why? Because it is a trisomy condition. You have an extra X chromosome. Sometimes instead of writing like this, they also write it like this: 47, comma, XXY. That means 47 chromosomes are there and probably the extra one would be this X. So you can either write like this or you can write like this. This is known as the karyotype of Klinefelter's. And I told you what happens in Klinefelter's, you have an extra X chromosome. This is a trisomy condition, right? And because of this, the males which are there, they have a overall masculine development. Okay, males will be having overall masculine development, but they will also have gynecomastia, as in there will be development of breast. There will be development of breast in this particular case. Apart from that, my dear children, what is the next thing that you have? Apart from gynecomastia, there will be underdeveloped secondary sexual characters. Do you think they can be fertile? No, they will be infertile. Why? Because there is no spermatogenesis and anything happening over here. Do you think they will have height and everything out there? Yes, they have tall stature with long arms and legs. And learning, okay, so this is all is not important. At least remember these characters out there. Apart from it, you have multiple other examples for trisomy, autosomal trisomy. Listen, this is a trisomy, okay, but this is allosomal. Why allosomal? The sex chromosomes are known as allosomes. So here the sex chromosome is extra, that is why allosomal trisomy. Similarly, you have autosomal trisomy, that is Down syndrome, where the 21st chromosome is affected. 21st chromosome is an autosome? Yes, it is an autosome. Okay, so 21st chromosome, my dear children, over here, when it is being affected, how it is affected? You should be having 2 chromosomes, right? But you have an extra copy of the chromosome. Trisomy of 21st chromosome, which is why this person will be having symptoms related to Down syndrome. But again, what is the karyotype over here? Usually you should be having 44 autosomes and then XX, that is for females, 44 autosomes and XY for males. Here you have 45 autosomes. One extra autosome is there. Yes, one extra autosome is there. So can I write over here 45 autosomes XY if it's a male who is having a XX is if a female is having Down syndrome, 45 autosomes XY if it's a male who is having Down syndrome. Now what about Turner syndrome? In Turner syndrome, it's a monosomy condition. You should be having XX, but you just have XO. There is only one X, there is no other X. So again, how do you show that? Autosomes are normal, 44A, but sex chromosome is like this. So you write 44A XO. So 44 and 45th chromosome, that's all. Otherwise, you can also write in total how many chromosomes are there, Baba? Instead of having 46, you just have 45. So 45, comma XO. This can also be written, but I prefer this. This is uniform. This will be easy for you to remember. Okay? Yes. Perfect. So aneuploidy is a condition in which organism has an abnormal number of chromosomes, either due to what? Gain or loss of one or more chromosomes. It is caused by non-disjunction. This is very important. How it is caused? It is caused by non-disjunction during cell division. Okay, there is no proper separation that is happening during cell division. That is why this particular thing is happening. Non-disjunction during cell division. That leads to aneuploidy. Okay, you might have also heard about polyploidy. Polyploidy is basically the overall number of chromosomes is being increased. The whole set of chromosomes number is being increased. That usually happens in the case of plants. Okay, it happens in the case of plants. And that is not because of non-disjunction during cell division. That is because of basically the karyokinesis took place. Nucleus division took place. Okay, but the cell division did not take place, which is why a cell will be having double the number of chromosomes. Okay? Okay. Now, represent schematically the results of one gene interaction between pure tall pea plant and pure dwarf pea plant. Pure tall pea plant and pure dwarf pea plant. How do you explain that? How do you show one gene inheritance? This is very, very simple. Okay. So, pure tall looks like this, capital T. This will be small t, small t. Here the gametes. So, first write the parents. You have to write it. Okay. I'm not writing it, but you need to write it. Now, the gametes. From this 2 chromosome, one can go into a gamete. So either way it will be this. Similarly, here it is going to be this, right? Now you do the fusion. This and this, you get capital T small t. This and this, you get capital T small t. This and this, capital T small t. This and this, capital T small t. So what do you write? This is going to be the F1 generation, and you write basically all of them are tall. Okay? Phenotype, physical appearance which is there, all of them are tall. What is the genotype? Genotype is all of them are capital T small t. Why they are tall? Because the capital T is dominant over the small t, right? Now you do F2 generation. How do you do F2 generation? Basically over here, let me make Punnett square. So in Punnett square, you just have to write 2 parents. So one is, I am crossing between what 2 parents? I am crossing between capital T small t and capital T small t. Basically I am doing what? Selfing. So how do you write it? One parent you write it over here, capital T small t. Another parent you write over here, capital T small t. When this and this fuses, you get capital T, capital T. When this and this, you get capital T small t. When this and this, again capital T small t. When this and this, you get small t small t. What exactly happens over here? Capital T, this is going to be tall. What about this? This is also going to be tall because you have a dominant allele. This is also going to be tall. So basically out of 4 of them, 3 are tall. So if at all, I have to write the phenotypic ratio, physical appearance ratio. How do I write? 3 of them are tall. So 3 bracket tall, colon, 1 of them is going to be dwarf. So phenotypic ratio is what? 3:1. Okay, ma'am, what is the genotypic ratio? Genotypic ratio, my dear children, if at all you see, 1 of them is capital T, right? Only 1. 2 of them which is there, it is going to be capital T small t. So capital T small t. And 1 of them is going to be small t small t. So write like that and you have got the answer. So this is how you are going to show Please make sure you write all the ratios properly. Mention what does 3 stand for, what does 1 stand for. It's 3 of them tall, 1 of them dwarf. Done? Done. You can get question related to this. As in basically either dihybrid cross or monohybrid cross, or basically incomplete dominance and things like that. Now categorize following as either autosomal or sex-linked disorders. So in sex-linked, in your NCERT you just have 2, and that is hemophilia and color blindness. So this one goes like sex-linked. This one, this one, this one. Thalassemia, phenylketonuria, myotonic dystrophy. All of them are autosomal. Now, in your NCERT, the sex-linked, autosomal, everything is recessive. Okay. Only one particular diagram has been given in your NCERT in the pedigree chart, and that example is of myotonic dystrophy. This is autosomal for sure, but it is autosomal dominant disorder. This is the only one which comes under autosomal dominant disorder. In your NCERT, one more thing is given, cystic fibrosis. They have not explained it, but you should know it is also autosomal recessive. So only one dominant disorder is there, and that too autosomal dominant, which is myotonic dystrophy. Okay, done. Now what conclusions will you arrive after careful observation of the below given diagram? So below given diagram has something like this: 1, 2, 3, 4, 5, 6. 6th position you have valin, you have GUG over here. Now valin which is there at this particular position, you need to understand that basically here point mutation has taken place. You just have to explain about the sickle cell anemia. Say that point mutation took place, you had GAG, that code for glutamic acid, but now it is replaced by GUG, which is coding for valine. Explain that, and that's all your answer about. From the diagram, we conclude that a single base substitution in the mRNA codon has occurred. The codon becomes GUG, which codes for valine. So in the beta globin chain, glutamic acid at the 6th position is replaced by valine. Just explain that. Got it? Yes. Now what? Identify the disorder by the following symptoms. Individuals with overall masculine development with gynecomastia. Overall masculine development is there. Overall masculine development. Okay. But they have gynecomastia, as in basically breast development. This is seen in the case of Klinefelter syndrome. So you have to identify the syndrome, and that is Klinefelter syndrome. Okay. Now, schematically represent the incomplete dominance. Incomplete dominance is basically what? It is not completely dominant. So in the case of capital T, small t, we saw that it was completely dominant. That is why here the character was tall. But in incomplete dominance, things are going to be different. How different? Let's see. So over here, you had a flower whose name was snapdragon flower, or you also call as Antherinum majus. Okay, Antherinum majus. So over here, you take the red plant, or red flowers, and basically white flowered plant. You mentioned it as small r. This is red and this is white, and you cross it. Let's see what is going to happen. Let's form the gametes first. These are the gametes. When you cross this and this, you get capital R, small r. This and this, capital R, small r. This and this, capital R, small r. This and this, again capital R, small r. Everywhere you have one capital R which will be dominant over the small r, right? So the feature should be what? Red. In the first generation, it should be red. But what do you think is the color? The color is actually, all of them are pink in color. Oh my God, why is that happening? Because capital R is incompletely dominant over the small r. This is an exception to Mendel's law. Okay, Mendel gave law of dominance, right? According to it, this was not the case, but this is an exception. Again, okay, they were shocked, so they crossed it once more. So capital R, small r, capital R, small r. Gametes are going to be capital R, small r, capital R, small r. Let's cross and see. This and this fuses, you get capital R, capital R. It's going to be red. Can I write red? Let's write red. This and this fuses, you get capital R, small r. What is this now? This is going to be again pink in color. Why? It's because it's incomplete dominant. Okay, when this and this was crossed, again capital R, small r, again pink in color. When this and this small r came in, and that is what? White in color. That is white in color. So what is the phenotypic ratio? Until now it was 3:1. All of them were red, right? Usually it should have been red, but now it is not like that. So phenotypic ratio, my dear children, is what? Is basically 1 red is to 2 pink is to 1 white. So 1 is to 2 is to 1. What about the genotypic ratio? Genotypic ratio is also like that. 1 capital R, 2 capital R, small r, and 1 small r, small r. Got it? That is going to be the genotypic ratio. So genotypic and phenotypic ratio are going to be exactly the same over here in the case of incomplete dominance. Done. Okay. Please note that they do not follow law of dominance. They do not follow law of dominance over here. Similarly, in the case of single gene, one gene inheritance, what do they follow? They follow law of dominance and law of segregation. Law of segregation means when you form a gamete, only one gene is going to go. Okay, only one gene or one allele is going to go into a gamete, not both of them. So yes. Now, the next question which is there, just identify the genetic disorders. Okay, we have to identify. Sex-linked recessive disorder occurring in about 8% of males and only about 0.4% of females. 8% and 0.4, it's not even 4, 0.4% of females. Leading to defect in the cone of the eye. So listen, leading to defect in the cone of the eye, that means it has to be color blindness. It has to be color blindness. Why do you think it's the males who are affected more? Because males just have one X chromosome. Right, they have another Y chromosome. In the case of females, you have two X chromosome. So if one is affected, other one is there to overshadow the effect, because it's a recessive disorder. In a female to have this, the female should be having both the genes being affected. So since it is a recessive disorder, even if one chromosome is affected, other chromosome is there, not there to overshadow, it will be showing up the disease. You get it? That is why males, they are generally affected. In the case of females, they can be carriers. They will be carriers, they will not have the disease, because there is one to overshadow the effect. So the first disease that we have is what? Color blindness. Now what do we have? Sex-linked recessive disorder affecting a single protein involved in the clotting of blood. Clotting of blood means hemophilia. It's a blood disorder, but it is sex-linked blood disorder. Okay, next. Inborn error of metabolism, autosomal recessive trait, accumulation of phenylalanine. Phenylalanine getting accumulated in phenylketonuria. And phenylketonuria is again an autosomal recessive disorder. I told you, only two sex-linked disorders are there. That is color blindness and hemophilia. Rest apart from that, everything. Let me take sickle cell anemia or thalassemia or basically phenylketonuria. All of them are autosomal recessive. Over here, your answer is phenylketonuria. Please know the symptoms of all of it. Okay, there will be mental retardation. Apart from that, in the urine, it will be a foul smell, because this particular thing which is being accumulated, it's not absorbed by the kidneys, it is being moved out, which is why there is a smell. Okay, now autosome linked recessive blood disease, substitution of amino acid in beta globin chain of hemoglobin. Autosome linked recessive blood disease. Substitution of amino acid in beta globin chain. Substitution word came, that means it is sickle cell anemia. Autosome recessive blood disease, again, reduced rate of synthesis, reduced, that means quantity, that means it is going to be thalassemia. Got it? Yes, ma'am. Okay. Next, represent schematically the result of two gene inheritance. Now, how many questions do we have over here? We have, okay, we are going to complete this. Okay, let's do. Two gene inheritance of seed shape and seed color. This is very easy. Okay, if you understand, try to understand, it's going to be super easy. I'm talking about dihybrid cross. That is why two gene inheritance, right? Dihybrid means I am going to talk about two characters at once. So seed shape and color. Similarly, shape and color. Let's talk about shape first. Round. Here it is going to be wrinkled. Just think of tall and dwarf, just like that. Similarly over here, it is yellow. Here it is going to be green. Okay, how do you show that? Capital R, capital R, capital Y, capital Y. Similarly, small r, small r, small y, small y. Which one is dominant? You know this one is dominant, right? Now, how do you form the gametes? Listen, this is important. This is where most of you do not understand things. So you have capital R over here, capital Y over here. When you make the gene from a pair of chromosome, only one will come. Sorry, when you make a gamete from a pair of chromosome, only one is going to come. One allele is going to come. So either this or either this. Whatever you do, it will be capital R from here. Here also, whatever you do, it will be capital Y only. Right? Similarly, here, whatever you do, one will be small r from one and one will be small y. Agreed? Now, when both of them come together, it will be capital R small r, capital Y, small y. Now you have to use your imagination. It looks like this. Capital R, small r, one chromosome came from here, one chromosome came from the other parent. Similarly, capital Y, small y. This is how it looks. Which is why all the plants are going to be what? Round and yellow in nature. They are going to be round and yellow. Why round and yellow? Because over here you have one to dominate, here also you have other one to dominate. Right? Now, when you do selfing, in the first generation you only saw the parental character, first dominant parental character, that is round and yellow. So they did selfing. In selfing, what is going to happen? Let's do the selfing and see. Can I make it here itself? No. I would need another one. So listen. I have now round and yellow like this. Similarly, round and yellow like this. Now, you have to use your imagination. It goes like this, capital R, small r, and capital Y, small y. Listen, now when I form the gametes, from here one can come, it can be capital R, from here one can come, it will be capital Y, suppose. From here one can come capital R, from here one can come small y, suppose. From here one can come small r, from here one can come capital Y, okay? From here one can come, and from here one can come. Got it? These are going to be the 4 gametes up there. These are going to be the 4 different types of gametes. Why do you think they formed? Because of law of independent assortment. In dihybrid cross, you have law of independent assortment, which says that whenever you are forming the gametes, the alleles of one particular character, that is shape, will be independently assorting from the alleles of another particular color. So does capital R say I will only go with capital Y? No, there is nothing like that. Which is why in future you will be able to see non-parental character. So you cross round and yellow and wrinkled and green, but now you will see round and green, which was not there in the parental. Why? Because of law of independent assortment. Please know. Please know, this is very, very important. They ask you, in Mendel's experiment, how did he show non-parental character? Because law of independent assortment was being formed over there. Okay. In T.H. Morgan, how was he able to say non-parental character? Because crossing over between the genes was being seen. That is totally different. The linked genes will have less recombination. The loosely linked genes will have more recombination. That is what he said. This is different. That is different. Please know about that. But both of them are going to lead to what? Non-parental characters. Now, you have formed the gametes. Write the gametes on this side, because the same parent will produce, this parent will also produce these type of gametes. Now you cross them and it looks something like this. Where is it? It will be looking something like this, right? So when you cross capital R, it will be capital R, capital Y, it will come like this. If at all you see, there are 4 into 4, 16 progenies will be produced. How many? 16 progenies. Now this is something that you have to remember for sure. And the ratio of this, phenotypic ratio, is what is there. Shall I remove this? Is it going to be a problem for you? Okay, I'll remove this. So it goes like this, okay? Where am I? Oh my God. 9:3:3:1. 9 of them are going to be round and yellow. That is a dominant trait. One of them is going to show the recessive trait, which is what? Wrinkled and green. Wrinkled and green. Now 3 and 3 are over here. What are they going to be? If this is round and yellow, this is going to be round and green. It's going to be ulta, round and green. If this is wrinkled and green, it is going to be wrinkled and yellow. Got it? Wrinkled and yellow. So this character, did you see any time until now? No. So these 3 plus 3, 6 characters are non-parental thing. They are non-parental progenies. If at all in a question it comes that 64 progenies are produced. I'm telling, I've repeated this question so many times, but telling you now also. If at all, they're asking you 64 progenies are produced. Out of them, how many of them are going to show non-parental character according to dihybrid cross experiment? So 64 are there in general. Here, out of 16, how many of them are non-parental? 6. So I'll write 6 by 16 into whatever number is given. If I do the calculation, it comes out to be 24. So 24 out of 64 will be what? Non-parental. Similarly, you should be able to do the calculation. Okay? Done. Perfect. Now, Mendelian disorders may be autosomal or sex-linked, dominant or recessive, homozygous or heterozygous. Pedigree symbols are used to analyze these disorders in human population. Mention the genotypes for the following pedigree symbols. Okay. So, you may get a question based on pedigree analysis as well. Make sure you solve it. Autosomal dominant disorder is this. And I am saying that this particular person, that is a man, he is being affected. Right? Because the color is different. Color is like that. So this person has to be affected. I'm talking about autosome and my genes are like this, capital A, small a. What should be my genetic combination over here? It should be either this or it can be like this. Because you have a dominant allele, the dominant allele will show the effect and that person will be having the disease. Here both the alleles are dominant, which is why again the person will be having the disease. So it can be like this or this, then only the person will be affected. See, you have to check over here and here. Similarly, autosomal recessive. Autosomal recessive disorder is what we're talking about, and here there is a lady, circle means lady, right, and she is not having the disease. That means it should not be in recessive condition. Listen, if at all it is in this condition, then you will get someone like this. They will have the disease. Why? Because it is an autosomal recessive disorder. Now you have to make sure that that person does not have the disease, because over here it is not being colored. So you have to do the ulta of small a. That will be either this or this. You should be having one dominant allele coming up. So it will be just similar to this. Got it? Getting it? Next thing. Sex-linked dominant. Sex-linked dominant means, my dear children, over here, suppose you have XA and XA. This is a dominant one, this is a recessive one, because you have small a over here, right? So when I say this is a guy has two chromosomes X and Y. And when I am saying that is X-linked dominant disorder, but this particular person does not have it, that means that this particular will be having a recessive allele. If at all the person had something like this, then he would have been affected. But here you are saying this person does not have this. Right, so you have to make sure it's the recessive allele that you have to put. So over here that is being mentioned. Similarly, sex-linked recessive disorder. Now, sex-linked recessive disorder is given and both the male and the female are affected. If it is recessive disorder and if you want to make both the male and female be affected, you have to put the recessive allele in the case of male because only one X is there. In the case of females, you have to make sure it's not just on one chromosome but on both the chromosome. Then only she will be showing. Otherwise, in her, the dominant allele will be taking up over that. So both the alleles have to be affected for her to be affected. Get it? So this is what you have to write. Is there a confusion? If there is a confusion in this question, go back once more and go through it. You will be able to do it. Got it? And this is very, very important. Very, very, very important. Cool? Yes. Next thing. Now, observe the representation of a dihybrid cross and answer the questions. This is the dihybrid cross experiment that we just did. Phenotypic ratio, we know it is what? 9:3:3:1. Do you know the genotypic ratio? It's very simple. 1:2:2:2:1 and then 2:4:2 and then again 1:2:1. So that is the genotypic ratio. Usually it does not come, but no. 1:2:1, 2:4:2, 1:2:1. Okay? Now let's come back over here. Okay. Identify homozygous dominant genotype in the progeny. Homozygous dominant. Homozygous means both the alleles have to be same. Homozygous, and I am saying dominant. So basically this is the condition. You cannot say this one, okay, because why? Both the chromosomes, both the alleles are not homozygous over here. You have to make homozygous, that means both the alleles have to be same. Cool? Next thing. Identify recessive genotype in the progeny. Recessive, if at all you're talking about recessive, it has to be this. It has to be this. Okay, there is no other progeny which is recessive. Recessive condition will always have both the alleles to be similar. Now, if at all you were asked about heterozygous dominant and all, you could have given the answer like this is heterozygous dominant. Why? Because here the alleles are different. Here you were asked about homozygous dominant. So again, yeah. Now, number of parental type of progeny. How many of them are parental type? You know the ratio 9:3:3:1. I told you 9 and 1. This is round and yellow and this is wrinkled and green. 9 plus 1 is equals to what? 10 of them are like parents. How many recombinant type progenies are there? 3 and 3. 6 of them are going to be non-parental, or basically what do you call as recombinants. Got it? So this is the thing that was given, so I thought I'll explain. Fine. Next thing. So here the answer is also given. If at all you want to see, you can check it out. Now, yeah. I think this is going to be, no, second last question of this chapter, okay? This is a hot question. A cross between a normal couple resulted in a son who was hemophilic and a normal daughter. Let's do. A cross between a normal couple. So that means, okay, resulted in what? A son who was hemophilic. Son who is hemophilic. That means you have to color it up. Okay? And a normal daughter. And a normal daughter. Normal daughter is why you're not coloring it up. The daughter was married to a normal man. Daughter is married to a normal man. Okay? To their surprise, the grandson was hemophilic. Now their son, grandson means basically, okay, their daughter's son, grandson. To their surprise, the grandson was having hemophilia. Okay. Represent the cross in the form of pedigree chart. I have already done that. Give the genotype of the daughter and the husband. So if I say that the male is having the hemophilia, that means he is something like this, Baba. Agreed? One gene is going to be affected, that obviously is going to be the X chromosome. X chromosome, do you think they can get from the father? No. X chromosome you get from the mother only. Y chromosome you get from the father. So here, if I am making it like this, you should understand, I should have one normal X chromosome to overshadow the effect. Then only she will not be affected. Here it is mentioned that she is not affected. She could be a carrier. She can be a carrier. So her genotype is like this. Only if the condition is like this, then you get one X chromosome from here, one normal Y chromosome from there. This affected X chromosome would cause disease in the case of the guy. You get it? Use the logic. You never can make it something like this. Otherwise, she should also be having hemophilia. Okay, because this is what? A recessive disorder. So since she has an extra X chromosome that overshadows the effect, which is why she is not growing. Now if at all it's just normal XX, it's normal XX, then how will he get the disease? He's disease, that means he have got one X chromosome at least, right? So yes. So this would be the genotype of the female, and that is a daughter. And you're asked about the son, right? And her husband, daughter and the husband. So husband is normal, it will be just XY. Husband is normal XY and female daughter is like this. Okay. Now write the conclusion you draw of the inheritance pattern. This is what? Sex-linked recessive disorder. And since it is recessive, it will be skipping generation. So over here the parent does not have it, but here the child had it. Here the parent does not have it, it has it. Okay. Now sex-linked recessive disorders, in that case the males are the ones which are mostly affected. So you can write that also. Okay. So I have written everything. Write the conclusion you draw. X-linked recessive, it is through transmitted to carrier females. It mostly occurs in males, and trait can skip a generation because it is recessive. Cool. Now we have one more question, the last question from this chapter, and that is, refer to the representation of a pedigree chart of a particular trait and answer the question that follows. So here is the question. So there is a circle and what do you call a square, and then you have to mention the rest. So which genetic disorder does the pedigree represent? Which genetic disorder does the pedigree represent? So over here you see there is skipping of generation. Here the parent does not have it, but the children have it. Right? Skipping of generation is there, that means it is going to be recessive. Now, is it sex-linked or autosomal? In sex-linked, obviously the males are the one which are mostly affected. In the case of autosomal, it can be male and female. So over here you can clearly see in one generation both male and female that is being affected. So I can say it probably could be autosomal. And let me tell you, in your NCRT, this particular pedigree chart is given, and directly they are saying it's what? Sickle cell anemia. So they have given you two pedigree chart. Please know it. One is myotonic dystrophy, which is autosomal dominant. It will have a diseased person in every single generation. This is autosomal recessive. This will be skipping generation. So as I said, here the parents do not have it, here the children have it. Here the parents do not have it, here the child has it. You get it? So please know that. And that is myotonic dystrophy. It's autosomal dominant. This is recessive. Now, which genetic disorder does the pedigree chart represent? Sickle cell anemia. Mention the cause of the above trait. Just say single mutation is happening out there. Just what we studied about sickle cell anemia. Is caused by point mutation. Due to this mutation, valine replaces glutamic acid at the 6th position of the beta globin chain. Okay. Now write in 3 features of sickle cell anemia. So autosomal recessive, red blood cells become sickle shaped under low oxygen tension, heterozygous. Okay. Suppose, my dear children, there is a heterozygous condition. One person has something like this. This gene is affected, this one is normal. Okay, so what is going to happen? Basically, if suppose two heterozygous people are marrying, two carriers are marrying, they do not know that they are carriers of hemophilia. Listen, if at all I do this cross, it comes out to be something like this. Very, very important. This is same for thalassemia and everything, phenylketonuria, because all of them are autosomal recessive, right? So, two people can be carriers. They might not have the disease, but the child might have. How much percent of the child? Let's see. This and this crosses, you get HBA, HBA. A is a normal one because that's why the person would be normal. They will not have the disease. This and this crosses, you get HBA, HBS. Here, in this particular condition, again, they would be carrier. They have one bad gene, but the other gene is there to overshadow the effect. This and this crosses, you get again HBA, HBS. Again, over here, what is going to happen? What is the thing that is you're going to see? Is that, okay, is that basically again here they are going to be carrier only. They do not have the disease. Now, when lastly this and this crosses, you get HBS and HBS. And when this comes in, when HBH, HBS comes in, that means that person will be having disease because this is a recessive condition. So, out of 4, 1 is affected. So, I can write 1 by 4 is equals to what? 25% of the progeny is going to be affected. Got it? As simple as that. So, please know that 25% chances of a progeny being affected is there. Cool? Cool. And now human health and disease is the next chapter. Over here, I have the first question that is up there. Human immunodeficiency virus replicates in. So, I think we have to be quick because it's already 1 hour 50 minutes, right? So, let's go. Human immunodeficiency virus replicates in B cells, NK cells, helper T cells and PMNL. What do you think is your answer? HIV replicates in actually macrophages first of all. Then later it is going to affect the helper T lymphocytes. This is the major target, but first it goes and infects the macrophage and increases the number. That is why macrophage is known as HIV factory of the cell, and then later it affects the helper T cells, which is the major target as well. But anyway, since macrophage is not been mentioned, you have to go with this. Not B cells, not natural killer cells. PMNL stands for polymorphonucleoleucocyte. You also call them as neutrophils. That is not the answer for this question. Okay. Next thing, the drug which is depressant and slows down the body function is. It is a depressant, okay, it slows down your body function as well. You just have one drug such like that, and that is known as heroin. Okay, it is known as heroin. And do you see heroin in these options? No, that means other name of heroin is given. What is the other name of heroin? Diacetylmorphin. Because you have morphine and heroin in one particular category. When you do acetylation of morphine, that's how you get what do you call heroin. Okay, so it is known as diacetylmorphin. If that name is also not given, you have to go for the other name, that is SMAC. That is SMAC. Okay, so heroin, SMAC, diacetylmorphin, all of it is same, bitter, odorless, colorless, a crystalline substance which is there. Okay, it can be taken using snorting or injection. Yeah? Yes. So please know about that. Next one, and you get it from the latex of the poppy plant. So morphin, I have written diacetylmorphin, or you also call this SMAC. Next thing, symptoms of constipation, abdominal pain and cramps associated with the disease were what? So there is constipation, okay, because of which you have abdominal pain, and there is cramps also. This is seen in which of the diseases? This is your MQP1 question. Amoebiasis, richness, non-stop ovulation, colostrum and diversity. Just fill in the blanks question. The answer is amoebiasis. And who is the causative agent? It is Antamoeba histolytica. It causes amoebic dysentery, or you also call this as amoebiasis. Either way, that is your answer. Okay. The next question that we have is question number 4. Antivenom given against snake poison contains what? Antigens, antigen-antibody complex, antibodies or enzymes. What do you think is the answer for this? Antivenom which you give against snake poison. Suppose a snake came and attacked you. Okay, now the doctor will give antivenom. Why? Why antivenom? Because at that particular point of time, basically the body needs ready-made antibodies. Okay, you cannot wait for your body to produce antibody by itself. Please understand that. You need ready-made antibodies, and that is what you get from antivenom, or basically antitoxin that is given in the case of tetanus injection also. Okay, so please know, it is not antigens, it is not antigen antibody complex, it is just antibodies that is being given. Okay, and not even enzymes. Yeah. Next one, the substrate produced by a cell in viral infection that can protect the other cells from further infection is. So there is a substance that is produced from the cell and it is protecting the other cell from infection. So this cell has been infected from virus. What is it releasing? So that it protects the other cell, it is going to be cytokines, and the name of the cytokine is interferon. Cytokine means some secretions of the cell, okay, and here you have a specific name for that. It is interferon. Always remember, interferon is a famous guy among the virus infected cells. Now there is one more interferon being mentioned in NCERT, and that is alpha interferons. Alpha interferons are given to cancer patients because it is a biological immune response modifier. It's something that is boosting up your immune system. That is the case of cancer, and that is alpha interferons. This is plain interferons, okay? Please remember that. Okay. Now, DASH is a very effective sedative and a painkiller. Sedative. So there are two drugs, as I told you, which is responsible for depression and all, depressant and slowing down of your body. Okay, one is morphine. It is actually sedative. It makes you sleep, and also painkiller, which is why doctors use it on patients who have underwent very bad surgeries. Okay, and after that, as I said, from morphine you get heroin, which is depressant. It slows down your body as well. Here you have been asked about sedative. That means this particular drug makes all people sleep. Okay, sedation, it gives sedation. And also it's a painkiller, it has to be morphine only. Okay, and both of them are opioids. Yes. Now, which of the following pairs correctly matches a disease and a pathogen causing it? Typhoid is caused by Salmonella typhi. Is it? Yes, definitely. This is the first one itself is the correct one. Let's see why others are not. Pneumonia is caused by Haemophilus pneumoniae. Oh, Haemophilus pneumoniae. This also is correct, right? No. Haemophilus pneumoniae is not the correct one. It is Haemophilus influenzae. It is Streptococcus pneumoniae. Two bacteria causes pneumonia, right? So that is why it's wrong. Malaria is caused by Ascaris lumbricoides. Papa, Ascaris lumbricoides is like, I did not cause this. Do not blame me for this. Okay, so malaria is caused by Plasmodium, not by Ascaris lumbricoides. And it causes ascariasis, by the way. Ringworm is caused by Antamoeba histolytica. Okay. Is it? No. Ringworm is not caused by any worm also. It is caused by fungus. Trichophyton, Epidermophyton, Microsporium and things like that. Which is why this is also wrong. So your only answer for the question that you have is A. And by this you understand that all the diseases and the causative agents are important for you to know. Yes. Next. Hemozoin is a dash. Precursor of hemoglobin. Is it? Hemozoin, is it a precursor of hemoglobin? No. Hemozoin is basically what? Basically, the plasmodium which is there, it infects your RBC. Inside that, what do you have? Hemoglobin. This globin is what the plasmodium needs. Okay, but it breaks it down. You get heme plus globin. This heme which is produced is very, very toxic for the plasmodium, which is why it converts into hemozoin. The thing is, this hemozoin, when it is released from the cells, how it is released? Over here you have so many plasmodium also. When they flourish too much, it is going to burst open, and that's how hemozoin is going to be released. This hemozoin will be causing chills and fevers to us because it is toxic for us instead. You get it? So over here, it is precursor of hemoglobin, wrong. Toxin released from Streptococcus infected cells. Streptococcus, papa, it causes pneumonia, so wrong. Toxin released from Plasmodium infected cell? Yes, exactly. Toxin released from Haemophilus infected cells? No. So your only answer for this question is going to be C. And over here, I have explained that again in the life cycle of Plasmodium. I will be talking everything in detail. Okay, so Plasmodium enters in our body in sporozoite stage, exits from our body in gametocyte stage. No sexual reproduction happens in our body. Okay, sexual reproduction happens in the mosquito's body. Okay, so please remember all those things. Sporozoite is the place where it enters through saliva, and basically it exists in the gametocyte stage. No sexual reproduction happens in human body. Only asexual reproduction happens in the human body. It affects your liver first, then your RBCs. Cool? Cool. Yes. Fine. Now, next question. Which of the following is an autoimmune disease? Autoimmune disease means your cells do not know that, okay, these are also my cells, I should not be killing them. So that is autoimmune disorders. Okay, whenever your cells, your body is not able to recognize what is self and what is not self and keep on killing your own cells also, that leads to autoimmune disorders. And over here in your NCERT you just have one example, and that is rheumatoid arthritis. Rheumatoid arthritis is the only example for autoimmune disease in your NCERT. Yeah? Yeah. Cool. Next question. The site where lymphocytes interact with antigens and proliferate to become effector cells are. The site where lymphocytes interact with antigens and proliferate to become effector cells. Effector cells means actually do their function of killing the antigens. So whenever lymphocytes are going to kill the antigens, that is going to happen only in the secondary lymphoid organs, not in the primary lymphoid organs. In the primary lymphoid organs, they become antigen sensitive. Remember the term, antigen sensitive. They know that I have to kill an antigen and things like that, but they don't kill it. They kill only in the secondary lymphoid organs, and that is when we say that they have become what? Effector cells. So where do they become effector cells? You have to spot the secondary lymphoid organs. Thymus is a primary lymphoid organ, which is why it is wrong. Bone marrow and thymus, again wrong. Both of them are primary lymphoid organs. Peyer's patch and tonsils, both of them are secondary lymphoid organs, which way this could be the answer. Bone marrow and spleen, wrong. Listen, over here, lymph nodes, spleen, Peyer's patch, tonsils, all of them are secondary lymphoid organs. But here they are paired with primary lymphoid organs, which is why they are wrong. Only one option is correct, and that is C. Cool? Cool. Next. Injection of antitoxin in tetanus confers which type of immunization? Listen, over here, antitoxin, I told you, you are giving ready-made antibodies. You're not waiting for your body to produce antibodies. So this type of immunization is passive immunization. When your body produces antibodies by itself, you call it active immunization. That happens in vaccination. That happens when you get infected with something naturally. Your body produces antibodies, right? That is active immunization. Over here, you are directly giving ready-made antibodies, which is why it is passive form of immunization. So your answer for this question is going to be B. Cool? Cool, ma'am. Okay. Tobacco consumption is known to stimulate secretion of adrenaline and noradrenaline. The component causing this could be, in tobacco, what is that something that increases your blood pressure? It is nicotine. Nicotine, my dear children, is responsible for basically stimulating the secretion of adrenaline and noradrenaline. Now, when these hormones are secreted, your blood pressure is going to go high. Always remember, if there's a question coming in, which is the drug responsible for increase of blood pressure? It is nicotine, and you get it from tobacco consumption. Okay? Okay. Next one, charas and ganja are the drugs which affect. Charas, ganja, hashish, marijuana, la 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 la. Everything you get from cannabinoids, and cannabinoids affect your cardiovascular system. The only ones which are affecting cardiovascular system. Rest, opioids as well as coca alkaloids, both of them affect the CNS, okay, but not the cardiovascular system. So this guy affects the cardiovascular system. These are cannabinoids. They are cannabinoids. Okay. Next one, a chemical structure is given below. Identify its plant source. So it looks like an O, which is why I'll say it's opioid. And in opioid, you have two, morphine as well as heroin. Which one is this? It is actually morphine. And you get it from Papaver somniferum. You get it from Papaver somniferum. So basically, they're not even asking you what is the structure. They're asking you from where do you get it. So high level of things, you need to an understanding of that, right? So C. Fine. Next, the disease chikungunya is transmitted by. How do you think chikungunya is going to be transmitted? Okay. It is what? Sometimes I forget that I'm a teacher, and sometimes, most of the times, guys, first of all, let me tell you, I'm your teacher, but I'm also your friend. So yes, please consider me like that also. So your answer for this question is going to be Aedes mosquito. Okay, it is going to be Aedes mosquito. It is the one that causes chikungunya as well as dengue. You have Anopheles mosquito that causes malaria, and you have Culex mosquito that causes filariasis. Okay, so B is your answer for this question. Cool. Next one. Identify the enzymes which deviates from the central dogma of genetics during HIV infection. Okay, what is central dogma of genetics? The DNA is going to form the RNA. The RNA is going to help in the formation of protein, and DNA forms itself. That is known as replication. This is central dogma. Now you have been asked, there is an enzyme which deviates from this. That deviates from this. What is that enzyme? It is reverse transcriptase. Why? Because in the case of HIV infection, the RNA of the HIV virus reverse transcribes to form the DNA, and that has been done using the enzyme reverse transcriptase, because our cell has DNA and it wants to incorporate it into our DNA. That is why it does reverse transcription, and that is opposite of the central dogma which was given by Watson. And now we come into the next chapter, that is human health and diseases. So by the way, have you reached until here? I hope you have. Well, my dear children, I hope you're understanding. I might be going in a different pace today because I feel that these are more of like a repeated concept out there. But if at all you have any confusions, please ask me during live sessions. I am really there for you. I do not move ahead without you understanding things, and you know that, right? And today I'm a little bit tired also. So just finish the live off and then had the recording. So yes. Yeah, but be there on Wednesday. I have a live session out there. You can be there and ask me doubts. You can note the doubts and ask me then if you have any. And if at all everything is fine, please definitely do not forget to hit the subscribe button and click on the like button as well. I'll just take a quick break and I'll come back. Okay? The first question that we have over here is, name the causative organism of the following diseases. Filariasis, my dear children, is caused by what? The filarial worm, which is known as Wuchereria bancrofti or Wuchereria malai. Ringworm is caused by, it's not caused by worm, it's caused by fungus. So two fungus are fighting, epidermophyton and trichophyton. Okay, epidermophyton and trichophyton, they are fighting, and also they are fighting for one very important thing, a small spore that is known as microspore. So microsporum, epidermophyton, trichophyton, they come under. They are the ones that causes ringworm disease. Now malaria we all know, Plasmodium falciparum, Plasmodium vivax, Plasmodium malariae. These are the three things, but the most malignant form of malaria is caused by Plasmodium falciparum. So over here I have the answer for all of your questions. Please go through it, it's super important. Over here, please do not do any conjusi, please write epidermophyton, and also what do you have? Microsporum. Cool? Yes. Perfect. Now, mention the causative organisms for the following disease: Escherichiasis, typhoid and filariasis. Escherichiasis, my dear children, is the one that is caused by what? Ascaris lumbricoides, that is a roundworm. Typhoid, which is there, it is caused by what? Salmonella typhi, which is what? A bacteria. Now filariasis is caused by what? Wuchereria bancrofti. We just studied it. The reason I am showing you these questions again and again is that there will be one question coming from the causative organism of this particular disease. Three diseases will be given. Any three diseases, you should be able to know it. Okay, so please go through it. Now, name the causative. See how many times the question was repeated, model question papers. Name the causative organism of the following disease. Escherichiasis, we just received Ascaris lumbricoids. Ringworm, we know Microsporum, trichophyton, epidermophyton. And then typhoid, it is Salmonella typhi. Okay. Now name the group of genes that have been identified in normal cells that could lead to cancer. What are these these genes which are identified in normal cell? These are known as proto-oncogenes. Proto-oncogenes means as in they are not problematic, but if at all they have any mutations around it, basically they can turn the whole cell into a cancerous cell. These are known as proto-oncogenes out there. Now, which techniques are useful in detecting cancers of internal organs? So you have computed tomography, CT scan, you have X-rays, that is radiography. Okay, radiography, you have computer tomography, and where you are again using X-rays only, but in computer tomography you get three-dimensional image of the internal organs. Then you have MRI, magnetic resonance imaging, where you use magnetic fields and non-ionizing radiation for getting an idea about the internal living tissues out there. So these three things you can mention. Okay. Got it? Now, why are cancer patients often given alpha interferons as the part of treatment? Listen, alpha interferons is a biological response modifier. That means, basically in the case of cancer patient, the immune system is very weak. So to make it a little bit hyped up, to increase it, motivate it, what do we give? Alpha interferons. They are modifiers, they are response modifiers. Okay, so they increase the immunity a bit more. That is why we give it. So your answer for all the questions are given. Proto-oncogenes is a gene that has been identified in normal cell that can lead to cancer later on. Now, which techniques are useful in detecting cancers of internal organs? Biopsy and so basically these two, and also your radiography. That means basically just normal X-rays. So biopsy and histopathology helps in examination of tissues which are there. But yes, if at all you just want to find the internal organs, you can have these things. Please know that. Now, why are cancer patients often given alpha interferons? Because they are biological response modifiers. They activate the immune system out there. Okay. Now name the drugs and the source, and the nature is given in the table. Okay, let's go ahead. So, name of the drug. Source plant is Papaver somniferum. Okay, that is opium poppy. And the nature is a depressant. So, a depressant that you get from Papaver somniferum is what? Think about it. You have two drugs, morphine and heroin. Morphine is a sedative and a painkiller. Heroin is a depressant and slows down the body. So, your answer for this question is going to be heroin. Okay. Now, cannabinoids is the name of the drug. Source plant is what? Source plant of cannabinoids is what? Cannabis sativa. Cannabis sativa. Where do you get it? Sorry. What does it do? It is affecting your cardiovascular system. Remember, cannabinoids is a drug that is abused by sports people. Done? Done. Next thing which is there, name of the drug is cocaine. The source plant that we have over here is Erythroxylum coca. Is what? Erythroxylum coca. What is the nature of Erythroxylum coca? Sorry, what is the nature of this particular drug? It affects the dopamine transmission. It actually affects your CNS, central nervous system. In the central nervous system, it affects the transport of dopamine. So please know about that as well. Okay? So yes. So heroin, cannabis sativa, effects on the central nervous system is what you need to answer. Cool? Cool. Next thing. Describe any three properties of cancerous cells. Cancerous cells, my dear children, first thing, they show uncontrollable growth. They divide, it goes on. And they have loss of contact inhibition. What do you mean by that? Basically, if the cells, when they come in contact, they are not going to divide any further. Okay? Generally, the normal cells are there, but these cells are not like that. These cells, even though they come in contact, they will keep on dividing. That is loss of contact inhibition. The third feature is metastasis. What do you mean by that? The cells which are there, they can travel throughout the body and can cause trouble at each and every place, at different parts of the body. That is known as metastasis. So these are the three properties of cancer cells which you have to remember. Done? Done. Okay, my dear children, let's go ahead with the next chapter, that is human health and diseases. And the first question that you have over here is, what are lymphoid organs? Write the functions of the following in response to immune system of our body. Now this question is very simple, very straightforward, but you should know the logic and some keywords out there. So always remember, the lymphoid organs are the ones where your lymphocytes are going to develop, they are going to become mature, and also they start doing dishum with the antigens or with the pathogens out there. So their major function is to kill the pathogens, right? So the exact thing is happening over here, but in a structured way. So there are two types of lymphoid organs. First one is the primary lymphoid organ. Okay, and the second one would be the secondary lymphoid organs. In primary, you just have two of them. Always remember, only two. One is your bone marrow, and the other one which is there is going to be the thymus. Now, bone marrow is something that is found inside your bone. Inside each of your bone, my dear children, you would be seeing something. There is red marrow and there is yellow marrow. Yellow marrow contains the cell which is responsible for formation of fat. Apart from that, the red marrow contains some stem cells. Some cells which has the potency to become different kind of cells. That is what you call as stem cells. Now, over here, they are hematopoietic stem cell, which means that they are going to form different forms of the blood cells. Not any other cell, just the different forms of blood cells. You know, there is RBC, there is WBC, there is platelets and all of it. So all these kind of cells are being formed inside the red marrow of your bone marrow. Now when I say red blood cells, white blood cells are being formed, you should know that under white blood cells comes what? Your lymphocytes. That is your B and T lymphocytes. Okay, under white bloods comes your B and T lymphocytes. Apart from that, you have neutrophils, you have so many other cells, macrophages, everything out there. Now, so we got an understanding that, okay ma'am, I got an understanding that B and T lymphocytes, which are a major cells out there, they are definitely formed inside the bone marrow. So are they just formed over here? Yes, they are just formed over here, but also you need to understand they mature as well. They become mature over here. But here there is something that you have to note. The T lymphocytes which is there, it goes to the thymus. Now thymus is somewhere over here, that is behind our sternum. So sternum is a place where all your ribs are going to be joined. Okay, so behind your sternum, or what you call as chest bone, you have a small little thing which you call as thymus. So the T lymphocytes which is there, it comes over here into the thymus and becomes mature over here. It is formed there, but it comes over here for maturing. B lymphocytes stay there and matures there itself. Now, one more very important thing that you need to know about primary lymphocyte is that primary lymphoid organ is that this is the place that the lymphocytes which is there, it becomes antigen sensitive. What do you mean by that? Antigen sensitive means they know that they would be having some some cells which they have to fight against. Okay, they are becoming antigen sensitive. They know that these are the pathogens, these are enemies, and I have to kill them. But do they start killing them over here itself? The answer is no. They just become sensitive. They do not start their work over here. So then, ma'am, where do they actually meet the pathogens and kill them? It is going to be the secondary lymphoid organs, where these cells, my dear children, are going to become what? The effector cells. Effector cells means that they are going to show their effect of killing the antigens. So always remember, whenever the term antigen sensitive comes, it's in the primary lymphoid organs. Whenever the term comes that they become effector organs, where do they become effector organs? In the secondary lymphoid organs. Got it? Now, under the secondary lymphoid organs, you have multiple things. For example, the most famous one that is there is lymph node. Is what? Lymph node. So basically, just like how we have our arteries, veins and capillaries, that is a blood vessel out there, we have lymphatic vessels. We have lymphatic vessels, which is responsible for carrying anything extra which is there in between the cells back and giving it back to the heart. That is the responsibility of the, the lymphatic vessels. The lymphatic vessels which are there, they would be having some places like this. Okay, some places like this, which is kind of enlarged. Why do you think they have places like this? Well, in these places, you have so many lymphocytes already standing up there. Why do you think that? Because whenever the lymphatic vessel is basically carrying any extra fluid between the cells, and suppose there is some pathogen in between those particular cells, they also come in through the lymphatic vessel out there. Now there should be someone to trap them. Who is that? It is going to be the lymphocytes which are there in the lymph nodes. So don't you think this is the place that they are going to show their effect, that they become effector cells? They are going to do dishum. The answer is yes. So you have lymph nodes or lymphatic vessels throughout your body, and you might have seen when you go to doctor checks over here and touches and see that whether it's swollen or not. What is he or she checking? He or she is checking basically, are your lymph nodes swollen? If at all it's swollen, that means there's an infection going on and probably your body is reacting to it. You got it? Okay, fine. So that is one very important thing. Apart from that, you have spleen. Now, spleen is just beside our stomach, my dear children. Spleen is popularly known as the graveyard of RBC. This is where the old and worn out RBC is going to go and die. Now, when I say that it has blood supply to it, and when I say that RBC are coming, I can also imagine that there will be some pathogens also which are there in our blood that will be coming to the spleen. Now here, my dear children, within our spleen, there are soldiers lined up. Soldiers as in lymphocytes lined up there. So whenever some pathogen comes in, those pathogens are being killed, or those antigens are being killed. So can I say again that here is a place where they become effector cells? Absolutely yes, I can say that. So that is clear to you. Now we have the next thing, that is MALT. MALT stands for mucosa associated lymphoid tissue. What do you mean by that? So basically our intestinal tract overall, the innermost lining of the intestinal tract, which you cannot see, which is innermost, my dear children, which lines up everything, it is known as mucosa layer. It is known as mucosa layer. Now this mucosa layer can have a group of lymphocytes around there at different locations. For example, we have that in our tonsils. So tonsils that you see, my dear children, is a part of mucosa associated lymphoid tissue, and here, my dear children, you would be having so many lymphocytes, which is why whenever something comes up, we have throat pain and things like that, probably because our body is reacting to it. Okay, our body is fighting for us. Similarly, so under MALT comes tonsils. Apart from that, there is one more thing, that is Peyer's patch. Have you heard about it? Peyer's patch. So this is again found in the mucosa layer, but found where? Not over here. It is found towards the intestinal region. So it is found towards the intestinal region, which is having high number of lymphocytes out there. Apart from it, you also have appendix, which is there. Always remember, mucosa associated lymphoid tissue occupies around 50% of the whole lymphoid organs. So 50% of the whole lymphoid organs, basically secondary lymphoid organs, are coming under MALT, mucosa associated lymphoid tissue. Got it? So now, how do you frame this answer? First of all, what are lymphoid organs? Organs where origin, maturation and proliferation. Origin as in they are formed, maturation as in they are going to mature, they are going to differentiate, they will be knowing that what I have to do in future. And proliferate as in increase the number of the lymphocytes occur over here in the lymphoid organs. Now primary organs such as bone marrow and thymus produce antigen sensitive lymphocytes, okay, which migrate to secondary organs such as spleen, lymph nodes to become what? Effector cells whenever an antigen comes into an action. Now bone marrow which is there, it is a main site where all blood cells including lymphocytes are formed. Agreed, okay. Provides microenvironment for B lymphocyte maturation. I told you B lymphocytes stay there and mature their self, correct. Thymus, primary lymphoid organ for T lymphocyte maturation. Here the T lymphocytes mature, which is why you have T name over here, because it is maturing in the thymus. Now immature T cells become antigen sensitive T lymphocytes and later differentiated into what? Effector cells. Later, not here, it will become effector cells in the secondary lymphoid organs. Also remember, the size of the thymus is going to reduce as we are gonna grow up, okay, and the thymus would be somewhere over here. Now, we are asked for the next thing, that is spleen as well as lymph nodes. Secondary lymphoid organ that filters blood and traps blood-borne antigen, contains lymphocytes and phagocytes that help activate antigen-sensitive cells into effector cells. Or basically, I can just say that they become effector cells over here and start killing the antigens. Lymph nodes, same thing, that is there. Trap antigens from the lymph entering the body. Lymph as in the extra fluid, interstitial, in between the cells, whatever fluid is there, that is known as lymph fluid. Okay, so that runs in the lymphatic vessels. So provide site for lymphocyte antigen interaction. Again, they become effector cells over here. Now this is the picture that is given of a spleen. So my dear children, if at all you see, this is your stomach and just beside it you have this spleen right over here. Similarly, if at all you see this, these are your lymphatic vessels. The green color thing is a lymphatic vessel, and in between that you can see small round things. These are the lymph nodes which are highly supplied with lymphocytes. Apart from it, what do you have? MALT, mucosa associated lymphoid tissue, which is the one that contributes majorly. It contributes 50%. So that is a very big number. Got it, ma'am. Perfect. Now we go to the next question, that is, give the schematic representation of replication of retrovirus. Now you might be asked to draw this whole thing, okay, you might be asked to fill in some steps over here. That means that your concept should be superbly clear over here. So please listen to me whatever I am trying to tell you. So first thing first, you have your retrovirus, that is your HIV. Do you know the genome of HIV? What is HIV genome made up of? For example, in our case we have a double-stranded DNA, right? But what about HIV, my dear children? It has single-stranded RNA. What does it have? Single-stranded RNA. How many of them are there? Ma'am, 2. So ma'am, 2 of them do not come together to form complementary double-stranded RNA? No, they remain single-stranded only. So always remember, you just have to write ssRNA. That means single-stranded RNA. Okay. Apart from it, it has also the protein code. So since there are chances that you might forget, I am just writing it. It has single-stranded RNA. This is what? The genome. Okay, perfect. Now, it also have the protein covering, that is the virus protein coat is there. Agreed. Apart from that, there will be some proteins also, as in some enzymes also within this. Now, the virus which is there, it is going to affect the normal cells. Always remember, HIV virus, first of all, will be going into macrophages. Its actual target is T helper lymphocytes. Its actual target is T helper lymphocytes. Why? Because T helper lymphocytes is something that is a mediator in our immune system. It's a mediator in our immune system, right? So if at all that guy is damaged, our immune system will go very low. And that is the exact thing that the virus wants. Okay. So it is the T helper cell which is the target, but initially if at all it wants to increase its number, first of all it will go and attack the macrophages. What phages? Macrophages. Those are also different type of cells. So it came and attacked the macrophage. What does it do over here? When it comes and attacks, it will be transferring its single-stranded RNA along with some very important proteins which are there inside into the cell. This is our cell. This is our macrophage. Now, the target is to utilize our machinery. How can it utilize our machinery? It has to get inside the nucleus and basically be part of our DNA. Because whatever is there in our DNA, that is going, whatever information is there, that is going to be transcribed, translated, and the proteins and everything are going to be made, right? So if at all it wants to use our machinery, the first thing that it would have to do is, this is an RNA, it has to transform itself into DNA. Ma'am, is that possible? RNA to DNA? I have heard about DNA to RNA. That is transcription, right? DNA transcribes into RNA. That is transcription. Can't we do the opposite of it? We can. Only if if at all we have an enzyme for that, and that is known as reverse transcriptase. Okay, it reverses the transcription process, which is why it is known as reverse transcriptase. Did the virus have it? Yes, the virus brought this particular enzyme also. I told you there are some proteins inside, right? So this is one such thing. So what does it do? It does reverse transcription with the help of very important enzyme, that is reverse transcriptase. And remember, this is against what is followed in the central dogma. The central dogma says from the DNA you get the RNA, from the RNA you get the protein, and basically DNA to DNA is known as replication. This is what is central dogma. But now since ulta is happening, I'll say this is against the central dogma, and it is because of the enzyme reverse transcriptase of the of the virus up there. Now you have got the DNA. What does the DNA do? DNA will be slowly getting inside. The viral DNA incorporates into what's the host genome out there, that is our genome. It gets inside again with the use of some enzymes only. You call this as integrase. It's not given in NCERT, so you did not learn it. Okay, so it now incorporated over here. After incorporating, it is going to use our machinery, and what does it do, Baba? It will form new viral RNA. It has to form RNA again, right? It has to come back in this form, which is why whatever was there initially will be formed here. So new viral RNA is being produced with the help of information given by the DNA. Now new viral proteins are also being produced, and finally you have the RNA, you have the protein covering, and they would be moving out. So please remember these steps. New viral RNA is produced by the infected cell, and new viruses are produced again. Got it? This is the basic funda. You should be knowing each and everything about it, otherwise you might lose your marks. Now I'll be showing you similar question that has come this year, okay, and it goes like this. Just a second. Okay, let's solve this. So what you have to do is fill in the missing data in boxes 1 and 2, that is this and this. Give an example for retrovirus. Retrovirus is basically HIV. So example for retrovirus is what? HIV, human immunodeficiency virus. Now name the disease caused by the retrovirus. It is AIDS. What do you mean by AIDS? Acquired immunodeficiency syndrome. That means you are acquired it during your lifetime. You got this disease during your lifetime, and it makes your immune system deficient. That is why acquired immunodeficient, okay, syndrome, that is there. Syndrome means group of symptoms will be there. Since you're immunodeficient, what is going to happen? There will be a group of symptoms also being shown up. Now name the diagnostic test for the disease. What is the diagnostic test? It is ELISA. What is it? ELISA, enzyme linked immunosorbent assay. So over here I have written everything. Enzyme linked immunosorbent assay. How does it work? It works on the principle of antigen antibody binding. Antigen antibody binding, as in, I'll just give you an example, so please remember that, okay? Antigen antibody binding, that is a principle of how enzyme linked immunosorbent assay works. Suppose I already have some setup like this, where I have the antibodies which are produced against HIV. I know that these are what? Antibodies which are produced against HIV. Now I'm testing whether Pappu has HIV or not. What do? I take some blood from Pappu, okay, and basically if Pappu has HIV, what is going to happen? In that blood there will be some antigens, as in there will be some pathogens, and these antigens will be coming and attaching over here, if at all they are correct match, because these are HIV specific antibodies, right? So onto this the antigens from the blood will be coming and binding. Again, we will not be able to see it, which is why we will need another antibody out there, which is also pre-prepared. Now this antibody which is there, my dear children, it will be linked to an enzyme. It will be linked to an enzyme. Okay, now this antibody comes, it is linked to an enzyme. Now I want to see whether the actual binding has taken place. What do? I'll be putting some coloring substance. When I put the coloring substance, if at all the enzyme reacts to it and gives a color change, that means that all of this reaction has taken place. That means that the person has HIV/AIDS. You get it? So it is dependent on antigen antibody binding principle. Okay, now we come back to this question and we fill in the missing data which is there. Now this time you have to try. Retrovirus came. Virus infects a normal cell. Okay, it infected a normal cell. Now viral RNA is introduced into the host cell. Agreed, it is now inside the host cell. Reverse transcription absence. After that, what happens, my dear children? After reverse transcription, what do you get? Viral DNA is going to be formed over here. That is the next step. Now viral genetic material in the nucleus of the host cell, as in the viral DNA is now inside the host cell. What will happen? The viral viral DNA incorporates into the host genome, and after that what will happen? After it incorporates, it will give information to form the viral protein as well as the viral RNA. So viral protein and viral RNA is being formed. The viruses comes out of the host cell because everything is now assembled, and that's how it goes. So if at all you know the flow, you will be able to write the answer. If not, you will not be able to write the answer. So viral DNA is formed, that is the first one. Next thing, viral components such as proteins and RNA is being synthesized. Then similar to that, we have one more question. Now this is all from the question bank, and they have been put under the 5-marker category, which is why I thought I'll explain it to you. Okay, so a person has been diagnosed as HIV positive. Okay, they have been diagnosed as HIV positive. Name the diagnostics test. It is ELISA. Now write the full name of the pathogen: human immunodeficiency virus. Human immunodeficiency virus. Now name the genome of the pathogen. What is it? What is the genome? Single-stranded RNA, right? Single-stranded RNA. Also you call it as ssRNA. Now name which particular cells of this person are likely to get destroyed. What is the cell that are going to be likely to get destroyed? It is first macrophages, but my dear children, ultimately it is T helper cells. Ultimately it is T helper cells. So even though if we do not write this, at least write this. At least write this. This is going to be the major target. This is the one which is why we have less immunity, because this guy is being targeted. Macrophages, initially they are being attacked, and they are basically, we say they are factory of HIV. Why? Because HIV viruses, they are going to replicate the number over here and then attack the T helper cells. But if at all you get a question like this, I will prefer you write this first. You write this first, and probably you can write macrophages in bracket, you can write factory of HIV. That's all. First this. Okay. Now suggest a partially effective treatment to treat him. So HIV, do you think HIV/AIDS has a treatment? Okay, there is no fully curative treatment that has been found out until now. Okay, there are things that are ongoing, but generally the partially effective method that we have is having antiretroviral drugs. Okay, so antiretroviral therapy is something that we have because it's a retrovirus, so against it we have antiretroviral therapy. So you can just remember that. I hope that works. Yeah? Okay, perfect. Now we'll come back to the questions that we were doing. So yes, we were here. Differentiate innate immunity innate immunity and acquired immunity. First question, innate and acquired. Innate means what? Something, immunity that you have from your birth, and also it is not specific. So example, when I say that I am born with skin, this is an example of innate immunity. I am born with skin, and the skin prevents entry of anything inside my body, right? And do you think it's specific such that, do you think it will allow a virus, you come in, and bacteria, you do not come in? Is it like that? Is it specific? No. It's the same for all. It acts the same for all of them. So this comes under what? Innate immunity. Basically, you need to remember they are from birth and they are not specific in nature. Okay. Under this, you have multiple barriers. This is your homework. Please go and study the different barriers of innate immunity. Very, very important question. Physical barriers, as in your skin as well as your mucus, and then you have physiological barrier, for example, HCl being secreted or your tears being secreted. All of them, they come under what? Physiological barriers. Cellular barriers, for example, you have macrophages, you have polymorphonuclear leukocytes, that is known as neutrophils, and all of them. Okay, natural killer cells. Now you have cytokine barriers, as in cells releases some things which are non-specific. It is known as, in example is interferons. All of them, please go through it once. Okay. Now acquired immunity is something, it's not that we get from birth, we acquire it during our lifetime, which is why you call it as acquired immunity. And this is going to be super specific. It is going to be super specific. For example, I tell you that in our body, whenever some antigen comes, okay, whenever we say that, okay, I've got an infection, when the antigen comes in, my body is going to react to it in two ways. One is, it will be showing humoral immunity. The second thing it will show is cell-mediated immunity. Okay, humoral immunity as in, basically it is going to release, or basically I can say humoral immunity is the when, basically, seeing a pathogen, my body will be producing some B lymphocytes. And these B lymphocytes in turn are going to produce two other things. One is going to be the antibodies, and the other one which is there is going to be some memory cells. is going to be some memory cells. So antibodies and memory cells are being produced. These antibodies are going to target the antigens and are going to target to destroy them. What about the memory cells? Memory cells are produced in a way so that it has an idea. Next time the same antigen comes in, I have to be more careful. I have to produce the antibodies in more time. So this might be for the first time it might be seeing an antigen out there, right? So obviously the first response, the primary response over here is going to be less. But the next time the same antigen comes into our body will react. So which is why every time when we have some infection for the first time, it will react very badly. The second time the same thing comes on, our body knows how to tackle it. Why? Because there is memory cells being produced over here. Why do you say that this is humoral immunity? Because, my dear children, B lymphocyte, which is a type of white blood cell, it is going to release antibodies directly into the blood. And blood is what? A fluid. Blood is what? A fluid. And you also call this fluid as humor. That is the interchangeable term that is there. Humor. So since in your humor, or basically in the blood, you have antibodies floating around, and that is there for trapping the antigens, you call this type of immunity as humoral immunity. This is one type of acquired immunity. Didn't you acquire it during your lifetime? Yes, you acquired it, right? You acquired it during your lifetime, you got some infection, and that is when all of this thing started happening. Similarly, you have cell-mediated immunity. Here in cell-mediated immunity, the major guy that plays a major role is T lymphocytes. T lymphocytes. Now over here also, whenever some antigen comes across some antigens, it is going to tell the other cells, hey, you have to be active, you have to go kill the antigen. Such as it will signal the B lymphocytes to produce antibodies, or probably it directly can go and kill the cells. Anyway, that can happen. Apart from it, it will be also releasing one very important thing, that is memory cells. Again, memory cells, such that the body has an idea. So one thing that is common in both of them is that memory cells are being produced. That means the body has a memory of it, that something has come. The next time you have to be very, very careful out there. Got it? So this is humoral immunity and this is cell-mediated immunity. This comes under acquired immunity, and this is very specific to specific antigens. Specific type of antibodies are going to be produced, a specific pathogen, specific type of T lymphocytes are going to go act. Why it is known as cell mediated? Because over here T lymphocytes mediates the other cell, or itself goes and kills the antigens out there. So there is cell to cell interaction out there, which is why you call it cell mediated immunity. I hope you got it. Now if at all you had to give difference between, so you have to say innate immunity is present from birth, acquired develops after an infection, or basically during a vaccination. Vaccination is what basically you get the weakened terms of pathogens, weakened type of pathogens, and that has been put inside your body such that it can ignite a response, right? So either an infection or a vaccination. And here in innate it is non-specific, but here it is very specific to particular antigens. Here there is no memory, here there is immunological memory. So example is skin, phagocytes as in your macrophages and things like that. Over here you have antibodies, T cells as an example for it. Cool. Now you have the next question, which is refer to the below given diagram and answer the question. First of all, the diagram of antibody is super important. You should be knowing it. This is an antibody. Okay, over here this long chain that you see, this very long chain that you see over here, these chains are known as what? Heavy chains. What do you call it? Heavy chains. So when I say H2, that means two heavy chains. H2 stands for two heavy chains. Okay, what about L2? L2 is basically these two light chains out there. These two light chains out there, they are known as what? Two light chains. Got it? This is super important. Now over here, my dear children, you should also be able to see that there is a binding site. Binding site for what? Antigens. Antigens are going to come and bind over here, and that is how the whole reaction is going to take place, right? So this is the antigen binding site. A and B is what? The antigen binding site. Do not forget it. Very important. So name the molecules trapped in the lymph nodes responsible for the activation of lymphocytes that bind to A and B site. What is the one that is responsible for activation of lymphocyte? It is going to be the antigens, and they bind at the A and the B site. Works? Works, ma'am. Perfect. So how do you write the answer? You write the answer like this. Okay, make sure you write it. Mention the cells involved in accomplishing the acquired immunity. It is B lymphocytes and T lymphocytes. B does humoral immunity, T lymphocytes show cell-mediated immunity. Then there's a question. If at all I have an organ transplantation, okay? You might have seen in most of the cases the organ which is transplanted is getting rejected. You call this as graft rejection. Who is responsible for that? B lymphocyte or T lymphocyte? It is mostly T lymphocytes which is responsible for that. Cell-mediated immunity is responsible for graft rejection. graft rejection. And since we're talking about antibodies, also remember antibodies are also known as immunoglobulins, IG. That is why you say IgA, IgG and things like that. IG stands for what? Immunoglobulin. Antibodies that is being produced, it is nothing but proteins. It is nothing but proteins, or basically polypeptides. Polypeptides come together to form proteins. So please know that. And how many antibodies do we have? You can remember it like this: GAMED, gamed. IgG, IgA, IgM, IgE, IgD. The first antibody which travels from the parent, that is from the mother to the child, through placenta is IgG. That is why first position. Now the second antibody that travels from mother, okay, mother's milk to the baby, from the colostrum, it is IgA. Okay, so you can remember IgA, A for Amma. A for Amma. So this is passed through colostrum to the baby. Now M function is not given, D function is not given, E is given. E is there for allergy. E, allergy. Please remember it. Okay, and if at all, whenever something comes in and you feel that, okay, I do not know about it, go refer your NCERT. This is very, very important. Okay, now name the cells involved in the humoral and cell-mediated immune response out there. So in a question paper, instead of C question like this, they have put another question: name the cells involved in humoral and cell-mediated. So you should know, in humoral it is carried out by B lymphocytes. They produce antibodies, and also they produce what? Memory cells. These antibodies bind to pathogens and neutralize them. Okay, so please remember, they produce antibodies and also memory cells. Okay, now cell-mediated immune response, this response is carried by T lymphocytes. No antibodies are produced over here. Instead, T cells directly attack and destroy the infected cells or the cancer cells, or signals other cells, and also produces what? Also produces memory cells. So please know about all this thing. Works? Works, ma'am. Perfect. Okay. Now this question we solved. We'll come to, I think, the last question of this chapter. Fill in the missing data. Escheriasis causative organism is what? Ascaris. What are the symptoms of this Escheriasis, my dear children? Escheriasis. Escheriasis is basically caused by Ascaris lumbricoides, which is a roundworm, and it affects your intestine. It causes intestinal blockage. This is the first point that should be coming to your mind. Since there is a blockage, always remember, and also this particular guy goes and perforates our intestine, which is why there will be internal bleeding. Okay, intestinal, first of all, intestinal blockage. Then you talk about what? This thing, intestinal blockage. After that, internal bleeding is something that you have to say. And since internal bleeding is happening, you will be suffering from anemia as well. So will you be able to write that? Please write it. Okay, internal. So I'll write it over here. I think that will be good. Internal bleeding, intestinal blockage, and then anemia. At least these points you have to write. Now what do you have? You have name of the disease you have to tell. Trichophyton is the causative organism, and basically appearance of scaly lesions on the various parts of the body is there. Scaly lesions of the body leading to itching. Trichophyton, epidermophyting, fighting for what? Microspore, small spore. They are all fungi that causes ringworm disease. It is not caused by any worm, it is caused by fungi, but it looks like a worm because the structure is like this on the skin, right? So I'll say it is ringworm disease. Okay, see question. Typhoid causative organism is dash, and it has high fever, weakness, headache, stomach pain and constipation. So what causes typhoid? The bacteria whose name is what? Salmonella typhi. Salmonella typhi. Now what do you have? Pneumonia. Pneumonia is caused by Streptococcus pneumoniae or Haemophilus influenzae. What is the thing that you have? Pneumonia is the place where your lower respiratory system is going to be affected. That means your lung is going to be affected. In your lung, what is being affected? Basically your alveoli. Your alveoli, which is there, which is responsible for exchange of gases, that is being filled by mucus, which is why there is no proper exchange of gases. So can I say that fluid filled alveoli is seen over here, and the symptoms, if at all you go talk about, so there will be what symptoms if at all fluid filled alveoli is there. So this is not a symptom. The symptom you have to write, right? The symptom will be breathing difficulty, you will have fever, And everything, very common thing is what? Fever. Apart from breathing difficulty, fever, what do you have above? You might have cold, or basically you can just write cough. Apart from that, what do you have? You might have headache also. Okay, if not headache, if you're very scared for writing headache right there, because some things are common between common cold and this, you can go with the extreme character. That is what? Your nails and nails and lips turn what? Bluish gray in color. So please write all this thing and you'll get your marks. Okay, so each and every part of this particular table is important. You can get similar type of questions also, so please know about all the diseases. Now the one that I have got is right over here. E is a disease. It is caused by rhinovirus. It causes nasal congestion, discharge and sore throat, okay, and also cough and headache. So what is this thing? It is going to be common cold. This is going to be common cold. So I think I have got this or something else. Okay, so these are things that you have to write. I hope this is clear to you. If at all until now it is super clear, definitely let me know, and let me just drink some water and I'll be right back. Okay, level 3 crossed. If at all you have crossed level number 3, let me know in the comment section. By the way, was it level number 3? Okay, I remember one more level number 4, but anyways, okay. Starting with the first question, select the correct match. Australian marsupials is given on this side, placental mammals are given on this side. So spotted cuscus is from team Australian marsupial. You have to find the opposite of it from here. What is the answer? Flying squirrel, bobcat, anteater, lemur. The answer is lemur. Ma'am, how do you remember it? For flying squirrel, the opposite one that you have is flying phalanger. For bobcat, you have Tasmanian cat. For anteater, you have numbat. Okay, so how do you remember all of that? Once you go through this, slowly you will be remembering all of it. Remember, placental mammals are the ones with smaller names. Australian marsupials are the ones with larger names. So if mole is there, marsupial mole is there. Okay, if anteater is there, numbat is there. This is something different, so you need to remember anteater and numbat. Mouse is there, marsupial mouse is there. Easy. Lemur is there, spotted cuscus is there. This is also different, so please remember it. Flying squirrel, flying phalanger is there. Okay, somehow different. Bobcat, Tasmanian tiger cat. Tiger cat is there, so it's easy. Wolf, Tasmanian wolf. So always remember placental mammals, smaller name. So placental mammals, Baba, smaller name will be there. Smaller name is going to be there. Here you have bigger names. So mole, marsupial mole. Which one is placental mammal? This side is placental mammal. Okay. Anteater, numbat. So one which I have colored have different names, which is why you need to remember them specifically. Anteater, numbat, lemur, spotted cuscus, flying squirrel, flying phalanger. Phalanger comes that side. Okay. In Australian marsupials it comes. In Australian marsupials it will be coming. Please remember. Okay. Yes. Okay. So I was thinking of a way for you to remember it, but apparently flying squirrel, no. Anyway, phalanger is a bigger name, so you can remember it comes from the Australian marsupial side only. Fine. Next question. Which of the ancient men protected their body with hide and buried their dead? Okay. So, you got Homo erectus. Okay. 1.5 million years ago, you got Homo erectus, which started hunting food as in meat. They have two things, the divergent of two things. One is Neanderthal man and one is Homo sapiens, that is us. First who came? Neanderthal man. And they were the ones with 1400 cc of brain capacity or cranial capacity, and they started hiding their body and also burying their dead. So it is Neanderthal man which is the answer for this particular question. They are very unique for that particular reason. Cool? Cool. Next thing. Okay. So I've explained everything. Again, you can go read the PDF, or I will be explaining you in 2 marker, 3 marker. Do not be worried. Okay. Next. Arrange the following animal groups in current sequence of their occurrence on geological time scale. Okay. Amphibians, fishes, reptiles, mammals. Very easy way to remember. Farm. First came fishes, then came amphibians. From amphibians you got reptiles. From reptiles you got both aves, that is birds, and also mammals. So you have to go with farm. Farm basically means second comes first. Second. So these two options are wrong. Okay. After second you have amphibians, 1. So this is your answer. 2, 1, 3, 4. 2, 1, 3, 4. Okay. Okay. Yes. Next one. The paleontological evidence for evolution is obtained from paleontological, as in fossils. Okay, you get the paleontological evidence of evolution from the fossils. So your answer for this question is going to be fossils. Easy question. Yeah? Yes. Next thing. Observe the following diagrammatic representation of operation of natural selection on different traits here. Natural selection depicts what? Stabilization, directional, disruptive cannot be depicted. Listen, over here you see two bumps, right? So listen to me carefully. If at all you have tall and you have dwarf, and if both the characters are being selected, you say that this is disruptive. Two extremes are being selected. If at all we say the median character is being selected, that is middle height, then it is going to be stabilizing. If at all we say that only one, dwarf ones are selected, then I say it is directional. Or tall ones are only selected, then I say it is directional. So this is disruptive where both the extremes are selected, stabilizing as in basically the middle character is going to be selected, and directional as in basically one extreme is going to be selected. So here it is going to be disruptive. C is your answer, it is disruptive in nature. Cool? Cool. Fine. The next question. Dash stage in human evolution used hides to protect their body and buried their dead. So what is that stage that uses hides to protect their body and bury their dead? It is going to be Neanderthal man. We just discussed about it. Okay? Yes. Statement number 1. Man has domesticated dogs and has created different breeds by artificial breeding. Is it? Yes. It is definitely true. That is how we got different species of dogs out there. Statement number 2. Intensive breeding of dogs signifies divergent evolution. Is it? What do you mean by divergent evolution? Means you had a common ancestor. Now basically they have diverged into different species out there. So is it like that? Yes. Yeah, because so husky you find it in colder areas. Okay, other type of dogs you find it in different. Indian dogs are different from how the other dogs are being seen. You get it? Why? Because they are in different habitats. So obviously they have diverged, not converged. They have diverged. So can I say both statement 1 and 2 are correct? Yes, it won't be wrong to say that. Both statement 1 and 2 are correct. Next question. Statement number 1. Antibiotic or drug resistance is due to evolution by anthropogenic actions. Okay, what do you mean by anthropogenic actions? It means man-made actions. So if I say that we are resistant to antibiotics or drugs, do you think it's because of our actions? Yes. Why? Basically we are giving high doses of medicine and we are taking it very usually, such that the organisms which are there, they are mutating, and they are mutating in a way that they are okay with all these changes out there. Okay, they are being resistant to that. So yes, it is an example of evolution by anthropogenic action. That is absolutely true. Similarly, plants becoming resistant to pesticides, or basically not plants, the pest becoming resistant to pesticides, that is also an example for anthropogenic action. Now statement number 2. Many antibiotic or drug resistant organisms arise due to overusage of antibiotics. Yes, because they thrive, they want to survive, which is why they mutate, and then the one that is able to, it is just surviving and keeps on reproducing. So yes, I'll say both the statements again are going to be correct. Cool? Cool, ma'am. Okay. A brown-billed duck is newly added to a farm of white-billed duck. This indicates what? There were a brown-billed duck, and now soon I am adding some white ones here. Can I say it's gene flow or gene migration? From some other population, I'm adding something over here, which is going to change the frequency of the alleles over here. Isn't it? This is an example of gene flow. If at all I was doing, it was happening by chance. Suppose the black ones or the brown ones will be killed by some attack, and accidentally all the brown ones lost their life and the other ones were living. That means it is genetic drift. But here it is gene flow. Why? From outside I am bringing it inside, and that is changing the genetic frequency, which is why it is known as gene flow or gene migration. Suppose there are white people in America. You go there. Okay, we are blacks. Okay, that does not mean I'm racist. Okay, please understand that. I'm giving you an example, and by we I mean all of us. Okay, not you, it's we. So when we get inside, there's a change in the genetic frequency. Similarly, when they come over here, there's a change in genetic frequency out there here as well. That is gene flow or gene migration. Okay, okay, fine. Now change in frequency of genes and alleles is due to. Change in frequency of genes and alleles is due to mutation. Yes, mutation can bring in changes. For example, I had the brown ones and the black ones. Now because of mutations, I have blue ones also. So yes, it is changing the frequency of the alleles, correct? Gene flow, we just saw an example for that. Yes, that is also a reason. So can I say both A and B is the answer? Neither A and B cannot be true. So both A and B can be true, which is why I'll say C is the answer for this particular question. Works? Works. Definitely works. Next, the first amphibians that lived both on land and the water evolved from. The first amphibians that lived both on land and the water evolved from. Listen, you had fishes which was living in water, you had amphibians which lived on both land and water. So between fishes and amphibians, you had one organism that is known to be the connecting link, and that is lobe fin. By that I mean, this was just like a fish, but the fin which was there, it was very hard, such that with the fin it could walk around. And that is the connecting link between the modern day frogs and salamanders and also the fishes. So I'll say lobe fins, or they can just give you coelacanth. Coelacanth. Please know that this name is same only. They are used interchangeably. Okay. Similarly, between reptiles and mammals. Reptile and mammals, you have connected link which is known as shrews. Okay? Yes. Next, the biggest land reptiles that existed during Mesozoic era was. Mesozoic era is made up of Triassic, Jurassic and Cretaceous period. Okay? So over here, my dear children, the biggest land reptile that existed was Tyrannosaurus rex. Tyrannosaurus rex. That is going to be your answer. B. Okay. Next, statement number 1. Hugo de Vries believed mutation leads to spontaneous evolution. Hugo de Vries said that mutations are happening, which is why, and these mutations are really fast, it happens, and that is how new evolution is taking place. Yes, he was correct when he said that. Now, mutations are random, directionless, causes saltation. Mutation, do you think it is planned? No, it is very random thing. Yeah, does it have any direction? Does it support anything? No, it is directionless. It can happen in any direction. Okay, anything can be selected out there. Does it cause saltation? Saltation means one large mutation can lead to formation of a new species altogether. One step mutation can lead to formation of a new species altogether. Do you think that can happen? Yes, that can also happen. So can I say both the statement 1 and 2 are correct? Yes. So according to Darwin, he said that there are some changes happening that slowly leads to gradual changes out there, and that leads to evolution. Okay, so Darwin was promoting gradual changes. He was talking about mutations, which is very, very fast. Both of them were ideally correct in their own places. Both of them spoke about evolution and both of them were correct. So yes. Okay, but Darwin spoke about gradual ones. He spoke about spontaneous ones. Cool. Now, elongation of neck in giraffe due to inheritance of acquired character was given by. So basically, there was a guy whose name was Lamarck. He said that whenever you use an organ, basically because of its use, it stays there. Okay, and if at all it is not being used, basically it's not there. So there were actually smaller giraffe, but the smaller giraffe, they were using their neck to go up and take up the leaves out there, and since they were using, that is how their neck got elongated. But to contradict that, Darwin came and said that how can that be true? Acquired characters, something that you acquire during your lifetime, cannot be passed to the next generation. That cannot happen at all. Okay, prove it then. So basically, experiment was done with mouse. The tail of the mouse was being cut, and it was seen that, let's see, the babies of the mouse, will it have tail or not? Because anyway, it does not have tail, right? So let's see it if the babies have. The babies had tail. That means the acquired character of not having tail is not being passed down, right? Similarly over here, it's not like the giraffe is not utilizing its neck. Probably some mutations might have happened. Some might have shorter necks, some might have longer necks. The longer ones were able to survive, which is why they kept on reproducing, is what Darwin said. You get it? So the opposite was said by Lamarck, and please remember, it was being removed also. It was like, no, it does not make any sense. Cool? Cool, ma'am. Okay. Next, single step large mutation leading to speciation is also called as single step large mutation that leads to new species formation is known as what? Natural selection, branching descent, founder's effect, saltation. Answer is saltation. This comes below Hugo de Vries. Single step large mutation leading to speciation is known as saltation. Very, very important. Cool, cool. Next, the organic compounds found in the Miller's experiment are. Always remember, in Miller's experiment, he was only able to form one organic component, and that was amino acid. Nothing else. Rest of the experiments were done by different people, and they came up with different things. But Miller's experiment, Stanley Miller's experiment, he could only find amino acids. So your only answer is one only. Okay, one only. Nitrogenous base, pigments, fats and all were found by different other people out there, not by Stanley Miller. Cool. Embryological evidence for evolution was disapproved by. So embryological evidence said that basically fishes, amphibians, reptiles and mammals, that is how it goes, right? So since in the embryos of the mammals you have gill slits and you have tail also, but which usually goes in the adult state, it was said that whatever organism we have evolved from, basically the features of those organisms are seen in our embryo. So probably we might have evolved from fishes. That is what Ernst Haeckel said. Okay, that was embryological evidence. But then Carl Ernst von Baer, both of them are Ernst only, but the one with the bigger name was the person who disapproved this and said that that is not true, bro, because in that case amphibians features should also be seen in the case of mammals, reptiles features should also be seen. Why are we seeing only just gill slits which are there in the case of fishes, or just tails which are there in the case of fishes? That is absolutely wrong. If we have evolved from all these organisms, we should be having some characters of all these organisms. Why is it not that like that, right? So that is how it was disapproved, and it was Carl Ernst von Baer, who was responsible for disapproval of this. Okay. Arrange the following plants as appeared on the different geological periods. So over here you have conifers, knitales, dicots and monocots. Listen, in the case of angiosperms, dicots came first and then came monocots. So this flow is okay. Here also the flow is okay. Here the flow is not okay. Here also the flow is not okay. So I'll just cut it off. Okay. Now what do we have? Conifers, knitales. So listen, conifers came first, then came knitales, then came dicots, then came monocots. So here knitales is given and conifers is given at last. That cannot be true at all. Conifers has to be coming front, then knitales. So if you see that, this is going to be your answer. Listen, look at this particular picture. Ferns, spinopsids, basically you are pteridophytes. Then you have conifers, ginkgos, cycads, knitales. So conifers came first, then knitales, then dicots, then monocots. That is how it usually grows. Okay? Yes, so that is your answer. Now 19. Replacement of the lighter colored variety of peppered moth, Biston betuloria, to its darker variety in England is an example of. So you have seen industrial melanism. You have seen industrial melanism where what is happening? The white ones were selected first because there was white background, there was lichen. Okay. Later on when pollution was there, lichen is not there, white background is not there, which is why the white ones were spotted very easily. So it was nature which was selecting. It was not us, it was nature which was selecting. And that's all. So it is an example of industrial melanism, is an example of what? Natural selection. The one which is able to survive and reproduce gets ahead, right? So this is an example of natural selection. This is not regeneration, not genetic isolation, not even genetic drift. Cool? Cool. Yes. Now at a particular locus, the frequency of allele A is the frequency of allele A. When you denote it by capital A, you just understand that this is the frequency of the dominant allele. So it is 0.6. Okay, and the frequency of allele small a is, that means you're talking about Q, it is 0.4. What would be the frequency of heterozygotes in random mating population at equilibrium? So how do you denote heterozygotes? Always remember there's a formula, P square plus 2PQ plus Q square. P square denotes the homozygous as in condition of capital A, capital A. So you have to do this. Okay, you have to do this. That's when you get homozygous AA. When you do homozygous small a, so that means you do 0.4 whole square, that's when you get AA. You have been asked about heterozygotes. That means A and A. How do you find that out? So P into Q. P is what? 0.6. Q is what? 0.4. Is that all? No. You have to multiply 2 also. So 0.6 into 0.4 is 0.24. Okay, into 2 is equals to what? Is equals to 0.48. So your answer for this question is going to be 0.48. Okay, so do not forget to multiply the 2 over here. If at all you are asked about the percentage, write 48%. That's all. Now we move on to the chapter evolution. So there are more to it. There are more questions to it, and most of them would be 5-marker questions also, which we'll be dealing. And as I said, please check out the top 50 PDF at least and see if at all you are missing out on any important or difficult question out there, because there I have taken so many hot questions. Yes. So in evolution, we have the first question. Comment on the similarity between the thorn of bougainvillea and tendril of cucurbita with reference to evolution. So thorn of bougainvillea, okay, thorn of bougainvillea as well as tendril of cucurbita. How are they related? Basically, they had a common thing that is over here, which is axillary bud. They both came from the axillary bud. So structurally, they came from the same. Originally, they had the same origin, but since they are living in two separate habitats, one where it needs protection, one it needs support, right? So which is why it has been evolved differently. So there was a common point from there, they diverged. They show divergent evolution. And these type of organs which do not have any similarity in their functions, but have similarity in their structure are known as homologous organs. So homologous organs show divergent evolution, always remember that. Okay, HD. So thorn of bougainvillea and tendril of cucurbita are homologous organs. Both originate from the same basic structure, that is axillary bud, but perform different functions. Thorn protection, tendril climbing. Evolutionary significance is what? The similarity indicates divergent evolution where structures with common origin evolved to perform different functions due to their adaptation. Okay, works? That's all. Perfect. Next thing, state the evolutionary relationship giving reasons between sweet potato and potato. Sweet potato and potato, my dear children, on the other hand, are going to show something else. So here, suppose there's a stem. The stem basically went ahead and they are forming a storage unit. In this particular case, there is a stem and there are roots. The roots basically have modified to form what? To form storage units. This is a root modification, this is stem modification. Stem modification is potato. Root modification is sweet potato. So you can remember, S is not for stem here. This is ulta. So, okay, so here potato is something that comes from stem and sweet potato comes from root. Both of them has a similar function, storage. But do they have a common origin? No. So this is opposite to homologous organs. They are analogous organ. Function is same, but the origin is different. What type of evolution is this? Convergent. Both of them are staying in the same habitat, which is why they have converged to a point that they have similar functions. So always remember AC. Okay, analogous organ, convergent evolution. So sweet potato is a modified root, whereas potato is a modified stem. Both store food but differ in origin as well as structure. Such organs with same function but different origin are known as analogous organs, and their occurrence is due to convergent evolution where unrelated structures evolve similarly to perform the same functions. Okay, done? Done. Let's go ahead with the next question. Name the evolution of any two plant forms through geological periods from Tracheophyte ancestors. So my dear children, if at all you see over here, we have got Tracheophytes. Basically by that I mean the ancestors of Pteridophytes, because Pteridophytes are the first organisms to have what? The vascular system. And Tracheids is part of a vascular system, xylem, right? So Tracheophyte gave rise to Rhinia type plants. Rhinia type plants gave rise to Xylophyton. From Xylophyton, what did you get? You got spinopsids, which are what? The pteridophytes out there. Or basically you got what? Ferns. So you can write this particular thing from Tracheophyte, Rhinia type plant, Xylophyton, and then ferns, which is what? A pteridophyte. That evolution could take place. Otherwise, you can also write this Tracheophyte, then Rhinia type plants, then Xylophyton, then progymnosperms, yeah, then seed forms, and then angiosperms. Or if you feel it's too difficult for you, just write this Xylophyton and then conifers, and that's all. Basically, conifers and ginkgos, all of them, they come under gymnosperms. So here you, from ferns and spinopsids, they are, what do you call, pteridophytes. These ginkgos, conifers, nettles and cycads, they are basically what? They are gymnosperms. So you can write this, or you can write this, or you can write this also, either way. So for that you need to know the root, and always remember they were ginkgos, conifers, nettles, cycads, and also spinopsids and ferns. They were very much abundant during Jurassic and Cretaceous period of Mesozoic era. Jurassic and Cretaceous period of Mesozoic era. This is very important. And towards the end you got the what do you call, angiosperms. First came the dicots, then came the monocots. First came the dicot, then came the monocots. Monocots came in Cenozoic era. Mesozoic era, these guys were there. Okay, gymnosperms and dicots. Towards the end, you got the dicots. That comes under angiosperms. Fine. So, GCGC. GCGC. Got it? Please remember that. So, CC comes first. They came first. Then only you got all these things. Fine. Now, draw a label diagram of Stanley Miller experiment in relation to chemical origin of life. So what is that origin of life? Basically it means that inorganic things came together to form organic compounds, and then later this organic things came together to give rise to life. Didn't understand? Listen to me carefully. Inorganic things, for example methane, for example ammonia, for example water vapor, for example hydrogen, all of them which were here, okay, they came together under two very important things. Under high temperature, super high temperature, that is 800 degrees Celsius was maintained over here, and this whole setup, if at all you see, there is no leakage over here. Oxygen is not going to be there. There is a reducing environment. There is no oxygen up over here. Now what happens at this particular point of time is that basically, if at all you see over here, there will be liquid water in the trap, and this liquid water will be having amino acid, and amino acid is what? An organic compound. So from inorganic things you got the organic thing, that is what? Amino acid. Got it? This is super important. Fine? And only amino acid. Do not go and write that from S.L. Miller's experiment you got nucleic acid and things like that. You got only amino acid. Fine. Now, Australian marsupials and Australian placental mammals explain convergent evolution and adaptive radiation. Justify the statement. So, Australian marsupials, my dear children, are them. Marsupial, mole, numbat and things like that. Placental mammals are them. Mole, anteater, mouse. So, mole, marsupial mole. Anteater, numbat teater. So, numbat anteater. Mouse, marsupial mouse. Why do you see all this thing which is very common in nature? They are different organisms. But how do you think they are similar? The name, the structure is almost similar. Why? So, listen to me very carefully. So initially, my dear children, over here, suppose this is an island. First came Australian marsupials. They moved here, some of them 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 moved here. What type of evolution does it show? Diversion, because they are diverging, right? So if I talk about Australian marsupials alone, suppose mole and anteater, they showed divergent evolution. They had a common ancestor, but now, suppose I am talking about one more group of organisms that is up there, which is placental mammals. Placental mammals also, some moved here, some moved here, some moved here, some moved here, and some moved here. Basically, what are you doing? Again, here divergent evolution is happening by placental mammals. But if at all you see this, aren't they converging to a point? Aren't they converging to a point? Yes. So when you talk about two organisms in the same place and when they are showing divergent evolution, we say at the end, both of them are converging. They are showing convergent evolution if I talk about them together. Separately if I talk, then divergent. Together if I talk, then convergent evolution. This is very important thing. Keep in your mind. Okay, which is why they are very similar in look-wise. And also, yes. So you have to write, in Australia, many marsupials evolved from a common ancestor and diversified into different forms, adapted to different habitats, which is why we say that they show adaptive radiation. They're radiating and they're getting adapted there. Similarly, placental mammals also did that, and since they were in the same habitat, you see convergent evolution. Please know the examples for this. Always remember, placental mammals' name are small, Australian marsupials' name will be big. So mole, marsupial mole. Anteater, numbat. This is kind of unrated name, so you have to remember it. You might be given match the following question. Mouse, marsupial mouse. Lemur, spotted cuscus. Lemur, spotted cuscus. This is the most frequently asked thing. Again, bigger name is over here. Flying squirrel, flying phalanger. Bobcat, Tasmanian tiger cat. Wolf, Tasmanian wolf. Okay? Perfect. Now mention the evolutionary significance of the following organisms: shrews and lobfins. So my dear children, I always say farm. What do you mean by farm? Fishes, amphibians, reptiles, mammals. From fishes came amphibians, and the connecting link that you have over here is lob fins. So they were fishes itself, but their fins were lob fins. So with the fins they could walk around, and that is the evolutionary relationship that we have between amphibians and reptiles. So your modern day frogs and salamanders have come from lob fins. You also call this as coelacanth. Got it? This is super important. Now what do you have? Reptiles to mammals. Again, reptiles are the ones that gave rise to birds, also mammals, both of them. But between reptiles and mammals you have a connecting link, and that is shrews. So always remember, that is the evolutionary significance out there. So shrews are among the earliest mammals and represent the primitive ancestors of modern mammals out there. And lobfins, they mark a critical step in vertebrate evolution, bridging the gap between fishes and the land-dwelling animals. Okay. Yes. Now what are analogous organs? Give an example. The organs which are different in structure and origin but perform the same functions. We know wing of the bird and the bats and the insects, all of them have different wings, but they live in an aerial environment, which is why they have wings. That is a common thing that they have. But do they have the same origin? No. Eye of octopus and mammals, both for vision only, but do they have the same structure? No. One is invertebrate, one is vertebrate. Flippers of penguins and dolphins. Penguin is a bird, dolphin is a mammal. Flippers are both of them there. It is used for swimming itself, but again, not the common origin. Root modification is sweet potato and stem modification in the case of potato. That is also there. Now state Hardy-Weinberg principle. Explain any 4 factors affecting the Hardy-Weinberg principle. Hardy-Weinberg principle says that the allelic frequency is going to remain the same generation after generation. It will be remaining the same generation after generation. That means, for example, if I talk about this, okay, over here, suppose I am saying capital T, capital T, capital T, capital T is this much, and I say small t, small t, small t, small t is this much. I try to mean that allelic frequency is like this, and now according to Hardy-Weinberg says that it is going to remain the same. This is how exactly it will be throughout. Do you think that's true? No. What if some tall people came from elsewhere and came over here and settled? You will have more capital T gene. That is known as gene flow or gene migration. Okay, the sum total of genes occurring in a sexually reproducing. The transfer of genes between population of different gene pool which differs genetically but can interbreed is called gene flow. So basically just understand, if at all there is a gene migration happening, if there are people are coming from outside to a different place out there, or some people are moving from here to outside, there will be change in the allelic frequency, which again is going to create an imbalance in the genetic equilibrium. Next thing, genetic drift. For example, some organisms are up there, okay, and suppose these organisms which are up over here, such as I have the yellow beetle and the white ones. So the yellow ones are strictly more in number, but one day suppose a big elephant came and stamped over it. Do you think the allelic frequency is going to remain the same? No. Do you think it's a process that was planned? No. It's a very random process. So this is known as genetic drift. Because of from random changes, there will be change in the genetic frequency. That is known as genetic drift. Now the next thing that we have is mutation. Is what? Mutation. What exactly happens in mutation? You have yellow ones, you have white ones. Suppose you have the new ones being added. Why? Because of mutation. Again, do you think there is a change in genetic frequency? Obviously, there will be. Next thing, recombination. During sexual reproduction, there is crossing over, there is new things being formed, which was not there initially. Don't you think there is a change in genetic frequency? Yes. So recombination is also one. Natural selection, the last one. Natural selection is basically when nature is selecting. The white ones were more before industrialization. The black ones were more after industrialization. So again, the allelic frequency, isn't it changing? Yes. So all these things are the factors that affects the Hardy-Weinberg principle. Please know about it. It's very, very, very important. Okay. And if at all you have seen the graphs also, in natural selection there are 3 types of graphs. One is stabilizing. For example, you say capital T and small t is there. None of them are tall or none of them are dwarf. In between, you get the middle character, that is medium heightedness. This is what? Stabilizing selection. Okay. Then over here, you have capital T and you have small t. Both of them are being selected. Both the organisms are there. Both the extremes are being selected. This is going to be known as disruptive selection. And then suppose over here you have capital T and over here small t. Only one is being selected. That is known as directional selection. Only one extreme is selected. That is known as directional selection. These are the few important things that you have to keep in mind. And we'll go on to the next chapter, that is, okay, first of all, evolution. There is no 5 marker, which is why it is not going to be discussed over here. And now we enter the next chapter, that is microbes in human welfare. One of my favorite chapters out there, and I see that 2 hours 23 minutes somehow is always has been done already. But are we going strong? I hope we are going strong, and we have reached the level 4. So level 4, if at all you have crossed, do let me know, and let's go ahead. Saccharomyces cerevisiae, commonly called as brewer's yeast, is used for fermenting what? Malted what? Malted pulses, malted cereals, fruits, vegetables. Saccharomyces cerevisiae, which is known as brewer's yeast. In bakery industry, baker's yeast, okay. It is used for fermenting malted. Malted means something that has started with germination but process got stopped in between. That is malted condition, okay. So it was malted cereals that were being used. Malted cereals and also fruit juices are being used in this particular industry. Malted cereals and fruit juices, okay, in the brewery industry. So your answer for this question is B. This is this year's MQP question. Now, cyanobacteria is an autotrophic microbe widely distributed in aquatic and terrestrial habitats, can fix atmospheric nitrogen is cyanobacteria. In your NCERT, there is the name of 3 cyanobacteria that are given. One is nostoc, one is anabena, and the other one which is there, it is nostoc, anabena, oscillatoria. Okay, nostoc, anabena, oscillatoria. Okay, so instead of uno, you can put ano. That's all. So here nostoc is an option, so we'll go with that. If at all you're talking about bacteria, plain bacteria, there is rhizobium, there is azotobacter, and that is azospirillum. 3 things are also given. This is cyanobacteria, okay? They are autotrophic in nature. They can prepare their own foods. Normally, bacteria cannot prepare its own food. So here talking about own food preparing bacteria that can fix atmospheric nitrogen as well. That is ano. As I said, anabena, nostoc, and oscillatoria. Okay? They are used in paddy fields as well. Now, because of the presence of which of the following vitamin, curd is considered as nutritionally richer than milk. So, in curd, what do we have? We have vitamin B12, my dear children, which is why we consider it nutritionally more richer than milk. Okay, so when the lactobacillus or basically lactic acid bacteria is secreting lactic acid and converting or breaking down the casein protein which is there in the milk, it also adds up vitamin B12, and that is why the curd is rich in vitamin B12. Okay. Next, mycorrhiza is involved in absorption of dash from soil and passes it to the plant. What is mycorrhiza? Mycorrhiza is a combination of a fungus which belongs to glomus. And also the root of the plant. So basically the plant is going to be giving the product of photosynthesis, the organic food. Fungi, my dear children, provides phosphorus. Phosphorus, this is very, very important. Phosphorus, not nitrogen and potassium. It is phosphorus. Okay, from the soil, because fungi can go deep in the soil and can trap it up. Apart from phosphorus, there are other things also that you get from this particular mycorrhiza. That is basically, it provides tolerance, it provides resistance, and it increases the plant growth and development and all of these things. Okay, but here your only option that is given is phosphorus. Absorption of what? Phosphorus. Okay. Next thing, during sewage treatment, the gas produced in the anaerobic sludge digester includes. Listen, whenever the term anaerobic comes and whenever the term gas comes, just understand you're talking about biogas. Biogas is produced in anaerobic conditions by anaerobic bacteria. You call them as methanogens. Okay, and when you talk about biogas, biogas is made up of 3 very important things. One is methane, other one is hydrogen sulfide, and the third one is carbon dioxide. So your answer for this question is going to be C. So methane can be denoted like this. H2S is hydrogen sulfide, and carbon dioxide, you know how it looks. Sometimes instead of putting hydrogen sulfide, they can also put hydrogen. Please do not get confused. Your answer is going to be the same itself. Okay, done? Yes? Okay. Next question. Dash is a producer of butyric acid. Butyric acid is produced from a bacteria whose name is Clostridium butylicum. So if at all, again, you studied microbes in human welfare from me, I think some of you might be remembering the things that I have taught you, right? So Clostridium butylicum is the producer of butyric acid. It's a bacteria. Now, other name of the industrial chemicals are also given over here. You can just have a quick sneak peek, pause the video, and can just look at it. Now, Propionibacterium shermani is used in the production of Swiss cheese. Okay, not in Roquefort cheese. In Roquefort cheese, you need a fungi. A fungi helps in the production of that. Swiss cheese is where basically big holes are there because CO2 is being expelled through that. Okay, and it is being produced by Propionibacterium. Okay, and it is Swiss cheese as the answer. So B is the answer for this particular question. Okay, I have mentioned it wrong. This is the answer, okay? So probably while copying and doing things like that from different there is a typist out there involved. There are a lot of things involved probably, which is why all these mistakes come. Do not be worried, I am here to correct them as well. Okay? Yes. Cool. Alexander Fleming discovered penicillin while working on. So Alexander Fleming was working on one very important thing that is known as, any idea? Any idea? Is it Streptococcus or Staphylococci? It is Staphylococci. He was working on Staphylococci. That's when he got to understand, he didn't wash the dish once and he thought that the bacteria is going to grow. But next day he saw that a mold, a fungal mold is growing out there, and because of this mold, the bacteria was not able to grow. Why? Because probably the mold was releasing something. What was it releasing? Antibiotic, that is penicillin. So the mold was known as Penicillium notatum and Penicillium notatum, and basically it was releasing an antibiotic penicillin, and that did not allow Staphylococci to grow. So it is not Streptococcus. Streptococcus is responsible for production of, streptokinase, right? So remember that. The Staphylococci. And Alexander has L over here, Staphylococci also has an L. Okay, remember like that also. This is the guy. Yes. Now, which one of the following alcoholic drinks is produced without distillation? So, distillation basically means that I am making the water content to go away. When water is going away, the level of alcohol will be more concentrated, right? So, after drinking something, if at all it goes like this, that means it has underwent distillation in more amount. So, whiskey, rum, brandy undergoes distillation. You have been asked which is produced without distillation. That means water content is still there. It is not being removed too much. It is not too much concentrated alcohol out there. That means wine. Wine is something like that. Apart from wine, you also have beer. Cool, ma'am. Okay. Next, activated sludge is a sediment deposited in. Activated sludge. Think about it. So you had the primary effluent. You made it treat with one very important thing, that is aerobic bacteria and the fungal filaments together form the flocks and things like that, right? It treated the organic matter out there. Now you transferred it. This was in the aeration tank. You transferred into settling tank. In the settling tank, the flock, my dear children, got settled down. And whatever is clean, that was sent into the river. This thing is known as activated sludge. The flock, basically, after the use, the flock which is there, it is the activated sludge. Where was it settling down? In the settling tank. So the first process is done in the settling. Primary treatment is done in the settling tank. Then for secondary treatment, you transfer it to aeration tank. Then back into settling tank. Then into anaerobic sludge digester. Okay, so first settling tank, then aeration tank, then again settling tank for this, and then you put this particular thing in anaerobic sludge digester. That is how it goes. Okay, so it settles down in the settling tank. A. Yes. Biogas is a mixture of, you tell me, biogas is a mixture of CH4, CO2, CH2. Do you have H2S anywhere? No, you have hydrogen. Is that wrong? No, it's not wrong. So basically, this combination should be correct. Do not go and write CH3 out there. It is CH4. Okay? Okay, ma'am. Next. Match the column 1 with column 2 and select the correct option using the codes given below. Citric acid, cyclosporin A, statins, butyric acid. Okay, I'll just zoom this in and see. So citric acid is produced by Aspergillus niger. It is a fungus that produces it. Butyric acid by Clostridium botulicum. So A goes with 3. A goes with 3. A goes with 3. I have 3 options like that. I can just remove this option. Okay. And what about D? D goes with 2. D goes with 2. I have 2 options like that. So I have confusion between this and this option. Now cyclosporin A is basically an immunosuppressive agent which you get from Trichoderma polysporum. So B goes with 1. B goes with 1. So this could be the answer. And C, serratin, which is there, it stabilizes or lowers the blood cholesterol level. You get it from Monascus purpureus. So C goes with 4. So basically your answer for this question is going to be B. It is going to be B. Got it? This is question bank question. Fine. Fine, ma'am. Next. Which one of the following is not an advantage provided by mycorrhiza to the plants? This is an important thing. Okay. Which one is not an advantage provided by mycorrhiza to the plants? Resistance to root-borne pathogen. Does it provide resistance to root-borne pathogens? Yes. Increase in the yield quality? No. It does not increase the yield quality. I told you. Resistance, tolerance, overall increase in plant growth and development, definitely yes. And also phosphorus absorption, but not yield quality increase. It has nothing to do with that, which is why B1 is the odd one out. Okay, B is going to be the odd one out. Now what do you have? The microbial biocontrol agent for butterfly caterpillar is butterfly, all the insect group specific, the butterfly armyworm and all the things, my dear children, tobacco budworm, all of them are being treated with Bacillus thuringiensis, BT. So this is your answer. These are insects ones, okay, insect killing ones. Okay, next one, these are straight questions, right? So which one of the following is a bacterial biofertilizer? Bacterial, I'm not talking about cyanobacteria, bacterial. So instead that Anabaena, Nostoc, Acetylatoria, A and O cannot come in. What can come in? Rhizobium. Rhizobium, Azotobacter, Azospirillum, they are bacterial biofertilizers. They fix nitrogen, they do all this thing. Rhizobium is symbiotic, shows symbiotic association with the plants. Azotobacter and Azospirillum, they are free living, but they are basically providing nitrogen. They fix nitrogen. And they are all bacterial biofertilizers. Know the difference between cyanobacteria and bacteria. Cyanobacteria prepare their own food. Okay, so over here also you get 2 and 3 marker questions. Just give me 1 minute, I'll be right back. So the first thing is describe briefly the primary treatment of sewage. So I will talk about the sewage together itself. Primary treatment, secondary treatment, that will be a big thing to do. So let's talk about that first and then we'll go with anything out there. I think then half of the chapter is being done already. So listen, first thing that we have is treatment, right? And that is primary treatment. What do you mean by primary treatment? Primary treatment. Where do you do it? You do it in settling tank. You do it inside a tank that is known as settling tank. Why? You want the larger things to settle down. You want the things that can float to float around. The floating things are removed through the first thing that is known as filtration. And the things that are sedimenting down, you remove this through sedimentation. Through sedimentation. So you have filtration and you have sedimentation. Now whatever is left behind over here, that is known as the primary effluent. The primary effluent. What do you do with that? That goes for secondary treatment. In secondary treatment, you put it inside the aeration tank. Aeration tank means basically here air is going to be supplied. Why aeration tank? This is the most important thing. Here because aerobic bacteria which is there, the ones that needs oxygen, is going to act on the organic matter of the primary effluent which is there. Can they do it alone? No, they require some fungal filaments also. Together this fungal filaments and aerobic bacteria form something that is known as flux. What does the flux do? Basically they act on the organic matter. For acting on it, do you think it needs oxygen? Yes. So can I say the biochemical oxygen demand, the demand for oxygen will be high if the organic matter is more? Yes, I can say that. Now on the other hand, my dear children, I will be also saying one more very important thing. If at all the organic matter is less, that means the aerobic bacteria is less. If aerobic bacteria is less, the demand for oxygen is also going to be less, right? So I will measure the pollution by finding the BOD. If BOD is less, I understand aerobic bacteria is less, that means probably organic matter will also be less. So I have to work in here until the BOD becomes reduced. BOD, flux, super important. Flux is what? A combination of aerobic bacteria and fungal filaments which acts on the organic matter, causing reduction in the pollution out there. BOD is a sign for pollution. We get to understand the demand for oxygen in a particular time out there from the aerobic bacteria. Now once it has been done, I transfer it again back into a settling tank. The name of the tank is important, settling tank. What is happening in the settling tank? All the flux which is there, it is going to be accumulated over here in the settling tank. What about the other things out there? What about the rest of the things which is up over here? Okay, what about the things which is rest up over here? It is going to be transferred to some river or stream because it is already clean. Now whatever is kept over here, the flux which is there is now known as activated sludge. Why do I call it activated sludge? Because my dear children, it is this flux and basically it is now going to be treated again. By the way, some amount of it is given back into the aeration tank so that the next time something comes, it can be treated. You need some aerobic bacteria, right? So it acts like an inoculum as well. Please remember that. Now, what do you do with the activated sludge? The activated sludge you find in the settling tank and later you transfer it to the next thing, that is anaerobic sludge digester. What happens in anaerobic sludge digester? Over here, my dear children, there is no oxygen. Remember, there is no oxygen. There are anaerobic bacteria. anaerobic bacteria. They do not need oxygen. And this anaerobic bacteria, my dear children, are going to act on the floc, or basically the activated sludge. The aerobic bacteria as well as the fungal filaments, producing what? Producing the biogas. What is the biogas made up of? Biogas is made up of methane, which is known as CH4. Biogas is made up of H2S, hydrogen sulfide. Biogas is made up of CO2. Three gases comes together to form biogas. Always remember that. So know the flow of it and you know the whole thing. Okay, so we'll remove the multiple form of questions that have come. The first one that is there is this. Briefly in the primary treatment. First one, in primary treatment, what do? You do is the first physical step in the wastewater treatment. Floating debris is removed by filtration. Grit is removed by sedimentation. The settled solid is the primary sludge. The upper clear liquid is known as effluent. This effluent is sent for secondary treatment. That's all. So filtration and sedimentation is something that you have to write for sure. Now I'll come to the next thing. What are flocs? Write their significance. Flocs are loose aggregates of aerobic microorganism, especially aerobic bacteria with fungal filaments. What is significance? They break down the organic matter. This reduces the BOD. Lower BOD means reduced pollution. They help in purifying the sewage. Done and dusted. Now what do we have? We have stages of, explain the stages of sewage treatment. You know that I have written over here. You will get the PDF, join the WhatsApp channel, everything is sorted. The same thing that I explained over here is being put in the terms of words. You can just go through it. Now what do we have is this thing. That is, explain the role of fungi as biofertilizer. Listen, fungi, how can it act as a biofertilizer? Fertilizer is something that increases the nutrient quality of the soil. An organism can also increase the nutrient quality of the soil. How do you think so? By basically doing one very important thing. By absorbing something for the other plant. So suppose this is a plant with the roots and everything. This is the root. Along with the roots, you have some combinations out there, that is of the fungus. What does this fungus do? Fungus go deep into the soil and enhances the absorption of phosphorus. Enhances the absorption of phosphorus. Here phosphorus is what? A very important thing. Okay, very important nutrient, and that is letting the plant grow. Apart from that, this particular, so that is the reason why it is acting like a biofertilizer. Okay, apart from that, please know the fungus has some other functions also. It increases resistance. Resistance to what? Pathogens. It increases tolerance. Tolerance to what? Drought conditions, salinity and things like that. Okay, and always remember they have a symbiotic association. Why? Because the plant roots provide food to the fungi. Fungi, which belongs to the glomus, my dear children, it absorbs phosphorus, it provides resistance, tolerance, and also increases overall plant growth and development. Overall plant growth and development, that is also important. It does not help in any increase in yield. There is no increase in yield that has been affected by this. So this is the role of fungi as a biofertilizer. Got it? Got it. Now the next thing that I have is mention the uses of following bioactive agents. Streptokinase. Streptokinase, what does it do? It is a clot buster. It bursts down the clots. It is used in patients who are having myocardial infarction or basically heart attack. Okay, they bust the clots. You get this from Streptococcus. Very important thing. All these things are very important. They are bioactive agents. Cyclosporin A is basically an immunosuppressive agent. Suppose I take a kidney from some other person. Now I want my body to accept it, not reject it. My body will usually reject it thinking it's a foreign thing, right? So for my body to become little immunosuppressive in nature, little less dominant nature, I'll take immunosuppressants such as cyclosporin A. I get it from Trichoderma polysporum. This is a fungi. This is a fungi. That was a bacteria. Statin stabilizes the blood cholesterol level or lowers down the blood cholesterol level. How? By inhibiting with the synthesis of cholesterol. Okay, so there's an enzyme that helps in synthesis of cholesterol. It basically goes and inhibits the enzyme out there. You get it from Monascus purpureus. Again, this name is important. You get it from a fungi itself. Cool? Cool, ma'am. Next thing, name the microbes that helps in the production. Cyclosporin A, you know, Trichoderma polysporum, a fungi. Streptokinase, Streptococcus. Statin, you know, it is Monascus purpureus. Now, explain the role of microbes in industrial products. So, you have to remember it. Okay, there is no other way. Aspergillus niger, it's a fungi producing citric acid. Acetobacter aceti, it's simple. Acetobacter aceti, acetic acid. Clostridium botulicum, butyric acid. Lactobacillus, lactic acid. Saccharomyces cerevisiae, ethanol. So, yeast, my dear children, is used for fermentation. It produces ethanol, alcohol, right? So, this is a fungi. Okay, sorry, this yeast is a fungi. Aspergillus niger is also a fungi. So citric acid does not have a name in common in over here, which is why you might forget it. Please remember it. Aspergillus niger, citric acid. Yeast, Saccharomyces means ethanol. Apart from it, Penicillium notatum, penicillin. That is the first antibiotic. Penicillium notatum. Penicillium notatum. It's a fungi. It's a fungi. Streptococcus, streptokinase, trichoderma, you know that. Okay, so please know all of it. Apart from that, pectinase. Okay, pectinase, protease and everything, which is very much important for clearing up the mess in the bottle juices out there. It brings out clarity. Please know that. Lipase is used in what detergents to remove the oil stain. Lipase acts on the lipids. So that is also there. Now explain the role of microbes in household products. You have to talk about lactic acid bacteria that converts milk to curd, and while it does it, please remember it improves the vitamin B12 quality. It increases the amount of vitamin B12. Next thing, baker's yeast, Saccharomyces cerevisiae, is used for making bread. Okay, toddy is a traditional thing which you get from the fermentation of palm sap. Again, you need organisms to doing that. And you have Swiss cheese. You might have seen Tom and Jerry in that very big holes in the cheese. This is Swiss cheese. Why do you have holes? Because CO2 is being released. Who does that? Propionibacterium shermani. So it is getting shy because it is releasing CO2. You can remember like that. Propionibacterium shermani. Okay, the last is Roquefort cheese. You get it from another fungi. Okay, a fungi is involved over here for the ripening of Roquefort cheese. Please remember that also. Propionibacterium for Swiss cheese, which is there. Propionibacterium shermani, SS. Always remember SS. Over here, it is a bacteria. In this Roquefort cheesecake, it is a fungi. Cool. So what do we have next? Biocontrol agents as an organism. Again, I do not want pesticides. I want organism that will feed on another organism. Those are biocontrol agents. So please know about that. Ladybird beetles are there. These are insects which will go and eat other insects. Ladybird beetle feeds on aphids out there. Okay. Similarly, dragonfly which is there, it feeds on mosquito larva. Ladybird beetle on aphids, dragonfly which is there, mosquito larva. Bacillus thuringiensis, basically you all know BT cotton, BT brinjal, they kill the insects specifically, and they are usually sprayed as dried spores, not as fresh spores, dried spores on the plants. Okay, and yes. Now next thing is fungal biocontrol agent, trichoderma. Trichoderma polysporum produces cyclosporine. Apart from that, trichoderma fungi is also there in the root of the plants, and they do not allow another fungi to grow. Okay, then you have viral biocontrol agent, baculoviruses. They come under nucleopolyhedrovirus. Very important. Baculovirus, they come under nucleopolyhedrovirus. They infect only specific insect pests out there. Okay, so these are the things that you need to remember. Please go through it. All of them super important. Biofertilizer, mycorrhiza fungi you studied. The mycorrhiza is basically fungi on the root of the plants, about which we studied, right? Apart from that, there is nitrogen fixing bacteria. Always remember, in bacteria, two things you have to remember. One is nitrogen fixing plain bacteria that do not make their own food. One is cyanobacteria, who has the ability to make their own food, and also they help the plants as well. That is, in cyanobacteria, you have ano. Instead of uno, you have ano. Anabenna, nostoc, and oscillatoria. Anabenna, nostoc, oscillatoria, they basically again fixes the atmospheric nitrogen, because plants cannot take the nitrogen out there. It is having triple bonds, it cannot break it by own. So it fixes nitrogen and increases soil fertility. This is a basic biofertilizers in paddy fields, and they increase the organic matter of the soil as well. Whereas when you talk about plain bacteria out there, nitrogen fixing bacteria, first one you have is rhizobium. Rhizobium has a symbiotic association. It always is in relation with the plant itself. It fixes nitrogen for the plants, in the leguminous plants. Apart from that, you have free living bacteria, as in these organisms are free living. They are not symbiotic association. They live free, they do their own work only. And over here, example is Azospirillum and Azotobacter. They also fix nitrogen. Okay, so this is symbiotic, this is free living, and this makes their own food by themselves. Cool? Cool. Now what do we have? We have explained a typical biogas plant with a neatly labeled diagram. So over here in the biogas plant, what do you have? Here you fill the dung and the water, you mix it up. In the dung, what do you have? In the dung, you have methanobacterium. Methanobacterium, they are anaerobic bacterium, anaerobes. They act on aerobic things, or they act on aerobic matter and organic matter, and they produce what? Biogas. So that is the logic. So this dung has the methanobacterium. It's now over here passed in a digester. In the digester, they will be producing the the gas out there, biogas, methane, H2S, as well as CO2, and that is going to be going to different households. Whatever is left behind is passed into the sludge, and this sludge is used as fertilizers or manure. That is the only logical thing. So know the diagram of it. Remember, methanogens are anaerobic in nature. They can never be aerobic. And when I say biogas, it's not just methane. It's not just CH4. It is also H2S, hydrogen sulfide, or you can be H2 also, hydrogen as well. And then CO2. And then CO2. So the technology of biogas production was developed in Indian Agriculture Research Institute, which you call as IARI, and also Khadi and Village Industries Commission, KVIC. This is very important. These two organizations came together for the biogas production. Okay, and this diagram is important. You need to know it. Microbes and human welfare also, there is no 5 marker. There's only 2 and 3 marker. We have discussed it already. Now we enter into the next chapter, that is biotechnology, principles and processes. So we have, I think, 5 more chapters, which is why I'm increasing the speed a bit. Otherwise, it can keep on going. Yes. So, yes, let's go. So in the year 1963, the 2 enzymes responsible for restricting the growth of bacteriophage in E. coli were isolated. 2 enzymes responsible for restricting the growth of bacteriophage in E. coli. In E. coli, one bacteriophage was trying to grow, but the E. coli was producing some enzymes which were not allowing the growth of the bacteriophage. One of them cut the DNA and the other added the group. What? So that is being asked. So what do you think is the answer for this particular question? It's a very easy question. Listen to me very carefully, okay? So this is the thing that we have. This is the thing that we have, that is E. coli. E. coli will have its own genetic material, agreed? Now suppose a bacteriophage came in and put its genetic material inside. So the major target of this bacteriophage is to integrate this into the genetic material of the E. coli. And you have to stop that. Which is why E. coli will do the first thing, that is produce restriction endonuclease, as in restriction enzymes. And that is going to cut the genetic material. But if it is producing restriction endonuclease, that can act on this particular genetic material as well. What to do with that then? You have to protect your own genetic material. So basically, you will need one very important thing. You will need to add some groups over here. These groups, my dear children, are going to protect, okay, or is going to protect the E. coli's genetic material to not be degraded. Okay, so whenever methyl group is added, basically restriction endonuclease will not be cutting out there. That is why I say A is going to be your answer for this particular question. Okay? Okay. Next one. Which of the following is the correct representation of primer annealing step in PCR? Primer, where does it go and attach? It attaches at the 3' end. Why? Because DNA replication happens from 5' to 3' direction. So in the opposite side, it has to be 3', then only it can do 5' to 3' thing, right? So it has to attach only at the 3' end. So here it is 5', here it is 3', there it is 3' end, but here it is 5'. So both of these are wrong. What about this? 5' again, wrong. What about this? Yes. Here at 3' and here also at 3'. So yes, probably that can be correct. So D is your answer for this particular question. Cool? Now, the chemical nature of the primer used in polymerase chain reaction is, okay, the chemical nature of the primer that you use. So usually when DNA replication happens in our body, at that time the primer is made up of RNA nucleotides out there, okay? But here it is going to be deoxyribose nucleotides. Okay, it is going to be deoxyribose nucleotides. It is not going to be ribonucleotides. It is going to be deoxyribonucleotides that you are going to use in the PCR. Always remember this. This is very, very, very important. Okay? Okay. Now, in the given diagram of PBR 322, identify the restriction site A, B, C and D. A, B, C and D. A is basically ampicillin resistant site. Okay, B is basically ampicillin resistant site. C is tetracycline resistant site. So B is ampicillin, C is tetracycline. What about A? A is the ORI site and this is the ROP site. So A is the ORI site. Yes, A is the ORI site. B is ampicillin resistant site. C is tetracycline and D is what? HIND3. D is HIND3. For this, if at all you want to know the answer for this very, very fast, you definitely have to remember this. Again, I've taught you in master stroke. Please remember. Okay, I can make it for you guys also. Listen, here you have the origin of replication from which replication is going to begin. Here you have the ROP site. This protein is super important for replication to happen. Okay, apart from that, you have ampicillin resistant gene over here. You have tetracycline resistant gene over here. Okay, now listen. There is one restriction enzyme which you call as PVU2, that is over here. Here you have two restriction enzyme site. One is BAMH1 and one is SAL1. Here also you have two. One is PVU1 and one is PST1. Two of them. Here you have three. One is going to be EcoR1, one is going to be CLA1 and one is going to be HIND3. So listen, 1, 2 and 3. One is here, two of them are here and three of them are there. Okay, here all of them are 1, 1, 1. Here it is 2 and here it is going to be 3 at the end. Always remember like that. Narrow P, you have PVU2, here you have PVU1. Cool? Cool. Yes. Now restriction site for EcoR1 enzyme is 5-GAA-TTC. Okay, 5-GAA-TTC3. So 5 comes first, then comes 3. Always remember, do not go and write this is the answer. Your answer for this question is going to be A. Cool? Cool. Now, next one. Fill in the blanks by choosing the appropriate word from those given in the bracket. The plasmid DNA acts as dash to transfer the piece of the DNA attached to it. The plasmid DNA acts like a carrier. What do you call the carriers as? Vectors. What do you call them as? Vectors. So onto the plasmid DNA, if at all I'm putting my gene of interest, it is going to carry it, is going to transfer it to the next organism out there. So which is why I'll call this as a vector. So your answer for this question is this. Next question. Which of the following is not a feature of a plasmid? Extra chromosomal? Listen, in a bacteria, in a prokaryote, it has its own genetic material. This plasmid which is there is an extra chromosomal. It is going to be circular. It is going to be double-stranded, not single-stranded. Independent replication. Why? Because it has origin of replication. So this one is the odd one out. This is not the feature of a plasmid. Okay? Yes. Cool. Next one. Agrobacterium tumefaciens is used as a vector for cloning genes in the case of plants, animals, bacteria or viruses. Agrobacterium tumefaciens is for plants because this is a generic bacteria that is there. It causes tumor in the case of plants. So if at all I remove the gene that is causing tumor and I put my gene of interest, basically it will still go and infect the plant, but this time transfers my gene of interest out there. You get it? So basically Agrobacterium tumefaciens, always remember, is there for plants. And for animals, you have retroviruses. For animals, you have retroviruses. For plants, you have Agrobacterium tumefaciens. Cool? Cool, ma'am. Next. Agrobacterium tumefaciens transform normal cells into tumor by delivering a piece of DNA known as. So listen, I'll talk about it then. So there is a plasmid. This plasmid is known as TI plasmid. TI stands for tumor-inducing plasmid. Why tumor-inducing? Because it has a segment of DNA which is known as tDNA. Transfer DNA. When this transfer DNA is transferred to the plant, basically it causes tumor in them. Okay? So Agrobacterium tumefaciens transform normal cells into tumor by delivering a piece of DNA known as what? tDNA. Known as tDNA. Very, 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 very important. Please remember it. Okay? Okay. It is also known as transfer DNA. Now, which of the following is the correct order of processing of PCR? First of all, PCR means you want to replicate the DNA, right? So DNA strand comes like this. You have to denature it. You have to separate it. That is denaturation. After denaturation, you have to attach the primers. That is known as annealing. After that, you have to extend the strand out there. That is known as extension. So first one is denaturation. Second one is going to be annealing. Third one is going to be extension. So over here, instead of DNA, it is DNE. Denaturation. I hear you. Just a second. Just a second. So instead of, okay, I'll not, okay. Let's not have anything because I was thinking something else. Okay. So denaturation, annealing, and then extension. This is your answer. Okay, denaturation, annealing, extension. The highest temperature is required for denaturation, then comes annealing, then comes extension. Okay, so denaturation requires higher temperature, that is 95 degrees Celsius. Then you lower the temperature in annealing, then you again increase the temperature, as in it brings out to 75 degrees Celsius in the case of extension. Okay, okay. Next one. Statement 1. Sticky ends are resulted due to action of restriction enzymes. Is it? That's how you get sticky ends when you cut with restriction enzymes? Yes. Stickiness facilitates the action of DNA polymerase. Is it? Now stickiness which is there, once you get sticky ends and once they join, okay, once they join, do you think that is facilitating the action of DNA polymerase? No. Basically, who is the one that will be fixing all the gaps out there at the end? It is DNA ligase. So stickiness is helping in the functioning of DNA ligase, not DNA polymerase. So I'll say this is correct, but this statement is false. Statement 1 is correct, but statement 2 is false. Yes. Okay? Okay. So the purpose of stirred tank bioreactor is, what is the purpose of stirred tank bioreactor, my dear children? Provide anaerobic conditions? No. Aerobic bacteria you're putting, then you put anaerobic condition. No, no. Purification of the product? Addition and proper mixing of preservatives? Availability of oxygen throughout the process? Yes. Which is why you have stirrer in the middle that is going to stir things. Okay, and it has flat-bedded impellers also. Got it? So your answer for this question is going to be D. Cool? Cool. Next. Identify the correct statement. Hint 2 cut the DNA at specific sequence of 6 base pairs. Hint 2 was the first discovered restriction endonuclease. Okay, and it cuts the specific sequence of 6 base pairs. That is also correct. Microinjection is a technique of introducing rDNA into the plant cell. No. With the help of injection without using a vector, can you transfer the DNA? Yes, but not into a plant. Why? Because plants have a cell wall. The injection would not enter. This you do in the case of animals. This is vectorless method in the case of animals. Usually the vector that we use is retrovirus. If at all we are not using retrovirus, basically we can use a vectorless method, that is known as microinjection. Over here you are not using plant cell, instead you should use animal cell. Now, biolistic is a technique of introduction of rDNA into animal cell. Biolistic is a method, it is also known as gene gun, because the plant cell has a cell wall and it will need a gun. That's why you use that, and it's not for animal cell, it is for plant cell. So this is wrong. So this is gene gun method, and basically you introduce recombinant DNA into the plant cell. Over here, you use the particles of gold and tungsten and you cover it up with the DNA and then you do the bombarding. Okay? Okay. Now, retroviruses are commonly used cloning vectors in plants. Retroviruses are commonly used cloning vectors in plants? No, it's in the case of animals. So this is also wrong. So over here, the only option that is correct is going to be A. Cool? Cool. Biotechnology principle and process, there are no 2-3 marker coming. That's why there's only 5 marker not discussed over here. Biotechnology principles and processes, my dear children, again you have 5 markers, so you have to start discussing this. So explain the process of preparation of competent host for transformation with recombinant DNA. Listen, competent host means a host which is ready to take up the recombinant DNA. So what is the process that I will be utilizing for making a host competent, for being ready? So in the case, this is usually discussed in the case of bacteria, and I'll tell you in bacteria what you do is, suppose you have the organism up there, and basically in this medium you also put the DNA which you want to insert, right? And this medium is going to be a medium that has calcium in it. Why calcium? Why calcium? That is the first and the foremost thing that you should be doing. So okay, so bacterial cells are treated with divalent cations, as in basically CaCl2, to make them competent. Over here, when I say divalent cation, you should know the charge is over 2+. That is why I call it divalent cation. It is calcium. The calcium ions are going to neutralize what? The negative charges. On what? On the DNA and also on the cell membrane. Listen, the DNA and the cell membrane, both the charges have to be neutralized. Otherwise, do you think they will get inside? No. Basically, they will repel. There is no chances of the DNA passing through it. So we have to make sure that the neutralization happens, and that is one major role of Ca2 plus ions, neutralize the negative charges on the DNA and the cell membrane. And also need to understand the second function of calcium, and that is it increases the permeability. That means it increases the permeability. There will be some pores in the cell membrane that is created because it is being treated with the calcium ions out there, right? So now there are some pores over here, negative charges is also, it is neutralized over here, everything is okay. There are few pores over here. The next thing that you have to do is, first of all, you have to keep in a medium that is having ice. Okay, now you shift the thing into a hot medium. That means basically you are giving a heat shock. And then again what do? You treat it back with ice. So the thing is that whenever you do that, there is basically opening of the pores very much, okay, and there will be closing of the pores when you treat it back with the ice. So since there is temperature change out there, the permeability of the membrane is going to be changed, which is exactly why whenever it opens, the recombinant DNA is going to get inside, and basically when I put in the ice, it will be closed, which is why it cannot move back outside. You get it? So this is the thing that we generally use. So the mixture of cells and the DNA is then put into ice, then given a heat shock, 42 degrees Celsius for 90 seconds, just for 90 seconds, and then back cooled on ice. This allows the recombinant DNA to enter the bacterial cell and not to move out as well. This is how you make the cells competent generally. Okay, this is how you make a bacterial cell competent. Works. Now, next thing is that mention the restriction enzyme that can be used for insertional inactivation of antibiotic resistant gene over here. So, oh my dear children, listen to me. This is a plasmid, right? This thing that you see is a plasmid. That is PBr322. PBr322 has an ORI site, that is origin of replication. Apart from that, it has ROP site. This protein is super important for the replication process to happen. Always remember that. Okay. Now ORI ROP site is done. Here you have two sites. If it has this gene, it allows it to be resistant to ampicillin, as in basically I can grow an organism that has PBr322 and it will still survive. Why? Because it has a resistant gene, ampicillin resistant gene. Ampicillin is what? An antibiotic. So if when I have a gene against it, is basically like having a superpower against it. So this is ampicillin resistant gene. This is tetracycline resistant gene. This is another antibiotic resistant gene that is out there, right? Okay, now listen to me. These are enzymes, these small things that is seen over here, these are enzymes, and these enzymes have specific site. That means these are the places where they will make a cut, and if at all needed, they will, whenever needed, we can insert the DNA at this particular site out there, right? So PVU2 has a site in the ROP site. BAM-H1 and SAL1 have a site over here. So if at all I am cutting anything with BAM-H1, I'll make a cut over here, I'll insert my new gene over here. When I insert my new gene, my dear children, do you think it will be tetracycline resistant anymore? No. Why? Because at this place I am inserting something else. I have lost the older gene which was making the cell resistant to tetracycline. Now is it tetracycline resistant? The answer is no. So if at all I want to make sure that the plasmid does not have tetracycline resistant, what do I have to do? I basically have to cut this particular plasmid with either BAM-H1 or SAL1. Now, suppose I do not want the organism to have ampicillin resistant. Again, what do? I can cut it with PVU1 or PST1, because anyway if I make a cut over here and I insert a gene over here, it would not have the older gene which is ampicillin resistant. This process of inserting something and inactivating the other thing out there is known as insertional inactivation. What do you call it? Insertional inactivation. I'm inserting something, which is why something else is getting inactivated. So if at all I want to inactivate ampicillin resistant, I have to use either PVU1 or PST1. If at all I want to inactivate tetracycline resistant, then I have to use either BAM-H1 or SAL1. These things are something that you have to remember. Now there can be a question, that is, basically I do not want to inactivate any antibiotic gene out there. Which are the sites that I can use? I can use EcoR1 site, I can use CLAV1 site, I can use HIN3 site, because they are not coming under what? Any of the resistant genes out there, right? So EcoR1, CLAV1, HIN3 and also PVU2 But if at all you're cutting with PVU2, you will not get replication protein being made, and that is super important for replication process to happen. So please know that. Okay, so your first question is, mention the restriction enzymes that can be used for insertional inactivation of antibiotic resistant gene. So what are the restriction enzyme that I can use for insertional inactivating these two resistant genes out there? I can use PVU1, PST1 for ampicillin resistant, okay, BAMH1 and SAL1 for inactivating tetracycline. This is your answer. So exact thing is written over here. Got it? For that you need to know the location. We will draw it once and we'll remember it. Okay. Now one more question is there. How many cloning sites are present in the given PBR 322? As in how many sites for the enzymes are present in PBR 322? So how many sites are there? Let's see. 1, 2, 3, 4, 5, 6, 7, 8. 8 cloning sites are there and you can just write it. Number of cloning sites in PBR 322 is 8. 8 unique restriction sites are there and these are the restriction enzyme sites. Got it? Please write this. And since if at all you are getting confused by any chance, I can draw it and I can help you remember this. So this is your PBR 322. Here what do you have? You have origin of replication. Okay, here what do you have? You have ROP site. Here what do you have? You have tetracycline resistant gene. Here what do you have? You have ampicillin resistant gene. Done? Okay. Now we start making things. Here my dear children, you have PVU2. What do you have? You have PVU2. Here you have BAMH1. Here you have SAL1. 1. BAMH1, SAL1. Here you had PVU2, right? Here you have PVU1 and here you have PST1. PVU1, PST1. Okay? 1 here, 2 of them here, 3 will be over here. So the first one that you have is EcoR1. This is very famous restriction enzyme. Then you have a dundee like this. This is known as CLAV1. Then you have something like this. This is known as HIND3. So listen to me very, very carefully over here. 1, 1, 1, 1. Everything is 1. Here it is 2. Here there it is 3. Okay, 1, 1, 1, 1, 1. But here it is 2 and there it is 3. Here you just have 1 restriction enzyme site. Here you have 2. Here you have 3 of them. 1, 2, 3. You can remember like that also. This is super, super important for you to know. And remember, an enzyme will be having only 1 site over here. Okay, a particular restriction enzyme will be having 1 cloning site. Multiple restriction enzyme can have multiple cloning sites, as in basically when I say PVU2, do you see PVU2 elsewhere? No. It will only make a cut over here. Suppose you had a PVU2 site elsewhere also. What is going to happen? It will make a cut and will destroy the whole plasmid out there. We do not want that to happen. So per restriction enzyme, only 1 cloning site is there. But in one particular plasmid, you can have multiple cloning sites of other restriction enzymes. Please know that also. Done? Done, ma'am. Perfect. Clear. Let's go ahead. Now, give reasons for the following. You have to give reasons. Restriction enzymes are called molecular scissors. Why do you call them molecular scissors? Restriction enzymes, these enzymes that we saw, you call them molecular scissors because they are going to cut your DNA. There are 2 types of restriction enzyme. One is endonuclease, one is exonuclease. Endonuclease goes within the DNA strand and makes a cut in the middle. Exonuclease cuts towards the outside. Anyway, isn't it cutting the DNA? Yes. Which is why you call them restriction enzymes as molecular scissors. So, because they recognize and cut the DNA at specific base sequences, that is recognition sites, just like scissors cut at precise points, you will call them as molecular scissors. And also since they are cutting in the big molecules such as DNA, RNA and things like that, that is why you call them molecular scissors. You please write that also. Okay, now DNA ligase is used in genetic engineering. Why is DNA ligase being used? So when I make a cut and I put this particular gene somewhere else, do you think it will slowly fit in nicely? Suppose I've cut this particular plasmid and I am inserting a new gene over here. Do you think it will perfectly stick? The thing is that you will need something to glue it up really tight, and that is the role of DNA ligase. It is there for ligating things. Okay, it is there for ligating things. So DNA ligase is used in genetic engineering because it joins the 2 DNA fragments by forming what? Phosphodiester bonds. It helps in the formation of phosphodiester bonds between their sugar and phosphate backbones, helping to seal the foreign DNA into the vector. So these key points are super important. Over here also, why they are known as molecular scissors? Because they cut the molecules. Molecules, big molecules such as DNA. Also write this, very important. Here they form the phosphodiester bond, sealing the sugar phosphate backbone out there, right? Next thing, Chilled ethanol is used in DNA isolation. So suppose, my dear children, I want to isolate the DNA from a cell. What do? I take a test tube over here and I put chilled ethanol. In this chilled ethanol, your DNA will be what? Precipitating out, or basically moving out, separating out. That is why I used chilled ethanol in DNA isolation. Okay, so because DNA is insoluble in cold ethanol, so you have to explain like this. It is insoluble. Okay, DNA which is there, it is insoluble, and since it is insoluble, do you think it will mix up? No, it will not mix up, rather it will be precipitating out from the cell extract, making the DNA visible and easy to collect. So this point is important over here. DNA is insoluble in cold ethanol. Works. Okay, smaller DNA fragments move farther in a gel slab. So if at all you have seen gel electrophoresis, my dear children, it goes like this. You have wells over here, you have cathode over here, and you have anode over here. Cathode and anode. Always remember, cathode is negatively charged, anode is positively charged. Anions are negative, but anode is positive. Always remember. Your DNA ends with A, so it moves towards A. You can remember like that. DNA is also negatively charged. Negative will repel. What will happen? They will separate. The smaller ones which are there, they move the farthest. The larger ones which are there, they will move slowly. So who moves the farthest? The smaller one. Why? Why do they move faster? Because they will be having less inhibiting effect. Always remember, in gel electrophoresis, you use agarose gel. And the DNA is not going to float around the agarose gel, it is going to show the sieving effect. It moves within the agarose gel. Sieving effect is something that you should not be missing, right? When it shows the sieving effect, it gets the least hindrance. It will be able to move very easily. That's the reason why smaller fragments would be able to move faster, or basically farther. So because during gel electrophoresis, smaller fragments face less resistance from the gel matrix and therefore migrate faster and farther towards the positive electrode, that is anode. Now one more very important thing. Who moves first? This is also a question. So sometimes we confuse this. When I say smaller fragments move faster, people confuse it. Does it move first? So from the well, it can be either the longer fragment or basically the shorter fragments that can move first. We never know. It can be anything. So if at all the question says, the first one to move is a smaller fragment, there is no clarity regarding that. The one that moves the farthest is basically the smaller fragment. That is something very sure of. Cool. Now thermostable DNA polymerase is used in polymerase chain reaction. Thermostable DNA polymerase, as in a DNA polymerase which can resist high temperature, is being used in polymerase chain reaction. Why do you think so? Because in polymerase chain reaction, my dear children, there is denaturation. Your DNA, when you say that you have to replicate the DNA, first of all, you have to separate them. And for separating them, you need a degree that is 95 degrees Celsius, which is too hot. Okay, after denaturation, basically, I have to do the annealing process. It also happens at high temperature, around like 55-65 degrees Celsius, which is lower than this, but yes. And then elongation, which again requires, elongation of a new strand again requires almost 75 degrees Celsius. Now, for making a new strand at this particular temperature, obviously you have to make sure that the DNA polymerase used is what? Thermostable. It can resist high temperature. Okay, that is the reason. So because enzymes like Taq polymerase, as in the DNA polymerase which we extract from Thermus aquaticus is known as Taq polymerase, can withstand high temperature used to denature the DNA strands, allowing the PCA cycle to continue without the enzyme breakdown. Okay, so you have to make sure the enzyme does not break down at these high temperatures out there. It's not just for denaturing the DNA strands, but also for the process of extension and everything, you need high temperature, right? And you have to make sure that the enzyme does not break down during these conditions. Now, here I have a picture so that you can remember things a lot better. This is chilled ethanol and your DNA is insoluble, which is why it is moving out. Okay, here gel electrophoresis, the smaller fragments, if at all you are able to see, they move the fastest. Here this is DNA ligase. It's like a glue. It glues up the things by making the phosphodiester bond. Cool ma'am, perfect. Next, genetic engineering is a core technique of biotechnology. In the background of this, complete the following table. Okay, let's complete the following table. Over here, what do you have? Gel electrophoresis dash visualization of the DNA fragments. Here the things which are used are being written. So gel electrophoresis, in gel electrophoresis, what do you use such that the DNA becomes visible to you? You use a stain which is known as ethidium bromide. What do you use, Baba? You use ethidium bromide. So please write that, ethidium bromide. Now next thing, dash DNA polymerase is being used and dash is the technique's name. This is going to be a technique's name. Okay, no, this is going to be the technique's name. Over here, gel electrophoresis, gene gun and everything is written. So this is going to be the technique. This is going to be the use of it, right? The use of it. So basically, what is the technique where you use DNA polymerase? You are going to basically call this technique as polymerase chain reaction PCR. Okay, what do you get after the end of it? You get amplification of the DNA, right? Your DNA is going to get amplified. So amplification of DNA. When these type of question comes, read to the entire question and understand what is being asked over here. Do not make it ulta sida. Okay. Now gene gun method is there. Gene gun is basically a vectorless method. A vectorless method for transferring DNA inside what? The plant cells. Inside the plant cells. How do you transfer it inside the plant cells? So plant cell has a cell wall, which is why you use a gun over here. Otherwise you could have just injected, just like animal cells, you do micro injection. But in plant cell you have gene gun, where what do you take? That is the next question. What do you take over here? What is the thing that you use over here? You take, okay, you take particles of gold and tungsten. Particles of gold and tungsten. And what do? You wrap it around with the DNA. You wrap it around with the DNA that you have to insert. What will happen later? Transfer of recombinant DNA into the host nucleus will be happening up there. Gene gun is also known as biolistic method. Please do not forget it. This is found only in the case of a plant cell. Only in the case of a plant cell. Got it? Got it, ma'am. Now isolation of DNA is there. What do you use for isolation of DNA? That such that it precipitates. It is chilled ethanol. So E is going to be chilled ethanol. Got it? Perfect. Okay. So exact thing I have written over here. You can just go through it once more. The next question that we have is, given below are the diagrams of plasmid A and B. This is plasmid A and this is plasmid B. Now observe meticulously and answer the questions that follows. Okay, let's see. So this is PBR 322 and this is another plasmid which is given in the NCERT. LAG-Z gene, okay, blue-white colonies, you might have heard about it, right? Now which plasmid is or are you going to select for the cloning NY? For cloning NY. What do you think is the answer for this? Which one will we use for cloning? Listen to me very carefully. I will be using plasmid B for cloning. The reason is very simple. In plasmid B, suppose the LAG-Z gene over here, I am inserting something else here at this particular position. What is going to happen? LAG-Z gene is not going to produce the same thing that it was producing earlier. What was it producing earlier? It was producing beta galactosidase. What was it producing? It was producing beta galactosidase. Now this beta galactosidase, which is usually being produced, what does it do? If I add a chromogenic substance as in a coloring agent, this beta galactosidase will act on it and give a blue color thing. Now suppose I am inserting some gene over here. I am inserting my gene over here, and then to check whether the plasmid has got the gene or not, it has become recombinant or not, what do? Basically again I'll put the chromogenic substance. If at all there is no color change happening, I'll just easily get to understand that that over here I have got my gene being inserted, because there is no color change. Otherwise it would have given me a blue color thing, right? Over here the steps are pretty much very long and I'm going to explain that, do not worry. So the thing that I will prefer is plasmid B, because it is very easy for me to use. Plasmid B is selected because it has LAG-Z gene, which helps to easily find the recombinant colonies. When a foreign DNA is inserted into the LAG-Z gene, the gene becomes inactivated. What happens? It becomes inactivated. This allows blue-white screening. White colonies contains recombinant DNA. The blue colonies contains the non-recombinant DNA. So basically it's very easy for me to understand. Now, what is insertional inactivation is the next question that is given. Insertional inactivation is common for both of them, where what is happening? It means the gene stops working because a foreign DNA is inserted into it. Now if you want to give an example, you can give. Example, in plasmid B, when LAG-Z gene stop producing the enzyme beta galactosidase, after insertion of what? The foreign DNA out there. That is why I get the white colonies, right? Right. Now, will the number of culture plating be same or different over here? Comment. What do you mean by that? So here, my dear children, where is it? So here in this particular case and this particular case, the number of plating that you require for finding the recombinants, will you think it will be the same or different? That is a question and we have to find the answer. For this, you need to have patience. For understanding this, you would require patience, and also this is a very important concept which is being asked, and you guys mostly get confused between this. So make sure you listen to me very, very carefully. So it goes like this, okay? This is the condition for plasmid A, and this is the condition for plasmid B. So I'll make the plasmid B first, okay? In plasmid B, my dear children, suppose here you had lack Z gene, okay? And here, now in this particular case, what do you have? You have the gene of interest. So this is going to be what? My recombinant DNA. This is not a recombinant DNA. Now what do? Basically, I put this inside a cell. Similarly, I put this also inside a cell. Later, I have a plate right over here, where I am going to add the chromogenic substance. Chromogenic substance. Okay. Here the cells which have the plasmid, the older plasmid out there, it will look like this. And here the cell which have the new plasmid, it is going to look like this. But can I identify just on the basis of the looks? No, obviously it is not visible to me. DNA, how will I get to know that? So over here, when I put the chromogenic substance, the thing is that this one will be turning blue in color, whereas this one will not have any change. It will be basically white in color. I say colorless. Over here it is blue. Why? Because still LAC-Z gene produces beta galactosidase, and it is going to act on chromogenic substance, which is why I get blue color colonies. How many plates do I require? How many number of plates did I require to basically find the recombinant DNA? Only one plating. In one plating itself, I got to understand that these ones are the white ones. Basically, it has a recombinant DNA. And this one is what? The non-recombinant. So this has been very, very simple for me. Now, let's talk about plasmid A. Let's talk about plasmid A. In that, my dear children, first thing that you need to realize is that you have to, so there is one cell over here, there is another cell over here, and there is one more cell, suppose. Okay, this cell, or basically I'll say, okay, this is ampicillin and this is tetracycline gene. Okay, this is another plasmid, and this plasmid has ampicillin over here, and the blue one is the gene of interest. So my first task is to find the transformants, or basically I'll write in yellow, it will be easy for you to understand. The first task is to find the transformants, as in basically a cell did not have this plasmid before. Okay, I have to make sure that they take up the plasmid. How basically I do the competent DNA technique by doing heat shock and this and that, calcium and stuff. So some might have taken, some might have not taken the plasmid, some have not taken, some has taken. We know because it is clearly visible, but how do we get to understand when we're doing it under the lab? We cannot see the DNA out there, right? So for that, the first plating we do is with ampicillin. We do the first plating in ampicillin. Suppose when we plate it with ampicillin, this is not be able to survive. Why? Because this does not have the plasmid out there. It does not have a normal plasmid. It does not even have the recombinant plasmid. Listen, some plasmid might have taken up the gene of interest, some plasmid might have not taken up the gene of interest. Anything can go inside the cell. It can be this or it can be this also. Okay, so suppose over here it has not taken any plasmid at all. So this is going to be what? A non-transformant. This is not transformed. Transform means basically to take up something new. It has not taken anything new. Okay. Now over here, these two are transformants. How do I understand this? Because this can survive and this can survive. Why can they survive? Because it has ampicillin resistant gene over here also. So what do I get to understand? That these two things which are there, these two cells are transformant and this one is the non-transformant. This is going to be the first plating. The next plating that I'm going to do is with, wait. So I have these two guys now, so I have to make that diagram. Let's do that first. Here, this one is this. Here, this one is this. Cool. This is my gene of interest and this does not have it. So basically I can clearly see and as of now tell that this is the recombinant cell with the recombinant DNA and this is the non-recombinant. Both of them are transformant because both of them got the plasmid which was initially not there. Both of them are transformant, but this is non-recombinant because it does not have the gene of interest and this has. Again, how do I identify it? The next plating that I am going to do is going to be in tetracycline medium. When I do in tetracycline medium, what is going to happen? This is second plating, okay? This is the first plating. This one will keep on increasing their number. Why? Because, ayyo, didn't go, my God. It went. Okay, anyways, that one which was there, it will be, okay, calm. This one which was there, this one was non-recombinant. Why? Because it does not have this. It has the recombinant DNA. When I grow it in tetracycline medium, which one is able to grow? This one. Why? Because it has a tetracycline resistant gene. This one will not be having. So which one will I select? This one is something that I will be selecting. The one which is not able to grow is the one which has my gene of interest. It is not able to grow because it is not having tetracycline resistant. It has become insertionally inactivated. So this is my thing that I would take. You get it? For this you require 2 plating, whereas in the other case you just required 1 plating out there. So will the number of culture plating be the same or different? Comment. The number of plates will be different for both plasmid. For plasmid A, ampicillin tetracycline needs 2 plates, one with ampicillin for finding the transformant, such as who has taken up the plasmid, and one with tetracycline to find which one has taken up the recombinant plasmid, right? For plasmid B needs only 1 plate with X-gal. X-gal is the chromogenic substance which is there. Okay, X-gal is the chromogenic substance onto which beta-carotidase is going to react. So white means it is recombinant. That means it has been insertionally inactivated. There is no production of beta-carotidase. The blue means it is non-recombinant. Hence, plasmid B is better because it allows easier and faster selection of recombinants in just 1 step. Got it? That's all. Perfect. Now, we go over here. Study the diagram given below and answer the question that follows. What is EcoR1? What is EcoR1? It is a restriction enzyme. It is a restriction enzyme. And how did it get its name? Listen, EcoR1 stands for Escherichia coli, E. coli basically. The first letter of Escherichia is being taken, that is E. Coli, 2 letters. The species name, 2 letters have been taken, that is by CO. And R stands for the name of the strain and 1 stands for the number of discovery, on the basis of number of discovery. So what is EcoR1? It's a restriction endonuclease. Endonuclease is something that will go cut in between, okay, in between the phosphodiester bonds within the DNA itself. So EcoR1 is a restriction endonuclease enzyme isolated from Escherichia coli. It recognizes specific palindromic sequence. It has a specific site where it will go and make the cut. What is the site? 5-GAATTC 3-. 5-, this is very important. GATTC, okay, it is 5-GAATTC 3-. This is the site that we have over here. And it cuts between what? Cuts between G and A. Here, this is the site. Here it makes a cut between G and A, and it's a palindromic sequence. Why? Because in a DNA, suppose I have GAATTC over here on the other side. This is 5-3-. Similarly, from here also, it will be GAATTC 3-. Always remember, Always remember, this is super important. It's in 5-3-. Do not make a mistake when there. And it cuts between G and A. So it makes a cut over here, it makes a cut over here. Now, how is the action of exonuclease different from that of endonuclease? I told you, exonuclease are going to cut the DNA from the ends, whereas exonuclease cuts the DNA from the end. Endonuclease cuts the DNA from within this DNA itself. Okay, it removes nucleotides one by one from the terminal ends. It breaks DNA at specific recognition sequence. So first thing that it does is that it goes on and search for the recognition sequence, then makes a cut out there. Example, enzyme that trims the DNA ends. Example, EcoR1, HIND3 and things like that. Which one was the first restriction enzyme to be found? The first restriction enzyme to be discovered? It was HIND2. It was HIND2. HIND3 is found in PBR 322. HIND2 is the first one. It also have a recognition sites of 6 base pairs, just similar to this. Okay. In E. coli also you found the restriction enzyme, but you could not discover it completely. You just knew that there was something out there. But the first one to be discovered was HIND2. Please know that. Now, how are sticky ends formed on the DNA strand and why are they so called? Sticky ends are found when a restriction enzyme such as EcoR1 cuts the DNA. Okay, I'll just show it to you. So over here I had the picture, right? Why do you get sticky ends? So suppose, my dear children, you have the vector DNA, as in the plasmid DNA. Okay, suppose the vector over here is the plasmid, and you have your foreign DNA. Now these guys have to come over here. That means this side and this side should come. This side and this side could come in contact, right? So what do? We cut it with the same restriction enzyme. Plasmid, I'm going to cut it with EcoR1, and in plasmid, GAA-TTC site is being searched. It makes a cut over here between G and A. It makes a cut over here between G and A. Okay, done. What is going to happen? This L-shaped thing is going to go. This L shape, when I make a cut over here and I make a cut over here, this L-shaped thing is free over here. It's not attached to any sort of DNA. So this one will go. I'll get something like this, right? This is my vector DNA, as in my plasmid DNA. Similarly, in my gene of interest also, I made a cut using the same restriction enzyme. Here also you had GAA-TTC site. Again, it cuts between G and A. The one which is not attached to the plasmid, or basically not attached to the DNA, is being cut. The L-shaped thing is being removed. Now, when I put one end and another end of the vector, this was the end of the plasmid, this is the end of the foreign DNA, and I keep it close. What is going to happen? They are going to be base pairing to each other. Why? Because they are complementary to each other. They are waiting for a hydrogen bond to be formed, and since both of them come together, you get everything fine. Now, for the last step, you need DNA ligase, but sticky ends formation is super important over here, which is why we cut the plasmid with the same restriction enzyme that we use for cutting the foreign DNA also. So sticky ends are formed when a restriction enzyme such as EcoR1 cuts DNA staggeredly between the G and A. This produces single-stranded overhangs. These are single-stranded overhangs, as in there are extra things which are there. They're complementary to what the sequences are there. They are called sticky because these ends can easily pair with the complementary basis of another DNA fragments, helping in joining foreign DNA with the vector DNA. Got it? That's all. Okay, that's all. As simple as that. And make the diagram and show it to them. It will be really useful. Now write 3 vector-free techniques that can be utilized to transfer recombinant DNA into a ready host cell. What is a vector-free technique that we can use without any vector? Any idea? So first one is micro-injection. Micro-injection is basically where I am going to so this is an animal cell. There is no cell wall over here. I can just use an injection which is have my recombinant DNA and insert into the nucleus out there. That is known as microinjection. This is the case for animal cells where I do not want a vector. Okay, so directly injecting the recombinant DNA into the nucleus of an animal cell using a fine needle. That's all. Now the next thing that we have is biolistic or gene gun. I just told you for plant cell you cannot insert like this because you have an extra cell wall, which is why you will need something else for entering things, right? That is known as gene gun method. Here high velocity tungsten and gold particles are coated with DNA and are shot into the plant cells. That is the exact thing that we do. Now the third thing is electroporation. Electroporation is basically electric pulses are created such that it creates temporary pores within the cell membrane, allows the DNA to enter. This is not given exactly in your NCERT, but please know that. Okay, otherwise you also have the calcium chloride medium where you are basically using divalent cations to make the bacterial cells take up the DNA. You can mention that also. That also works. It is also a vector-free technique which is there. Now, agrobacterium tumefaciens as a natural vector. So, listen to me carefully. The usual vector that we have is a plasmid. Fine. But in some cases, this is not the case. In usual, plasmids can act like a vector, but not all vectors are plasmid. We have vector-free techniques. We have some other vectors also. For example, in the case of plants, we use Agrobacterium tumefaciens. Okay, this is in the case of plants. In the case of animals, we have retroviruses. Always remember, this is what vector that we have for animals. You know, HIV is there for transferring things. Similarly, disarmed retroviruses are being used for transferring things into the animal cells. Please have that logic in your head. Here you have been asked about Agrobacterium tumefaciens, which is a natural vector in the case of plants. Why do you call it as a natural vector? That is a question. So Agrobacterium tumefaciens, which is a bacteria, it has a plasmid. You call this plasmid as tumor-inducing plasmid. What do you call it? Tumor-inducing plasmid. I can also call it as TI plasmid. How does it induce tumor? Basically, it has a segment over here, and this segment is known as tDNA, that is transfer DNA. Basically, this organism goes and infects a plant. It goes and infects a plant, and when it does, it is going to transfer this tDNA inside the plant. And when it does, the plant will be developing what? Tumor. Now, suppose I'm using the mechanics over here for removing the tDNA part and basically inserting my gene of interest. What is my gene of interest over here? Any gene that I want to insert. Then I make the Agrobacterium tumefaciens go infect. Again, my gene of interest is going to be transferred out there. That is why it is a natural vector that is being used in the case of plants. Again, if you want a vector-free technique for plants, you have biolistic. If at all you want a vector for animals, it is retrovirus. Again, if you want a vector-free technique, that is a microinjection. Please know about that. Okay. So Agrobacterium tumefaciens is a soil bacterium that naturally causes tumors, that is crown galls in plants. It carries TI plasmid that transfers a segment called tDNA into the plant cell, transfer DNA into the plant cell. In genetic engineering, the TI plasmid is modified to remove the tumor-causing genes and used to transfer the desirable genes into the plants. Hence, it acts as a natural genetic engineer or natural vector for the gene transfer in the case of plants. Done. That's all. Cool. Now, write a labeled diagrammatic representation of recombinant DNA technology. You have to explain everything. So, what do you do first of all, my dear children? You have the foreign DNA and you have the vector DNA. It can be a plasmid. It can be anything, but a plasmid, suppose. So, always remember, okay, I have discussed this so many times. What do? With the restriction enzyme, you cut the gene segment. Similarly, with the same restriction enzyme, you're going to make a cut over here also. Done. Now, with the help of DNA ligases, you're going to join this. You have got the recombinant DNA molecule. Now, what do? Recombinant DNA molecule is made to put inside the host. This is known as transformation, right? Now, basically, the host which is there, it is keeping on dividing, and it has the recombinant DNA. That is the whole thing. That's the whole thing all about. So, if you are asked about this, make the diagrammatic representation of this. If you are asked to explain the steps of each and everything in detail, you have to explain each and every step out there. First one is isolation of DNA. This is something that we have not discussed. So, how do you isolate the DNA? Suppose you are isolating the DNA from a fungi. A fungi will be having, so there are three cases. Suppose it's a fungi. Fungi has a cell wall and the cell wall is made up of chitin, which is why you will need what? Chitinase. You will need chitinase. Suppose this is a plant. A plant has cell wall made up of cellulose. So what do you need to break it down? Cellulase. Okay, suppose it is of a bacteria. A bacteria has the cell wall made up of peptidoglycan. So what do you need? You need lysozyme, and lysozyme is an enzyme that is there in our tears also. Okay, so please remember. So you need enzymes like this specifically for specific service thing. If at all you are asked about fungi, please know you would need what? Chitinase. If you are asked about bacteria, you need lysozyme. If you asked about plants, you would need cellulase. So please know all about that. There's one question that I saw, I think I have not included over here. I saw it somewhere in the previous year, where they gave you number of things which are used in restriction engineering, genetic engineering technique, and they asked you, if at all you want to insert something into a plant, what do you need? What are the things that you would need? So please know all this thing. In a plant, you would need cellulase. Apart from that, after getting inside the plant, you have the cell nucleus which has RNA and protein also. So you have to remove the RNA with the help of RNase. You have to remove the proteins with the help of proteinase. You have to remove the fat with the help of lipase. And finally, you have the DNA that you have to take it up, right? So please know about that. Okay. So after isolating, you cut the DNA using restriction enzymes. And after that, you separate it with the gel electrophoresis. How do you view it? You use ethidium bromide bromide to view it under the UV radiation, or basically autoradiography machine. Please remember, it gives a orangish red color, bright orange red color, ethidium bromide. Okay, and it will be only visible under UV radiation. So please know about that also. Now, insertion of the desired DNA into the vector. First of all, you separated the fragments. Now you took the fragments out there and you put it inside the vector. Now what do? You introduce the vector DNA into the host cell. So you put it inside the vector, right? Now this vector has to be put inside the host cell. So for that, what do? You use microinjection, biolistic, whatever method is there. Now after that, you do selection of recombinants, like with the help of antibiotic resistant gene or blue-white colonies, whatever is there. Finally, you check for the expression. And lastly, you do downstream processing. Downstream processing has two things: separation and purification. Okay, it is having separation and purification. Separation and purification, as in basically, if at all you want proteins, it's not like just proteins are going to be made. Lot of other things are also going to be made. You have to make sure that it's separated from everything. Apart from that, you have to purify it, and later you would also have to preserve it. Okay, so separation and purification are the two major things that comes under downstream processing. Later you would have to do the other things as well. For example, adding preservatives, do the quality checking and everything. All of them comes under downstream processing. So these are the few steps. Please know about that. As I told, give yourself a chance that if at all I want to insert a gene inside a plant cell and I'm taking this gene from a bacterial cell, what are the things that I will be needing over here in this entire process? You should know about what are the basic things that you will require. Okay? Yes. Biotechnology principles and processes, my dear children, again, you have 5 markers, so you have to start discussing this. Fine. Now we enter biotechnology and its application. The 4th last chapter, and if at all you have reached until here, then write level 5, crust. Okay, let's begin. Given below are the steps in gene therapy of patient with adenosine deaminase deficiency. So they do not have the gene for adenosine deaminase, which is why we need to insert it into what? Into the lymphocytes, the white blood cells. Okay, specific white blood cells. So lymphocytes, first of all, have to be taken from the patient, right? So lymphocytes are collected from the blood of the patient. This is going to be the first step. Third one is the first step. So I have 2 options like that. I'll cut off these 2 options. Now I'll check my answer with this. I'll check my answer with the options. After 3, it is 4. What is 4? The genetically modified lymphocytes are infused back into the patients. No. In between that, obviously, there will be a lot of other things. You have just taken the lymphocytes. You have to genetically modify it, no, first. So after 3, 1. 1 is what? Lymphocytes are grown in a culture outside the human body. Yes. So they are taken first and then they are going to be cultured. Fine. After culturing them, what do? You are going to alter with the functional ADA gene. That means you are going to put that gene which is going to produce adenosine deaminase. Correct? You did the modification. And then basically last thing that you do is genetically modified lymphocytes are infused back into the patient. So that is also correct, which is why your answer for this question is going to be C. It is going to be C. Got it? Clear? Clear. Now, the next thing that we have up over here is what? The next question. The plants developed by the fusion of protoplast of 2 different plants are. What do you mean by protoplast? Suppose there's a plant cell and I'm removing the cell wall. Over here, I am not putting the cell wall, I'm just removing it. And what is left behind? Basically, if cell wall is not there, the thing that is left behind is going to be known as a protoplast. Is going to be known as a protoplast. And when I am fusing 2 protoplasts together, what do I get? What do I get? When I'm fusing 2 protoplasts together, I get hybrids, somatic hybrids. So this can be of tomato, this can be of potato, both of them fused together, I get pomato. So this is what? The somatic hybrids that I am getting. So your answer for this question is going to be B. Got it? Clear? Okay? Yes. Soma clones is a result of tissue culture. Whenever you do tissue culture and when you get product on the basis of that, that is known as soma clones. All of them would be genetically similar. Here it's not like that. Okay. Identify the incorrect statement. The transgenic cow Rosie produced milk enriched with alpha-lactalbumin. Yes, it is a human milk protein, but you're making a cow produce that by inserting the gene. That is correct. So, Bt toxin genes Cry1AC controls corn borers. We'll come to that. Human protein alpha-1 antitrypsin is used to treat emphysema. Yes, alpha-1 antitrypsin is used to treat emphysema, which is a respiratory disorder, and it is since animals cannot repeatedly produce too much copies, we make the since human beings cannot produce too much copies, we make the animals produce it. Okay, so that is also correct. The commercial production of human insulin was made possible by the transgenic E. coli. Yes, E. coli was being used for production of human insulin. That is also correct. Bt toxin gene Cry1AC controls corn borers. No. So Bt toxin, okay, Bacillus thuringiensis has a gene that is going to code for the toxin, and the toxin is very much specific. So the genes are also specific. There is Cry1AC and there is Cry2AB. Okay, and there is Cry1AB. Always remember, easy way to remember. First letter starts with C. Sorry, first word starts with C, cotton. Second word starts with B, bollworm. Since this is a bigger name, you need 2 genes for that. Okay, you remember like that, chumma I am saying. Okay, now Cry1AB, there is only 1 gene, and this is for corn borers. This produces toxins for corn borers. So only 1 is there, starting letter is C, and the next is B. Okay, here also it is CB only, but there it is bigger name, so you need 2 genes. You remember like that. Why I am telling you this is sometimes you might write 2AC over here. I do not want you to do that. First letter C, second letter B in cotton bollworms. Here it is going to be Cry1AB. Got it? That's all. So the answer for this question is going to be, okay, this is the incorrect one. Next one. Which of the following statement is correct with respect to synthesis of insulin? Synthesized as proenzyme and needs to be processed to become functional enzyme. You're talking about insulin. Is it an enzyme? It's a hormone. So this is wrong. Synthesized as prohormone and needs to be processed to become functional hormone. Yes, it is a prohormone, immature form of hormone, and it needs to be processed to become functional hormone. Synthesized as proenzyme and needs to be processed to become functional prohormone. Enzyme and hormone? Wrong, Papa. Read the options carefully. Synthesized as prohormone and needs to be processed to become functional proenzyme? Again, wrong. So you just have one answer, and that is going to be this, B. Cool? Cool. That's all. Cool. Now, crystals of Bt toxin produced by some bacteria do not kill the bacteria themselves because. So, Bt bacteria produces toxin. Why does it not kill its own self? Because it does not have the same, what do you call it, is in an inactive form inside its body. It becomes only active when you provide the ambient temperature, or basic, not temperature, the pH. Alkaline pH. Alkaline pH is found in the case of inside the insect. So once this bacteria is inside the insect, that's when the protoxin transforms into the active version of toxin. So it's basically because the toxin is inactive inside the body of the bacteria. You need alkaline pH, you get it only within the insect. Cool? Yes. Okay. Statement 1. The plants genetically identical to the original plants are known as soma clones. Is it? Genetically identical, as in I have one plant, with the help of tissue culture, I took some part of it and I did tissue culture, which is why genetically I am getting same type of plants. Are they known as soma clones? The answer is yes. They are definitely known as soma clones. Now statement number 2. Soma clones can be obtained through micropropagation. Can they be obtained through micropropagation? Yes. So tissue culture is a method by which we can produce so many amount of plants, thousands of varieties of plants, which we call as micropropagation. So soma clones can be obtained through micropropagation. That is correct. So I'll say both statement 1 and 2 is correct. Okay. Somatic hybrids is different. Soma clones is a result of tissue culture or micropropagation. Always remember, they are genetically similar. Somatic hybrids are different from the parent. From the parent it was different. You got a hybrid thing, new thing out there. Now the pollen grains of a plant with 2n is equals to 48 are cultured by tissue culture to produce a callus. Listen, pollen grains of a plant. Okay, so what are you taking? You are taking pollen grains. The plant which was there, it is having diploid number of chromosomes, that is means 48 number of chromosomes. What will be the number of chromosomes in the pollen grains? Half of it only. Okay, so it will be 24 over here. Fine. Now what would be the number of chromosomes in the cells of the callus? Basically when you are taking the pollen grains and you are growing in a medium, the cells are going to divide and going to form a tissue sort of structure. It is known as callus. You are being asked, what is the ploidy of this particular callus, or what is the number of chromosomes in the callus? It is made up of the cell, that is pollen grains. It had 24, so all of them is going to be having 24 number of chromosome itself. So your answer is going to be B. Again, read the question carefully and then only go ahead. Pollen grain has to be having haploid number of chromosome. The plant can be diploid, but here it is going to be having haploid only. Okay, so make the half of it and then everything will be the same. Next one. Statement 1. Bacillus thuringiensis produces insecticidal protein crystals called Bt toxin. Bacillus thuringiensis produces insecticidal protein. Does it produce insect killing proteins out there called Bt toxin? Yes, they do. Statement number 2. These toxins exist as pro-hormones before entering into the insect gut. It's pro-toxin, not pro-hormone, right? So which is why it will be wrong. So just children, sometimes they just see this, do not see this, do not do that mistake, okay? So your answer for this question is going to be statement 1 is true, but statement 2 is false. Cool? Yes, ma'am. Next question. DNA in a clone of cells followed by detection using autoradiography is called. DNA in a clone of cells followed by detection using autoradiography. So basically some DNA which is there, it is attached to some radioactive thing. And when it is attached to radioactive thing, even in between lots of other clones out there, you can still spot it. Why? Because it is attached to radioactive thing and you are viewing it under what? Autoradiography, which is why it is clearly being seen. This DNA which is attached to radioactive thingy is known as a probe. You call this as a probe. So B is your answer for this particular question. Got it? Yes, ma'am. Okay. Next thing. Use of bioresources by multinational companies and other organizations without proper authorization and compensatory payment is referred to as. So basically, suppose America, in US, my dear children, as I told you, I have not told you, okay. So basmati rice was being patented. They said that no one has the authority to produce basmati rice now, we are going to do it. But do you think that's correct? Because basmati rice origin is in India, the farmers do that, right? So without giving compensatory payment to that, can you just say that I am going to use the bioresources and I am going to put a patency on this? No. This is referred to as biopiracy. It is referred to as biopiracy. So your answer for this question is going to be A. Okay? Yes, ma'am. Fine. Next. Which of the following statement is not correct? Is not correct, okay? Most Bt toxins are insect group specific. They are definitely insect group specific. RNA takes place in all eukaryotes as well as prokaryotes organism as a method of cellular defense. RNAi, RNA interference. Do you know what is that? I'll talk about that. The recombinant therapeutics do not induce unwanted immunological response in humans. Yeah, they do not. Okay, the recombinant therapeutics that we use, they should not actually induce any unwanted biological response, immunological response, right? So this is also correct. Adult onset diabetes can be controlled by taking insulin at regular time intervals. Yes, because diabetes is caused because your insulin is not being produced in the pancreas, right? So obviously, if at all you give insulin, enough amount of insulin, it's all going to be sorted. So this is correct. Now what is not correct is obviously this. But what is this? RNA interference is a method that you use for producing pest-resistant varieties of plants. So there's tobacco plant which is infected by Meloidigoin incognita, an aschelminth, or what do you call as nematode. Okay, so what this nematode usually does is that it has the DNA, it transcribes into mRNA. This mRNA translates into a protein. Now over here, I want to stop this. I want to stop the translation process, which is why what I do, it has to be single-stranded. This mRNA has to be single-stranded for undergoing translation. I am going to give this strand one more complementary strand, which is why it will become double-stranded. And do you think translation is going to happen? No. That means protein is also not going to be made. This is known as RNA interference. You're interfering over here. And this is actually a cellular defense mechanism that is found in eukaryotes, as in basically the developed organisms out there. Eukaryotes have a proper nucleus, proper cell membrane mount organelles and things like that, right? In the case of prokaryotes, they are primitive, they do not have all these things and they do not show RNA interference. Please know that. So RNA interference is something that you see in eukaryotes as a cellular defense, not in prokaryotes, but here it is given eukaryotes as well as prokaryotes, which is why this is not going to be the answer. So B is the odd one out over here. Got it? Yes, ma'am. Now, match column 1 with column 2 and select the correct options from the given codes. Column 1, alpha-1 antitrypsin. We just studied it, but let's see. We have to read all the options first. Transposon, ELISA, retroviral vector. Okay, we know this. Now read this. AIDS, gene therapy, emphysema, mobile genetic element. Okay. Alpha-1 antitrypsin, we all know it is there for treating emphysema, respiratory disorder. So A goes with 3. A goes with 3. I have 2 options like that. Okay. B goes with 4. Transposon is a mobile genetic element. Okay. I have just keep it on side as of now. Let's go with D option first. Retroviral vector. Retroviral vector is basically used for as a carrier or a vector in the case of animals, right? So here in gene therapy I might need it. Remember, in gene therapy I need to put the gene adenosine deaminase into the lymphocytes. That is an animal cell. For that I might need it. So D goes with 2. D goes with 2. I just have one option. Basically this is going to be my answer. If D is very difficult for you, ELISA, we all know it's a diagnosis test for what? Test for AIDS. So ELISA goes with 1. C goes with 1. And you just have an option like that. So basically you got the answer. Okay, so when you have confusion, see all the options, check out for it and then only go ahead. Transposons are mobile genetic elements, as in there are genes which jumps from one place to another genes. They are known as transposons. They can also lead to very bad mutations. Cool? Now, biotechnology and its application, again you get a 5-marker questions coming off from this chapter. This is going to be second last chapter. After that we have organisms and population, and then we are going to end up. So let's see biotechnology and its application. The first one is genetically modified plants have been developed through core techniques of biotechnology. How are these genetically modified plants useful to the humans? So basically they're asking you the importance of GMO plants, that is genetically modified organisms or genetically modified plants. So first thing, enhanced crop yield and efficient mineral usage. So genetically modified plants, my dear children, they can sustain with limited amount of minerals also. Okay, they do not need much amount of, because they're okay with this mineral usage out there. That is one very important thing, which is why farmers will not be need to spend too much on fertilizers out there. With the minimum nutrients out there, they can easily survive. Now, resistance to pests and diseases. So you might have seen the example of Bt cotton as well as RNA interference. Okay, this makes the plant resistant to pest. So that is also a good thing. Now, reduced use of chemical pesticides. Again, over here, since you are creating the plant itself resistant against pest, you will not require chemical pesticides, and obviously when you are not requiring chemical pesticides, the soil fertility is also going to be maintained, because all those pesticides are going to mix up with the soil and going to cause trouble out there, right? So that is also done. Tolerance to abiotic stresses. Plants can survive in conditions where there is drought, salinity, heat, cold, everything. It is going to sustain all of that. Now, next thing is improved nutritional quality. Okay, the plants are going to produce something that is more richer in nutrient. For example, you might have heard about golden rice. Have you heard about golden rice? Golden rice is a rice which looks golden color. Why? Because it is vitamin A enriched. Vitamin A enriched. So in places such as Africa, where there was too much malnutrition going on at that place, golden rice was being supplied at some point such that they could at least have vitamin A in their food. Okay, so that is one thing. Next thing, reduced post-harvest losses. So after harvesting also there will be so many losses, because the plants and seeds which are there, they could be damaged and things like that could be happening. But with the help of recombinant DNA technology, with the help of genetically engineered plants, they could survive all of it a lot better. Cool? So these are the points that you have to write. Now, briefly explain the use of transgenic animals. So transgenic animals basically are the ones where I am going to put my gene into an organism. So this organism has a recombinant type of gene out there. We call this as recombinant, or basically a transgenic animal. Why do we need transgenic animals? The first one is to study of the normal physiology and development. For example, there is a factor that is known as insulin-like growth factor. Now, this is a very important thing in our body. Now, I want to know, what does it actually go and do inside our body? What are the key aspects where it goes and shows its effect? What is its effect on the growth and development? So how will I get to know that? Basically, I put the gene inside an organism. Mostly 95% of the times, my dear children, the transgenic animal which is used is a mice. Always remember, it's a mice, not sheep, not goat, nothing. It's usually a mice. So when I put this growth factor inside the mice, what is going to happen? It is going to show inside the mice there will be some changes, effects on the growth, and with that changes I will be getting to understand that okay, while inserting this, all this thing happened, so probably these are the functions of insulin like growth factor. So for understanding the normal physiology and development, we use transgenic animals, so helps understand gene regulation and effects on growth and development and body function. Example is insulin like growth factor research. Second, disease study and human models. For example, I want to see someone who has cancer, what are the areas which is being affected, such that I can create drug in response to that, right? So similarly, organisms have been made models for diseases such as cancers, cystic fibrosis, arthritis, Alzheimer's. So C square and A square, please remember it. Cancer and cystic fibrosis, arthritis and Alzheimer's. These are the diseases which have been studying using animals out there. So we make some changes out there in the body of the organism and see what does it have the effect in the, or basically for finding drugs also. Okay, for finding drugs against these diseases, we can try it on the animals. Obviously, this is a very bad thing to do, but this is how it works. Now, biological products. Now, this point is very, very important for production of biological products. There are two things which are given in your NCERT. One is alpha-1 antitrypsin. Alpha-1 antitrypsin is something that is produced in our body. It's a protein. It's a protein that is not a hormone. It's a protein that is produced in our body. And this protein helps us protect against emphysema, which is a respiratory disorder. Okay. Now, I cannot tell my body to produce it more. So what do? I take the gene and I make an organism produce it for me. Okay, so isn't it production of biological products using a transgenic animal? Yes. So don't forget alpha-1 antitrypsin. And why do we use it for? For treatment of emphysema, that is a respiratory disorder. The second thing which is there is alpha-lactalbumin. It's not alpha-1 lactalbumin, it's alpha-lactalbumin. This is again a protein. This protein is found in human milk. Now, I made a cow. A cow produced this particular protein in their milk, and it was the transgenic cow whose name was Rosie in the year 1997. So it produced a nutrient-rich human-like milk that contained alpha-lactalbumin, and again it is super beneficial for organisms. Okay, so these two products, biological products, are super important. So first is study normal physiology and development. Second is disease study and human models. The third one is biological products. Now the fourth one is vaccine safety testing. So before vaccine were tested on us, it was tested on animals. Okay, just to see whether it is actually working or does it have any other issues being caused, side effects being caused. So polio vaccine, everything was being tested just like this. The next thing is chemical and toxicity testing. So basically what do? That you make an organism very sensitive to even little bit amount of toxins out there, and then when you insert the toxin, we'll see if an organism is this sensitive, what is the reaction that is going to happen. Because in humans also there are organisms which are very sensitive, there are organisms which are not so sensitive. So we have to look for the people who are very much sensitive. What are the effects that are going to be shown? If it is too much effects, then probably the toxicity level is going to be less. Get it? So that is the thing. So genetically modified animals used for quick and accurate toxicity test for drugs and chemical testing. Over here, always remember you need the approval of GEAC. That is Genetic Engineering Approval Committee for doing all this thing. Okay, Genetic Engineering Approval Committee for doing all this thing. Always remember that. Now here itself, go study about biopiracy and go study about patency, if at all you do not remember. I think I have taught you in 2 marker, 3 marker. No, I have not. In 1 marker, I think I have done. But please make sure biopiracy and patency is clear to you. Okay, done. Now, explain the application of biotechnology in the field of medicine. In the field of medicine, how does biotechnology play a very important role? Any idea? Any idea, my dear children? So just give me a second, okay? Let me see what all is left. Okay. First one is in the production of insulin. So, insulin which was there, it was produced when first of all, what is insulin? Insulin is a hormone. It's a hormone. Okay, it's a hormone produced by the pancreas of human body, and particularly this particular hormone is super important for one very important thing, that is for trapping all those glucose and converting into glycogen. What is the use of trapping glucose into glycogen? Basically, it makes sure about one very important thing. That is what? That is the blood glucose level is being maintained. It is being maintained. That is the role of it. So, insulin make sure that our blood sugar level is not too high. Suppose some person has some problem in their pancreas such that insulin production is not happening. What do? We have to give them insulin. So usually insulin was taken from cattle and pigs and cows and things like that, but then people started developing allergies in response to that, which is why a company came into the picture, and that was Eli Lilly. Okay, this Eli Lilly company faced a problem, that is, in the case of insulin, there is A chain, there is B chain, and in between this A and B chain, there is one more chain which is known as C chain. Okay, and over here you have the disulfide bonds. This is how it goes. This is what a prohormone, as in, this is not the mature hormone, this is a prohormone which is produced in our body. In our body, we have some system that is going to cut this particular portion from here such that we just have A and the B chain, and in between that we have the disulfide bonds. Now this is going to be the actual hormone. I'll call this as the mature hormone. So this machinery is there in our body in the case of eukaryotes, right? And you need something similar happening in some organisms' body also. So that is very, very, very difficult. Prohormone to mature hormone formation. Listen, this is a prohormone. Do not go and write proenzyme and come. It's a prohormone, and this is a mature form of hormone. Now, so Eli Lilly came into the picture and said, okay, let's take a prokaryote. We definitely understand that it cannot make a cut over here for this particular C chain because eukaryotes and prokaryotes are different. When I take a E. coli, that is a bacteria, it's a prokaryote and it will not have the same machinery like me. One thing that can be done is that in one E. coli, I can put the gene for A chain. In another E. coli, I can put the gene for B chain. For production of B chain, what do I get at the end? I get A from here, I get B from here. In the laboratory condition, can't we just make the disulfide bonds over here? Yes, and that is how we got the insulin, the human-like insulin, which is known as humulin, humulin, not milin, it is humulin. Please remember this. Okay, so what did we do? We took the A gene and the B gene, A polypeptide chain gene and B polypeptide chain gene. We put it in two separate E. coli. Both of them produced their own polypeptides, and now we joined it. That is humulin. It was produced by Eli Lilly company. Got it? So over here, what is the answer for this question? Biotechnology helps produce safe and large quantities of proteins using microbes. Example, human insulin, that is humulin. Earlier insulin was extracted from cattle, pig, pancreas, causing what? Allergies. Human insulin consists of A and B polypeptide chains linked by disulfide bonds out there. Eli Lilly created separate DNA sequence for A and B chains and inserted them into what? E. coli plasmid. Bacteria produce both the chains, which is purified and chemically combined to become the functional insulin. This allowed the large scale production. So please know, this is one application of biotechnology in the field of medicine, and this is super important. Okay, so pro-insulin to insulin, I hope you know, or pro-hormone to mature hormone, I hope you know the transition. Now the next one is gene therapy method. So gene therapy method is where basically this was used on a 4-year-old child who had a deficiency of one particular enzyme, that was adenosine deaminase. Adenosine deaminase, you call it ADA. So in her body, the cells did not have the gene. Okay, there was a deletion of the gene which was forming ADA, adenosine deaminase. Now, when there is a deletion of this gene, do you think adenosine deaminase is going to be produced? No. What is the problem if at all this gene is not produced? There will be toxin buildup. Okay, there will be buildup of some very not so important toxin which can create problems. Now, this is a very big difficult situation for certain cells which you call as lymphocytes, because in lymphocytes you cannot afford to have toxins being built up there. So that means we need this gene for survival. That is very important. So that's when gene therapy was being introduced, okay, into a 4-year-old child. What was being done is that the steps of this gene therapy is super important, so you should know this. Okay, first thing that was done, that lymphocytes were taken from here. So the first thing that was done is lymphocytes were extracted from the patient. First step. Second step, which we usually miss, we culture it. Okay, we culture the lymphocytes. Okay, we culture the lymphocytes outside the body. Now what do? We transfer the gene for ADA using a vector. Now you tell me, what is a vector? Over here, it's an animal cell. For animal cell, if at all you are needing a vector, I'm not talking about vector-free method, I'm talking about using a vector. What is a vector? Retrovirus. Retrovirus, disarmed retrovirus is going to be my vector over here. I transfer the gene. So basically in my lymphocytes, now I have this gene. Now what do? I basically put it back into the body. What is going to happen? After a few days, again the lymphocytes number are going to be less because it's not immortal. The number of lymphocytes are going to be less. So what do you need? Again, you will need to do transfusion again. So ma'am, gene therapy, which is a method, it's not completely useful. It is useful, but it will be completely curative if at all gene therapy is done in embryological stage itself. When an organism is in the form of embryo, at that particular point, if at all it has been done, that's when it will be a permanent cure. Otherwise, it is not going to be a cure. Why not cure? Because over here, the cells are not immortal. Similarly, there are other methods also. Bone marrow transplantation. Sometimes it does not work. There is another method where you give this enzyme repeatedly. Again, you need repeated transfusion. So always remember, gene therapy in adult stage or bone marrow transplantation or basically the enzyme replacement therapy where you give the enzyme again and again, all of them is not a permanently curative method. The only permanent cure that you have for this is doing gene therapy in the embryological stage itself. Okay, because those cells, if at all one cell has it, it will distribute it to all the other cells out there. Got it? Okay. So that is a general thing that we should know. So, yes, lymphocytes withdrawn from the patient and then cultured. This step has been missed, so I'm writing cultured outside the body. And basically, what do you transfer? I told you transfer the gene, but you call this as ADA-cDNA. Adenosine deaminase complementary DNA is what you take. Okay, complementary DNA. So please remember it as such. If at all you want an idea, I can give you, but I was thinking about the time. So listen, DNA is there. DNA transcribes into an RNA. You know, basically exons, introns, both of them are there. Here also exons, introns will be there in HNRNA. Now what do? You remove the introns, you will just have the exons only, which codes for things. That is known as an mRNA. Now, when you just have the protein and you just want this particular protein, what do? You make a complementary DNA. You make a complementary DNA out of it. So from an RNA, you're doing reverse transcription to make a DNA. This is known as complementary DNA because we do not want the introns and things like that. That is why we did this. And then we put it inside the body. That is why we call it complementary DNA. Just know about it. Okay? Works? Works, ma'am. Perfect. Now, ELISA is one of the methods of molecular diagnosis. What is the principle of this technique? I told you what is the principle. There is antibody antigen linkage interaction. A specific antigen, virus, bacteria, protein or specific antibody is attached to a plate. Either it can be antigen or it can be antibody against HIV virus, right? The patient sample is being added. If the patient has matching antibody or antigen, they will bind together. Over here we have taken antigen, which is going to bind. A coloring agent, chromogen, is also mixed. The enzyme reacts with the chemical to produce a color. If a color change is there, that means antigen antibody is being protected. So no color change means disease is not being present. So again, how do you do it with antigen? Basically, you know HIV viruses that causes things. So you put it over here. Now, the patient's blood is taken. In the patient, if at all that person is having the disease, obviously they produce antibodies against it. So the antibody will come and attach. Okay, so you get it? So this is the same logic that is working. But in general case, we just talk about antibody, antigen, antibody. So you can just also talk about that also. Now, disease is what? HIV, AIDS. Now, always remember in your NCERT there is one question. PCR, polymerase chain reaction, is also used for diagnosing diseases. Is the answer yes? Yes. Whenever the question comes that a method which is used for finding the trace amounts of organism, very little amount of thing will be there. Still you can find the disease using which method? It is PCR, because PCR you do amplification, right? And PCR can also be used for detecting HIV. Know that. Can be used for detecting HIV. Please know that. Now, traditional breeding techniques have failed to keep pace with the demand and to provide sufficiently fast and efficient systems for crop improvement. In this context, explain how the tissue culture technique has contributed to crop improvement programs by mentioning any 5 of its application. What do you do in tissue culture? First of all, you need to know that. Suppose this is a plant. You take the tip of the plant. Okay, why tip? Because over here you have meristematic cells. What do you call these cells as? Apical meristem. Okay, or probably you can take from the bud also, because this bud will be giving rise to what? A new branch and things like that. You call this as axillary meristem. So usually you take from apical and axillary. This is super important. Why? Because if at all you want to produce virus-free plants, you have to take from here. These parts will, suppose this plant has virus attack being onto it, okay? But these parts will be free of the virus. Apical and axillary will be free of the virus. That is why you take those particular portions. Now you take that, you now call this part as explant. What do you call it? Explant. Now explant you put in a culture medium. The cells will grow and it will fall something like this, which is known as callus. A group of cells which is known as callus. Now the callus is transformed into what? A nutrient medium containing rooting hormone and shooting hormone. A new plant is there. Suppose you make multiple plants like this, thousands of them, you will call this as micropropagation. You will call this as micropropagation. Now what is the use of this? First of all, regeneration of the whole plants from the explant due to property of totipotency. First thing, what is the use of tissue culture? Is that I can regenerate a whole plant from the explants. Why? Because there is totipotency. Totipotency means a cell has the potential to become anything it wants. It has maximum amount of potential, totipotency. So regeneration of the whole plant is possible from tissue culture? Yes, ma'am. Rapid production of large number of plants in a short time through micropropagation. When I say tissue culture is being used to produce thousands of them, I call this as micropropagation. So can I produce thousands of plants? Yes, ma'am. Production of genetically identical plants. Can I produce genetically identical plants? Suppose I have a plant which is super good and I want multiple copies. Can I produce with that? Yes. So genetically identical plants I can produce. Next thing, commercial production of crops like tomato, banana, apple. If at all I want to in a large scale, can I do it? Yes, ma'am. Recovery of virus-free plants. Can I produce virus-free plants also from this method? Yes. But for that, what do you need? Axillary and apical meristem. Axillary and apical meristem. Always remember this. Okay, and the plants that you get from tissue culture is known as soma clones. Soma clones. Why? Because they are all from the same cells, though it will be like clones. Somatic hybrid is different. Soma clones are different. Somatic hybrid is when you take two plants, two separate plants. For example, tomato, potato. You take two cells, remove the cell wall of both of the cells. What is left behind is known as protoplast. What do you call it? Protoplast. Protoplasts are fused. This is known as somatic hybrids. Now we will call this as pomato. What do you call it? Pomato. Potato and tomato both. Done, ma'am. Now what is RNA interference? How does RNA interference help to develop resistance to tobacco plant from nematode infection? Then that is the next question for which you should know the answer. So listen to me carefully. There is a nematode, an Aschelminthes, which is known as Meloidigoin incognita. This is that particular organism, right? Now what does it do is that it affects the root of the tobacco plant. And how does it do? It goes inside, forms a cyst and be there. It's not this big, okay? It's very small thing. It goes inside and basically have its DNA make the RNA, that is the mRNA. And this mRNA will undergo what? Translation to form the proteins, and thus proteins are going to cause trouble in this particular plant out there. Okay, so what is the method in which I can prevent it? There is only one way I can prevent it, and that is RNA interference method. RNA interference method. What am I going to do over here? How am I going to interfere in between this? Basically, I take some genes from here, from the same organism, and I put this gene inside what? Inside a vector, because for transferring something to the plant, I need a vector. Which is a vector? Agrobacterium tumescens. So here I put the gene of my interest instead of transfer DNA place. This goes and affect the cell in the plant. Now in the plant, my dear children, what is going to happen? In the plant, this particular gene that I have inserted over here is going to produce double-stranded RNA. The gene that I have taken from here and put it inside the agrobacterium tumescens is now inside the plant, and this gene is going to produce what? Double-stranded RNA. dsRNA is what I call it. It is like this. Okay, ma'am, what is the use of this? This double-stranded RNA is going to now target the mRNA. What? So listen to me. mRNA is actually single-stranded structure. Only when it is single-stranded, then only it can undergo translation to form the proteins. Now suppose this double-stranded mRNA came into this particular picture. This double-stranded RNA came into the picture over here. So what? Will it make triple-stranded RNA now? No. This double-stranded RNA, oh my god, just a minute. Okay. I need patience. Yes. So this double-stranded RNA was there, and your mRNA was right over here, which was going to undergo translation to produce the protein, right? Now what happens is that this double-stranded is broken down. One strand is free over here. That one strand comes and makes a bond over here. Now do you think mRNA is single anymore? No, Baba, it has become mingled. So it becomes double-stranded. Do you think double-stranded mRNA can undergo translation? Do you think it can undergo translation? The answer is no. Do you think proteins is going to be made? No. Do you think plant will be affected by myeloid incognita anymore? No, Papa. That is the logic. So we are interfering in the RNA, and for that you need double-stranded RNA itself. Out of that, one strand comes and binds, but always remember it's a double-stranded RNA that you are requiring over here. Very, very, very important. So how do you write this answer? Again, so identifying the nematode gene. Scientists first identify the genes in the nematode that are essential for its survival or ability to infect the plant. Using Agrobacterium tumefaciens to insert the gene. So to deliver the silencing gene into the tobacco plant, scientists use Agrobacterium. So I took the gene, I used Agrobacterium tumefaciens for that, and basically now the Agrobacterium tumefaciens which is there, it is going to infect the plant cell. Now in the plant, it will produce double-stranded RNA. Right? Now double-stranded RNA triggers RNA interference. It goes and does the RNA interference, silencing the targeted genes in the nematode body. What is going to happen? Basically the plant is not going to produce anything anymore. Got it? This is RNA interference. For this you need double-stranded RNA. Very, very important thing. Now there is one more application of biotechnology in agriculture, and that is BT, cotton and things like that. What do you do over here, and what is the logic being used over here? This is also super important. So let's see. Over here the first thing that you require is Bacillus thuringiensis, which you call as BT. Remember, it has a gene which is known as cry gene. This cry gene is responsible for production of inactive toxin. I call this as protoxin. Why is it inactive over here? That is the question. Why is it inactive? Why it does not affect the BT, that is Bacillus thuringiensis? Because for it to be active, you need some ambient pH, and that is not being given over here in BT. Okay, fine. Suppose an insect somehow took the BT inside. Suppose it's a butterfly caterpillar, and now inside the butterfly caterpillar you have this. Okay, so here you have the Bacillus thuringiensis, and this is suppose the mid-gut region. What is going to happen? The protoxin over here, the inactive protoxin over here becomes active, my dear children. Why it becomes active inside the midgut? Because here you are providing an alkaline pH. Because of this alkaline pH, what is going to happen? The protoxin becomes active form of toxin, which is exactly why there will be perforations found over here. There will be perforations, and later the organism will be killed. First of all, it targets the epithelial cells of the midgut region. Later leads to perforation, leading to the death. So, ma'am, the protoxin which is produced is inactive because there was no alkaline pH in BT cotton in the BT organism. Exactly, that is the reason. And when you got the alkaline pH, it becomes active. This is super important for you to know. Now what do is, I take this gene and I put it inside the plants. The plants will also produce these protoxins, which will become inside any organism that will try to eat the plant. Any organism as in, it is very specific, it has to be an insect. So this cry genes are insect group specific. Always remember that. So there are multiple type of cry genes in your NCERT. It has been written like this. So there is cry 1AC and there is cry 2AB. Okay, sorry, AB. Cry 1AC, the first letter of the first word is going to be starting with C. So I am writing cotton. C. Second word starts with B. Second word is bollworm. And does it start with B? Yes. So you have two genes, cry 1AC and cry 2AB, for killing cotton bollworms. Similarly, you have cry 1AB. Cry 1AB for corn borers, for killing corn borers. So how do I remember it? There is only one, that is here you had two of them, here you just have one, and basically the first letter is C and the last letter is B. Okay, I know it's confusing, but you need to remember it. It's been usually asked in MCQs in every question out there. Cry 1AC, first letter, first word starts with C, second word starts with B. So both of them are like CB only, but here it is pretty long, that is why you have two of them. Here you just have one, and basically C and B is starting and the end. Okay, so why do I ask you to remember it like this? That sometimes you might go write 2AC over here, 2AC or 1AB. So always remember only one is there and it's B at the end. So that is a corn borer. Done. Now, the cry gene, cry 1AC and cry 2AB from thuringiensis coding for the toxins are transformed to the cotton plants. The genes have been transformed. Proteins encoded by these genes control the cotton bollworms. BT cotton plants express the BT toxin by producing toxin that provides insect resistance. BT toxin exists as inactive protoxins. Insect injects the inactive toxins, convert to active in the alkaline pH, okay, which solubilizes the crystal. In this alkaline pH, it will be able to solubilize the crystal and the toxin will be there. Toxin binds to the surface of the midgut epithelium, creating the pores out there. Always remember that is very important, and they kill insects such as lepidopterans, coleopterans and dipterans. Lepidopterans, coleopterans and dipterans. Thus, this is very, very, very important. Cool. Now let's go ahead with the last chapter, that is organisms and population. With this, we will end it up. So it goes like this. Write an equation to calculate the population density at time t. So what is the population density at time t? How do you represent it? Nt is equals to n naught. This is the initial population plus b plus I. B is what? The birth rate. I is what? Immigration rate. Organisms getting inside the population, that is immigration. Minus death plus emigration, the organisms going out. Okay, B plus I minus death, D plus E. Okay, this is very, very important. So write an equation to calculate the population density at time t. This is the equation. Minus has to come. This has to be put inside bracket also. Now, population density fluctuates due to changes in the four basic process. Explain the four basic process. Natality. Natality means what? The birth rate. Okay, the number of individuals reproduced in a place at particular time. Mortality is the number of individuals lost from a population per unit time. Immigration is the next thing, where the number of individuals which are being added up. Emigration, number of individuals which are being living in the population. Out of all of them, these two are the most important thing. We consider immigration and emigration only when there is colonization, as in some organisms are coming from outside to inside and things like that. Okay, I'm going a little fast because yes. So what is mutualism? Explain any 4 examples of mutualism. Or there is a different question that I saw, that is, what is mutualism? Give any 2 examples. Plant-animal interaction often involves co-evolution of mutualist. Justify your answer with an example. This was also given. So what is mutualism? Is what? Where both the interacting organisms are getting the benefit. Example is lichen, example is mycorrhiza, and example is fig tree and wasp, and also rhizobium bacteria and leguminous plants. Listen to me very carefully. Lichen is a combination of algae and the fungi. Mycorrhiza is a combination of plant and the fungi. What is common over here? Fungi is common over here. Algae produces what? Algae can produce its own food, and this food can be given to the fungi. Plant can also produce its food and give it to the fungi. So in both of the places, the producers help in that case. In the case of lichen, the fungus help in providing protection and also absorbing water and nutrients. Listen, the fungi has the ability to go inside and take in everything which is there. So basically, fungus will be helping in absorbing water and the minerals, as well as it helps in the protection in the case of lichen. In the case of mycorrhiza, the fungi will go deep into the soil and get phosphorus. This is very important. You have studied it in microbes in human welfare. Apart from that, growth and development, overall growth and development also it helps. Resistance, to provide resistance again other pathogens also it helps. On the plant other hand, plant provides the food to the fungus. So both of them are getting benefited. Now rhizobium bacteria and leguminous plants. Here the rhizobium bacteria fixes nitrogen and gives to the plant. What does the plant do? Plant again makes food and gives it to the rhizobium bacteria, because rhizobium bacteria cannot prepare its own food. Got it? Again, both of them has a relation like that. Now fig tree and wasp is there. Wasp helps in pollination of the fig tree. Right, and fig provides what? Food and also space for the egg to be laid. So what is mutualism? It's an interaction where both the species are getting benefit. Right, plant-animal interaction often involves co-evolution of mutualist. Why? Because over here, my dear children, if one organism is getting any changes out there, if the other organisms do not evolve, do you think both of them will be able to sustain without each other? No, because if fig tree is not evolving, you will not get any space to lay the eggs. If wasp is not evolving, pollination is not going to happen. That is why covulation has to be taking place. So justify your answer. This is how you have to justify, by telling the examples of it. So 4 examples of mutualism is being mentioned over here. Please know different type of questions can also be framed. Next, name the type of interaction seen in each of the following. Clownfish living among the tentacles of sea anemone. So you might have seen so many tentacles that is there. In between that you have a fish. What type of relation does it show? It is showing commensalism. It is showing commensalism. Now this is super important. Commensalism is super important. What is exactly happening in the case of commensalism? One organism is getting the benefit. What is that one organism? Clownfish. And tentacles of sea anemone. Anemone is not getting benefited nor getting harmed. That is why it is a +0 relationship out there. Koil and cuckoo bird during the breeding season. Koil goes and lays Koil and cuckoo bird, it's the same thing. Koil and crow is being asked, okay? Koil and crow. So koil goes and lays the eggs in the crow's nest. What does it do? Basically, the koil has evolved. It's a parasite, okay? It has evolved in a way that its egg looks like the crow's egg. Now the crow papa guy will go provide nourishment to all of them and basically gives its energy to growing a koil's egg out there as well. So this is parasitism, where one organism is getting benefited, that is the koil obviously, and the crow which is there, it is getting negatively impacted. And this specifically, if we say, it's brood parasitism. This is what you call as brood parasitism. Always remember, host is the crow and parasite is the koil. The koil evolves to look like crow. It's not like host will evolve. It's the koil who is going to evolve. The barnacles growing on the back of the whale. Now barnacles which are there on the whale's body, my dear children, they are there for transportation. Barnacles, does it cause any harm to the whale? No. Whale, does it provide any good thing to the barnacle? Yes, it allows in transportation. So this is a +0 relationship. No harm, no benefit. So again, it is commensalism. Now we have marine fish and copypods. Just like how you have lice and humans, how you have ticks and dogs, similarly you have marine fish and copypods. Copypods, my dear children, they are parasites which are going to feed on the marine fish. So marine fish is negatively impacted, copypod is positive in that. This is parasitism. Now sexual disease in Meterin orchid, that is Ophrys. So over there, the bee that comes to the flower, that is Ophrys flower and orchid, it undergoes pseudocopulation. Okay, it undergoes pseudocopulation, thinking that's a female flower. So the female bee out there, right? On the other hand, so it gets the benefit of doing pseudocopulation out there. And it can also take some pollen grains also. That is one benefit. Next thing, the plant gets benefit because pollination is definitely going to happen. So this is mutualism, and this is also an example of sexual deceit, which has already been mentioned out there, which is why you just say it is mutualism. Actually, we cannot consider it like that, but yes. Now, explain the fascinating forms. I have already explained, just go through it. I'm not going to re-explain it because that will waste a lot of time. It is exactly what I have told you in previously. So yes. Now, mention the population interaction exists among the following. Abundant tortoise and the goats. So abundant tortoise were there on one particular area. They were feeding on the grass. When goats were introduced, what happened was the abundant tortoise become extinct. It's because of competition, my dear children. They were both competing for what? The grass out there. So competition led to competitive exclusion of abundant tortoise. This is competition. Tiger and deer, my dear children. Are you? Tiger and deer. Tiger will go eat the deer. So there is prey and predator relationship. This is predation. Sea anemone and clownfish, we just saw, it is commensalism. Wasp laying egg in the fig fruit, it's basically mutualism. Cuscuta growing on the egg plant, it will be parasitism. Why? Cuscuta does not have chlorophyll to make its own food. That is why it depends on the plants for food. Okay, so that is parasitism. Plus minus, because the plant is getting negatively impacted. Okay. Now, what is commensalism? Explained with 2 examples. Commensalism is an interaction where one organism benefits, other is neither harmed nor benefited. Example, orchid growing as an epiphyte on the mango branch. On the mango branch, you might have seen orchid. Orchid is not there for harming the mango plant. Orchid just needs sunlight. Mango plant, does it have any benefit or negative effect? No. But is orchid getting the benefit? Yes. This guy is not having any benefit nor harmed. So +0 relationship. Clownfish and sea anemone, we just told. Barnacle on the back of the whale, again, it is just for transportation. It does not cause any harm to the whale. Whale does not get any benefit also. Cattle, egret and grazing cattle. So whenever you have seen cows moving, you might have seen some birds. That is known as cattle egret. Why they are just around the cow? Because whenever the cow moves, basically it flushes off the insects. Now the insects are being fed by the cattle egret. Okay, so it is getting the benefit. Here it is neither benefit nor loss. Okay, now state Gauss's competitive exclusion principle. Two closely related species, okay, that survives on the same resources cannot coexist indefinitely. Two closely related species that feed on the same resources cannot stay together indefinitely. One will definitely be eliminated over time. That is known as Gauss's competitive exclusion principle. Example is Abingdon tortoise as well as this thing. Good. Okay, now what are ectoparasites and endoparasites? List any three parasitic adaptation. Parasites are the one that feeds on the ectoparasites are the one that feeds on the external surface of the host, and parasites that are living on the internal body, it's known as endoparasites. Okay, so ectoparasite is this that lives on the external surface, and endoparasite is this which lives inside our body. Now which lives inside our body, these parasites will be having some adaptations. For example, they will not have any unnecessary sense organs such as ear, things like that, because they want to move very easily and they want to make their bodies literally lightweighted. So this is first thing. Presence of adhesive organs and suckers, because they do not have hand to cling on to places, so they will be having adhesive organs or suckers. Third one, loss of digestive system, because if, for example, tapeworm stays in our body. In our body, digestion happens, the things are broken down to very small components. These things have to be just absorbed by the tapeworm. They do not have to break anything down, which is why again they do not have any digestive system. Now they will be having high reproductive capacity to increase their number, and mostly they will be having a very complex life cycle. Example is given in your NCERT and you should not be missing out on it. That is human liver fluke. Human liver fluke, my dear children, it has two intermediate hosts. The last host is human beings. It has two intermediate hosts. One is snail and one is fish. It has to get inside the snail, it has to get inside the fish, then it will come into our body. So we are the ultimate definite host, but here these two are the intermediate host out there. Okay, this is for human liver fluke. Very, very important. Snail and fish. Okay, snail and fish. Very, very important. Cool. Now, predators are prudent and conduits in nature. Explain and support your answer for giving reasons. So, why do we consider them as predators? Prudent means they are clever enough to understand that if I eat all of the prey, tomorrow I will not have food. So, they avoid over exploitation. That is why we say that they are prudent. So, predators play an important essential role in the ecological balance. They are considered prudent because over exploitation of prey would lead to their decline due to food scarcity, which is why they will not do over exploitation. Now, why do you call them conduits? Conduits as in they are channels. Channels of what? Energy transfer. So from the lower organism, they transfer the energy to the upper organism. How? By acting like a chain. So predators are conduits in the energy flow of an ecosystem, transferring energy from prey to the other organisms which are there. So lion, hunting, okay, that's all. So that is what you have to write for prudent and conduits. Now there's one more thing. They are asking about the defense mechanism that have been adopted by the prey population. And here you have defense mechanism in the case of animals and plants separately. In the case of animals, the basic example that you have is cryptic coloration or basically camouflaging. So if at all I put a green frog over here, will you be able to see it? No. Why? Because it has been camouflaged. So this is a defense mechanism that you have, camouflaging. The second one is some are poisonous and therefore avoided by predators. Some organism, for example, monarch butterfly. You might have seen the butterfly, right? Monarch butterfly is blue and orange in color. This butterfly produces highly distasteful substance such that the predator will not come and eat it again. Okay, so that is why. So some can produce poisonous substances or basically highly distasteful substances such that the organism will not eat it again. And monarch butterfly is an example for that. So in the case of animals, you can write camouflaging and also the example of monarch butterfly. Now in the case of plants, you can give the examples like this. First one is, you need to understand that most of the insects are going to eat the plant. Almost 25% of eats are phytophagous. That means the plant needs protection. First one is morphological protection in the terms of production of thorns. Now thorns are there in the case of acacia and cactus. And basically these thorns will be preventing any organism to come and eat it because they will have thorn attack being done. That is morphological difference. Then there is chemical difference, where what is going to happen? Some plants are going to produce some toxic substances, for example cardiac glycosides. This is very important term, cardiac glycosides. These are being produced. When these are being produced, what is going to happen? Basically the organism might even die. It can even die. This is seen in the case of Calotropis. You might have seen this. This is a wild plant that is being grown. You call this as what? Calotropis. So in Calotropis, what do we have? We have cardiac glycosides that is there for protection against herbivory. Please know about this. Okay, apart from that, there are certain plant defense chemicals that are being produced which we use for our use. That is nicotine, strychnine. Okay, nicotine, strychnine and quinine and this thing, what do you call it, caffeine. All of these things are something that we utilize for our purpose, but actually it's plant defense mechanism. So do not forget Calotropis and cardiac glycosides, thorn in acacia and this, monarch butterfly and also camouflage. These are the prey defenses. Okay, now what do we have? Write any 4 factors that influences population density. Okay. What are the 4 factors that influences population density? I told you: natality, mortality, immigration and emigration. Draw a graph showing exponential growth. Exponential growth, Baba, how does it look? It looks like this. Over here you have population density and over here you have time. Exponential graph will always grow up. It will always grow up and it produces what? A J-shaped curve. So ma'am, do you think it's realistically possible? No. Because in every environment, my dear children, it has a carrying capacity. We all fight for resources. At the end, we all will fight for resources. Why? Because resources are limited. Now this is a condition where resources are unlimited. That's when the organism will grow exponentially and will produce a J-shaped curve. J-shaped curve over here. In nature, we have another curve which is known as logistic growth, which goes like this. It grows and then it comes like, so this is a S-shaped curve. It was initially J, but it becomes S-shaped curve. Why? Because there is a carrying capacity being involved over here. There's a limitation out there. Always remember that. So here there is not. So how do you have different formulas for both of them? Yes. In exponential growth, the formula is dn by dt is equals to Rn. R stands for intrinsic rate of natural increase. That means it stands for birth minus death. Okay. So in a population where the birth rate is more, obviously population density is going to be more. If death rate is more, population density is going to be less. R is intrinsic rate of natural increase. I can also say that according to Darwin, it is said as reproductive fitness. So high R value means basically high, more reproduction. B is more, right? B is more, birth is more, means more reproduction. So you can also consume like that also. Now over here, how do you frame the formula? The formula is exactly similar up to some point. dn by dt is equals to Rn, but here you have to talk about K minus N divided by K. What is K? Carrying capacity. As I told, beyond carrying capacity, nothing can go ahead. So K minus N divided by K, that is super important for here. This is a formula. Please make sure you write it, you practice it. This is the population density. This is time. Okay, this is a realistic thing that is usually found. Now I think I have a question which was there in the question bank and I needed you to solve it. So please make sure this is not 50th question. This is going to be the 10th question. So I had made it solved in the 50 most expected questions, which is why I kept it. So refer to the graphs given below showing the growth pattern in organism and answer the question that follows. Curve A represents which type of growth? So curve A is given right over here. And which type of growth does it represent? It is exponential growth. dn by dt is equals to Rn. Curve B, my dear children, it represents what? Logistic growth. You also call this as Wurzburg's logistic growth. Okay. Now, which growth model is considered more realistic one in nature? Which is considered more realistic one? This one. Why? Because we definitely have limiting resources. That is why it is considered more realistic. Fine. Now, when bacteria are cultured under controlled optimum environmental condition, which curve is obtained? When bacteria are cultured under controlled optimum, listen, here they are asking you one very tricky question. Controlled optimum environmental condition. That means you have, optimum means you have everything which is needed for you to grow. And it is controlled also. Controlled as in you have to think in a way that I am making sure whatever extra is there, I am removing it. Whatever thing I have to provide, I am providing it also. So it is optimum condition, definitely optimum condition, and I am controlling it in a good way only. That is, I am removing the waste products that can create toxicity and everything. So this is a good thing. If this is a condition, then organism will be growing in an, which condition will it grow? It will grow in exponential phase only. Since optimum is given and since controlled is also given. If at all they had mentioned you only controlled environmental conditions, that means that basically here not everything is free. Okay, in that condition it would have been logistic growth. But here they have mentioned controlled optimum environment. That means everything which is necessary is given. It is controlled also. That means super good. It can easily grow. This type of questions can be confusing for you, to listen to me very carefully. Sometimes they give you ideal environment. If only ideal is given, then exponential growth. But if at all they are saying ideal environment with limiting resources, ideal environment is there, but sooner or later everything is going to get limited. Limited term comes into the picture. That means it's talking about the S-shaped curve. Okay, if just ideal, then this. If limiting term is coming, then this. Similarly, over here, controlled optimum environment means it's all good only. Only controlled, if it's there, it's bad. Then it will be this. Okay, if controlled optimum conditions, then it is this. Cool. So it is, remember, it is exponential thing that you are going to get. Next thing, the interactions such as predation, competition, parasitism can be represented by which curve? Whenever we say predation is there, parasitism is there, competition is there, which curve is there? You have too much competition out there, right? So obviously the curve would be this one. There is limiting factors. You do not have food. Why? Because there are predators and things. So I'll say in that case it will be logistic growth. Done? Done. Very, very, very important. And I think so these chapters do not have any 5 marker, and I think with this we complete all the 5 marker questions. Well, I really hope you watch the entire video, because it is definitely going to be useful for you. And if you have any doubts, do let me know. If you think that this video had helped you, definitely do let me know about that as well in the comment section below. Wishing you all the best. Please take care of yourself. Lots of love. Bye bye. And now we come to organisms and population. So, my dear children, how do you feel? Is it too much for you as of now, or are you able to take it up? So, I am trying my best to be in live loop, and 3 hours are already over. I am so sorry. But this is important, right? It's important, which is why I went ahead. So, please listen to me. 3 more chapters are there. We will finish it up very fast. Okay? If at all you have crossed this level, level 6 crossed, comment in the comment section. Let's go ahead. The organisms that reproduce only once in their lifetime, only once they're going to reproduce: oysters, pelagic fish, salmon fish, hippocampus. Only once they're going to reproduce. You call these organisms as simulparous organisms. What do you think is the answer? The answer is salmon fish. Along with salmon, you also have bamboo. Okay, bamboo. They produce only once in their lifetime. Always remember. Birds, mammals, all of us, they will produce multiple times, but they are not like that. Okay, so salmon fish and bamboo, do not forget, do not forget, do not forget. Okay, next thing. In a pond, if there were 20 lotus last year and through reproduction 16 are added, calculate the birth rate. Okay, so previously existing one, you put it down. The newly added one, you put it up. Time period, if at all it is mentioned, you can from last year. So basically just 1 year. Okay. Now what is the calculation? You do the calculation, you will get your answer. What is your answer? It is going to be 0.8. Now, you are being asked to mention the birth rate. So you have to speak about the time. You have to talk about the organism that is producing it. So I'll say 0.8 offspring per year. Is it correct? Not correct. 0.8 offspring per lotus per year. That is going to be the birth rate. Okay, so there's a formula over here. Number of new individuals added divided by the initial population size into the time that is mentioned. So over here only 1 is mentioned, so it is 0.8. Okay, please mention 0.8 offsprings per lotus per year. That is very important. Okay, this is the birth rate. Now, similarly, you can be asked about the death rate. 20 were there, 10 of them died. So please know about this formula. Okay. Now the correct order of various levels of biological organization in an ecosystem is: organisms came together to form population, population came together to form community, community came together to form ecosystem, ecosystem came together to form what? Biome. So this first option itself is the correct one. Organism, population, population, then biome, wrong. Biome, ecosystem, wrong. Biome, no. So this one is the only one. This is very easy question. I think you got it. I got it from model question paper is what I remember. Yes. Next. Jiva was growing a bacterial colony in a culture flask under ideal laboratory conditions where the resources sooner or later became limiting. Listen, this is a very, very important question. Jiva was growing a bacterial colony in a culture flask under ideal laboratory condition. Everything was ideal. But there is one thing that is clearly mentioned. Resources sooner or later become limiting. That means unlimited resources are not provided over here. Resources are limited. Which of the following equations represent the correct growth in this particular case? You are going to see logistic growth. That means there is a carrying capacity over here. There is a factor that is limiting, and that is what? Resource, which is why it cannot grow exponentially like this. There will be logistic growth. There will be a sigmoid curve that is going to be found. Okay, not a J-shaped curve, sigmoid curve. And whenever you have something like this, you say it is dN by dt is equals to Rn, intrinsic rate of natural increase, K minus N divided by K, where K is what? The carrying capacity. This capacity, carrying capacity has to be mentioned over here. If at all you are given everything is ideal, everything is ideal, it is going to be the other thing that is known as exponential growth, which goes like this. There is no carrying capacity. There the formula is dN by dt is equals to Rn. You do not include this. You get it? But here limiting factor is clearly mentioned, so please go ahead with this. So in preparatory also I saw a question which had lots of confusion. Remember, if you have not been given limiting factor, read the question carefully. Probably the answer could be exponential. Okay, this is a question that can be anything. Okay, logically speaking, exponential does not happen, but yes, if in the question it is given, you have to answer accordingly. Here it is logistic growth, okay, or S-shaped curve is being formed. S-shaped curve is being formed, okay, yes. Now, monarch butterfly is avoided by the predators because monarch butterfly is avoided by the predators, because, my dear children, it is highly distasteful. It tastes very, very bad. It's a what you call defense mechanism that this particular organism has. It is cryptically colored. No. It is highly distasteful. Yes. It produces poisonous cardiac glycoside. It actually does, but in the NCERT it is not written. It is written about Calotropis, a plant, but not about monarch butterfly. So we will not also study that. Just to know, it is highly distasteful in nature. Okay. It has stinging cells on its body. No, Baba. B is your answer. Next. What does the shape of the given age pyramid A to C reflect about the growth status? Okay, so there is post-reproductive, reproductive and pre-reproductive. So the bottom one is the pre-reproductive one, okay, and then it is reproductive and then it's post-reproductive. If I say the organism in the pre-reproductive age group are more, that means in future they are going to reproduce, right? So can I say it is going to be an expanding population? Yes. Over here, if at all you see reproductive and pre-reproductive, both of them are same. So I'll say it's what? It is a stable population that I get. Then over here, I have what? The reproductive one are very less. Pre-reproductive one are very less. Reproductive one are more, right? So that means basically in future you do not have many organisms to reproduce. So can I say it is going to decline? Yes. So basically, first of all, I say this is expanding. If the lower one is more, that means it is expanding. Here it is going to be stable and here it is going to be declining. In future, there are not much organism to reproduce. Cool? Cool, ma'am. Okay. Fine. On the rocky sea coast of Scotland, the larger and competitively superior barnacle Balanus dominates the intertidal area and excludes the smaller barnacle Thalamus from that zone. Which kind of interaction is being depicted by this example? What do you think? So there is intertidal rock, there were the superior barnacle Balanus which was there, and then there is Thalamus also. Slowly what happened is that these guys excluded these guys off from this area. Balanus one, they dominated the intertidal region and they pushed the Thalamus upwards. Why do you think this happened? Because of competition. Because of competition. And whenever an organism takes up the whole space, is known as competitive release. It is known as competitive release. They utilize the entire area by excluding the other organism somewhere else. Okay, this is a result of competition. Not predation, not parasitism, not commensalism. Okay, next one. Assertion. The epiphytes use the trees only for attachment and manufacture their own food by photosynthesis. So if at all you have seen the epiphytes, that is orchid growing on a mango tree. So it's just basically there for getting enough amount of sunlight and things like that, just like this. Okay, it's not for it is not deriving any food from mango plant. It is not harming the mango plant. So basically, it is commensalism which is being seen over here. One plant gets a benefit, that is the orchid, which is the epiphyte, and the mango tree, it's neither getting harm nor getting benefit. So there's a +0 relationship, that is commensalism. So that is correct. That is exactly correct. Now, as reason, commensalism results in negative effects on the growth and survival of one or both of the population. Wrong. Commensalism, as I said, one gets a positive thing, and the other one is neither harmed nor affected. So it's a positive 0 relationship out there. So there is no negative effects that you can see. So I'll say that assertion is true and the reason is false. C is the answer. Cool? Cool, man. Next one. Abingdon tortoise in I have written the answer, but still. Abingdon tortoise in Galapagos Islands became extinct after goats were introduced on the island. Mention the type of interaction. So I have seen a picture where on the tortoise, the goats are standing. So what is happening over here is that initially you had the tortoise, but later on when goats were introduced, what exactly happened is that the goats competitively excluded the tortoise. This is known as example of competitive exclusion. It excluded the tortoise, my dear children, altogether. Okay, why? Because of the same reason. Basically, it was stronger one. The goat was a stronger one, which is why in the picture there's a tortoise. On the tortoise you have the goat. This is an example of competitive exclusion. It got extinct. Just imagine the level of competition. Both of them were having grass. Okay. Now we have ecosystem, the second last chapter out there. Few questions are there. Over here, I want to tell you one very important thing. In ecosystem, biodiversity and conservation, all of it, there is number-based question which you have to remember. I'll request you, these two chapters, read from your NCERT very, very nicely. Each and every single line of your NCERT is important in these two chapters. Super, super important. Okay? And I'm not writing any levels over here. The last level is the one that I'm going to write later on. Okay. The first question that I have over here, the decomposers especially abundant in the bottom of the pond are. Decomposers especially abundant in the bottom of the pond are what? Only fungi, only bacteria, only fungi and bacteria, flagellates, fungi and bacteria. So usually we say it's FB, that is fungi and bacteria. Facebook, right? But always remember, in the case of pond, these things are changing. There are flagellates, there are fungi, and there is bacteria. Okay, please remember, it's there in your NCERT, one single line, and that came as a question. Okay, and that too in an MQP, so you can expect more of it. The correct sequence of steps in the processing of decomposition is, first thing, you have detritivores, the ones that eat on the dead and decaying matter, and you break the food down into easy ones, right? That is known as fragmentation. That is done by the earthworm and things like that. After fragmentation, whatever inorganic things are there, soluble things are there, it will go into the soil, deep into the soil, that is known as leaching. Now, we had broken down the big things into small things. Now they have to undergo decomposition, and that is known as catabolism. And while they undergo decomposition, then comes a middle stage where they are partially digested, where humus is being formed, and that is known as humification. It is known as humification. Now, this partially digested form again divides completely into inorganic things. So from organic, you're coming to inorganic things, and that is known as mineralization. So your answer for this question is FLCHM. Is there any way to remember it? FLCHM. If you have any ideas coming in your head, please share it in the comment section so that all of your friends can also get to understand it. Okay? HM, headmistress. But yes, you can think about it. Yeah? Yes. Next one. The rate of formation of new organic matter by producer is. Rate of formation of new organic matter by producer. Net primary productivity, secondary productivity, gross primary productivity. You're talking about just one producer. You're not talking about all the plants together. You will call this as primary productivity because is the primary organism out there producer? Is the primary producer only one producer is there? It's the primary producer. The productivity of it is known as primary productivity. If at all I talk about multiple plants and I say the productivity of all the plants together, then it will be known as gross primary productivity. Okay, here it's not like that, which is why it will be just primary productivity. Remember, okay, yes. Now, which of the following food chains is the major conduit for energy flow in terrestrial and aquatic ecosystem, respectively? So in terrestrial, which one is the major conduit of energy? Grazing food chain, detritus food chain, or what? It is actually detritus food chain, which is the major conduit of energy over here. In aquatic, it is going to be grazing. Why? Why like that? We expect the opposite to happen. We expect the opposite to happen, right? But why is it like that? Listen, in detritus food chain, decomposers plays a major role. Decomposers cannot undergo decomposition, okay, cannot help in decomposition if at all there is no moist environment. In the aquatic water bodies, the temperature is very cool, which is why it cannot favor detritus food chain. Get it? Why? That's the reason it has grazing food chain in there. In terrestrial, on the other hand, there will be detritus because you have the moist environment, aerobic nature, all the things that you need. Okay? So please remember this offbeat question. Okay, so here I have marked wrong. The answer is this. Okay? Okay, next one. If 10 joules of energy is available at the producer level, then the amount of energy present at the level of secondary consumer is, okay, 10 joules of energy is available at the producer level. So suppose plant is there, plant is eaten by a rabbit is eaten by a tiger, right? So this is going to be your producer, this is going to be your primary consumer, and this is going to be the secondary consumer, right? So over here, I am saying 10 joules of energy is right up over here at the producer level. The amount of energy present at the level of secondary consumer, this level. So 10% of 10 is 1 joule. 10% of 1 is going to be 0.1 joules. So your answer for this question is going to be 0.1 joules. You get it? You have to know about the secondary consumer, primary consumer, and all of it. Cool? Yes. Going ahead, what do we have? Next thing. Question number 6. Given below is a table of factors affecting microbial decomposition. Okay, and plus means favoring and minus means inhibiting or lowering. Select the correct option. So you have to say which one favors microbial decomposition. Decomposition process is going to be helped with the help of the ones mentioned over here, that is denoted by plus. Okay, let's go, then we'll understand. High temperature, do you think it favors decomposition? Here it is written plus, here it is minus minus. I think it is this high temperature actually favors decomposition. Lack of oxygen, does it favor decomposition? It does not. It does not. So this one is the answer. It does not. There should be oxygen. Moist environment, does it favor? Yes, it does. Lignin and chitin and detritus, in the detritus, it does not favor. Lignin and chitin is difficult to decompose, which is why it does not favor. So which is why your answer for this question is going to be B. High temperature you need, lack of oxygen is negative, is going to have a negative impact. Moist temperature is going to have a positive impact. Lignin and chitin are going to have a negative impact. On the other hand, if the detritus is rich in carbon as well as nitrogen, it would have a better impact. Okay? Okay, just a second. Okay, just a second. Here, this one is the answer. This is not. Okay, I'll quickly change. I think that's better. Just a second, okay? Okay. Yes, so I had marked it. I didn't get the color. Just I marked in red. I hope that works, yeah? You will be able to see in the PDF. Now, what type of ecological pyramid would be obtained by the following data? Okay, my voice is going. Secondary consumer 120 gram, primary consumer 60 gram, and primary producer 10 gram. So over here, producer is at 10 gram, primary consumer is at 60 gram, and secondary consumer is at 120 gram. Gram as in you're talking about biomass. And this is, do you think upright pyramid of biomass? No, this is inverted. This is ulta pyramid of biomass, right? Top one has the highest biomass. This is usually seen in the case of oceans. In the case of oceans, my dear children, the producer is phytoplankton. They have very less biomass. And then it is eaten by zooplankton, they have higher biomass. The small fishes have even more higher biomass. So this is inverted pyramid of biomass, my dear children, and this is usually seen in the case of oceans or aquatic things. Okay? Yes. Next question. Assertion. Some aquatic ecosystem has inverted biomass pyramids. We just studied about it right now. Yes, it does. Pyramid of energy is always is also inverted in this case. Energy pyramid can never be inverted. It will be always upright. Energy is going to go less. It cannot be more at the top and can be less at the bottom. No, that is not true at all. So assertion is true, but the reason is false. Assertion is true, but the reason is false. C is your answer. Cool? Cool. Next. The annual net primary productivity of the whole biosphere is estimated approximately. So both of these questions are very related, which is why I put it up. This is there in your question bank. Annual net primary productivity of the whole biosphere. What do you mean by net primary productivity? Listen, gross primary productivity means all the plants producing their things. That is the productivity of all the plants come together. That is GPP, gross primary productivity. Now, obviously it is not available completely to the next organism. Some of it is lost as respiratory losses, as in some of it will utilize it for itself also. That is why that is what I call as respiratory losses. So GPP minus R is equals to NPP, net primary productivity, which is actually available for the next group of organism out there. Okay, over here the annual net primary productivity of the whole biosphere is estimated to be 170 billion tons. This is the actual answer. Now out of that, the annual net primary productivity of ocean is only half of it, and that is basically 55 billion tons. Not even half, it's more, it's lesser than that also. So it will be 55 billion tons. Just a second. Okay, so I was completing this. Please remember this is a very important question. Oceans contribute less. Okay, the terrestrial area is contributing more than oceans over here. So 170 billion, not million, please remember. Here also 55 billion is there, not million. Cool? Cool. Biodiversity and conservation. This is the next chapter that we are having, and with this we end the whole MCQ portion. And I think, my dear children, this video had helped you in some ways. And if at all you have reached up to this particular level, put level 7 crossed, and it goes like this. The IUCN Red List documented the extinction of species in the last 500 years. Include how many species? This type of questions are coming from this chapter. So you have to remember the numbers. It is 784. It is 784 species extincted in the last 500 years. Please write this particular number. And out of that, 338 were vertebrates, 359 were invertebrates, and 87 of them were plants. You can get a next question related to this also. 338 vertebrates, 359 invertebrates, the more number of invertebrates got extincted, and 87 plants. This is the data that you need to remember. Okay, let's try it out once. 338 vertebrates, 359 invertebrates, 87 plants. Okay, total number is 784. 784. They can give 847. Listen, you see the options. You get to understand this, right? Yeah. Next thing. The following are the in situ conservation approaches except. In situ means conserving in their natural habitat. So following are the in situ conservation approaches except. National parks, it is in situ or ex situ? What do you think? In the natural habitat. Yes, it is in situ. Wildlife sanctuary, yes, it is in situ. Biosphere reserves, yes, it is in situ. What about botanical gardens? It is not. You take the plants, you grow it somewhere else in some areas, right? This is botanical gardens. This is ex situ conservation method, not in situ, which is why this one is the odd one out. Got it? Yes, bam. Fine. Next. Alexander van Humboldt observed that within a region, species dash increased with increasing explored area, but up to a certain limit. So basically, he said that when you move on to a different area, if you keep on going on, what is going to happen? The species richness is going to increase. There are more number of species that you're going to see when you explore more area. But this happens only up to a certain limit. And he gave a formula, he gave a graph and everything for that. We will be discussing about that also over here. So here it is species richness that you have to write. Okay, the term that you have to write is species richness, not diversity. You write species richness. Alexander ends with R, so you can remember richness like that. Cool? Yes. Honestly speaking, I'm tired, my legs are shivering now. Okay. The genetic diversity shown by Rauwolfia vomitoria growing in Himalayan ranges produces an active chemical called what? A genetic diversity shown by Rauwolfia vomitoria. So there is a, a species, or basically there is a plant, Rauwolfia vomitoria. Okay. Now, genetically there are different strains of the active chemical that is being produced, and which is why we say that it is an example of genetic diversity that is being shown over here. What is the component that is being produced? It is reserpine. It is what, my dear children? Reserpine. This is very important. It is reserpine. On the basis of how much amount of reserpine is being produced, they are being genetically diversified. Okay, if you have got the confusion, I'll just explain again. Genetic diversity shown by Rauwolfia vomitoria growing in Himalayan ranges produces an active chemical that is known as reserpine. Depending on how much quantity it produces, we say that is genetically diverse in nature. Okay? Yes. Okay. So as per the species inventory, 70% of the species recorded are, as in the most number of species which are there, they are recorded are plants, fungi, bryophytes or animals. What do you think is the answer for this? The largest number of species recorded are animals. They are definitely going to be animals. D is your answer for this particular question. Cool? Very important thing. Okay? Now, in the animals, if at all you are asked, most of them are going to be insects. Insects are the largest group in the animals. Now, always remember, in the case of plants, okay, in the case of plants, they have added fungi also, and they are saying fungi is the one that is having the highest. So in overall, it's the animals. In the animals, it is insects. If at all you're talking about plants, they have added fungi group, and they are saying that fungi is the highest. Okay, fungi is the highest, and yeah, so please remember that. And yes. Now, in general, species diversity dash as we move away from the equator towards the poles. When we move away from the equator towards the poles, what is going to happen? The species diversity is going to decrease. Why? Because poles is not a better environment, does not provide a better environment to survive, for the survival of the organisms. There is frequent glaciation, things like that, which is why the species diversity is going to go less, not high. So I'll say it decreases. It is going to decrease. Cool? Next, the graph given below shows species area relationship. I told you this is Alexander von Humboldt gas. So which of the following equation currently represent the curve? So over here you have the area, and over here you have the species richness. Listen, Alexander von Humboldt said that yes, it is going to keep on growing, but only up to a certain limit. So basically I can not say that S is directly proportional to A. Species richness is actually not directly proportional to the area explored. Okay, it's somewhat it is not. So how do you express that then? You basically remove the proportionality question, you put C over here and you put A, and then you say up to a certain limit that is represented by Z. So S is equals to CAZ. This particular line is representing S is equals to CA to the power Z, this much amount. Okay, then species area, and this is related to this much species richness, and area is related to in this much particular amount. Now, if at all you take the log value of this, then it will be log S is equals to log C plus log A. What do you do with the Z? Z you put it over here. So log S is equals to log C plus log A. Z, which was an exponent over here, you put it over here. And this is represented by a line like this. This is known as the slope of regression. Z represents over here the slope of regression. It means that it shows the relationship. So suppose over here I say that this much is the area. This much is the area that I'm exploring. Okay, or basically let me make two lines. This is one line, this is another line. So here, with this much area explored, the species richness is what? Less. Species richness is what? Less. Now if at all the slope is like this, I can say with this much area explored, the species area richness is this. This slope is usually found in larger areas. So even with less area, you can find more species richness. This is found in larger areas. Mostly, if at all we see this type of slope of regression. Where the area explored is more, the species richness is not that great. So over here, I'll say the Z value is going to be less. This is happening in the smaller areas. This happens in the larger areas. In the larger areas, it goes up to 0.6 from, it goes up to 1.2 is what I remember. Just a second, okay, I'll just check it. 1.2 is the thing. Yes, it was 1.2. So where was I? Yes, here. Are you? Yes. 0.6 to 1.2, this will be the condition when the area is large. This is not the usual one. The usual one has a Z value from 0.1 to 0.2. From 0.1 to 0.2. This is very, very important. Most of the ecologists say that the Z value is from 0.1 to 0.2. Only when the area explored is very large, at that particular time, the Z value goes up from 0.6 to 0.6 to 1.2. Very, very important for you to know. Got it? Here, which of the following equation correctly represents the curve? This curve is represented by S is equals to CAZ. So A is the answer for this particular question. Cool? Now, the given pie diagram represents the proportionate number of species of major taxa of vertebrates. Identify the group A and B. So A is this, B is this. You have to identify. So A is basically fishes, and here you have to just go like farm. It's very easy. F-A-R. This is going to be birds, and my dear children, this is going to be mammals. So in the farm, you have to include B also, that is birds. So what is A over here? It is fishes, and what is B over here? It is already birds. Okay, so A is fishes and B is birds. Your answer for this question is going to be B. Okay. So farm, you write, but in between that you include the birds. So fishes is the one that occupies the most in the case of vertebrates. Now, select the incorrectly matched pair. Tannins are narrowly utilitarian. What is tannin, my dear children? Tannin is a product that you get from the plants, okay? So, when you get products, whenever you are safeguarding the nature, ecosystem, because of the direct benefits of the products which are there or ecological services which are there, you say you are saving them under narrowly utilitarian point of view, okay? Not so ecological services, I'll say the products, directly products. If at all I'm gaining some products from ecosystem, and which is why I'm safeguarding the nature, it is safeguarding under narrowly utilitarian point of view. So this is correct. Tannins, resin, gums, everything I'm getting from the nature, and basically for that I am safeguarding the nature. Now, endemism means species confined to a particular region and not found anywhere else. Is it that? Yes. And these species are known as endemic species. That is also correct. Lungs of the plant is Amazon rainforest. Is it? Yes. Rajasthan is sacred groves. So in Rajasthan, sacred groves that are the last refuges for a large number of rare and threatened plants. In Rajasthan, you have Aravalli Hills, that is a sacred grove, definitely, but it is not the last refuge for a large number of rare and threatened species. Last way to conserve the nature is sacred groves in Rajasthan. Do you believe it? It's not. It's actually Meghalaya. In Meghalaya, sacred groves is the last refuge. Always remember. And Meghalaya, you have Khasi and Chansha Hills. They are the sacred groves. Cool. So this one is the odd one out. Next one. All of the following are included in ex-situ conservation except wildlife safari parks, sacred groves, botanical gardens, seed banks. Which one is included in ex-situ except? So wildlife safari parks, is it ex-situ? Yes. Wildlife national park is different, wildlife safari park is different, okay? Or wildlife sanctuary is different, sorry. Wildlife sanctuary is in situ, wildlife safari park is ex-situ. Secret grooves, my dear children, it is in situ. Why in situ? Because we are conserving organisms in their natural habitat. That is why you call them in situ form of conservation. Basically, I have some religious attachment to a place and I am conserving the nature, but accidentally the organisms who are there, they are also getting conserved. So this is in situ conservation. Botanical gardens ex situ, seed banks also ex situ. So this one is the odd one out because this one is in situ conservation and all of the others are ex situ conservation. Okay. Assertion. The variation in Rauwolfia growing in the different Himalayan ranges might be in terms of potency and concentration of the active chemical, that is rhizopine, that the plant produces. Yes. So there is no difference apart from that. It is just that there are different types of Rauwolfia and we differentiate it on the basis of the different potency and the concentration of the chemical that they are producing, which is known as rhizopine. Correct. Reason. A single species might show high diversity in the genetic level over its distributional range. Yes. In the distributional range, a single species is there, but they are showing different variations, genetic variations, because it is happening in the genetic level, not in the species level. It is happening in the genetic level, which is why different potencies or different concentration of chemicals are being produced over there. So this is correct. In the genetic level, they show variation, not in the species level. Very, very important. So I'll say both assertion and reason is true, and the reason is the correct explanation of assertion. Okay. Next. Assertion. Speciation is a function of time and tropical regions has got a long evolutionary time for species diversification as compared to temperate regions. Do you think so? Has tropical regions got more time than temperate regions? Yes. In temperate regions, there is more frequent glaciation and things like that happening, not providing enough amount of time, which is why I'll say this is correct. Reason. Temperate regions have undergone frequent glaciations in the past, whereas tropical regions have remained relatively undisturbed for millions of years. Yes. Okay, temperate regions have underwent so much frequent glaciation, which is why you cannot allow the organisms to stay up there. Organisms which are there, my dear children, they die because there is frequent changes in the environment up there. But in tropical, it is not like that. So again, the assertion and reason is true, and the reason is the correct explanation of assertion. Okay, next one. Robert May places global species diversity at about. What is the global species diversity, my dear children? It is 7 million. I added this question from the question bank because I thought these type of questions are important for you guys to practice. Robert May places global species diversity at about 7 million, not billion, 7 million. There it was 170 billion tons. That is net primary productivity of the whole thing. Right? Here it is 7 million, not billion. Please know. This is Robert May, global species diversity. Next, the share of global species diversity of India is. The share of global species diversity of India is what? 8.1, 8.5, 9%, 2.4%. It is 8.1%. It is going to be 8.1%. Again, in your NCERT. And so many other terms are also given in your NCERT, which is why I am requesting you read it. Okay? Next one. According to ecologists, the value of Z lies in the range of, as I told you, the smaller region, it is 0.1 to 0.2. In larger areas, it is 0.6 to 1.2. But you have to specifically focus on, most of the ecologists say it is 0.1 to 0.2. Okay, only in the larger areas, it is like this. So, which is why your answer can be picked up. This is a thing that most ecologists say. That is why this one. In frugivorous, in areas where you have frugivorous plants and animals, my dear children, there the Z value goes up to 1.15. Okay, 1.15. In the places where you have frugivorous, animals and other things, the Z value goes up to 1.15. Remember that also. Okay. So this is in the larger area. This is generally in the smaller area, but this is what most ecologists say. Fine. Next thing. The Earth Summit was held in Rio de Janeiro in the year. Earth Summit was the first thing to be held. Then came the World Summit. So Earth Summit was held in Rio de Janeiro. Do not forget that. And it happened in 1992. This one happened in 2002. Both of them 2-2. Okay. This was World Summit and this was on sustainable development. Okay. And it was held in Johannesburg. So first one in Rio de Janeiro in 1992, Earth Summit, then World Summit in the year 2002 in Johannesburg. Very, very important. Cool? Done. And I think that's how, there are a few more things in terms of numbers that you need to know. 34 biodiversity hotspots are there, not 32. In a question bank question, I saw 32. It's wrong. 34 biodiversity hotspots, and these hotspots are rich in endemic organisms, the organisms that you're not finding anywhere else. 3 of these hotspots are Western Ghats in India, okay, Western Ghats and Indo-Burma and Himalayas. Just a second. So Sri Lanka is also being mentioned over here. Just a second, sorry. So Western Ghats and Indo-Burma region and Himalayan region of India are the hotspots. Not know why Sri Lanka has been mentioned over here. And my dear children, although all biodiversity hotspots put together cover less than 2% of the Earth's land area, only 2%. But the number of species they collectively harbor is extremely high, and strict protection of these hotspots could reduce the ongoing mass extinction by about 30%. By how much percentage can it reduce? 30%. Trust me, these type of questions have come. Now, India now has 14 biosphere reserves, 14 biosphere reserves. This is different from biodiversity hotspot, okay? 34 biodiversity hotspot, 14 biosphere reserves, 90 national parks, 448 wildlife sanctuaries. This is in India. This is all over the world. 34 biodiversity hotspots all over the world, 3 of them in India, Western Ghats, Indo-Burma region, and also the Himalayas, okay? And over here, 14 biosphere reserves, 90 national parks, and 448 wildlife sanctuaries out there. Okay, and if at all we, even though the biodiversity hotspots just occupy 2%, but they can reduce the mass extinction by 30%. I know their number, I know it's like ratification, but yes, in this chapter you have to do this.